Introduction. Fantasy always runs like this: a team of us has fought our way into his secret bunker. Okay, it's a fantasy, let's go whole hog. I've single-handedly neutralized his elite guard and have burst into his bunker, my Browning machine gun at the ready. He lunges for his luger. I knock it out of his hand. He lunges for the cyanide pill he keeps to commit suicide rather than be captured. I knock that out of his hand as well. He snarls in rage, attacks with otherworldly strength. We grapple. I manage to gain the upper hand and pin him down and handcuff him. Adolf Hitler, I announce, I arrest you for crimes against humanity. And this is where the Medal of Honor version of the fantasy ends and the imagery darkens. What would I do with Hitler? The viscera becomes so raw that I switch to passive voice in my mind, to get some distance. What should be done with Hitler? It's easy to imagine, once I allow myself. Sever his spine at the neck, leave him paralyzed but with sensation. Take out his eyes with a blunt instrument. Puncture his eardrums, rip out his tongue. Keep him alive, tube-fed, on a respirator. Immobile, unable to speak, to see, to hear, only able to feel. Then inject him with something that will give him a cancer that festers and pustulates in every corner of his body, that will grow and grow until every one of his cells shrieks with agony, till every moment feels like an infinity spent in the fires of hell. That's what should be done with Hitler. That's what I would want done to Hitler. That's what I would do to Hitler. I've had versions of this fantasy since I was a kid. Still do at times. And when I really immerse myself in it, my heart rate quickens, I flush, my fists clench. All those plans for Hitler, the most evil person in history, the soul most deserving of punishment. But there is a big problem. I don't believe in souls or evil, think that the word wicked is most pertinent to a musical, and doubt that punishment should be relevant to criminal justice. But there's a problem with that. In turn, I sure feel like some people should be put to death, yet I oppose the death penalty. I've enjoyed plenty of violent, schlocky movies, despite being in favor of strict gun control. And I sure had fun when, at some kid's birthday party and against various unformed principles in my mind, I played laser tag, shooting at strangers from hiding places. Fun, that is, until some pimply kid zapped me, like a million times, and then snickered at me, which made me feel insecure and unmanly. Yet at the same time, I know most of the lyrics to Down by the Riverside Ain't Gonna Study. War no more, plus when you're supposed to clap your hands. In other words, I have a confused array of feelings and thoughts about violence, aggression, and competition. Just like most humans. To preach from an obvious soapbox, our species has problems with violence. We have the means to create thousands of mushroom clouds, shower heads and subway ventilation systems have carried poison gas, letters have carried anthrax, passenger planes have become weapons, mass rapes can constitute a military strategy, bombs go off in markets, schoolchildren with guns massacre other children. There are neighborhoods where everyone from pizza delivery guys to firefighters fears for their safety. And there are the subtler versions of violence, say a childhood of growing up abused, or the effects on a minority people when the symbols of the majority shout domination and menace. We are always shadowed by the threat of other humans harming us. If that were solely the way things are, violence would be an easy problem to approach intellectually. AIDS, unambiguously bad news, eradicate. Alzheimer's disease, same thing. Schizophrenia, cancer, malnutrition, flesh-eating bacteria, global warming, comets hitting Earth, ditto. The problem, though, is that violence doesn't go on that list. Sometimes we have no problem with it at all. This is a central point of this book: we don't hate violence. We hate and fear the wrong kind of violence, violence in the wrong context. Because violence in the right context is different. We pay good money to watch it in a stadium. We teach our kids to fight back. We feel proud when, in creaky middle age, we manage a dirty hip check in a weekend basketball game. Our conversations are filled with military metaphors. We rally the troops after our ideas get shot down. Our sports teams' names celebrate violence: warriors, Vikings, lions, tigers, and bears. We even think this way about something as cerebral as chess. Kasparov kept pressing for a murderous attack. Toward the end, Kasparov had to oppose threats of violence with more of the same. 1. We build theologies around violence, elect leaders who excel at it, and in the case of so many women, preferentially mate with champions of human combat. When it's the right type of aggression, we love it. It is the ambiguity of violence, that we can pull a trigger as an act of hideous aggression or of self-sacrificing love, that is so challenging. As a result, violence will always be a part of the human experience that is profoundly hard to understand. This book explores the biology of violence, aggression, and competition, the behaviors and the impulses behind them, the acts of individuals, groups, and states, and when these are bad or good things. It is a book about the ways in which humans harm one another. But it is also a book about the ways in which people do the opposite. What does biology teach us about cooperation, affiliation, reconciliation, empathy, and altruism? The book has a number of personal roots. One is that, having had blessedly little personal exposure to violence in my life, the entire phenomenon scares the crap out of me. I think like an academic egghead, believing that if I write enough paragraphs about a scary subject, give enough lectures about it, it will give up and go away quietly. And if everyone took enough classes about the biology of violence and studied hard, we'd all be able to take a nap between the snoozing lion and lamb. Such is the delusional sense of efficacy of a professor. Then there's the other personal root for this book. I am by nature majorly pessimistic. Give me any topic and I'll find a way in which things will fall apart. Or turn out wonderfully and somehow, because of that, be poignant and sad. It's a pain in the butt, especially to people stuck around me. And when I had kids, I realized that I needed to get a hold of this tendency big time. So I looked for evidence that things weren't quite that bad. I started small, practicing on them. Don't cry, a T-Rex would never come and eat you. Of course Nemo's daddy will find him. And as I've learned more about the subject of this book, there's been an unexpected realization. The realms of humans harming one another are neither universal nor inevitable, and we're getting some scientific insights into how to avoid them. My pessimistic self has a hard time admitting this, but there is room for optimism. The approach in this book. Make my living as a combination neurobiologist, someone who studies the brain, and primatologist, someone who studies monkeys and apes. Therefore, this is a book that is rooted in science, specifically biology. And out of that come 3 key points. First, you can't begin to understand things like aggression, competition, cooperation, and empathy without biology. I say this for the benefit of a certain breed of social scientist who finds biology to be irrelevant and a bit ideologically suspect when thinking about human social behavior. But just as important, second, you're just as much up the creek if you rely only on biology. This is said for the benefit of a style of molecular fundamentalist who believes that the social sciences are destined to be consumed by real science. And as a third point, by the time you finish this book, you'll see that it actually makes no sense to distinguish between aspects of a behavior that are biological and those that would be described as, say, psychological or cultural. Utterly intertwined. Understanding the biology of these human behaviors is obviously important. But unfortunately it is hellishly complicated. Point 2. Now, if you were interested in the biology of, say, how migrating birds navigate, or in the mating reflex that occurs in female hamsters when they're ovulating, this would be an easier task. But that's not what we're interested in. Instead, it's human behavior, human social behavior, and in many cases abnormal human social behavior. And it is indeed a mess, a subject involving brain chemistry, hormones, sensory cues, prenatal environment, early experience, genes, both biological and cultural evolution, and ecological pressures, among other things. How are we supposed to make sense of all these factors in thinking about behavior? We tend to use a certain cognitive strategy when dealing with complex, multifaceted phenomena, in that we break down those separate facets into categories, into buckets of explanation. Suppose there's a rooster standing next to you, and there's a chicken across the street. The rooster gives a sexually solicitive gesture that is hot by chicken standards, and she promptly runs over to mate with him. I haven't a clue if this is how it works, but let's just suppose. And thus we have a key behavioral biological question: why did the chicken cross the road? And if you're a neuroendocrinologist, your answer would be because circulating estrogen levels in that chicken worked in a certain part of her brain to make her responsive to this male signaling, and if you're a bioengineer, the answer would be because the long bone in the leg of the chicken forms a fulcrum for her pelvis, or some such thing, allowing her to move forward rapidly, and if you're an evolutionary biologist, You'd say, because over the course of millions of years, chickens that responded to such gestures at a time that they were fertile left more copies of their genes, and thus this is now an innate behavior in chickens, and so on, thinking in categories in differing scientific disciplines of explanation. The goal of this book is to avoid such categorical thinking. Putting facts into nice cleanly demarcated buckets of explanation has its advantages. For example, it can help you remember facts better. But it can wreak havoc on your ability to think about those facts. This is because the boundaries between different categories are often arbitrary, but once some arbitrary boundary exists, we forget that it is arbitrary and get way too impressed with its importance. For example, the visual spectrum is a continuum of wavelengths from violet to red, and it is arbitrary where boundaries are put for different color names. For example, where we see a transition from blue to green. As proof of this, different languages arbitrarily split up the visual spectrum at different points in coming up with the words for different colors. Show someone two roughly similar colors. If the color name, boundary in that person's language happens to fall between the two colors, the person will overestimate the difference between the two. If the colors fall in the same category, the opposite happens. In other words, when you think categorically, you have trouble seeing how similar or different two things are. If you pay lots of attention to where boundaries are, you pay less attention to complete pictures. Thus, the official intellectual goal of this book is to avoid using categorical buckets when thinking about the biology of some of our most complicated behaviors, even more complicated than chickens crossing roads. What's the replacement? A behavior has just occurred. Why did it happen? Your first category of explanation is going to be a neurobiological one. What went on in that person's brain a second before the behavior happened? Now pull out to a slightly larger field of vision, your next category of explanation, a little earlier in time. What sight, sound, or smell in the previous seconds to minutes triggered the nervous system to produce that behavior? On to the next explanatory category. What hormones acted hours to days earlier to change how responsive that individual was to the sensory stimuli that trigger the nervous system to produce the behavior? And by now you've increased your field of vision to be thinking about neurobiology and the sensory world of our environment and short-term endocrinology in trying to explain what happened. And you just keep expanding. What features of the environment in the prior weeks to years changed the structure and function of that person's brain and thus changed how it responded to those hormones and environmental stimuli? Then you go further back to the childhood of the individual, their fetal environment, then their genetic makeup. And then you increase the view to encompass factors larger than that one individual, how has culture shaped the behavior of people living in that individual's group, what ecological factors helped shape that culture, expanding and expanding until considering events umpteen millennia ago and the evolution of that behavior. Okay, so this represents an improvement. It seems like instead of trying to explain all of behavior with a single discipline, e.g., everything can be explained with knowledge about this particular take-your-pick hormone/gene/childhood event, we'll be thinking about a bunch of disciplinary buckets. But something subtler will be done, and this is the most important idea in the book. When you explain a behavior with one of these disciplines, you are implicitly invoking all the disciplines. Any given type of explanation is the end product of the influences that preceded it. It has to work this way. If you say, the behavior occurred because of the release of neurochemical Y in the brain, you are also saying, the behavior occurred because the heavy secretion of hormone X this morning increased the levels of neurochemical Y. You're also saying, the behavior occurred because the environment in which that person was raised made her brain more likely to release neurochemical Y in response to certain types of stimuli. And you're also saying, because of the gene that codes for the particular version of neurochemical Y. And if you've so much as whispered the word gene, you're also saying, and because of the millennia of factors that shaped the evolution of that particular gene. And so on. There are not different disciplinary buckets. Instead, each one is the end product of all the biological influences that came before it and will influence all the factors that follow it. Thus, it is impossible to conclude that a behavior is caused by a gene, a hormone, a childhood trauma, because the second you invoke one type of explanation, you are de facto invoking them all. No buckets. A neurobiological or genetic or developmental explanation for a behavior is just shorthand, an expository convenience for temporarily approaching the whole multifactorial arc from a particular perspective. Pretty impressive, huh? Actually, maybe not. Maybe I'm just pretentiously saying, you have to think complexly about complex things. Wow, what a revelation. And maybe what I've been tacitly setting up is this full of ourselves straw man of boo, we're going to think subtly. We won't get suckered into simplistic answers, not like those chicken crossing the road neurochemists and chicken, evolutionary biologists and chicken psychoanalysts, all living in their own, limited categorical buckets. Obviously, scientists aren't like that. They're smart. They understand that they need to take lots of angles into account. Of necessity, their research may focus on a narrow subject, because there are limits to how much one person can obsess over. But of course they know that their particular categorical bucket isn't the whole story. Maybe yes, maybe no. Consider the following quotes from some card-carrying scientists. The first. Give me a dozen healthy infants well-formed and my own specified world to bring them up in, and I'll guarantee to take any one at random and train him to become any type of specialist. I might select doctor, lawyer, artist, merchant, chief, and yes, even beggar, man, thief, regardless of his talents, penchants, tendencies, abilities, vocations, and race of his ancestors. Point 3. This was John Watson, a founder of behaviorism, writing around 1925. Behaviorism, with its notion that behavior is completely malleable, that it can be shaped into anything in the right environment, dominated American psychology in the mid-20th century, will return to behaviorism and its considerable limitations. The point is that Watson was pathologically caught inside a bucket having to do with the environmental influences on development. I'll guarantee to train him to become any type. Yet we are not all born the same, with the same potential, regardless of how we are trained for. The next quote. Normal psychic life depends upon the good functioning of brain synapses, and mental disorders appear as a result of synaptic derangements. It is necessary to alter these synaptic adjustments, and change the paths chosen by the impulses in their constant passage, so as to modify the corresponding ideas and force thought into different channels. Point 5. Alter synaptic adjustments. Sounds delicate. Yeah, right. These were the words of the Portuguese neurologist Egas Moniz, around the time he was awarded the Nobel Prize in 1949 for his development of frontal leucotomies. Here was an individual pathologically stuck in a bucket having to do with a crude version of the nervous system. Just tweak those microscopic synapses with a big ol' ice pick, as was done once. Leucotomies, later renamed frontal lobotomies, became an assembly line operation. And a final quote. The immensely high reproduction rate in the moral imbecile has long been established. Socially inferior human material is enabled to penetrate and finally to annihilate the healthy nation. The selection for toughness, heroism, social utility must be accomplished by some human institution if mankind, in default of selective factors, is not to be ruined by domestication, induced degeneracy. The racial idea as the basis of our state has already accomplished much in this respect. We must and should rely on the healthy feelings of our best and charge them with the extermination of elements of the population loaded with dregs. Point 6. This was Konrad Lorenz, animal behaviorist, Nobel laureate, co-founder of the field of ethology. Stay tuned, regular on nature TV programs. Point 7. Grandfatherly Konrad, in his Austrian shorts and suspenders, being followed by his imprinted baby geese, was also a rabid Nazi propagandist. Lorenz joined the Nazi Party the instant Austrians were eligible, and joined the party's Office of Race Policy, working to psychologically screen Poles of mixed Polish-German parentage, helping to determine which were sufficiently Germanized to be spared death. Here was a man pathologically mired in an imaginary bucket, related to gross misinterpretations of what genes do. These were not obscure scientists producing fifth-rate science at Podunk U. These were among the most influential scientists of the 20th century. They helped shape who and how we educate and our views on what social ills are fixable and when we shouldn't bother. They enabled the destruction of the brains of people against their will. And they helped implement final solutions for problems that didn't exist. It can be far more than a mere academic matter when a scientist thinks that human behavior can be entirely explained from only one perspective. our lives as animals and our human versatility at being aggressive. Oh, we have a first intellectual challenge, which is to always think in this interdisciplinary way. The second challenge is to make sense of humans as apes, primates, mammals. Oh, that's right, we're a kind of animal. And it will be a challenge to figure out when we're just like other animals, and when we are utterly different. Some of the time we are indeed just like any other animal. When we're scared, we secrete the same hormone as would some subordinate fish getting hassled by a bully. The biology of pleasure involves the same brain chemicals in us as in a capybara. Neurons from humans and brine shrimp work the same way. House two female rats together, and over the course of weeks they will synchronize their reproductive cycles, so that they wind up ovulating within a few hours of each other. Try the same with two human females as reported in some but not all studies, and something similar occurs. It's called the Wellesley effect, first shown with roommates at All Women's Wellesley College. Point 8. And when it comes to violence, we can be just like some other apes. We pummel, we cudgel, we throw rocks, we kill with our bare hands. So some of the time an intellectual challenge is to assimilate how similar we can be to other species. In other cases the challenge is to appreciate how, though human physiology resembles that of other species, we use the physiology in novel ways. We activate the classical physiology of vigilance while watching a scary movie. We activate a stress response when thinking about mortality. We secrete hormones related to nurturing and social bonding, but in response to an adorable baby panda. And this certainly applies to aggression. We use the same muscles as does a male chimp attacking a sexual competitor, but we use them to harm someone because of their ideology. Finally, sometimes the only way to understand our humanness is to consider solely humans, because the things we do are unique. While a few other species have regular non-reproductive sex, we're the only ones to talk afterward about how it was. We construct cultures premised on beliefs concerning the nature of life, and can transmit those beliefs multigenerationally, even between two individuals separated by millennia. Just consider that perennial bestseller, the Bible. Consonant with that, we can harm by doing things as unprecedented as and no more physically taxing than pulling a trigger, or nodding consent, or looking the other way. We can be passive-aggressive, damn with faint praise, cut with scorn, express contempt with patronizing concern. All species are unique, but we are unique in some pretty unique ways. Here are two examples of just how strange and unique humans can be when they go about harming one another and caring for one another. The first example involves, well, my wife. So we're in the minivan, our kids in the back, my wife driving. And this complete jerk cuts us off, almost causing an accident, and in a way that makes it clear that it wasn't distractedness on his part, just sheer selfishness. My wife honks at him, and he flips us off. We're livid, incensed. Asshole, where's the cops when you need them, etc. And suddenly my wife announces that we're going to follow him, make him a little nervous. I'm still furious, but this doesn't strike me as the most prudent thing in the world. Nonetheless, my wife starts trailing him right on his rear. After a few minutes the guy's driving evasively, but my wife's on him. Finally both cars stop at a red light, one that we know is a long one. Another car is stopped in front of the villain. He's not going anywhere. Suddenly my wife grabs something from the front seat divider, opens her door, and says, "Now he's going to be sorry." I rouse myself feebly, honey, do you really think this is such a goo? But she's out of the car, starts pounding on his window. I hurry over just in time to hear my wife say, "If you could do something that mean to another person, you probably need this," in a venomous voice. She then flings something in the window. She returns to the car triumphant, just glorious. "What did you throw in there?" She's not talking yet. The light turns green. There's no one behind us, and we just sit there. The thug's car starts to blink a very sensible turn indicator, makes a slow turn, and heads down a side street into the dark at, like, 5 miles an hour. If it's possible for a car to look ashamed, this car was doing it. "Honey, what did you throw in there, tell me?" She allows herself a small, malicious grin. "A grape lollipop." I was awed by her savage passive-aggressiveness. "You're such a mean, awful human that something must have gone really wrong in your childhood, and maybe this lollipop will help correct that just a little. That guy was going to think twice before screwing with us again." I swelled with pride and love. And the second example, in the mid-1960s, a rightist military coup overthrew the government of Indonesia, instituting the 30-year dictatorship of Suharto known as the New Order. Following the coup, government-sponsored purges of communists, leftists, intellectuals, unionists, and ethnic Chinese left about 500,000 dead. 9. Mass executions, torture, villages torched with inhabitants trapped inside. V.S. Naipaul, in his book Among the Believers, An Islamic Journey, describes hearing rumors while in Indonesia that when a paramilitary group would arrive to exterminate every person in some village, they would, incongruously, bring along a traditional gamelan orchestra. Eventually Naipaul encountered an unrepentant veteran of a massacre, and he asked him about the rumor. Yes, it is true. We would bring along gamelan musicians, singers, flutes, gongs, the whole shebang. Why? Why would you possibly do that? The man looked puzzled, and gave what seemed to him a self-evident answer, well, to make it more beautiful. Bamboo flutes, burning villages, the lollipop ballistics of maternal love. We have our work cut out for us, trying to understand the virtuosity with which we humans harm or care for one another, and how deeply intertwined the biology of the two can be. 1. The behavior. Have our strategy in place. A behavior has occurred, one that is reprehensible, or wonderful, or floating ambiguously in between. What occurred in the prior second that triggered the behavior? This is the province of the nervous system. What occurred in the prior seconds to minutes that triggered the nervous system to produce that behavior? This is the world of sensory stimuli, much of it sensed unconsciously. What occurred in the prior hours to days to change the sensitivity of the nervous system to such stimuli? Acute actions of hormones. And so on all the way back to the evolutionary pressures played out over the prior millions of years that started the ball rolling. So we're set. Except that when approaching this big sprawling mess of a subject, it is kind of incumbent upon you to first define your terms. Which is an unwelcome prospect. Here are some words of central importance to this book: aggression, violence, compassion, empathy, sympathy, competition, cooperation, altruism, envy, schadenfreude, spite, forgiveness, reconciliation, revenge, reciprocity, and, why not, love. Flinging us into definitional quagmires. Why the difficulty? As emphasized in the introduction, one reason is that so many of these terms are the subject of ideological battles over the appropriation and distortions of their meanings. Words pack power, and these definitions are laden with values, often wildly idiosyncratic ones. Here's an example, namely the ways I think about the word competition. A competition, your lab team races the Cambridge group to a discovery, exhilarating but embarrassing to admit to. The competition, playing pickup soccer, fine as long as the best player shifts sides if the score becomes lopsided. See competition, your child's teacher announces a prize for the best outlining your fingers. Thanksgiving turkey drawing, silly and perhaps a red flag if it keeps happening. Maybe complain to the principal. Decompetition, whose deity is more worth killing for? Try to avoid. But the biggest reason for the definitional challenge was emphasized in the introduction. These terms mean different things to scientists living inside different disciplines. Is aggression about thought, emotion, or something done with muscles? Is altruism something that can be studied mathematically in various species, including bacteria, or are we discussing moral development in kids? And implicit in these different perspectives, disciplines have differing tendencies toward lumping and splitting. These scientists believe that behavior, X, consists of 2 different subtypes, whereas those scientists think it comes in 17 flavors. Let's examine this with respect to different types of aggression. 2. Animal behaviorists dichotomize between offensive and defensive aggression, distinguishing between, say, the intruder and the resident of a territory. The biology underlying these two versions differs. Such scientists also distinguish between conspecific aggression, between members of the same species, and fighting off a predator. Meanwhile, criminologists distinguish between impulsive and premeditated aggression. Anthropologists care about differing levels of organization underlying aggression, distinguishing among warfare, clan vendettas, and homicide. Moreover, various disciplines distinguish between aggression that occurs reactively in response to provocation and spontaneous aggression, as well as between hot-blooded, emotional aggression and cold-blooded, instrumental aggression, e.g., I want your spot to build my nest, so scram or I'll peck your eyes out. This isn't personal, though. Point 3. Then there's another version of this isn't personal, targeting someone just because they're weak and you're frustrated, stressed, or pained and need to displace some aggression. Such third-party aggression is ubiquitous. Shock a rat and it's likely to bite the smaller guy nearby. A beta-ranking male baboon loses a fight to the alpha, and he chases the omega male when unemployment rises. So do rates of domestic violence. Depressingly, as will be discussed in Chapter 4, displacement aggression can decrease the perpetrator's stress hormone levels. Giving ulcers can help you avoid getting them. And of course there is the ghastly world of aggression that is neither reactive nor instrumental, but is done for pleasure. Then there are specialized subtypes of aggression, maternal aggression, which often has a distinctive endocrinology. There's the difference between aggression and ritualistic threats of aggression. For example, many primates have lower rates of actual aggression than of ritualized threats, such as displaying their canines. Similarly, aggression in Siamese fighting fish is mostly ritualistic. Getting a definitional handle on the more positive terms isn't easy either. There's empathy versus sympathy, reconciliation versus forgiveness, and altruism versus pathological altruism. 4. For a psychologist, the last term might describe the empathic codependency of enabling a partner's drug use. For a neuroscientist, it describes a consequence of a type of damage to the frontal cortex. In economic games of shifting strategies, individuals with such damage fail to switch to less altruistic play when being repeatedly stabbed in the back by the other player, despite being able to verbalize the other player's strategy. When it comes to the more positive behaviors, the most pervasive issue is one that ultimately transcends semantics. Does pure altruism actually exist? Can you ever separate doing good from the expectation of reciprocity, public acclaim, self-esteem, or the promise of paradise? This plays out in a fascinating realm, as reported in Larissa McFarquhar's 2009 New Yorker piece, The Kindest Cut. 5. It concerns people who donate organs not to family members or close friends but to strangers. An act of seemingly pure altruism. But these Samaritans unnerve everyone, sowing suspicion and skepticism. Is she expecting to get paid secretly for her kidney? Is she that desperate for attention? Will she work her way into the recipient's life and do a fatal attraction? What's her deal? The piece suggests that these profound acts of goodness unnerve because of their detached, affectless nature. This speaks to an important point that runs through the book. As noted, we distinguish between hot-blooded and cold-blooded violence. We understand the former more, can see mitigating factors in it, consider the grieving, raging man who kills the killer of his child. And conversely, affectless violence seems horrifying and incomprehensible. This is the sociopathic contract killer, the Hannibal Lecter who kills without his heart rate nudging up a beat 6. It's why cold-blooded killing is a damning descriptor. Similarly, we expect that our best, most prosocial acts be warm-hearted, filled with positive effect. Cold-blooded goodness seems oxymoronic, is unsettling. I was once at a conference of neuroscientists and all-star Buddhist monk meditators, the former studying what the brains of the latter did during meditation. One scientist asked one of the monks whether he ever stops meditating because his knees hurt from all that cross-leggedness. He answered, "Sometimes I'll stop sooner than I planned, but not because it hurts. It's not something I notice. It's as an act of kindness to my knees." Whoa, I thought, these guys are from another planet. A cool, commendable one, but another planet nonetheless. Crimes of passion and good acts of passion make the most sense to us. Nevertheless, as we shall see, dispassionate kindness often has much to recommend it. Hot-blooded badness, warm-hearted goodness, and the unnerving incongruity of the cold-blooded versions raise a key point encapsulated in a quote from Elie Wiesel, the Nobel Peace Prize winner and concentration camp survivor. The opposite of love is not hate, its opposite is indifference. The biologies of strong love and strong hate are similar in many ways, as we'll see. Which reminds us that we don't hate aggression, we hate the wrong kind of aggression, but love it in the right context. And conversely, in the wrong context, our most laudable behaviors are anything but. The motoric features of our behaviors are less important and challenging to understand than the meaning behind our muscles' actions. This is shown in a subtle study. Point 7. Subjects in a brain scanner entered a virtual room where they encountered either an injured person, in need of help, or a menacing extraterrestrial. Subjects could either bandage or shoot the individual. Pulling a trigger and applying a bandage are different behaviors. But they are similar insofar as bandaging the injured person and shooting the alien are both the right things. And contemplating those two different versions of doing the right thing activated the same circuitry in the most context-savvy part of the brain, the prefrontal cortex. And thus those key terms that anchor this book are most difficult to define because of their profound context dependency. I will therefore group them in a way that reflects this. I won't frame the behaviors to come as either antisocial, too cold-blooded for my expository tastes. Nor will they be labeled as good and evil, too hot-blooded and frothy. Instead, as our convenient shorthand for concepts that truly defy brevity, this book is about the biology of our best and worst behaviors. 2. 1 second before. Various muscles have moved and a behavior has happened. Perhaps it is a good act, you've empathically touched the arm of a suffering person. Perhaps it is a foul act, you've pulled a trigger, targeting an innocent person. Perhaps it is a good act, you've pulled a trigger, drawing fire to save others. Perhaps it is a foul act, you've touched the arm of someone, starting a chain of libidinal events that betray a loved one. Acts that, as emphasized, are definable only by context. Thus, to ask the question that will begin this and the next 8 chapters, why did that behavior occur? As this book's starting point, we know that different disciplines produce different answers, because of some hormone, because of evolution, because of childhood experiences or genes or culture, and as the book's central premise, these are utterly intertwined answers, none standing alone. But on the most proximal level, in this chapter we ask, what happened 1 second before the behavior that caused it to occur? This puts us in the realm of neurobiology of understanding the brain that commanded those muscles. This chapter is one of the book's anchors. The brain is the final common pathway, the conduit that mediates the influences of all the distal factors to be covered in the chapters to come. What happened an hour, a decade, a million years earlier? What happened were factors that impacted the brain and the behavior it produced. This chapter has 2 major challenges. The first is its god-awful length. Apologies, I've tried to be succinct and non-technical, but this is foundational material that needs to be covered. Second, regardless of how non-technical I've tried to be, the material can overwhelm someone with no background in neuroscience. To help with that, please wade through Appendix 1 around now. Now we ask, what crucial things happened in the second before that antisocial behavior occurred? Or, translated into neurobiology, what was going on with action potentials, neurotransmitters and neural circuits in particular brain regions during that second? 3 metaphorical, but not literal, layers. Start by considering the brain's macroorganization, using a model proposed in the 1960s by the neuroscientist Paul MacLean. 1. His triune brain model conceptualizes the brain as having 3 functional domains. Layer 1, an ancient part of the brain at its base, found in species from humans to geckos. This layer mediates automatic, regulatory functions. If body temperature drops, this brain region senses it and commands muscles to shiver. If blood glucose levels plummet, that's sensed here, generating hunger. If an injury occurs, a different loop initiates a stress response. Layer 2, a more recently evolved region that has expanded in mammals. MacLean conceptualized this layer as being about emotions, somewhat of a mammalian invention. If you see something gruesome and terrifying, this layer sends commands down to ancient layer 1, making you shiver with emotion. If you're feeling sadly unloved, regions here prompt layer 1 to generate a craving for comfort food. If you're a rodent and smell a cat, neurons here cause layer 1 to initiate a stress response. Layer 3, the recently evolved layer of neocortex sitting on the upper surface of the brain. Proportionately, primates devote more of their brain to this layer than do other species. Cognition, memory storage, sensory processing, abstractions, philosophy, naval contemplation. Read a scary passage of a book and layer 3 signals layer 2 to make you feel frightened, prompting layer 1 to initiate shivering. See an ad for Oreos and feel a craving. Layer 3 influences layers 2 and 1. Contemplate the fact that loved ones won't live forever, or kids in refugee camps, or how the Navis home tree was destroyed by those jerk humans in Avatar, despite the fact that, wait, Navi aren't real. And layer 3 pulls layers 2 and 1 into the picture, and you feel sad and have the same sort of stress response that you'd have if you were fleeing a lion. Thus we've got the brain divided into three functional buckets, with the usual advantages and disadvantages of categorizing a continuum. The biggest disadvantage is how simplistic this is. For example, A. Anatomically there is considerable overlap among the three layers. For example, one part of the cortex can best be thought of as part of layer 2, stay tuned. B. The flow of information and commands is not just top-down, from layer 3 to 2 to 1. A weird, great example explored in Chapter 15, if someone is holding a cold drink, temperature is processed in layer 1, they're more likely to judge someone they meet as having a cold personality, layer 3. C. Automatic aspects of behavior, simplistically, the purview of layer 1, emotion, layer 2, and thought, layer 3, are not separable. D. The Trian model leads one, erroneously, to think that evolution in effect slapped on each new layer without any changes occurring in the 1s already there. Despite these drawbacks, which MacLean himself emphasized, this model will be a good organizing metaphor for us. T. The limbic system. O makes sense of our best and worst behaviors. Automaticity, emotion, and cognition must all be considered. I arbitrarily start with layer 2 and its emphasis on emotion. Early 20th century neuroscientists thought it obvious what layer 2 did. Take your standard issue lab animal, a rat, and examine its brain. Right at the front would be these two gigantic lobes, the olfactory bulbs, one for each nostril, the primary receptive area for odors. Neuroscientists at the time asked what parts of the brain these gigantic rodent olfactory bulbs talked to, i.e., where they sent their axonal projections. Which brain regions were only a single synapse away from receiving olfactory information, which were 2 synapses, 3, and so on. And it was layer 2 structures that received the first communiques. Everyone concluded, this part of the brain must process odors, and so it was termed the rhinencephalon, the nose brain. Meanwhile, in the 30s and 40s, neuroscientists such as the young MacLean, James Papez, Paul Bussey, and Heinrich Kluver were starting to figure out what the layer 2 structures did. For example, if you lesion, i.e., destroy, layer 2 structures, this produces Kluver-Bussey syndrome, featuring abnormalities in sociality, especially in sexual and aggressive behaviors. They concluded that these structures, soon termed the limbic system for obscure reasons, were about emotion. Rhinencephalon, or limbic system. Olfaction, or emotion. Pitched street battles ensued until someone pointed out the obvious. For a rat, emotion and olfaction are nearly synonymous, since nearly all the environmental stimuli that elicit emotions in a rodent are olfactory. Peace in our time. In a rodent, olfactory inputs are what the limbic system most depends on for emotional news of the world. In contrast, the primate limbic system is more informed by visual inputs. Limbic function is now recognized as central to the emotions that fuel our best and worst behaviors, and extensive research has uncovered the functions of its structures, e.g., the amygdala, hippocampus, septum, habenula, and mammillary bodies. There really aren't centers in the brain for particular behaviors. This is particularly the case with the limbic system and emotion. There is indeed a sub-subregion of the motor cortex that approximates being the center for making your left pinky bend. Other regions have center-ish roles in regulating breathing or body temperature. But there sure aren't centers for feeling pissy or horny, for feeling bittersweet nostalgia or warm protectiveness tinged with contempt, or for that what is that thing called love feeling. No surprise, then, that the circuitry connecting various limbic structures is immensely complex. The autonomic nervous system and the ancient core regions of the brain. The limbic system's regions form complex circuits of excitation and inhibition. It's easier to understand this by appreciating the deeply held desire of every limbic structure to influence what the hypothalamus does. Why? Because of its importance. The hypothalamus, a limbic structure, is the interface between layers 1 and 2, between core regulatory and emotional parts of the brain. Consistent with that, the hypothalamus gets massive inputs from limbic layer 2 structures, but disproportionately sends projections to layer 1 regions. These are the evolutionarily ancient midbrain and brainstem, which regulate automatic reactions throughout the body. For a reptile, such automatic regulation is straightforward. If muscles are working hard, this is sensed by neurons throughout the body that send signals up the spine to layer 1 regions, resulting in signals back down the spine that increase heart rate and blood pressure. The result is more oxygen and glucose for the muscles. gorge on food and stomach walls distend, neurons embedded their sense this and pass on the news, and soon blood vessels in the gut dilate, increasing blood flow and facilitating digestion. Too warm. Blood is sent to the body's surface to dissipate heat. All of this is automatic, or autonomic. And thus the midbrain and brainstem regions, along with their projections down the spine and out to the body, are collectively termed the autonomic nervous system. And where does the hypothalamus come in? It's the means by which the limbic system influences autonomic function, how layer 2 talks to layer 1. Have a full bladder with its muscle walls distended and midbrain/brain stem circuitry votes for urinating. Be exposed to something sufficiently terrifying, and limbic structures, via the hypothalamus, persuade the midbrain and brain stem to do the same. This is how emotions change bodily functions, why limbic roads eventually lead to the hypothalamus. The autonomic nervous system has two parts, the sympathetic and parasympathetic nervous systems, with fairly opposite functions. The sympathetic nervous system, SNS, mediates the body's response to arousing circumstances, for example, producing the famed fight or flight, stress response. To use the feeble joke told to first-year medical students, the SNS mediates the 4Fs, fear, fight, flight, and sex. Particular midbrain/brain stem nuclei send long SNS projections down the spine and onto outposts throughout the body, where the axon terminals release the neurotransmitter norepinephrine. There's one exception that makes the SNS more familiar. In the adrenal gland, instead of norepinephrine, aka noradrenaline, being released, it's epinephrine, aka the famous adrenaline. Meanwhile, the parasympathetic nervous system, PNS, arises from different midbrain/brain stem nuclei that project down the spine to the body. In contrast to the SNS and the 4Fs, the PNS is about calm, vegetative states. The SNS speeds up the heart, the PNS slows it down. The PNS promotes digestion, the SNS inhibits it, which makes sense. If you're running for your life, avoiding being someone's lunch, don't waste energy digesting breakfast. And as we will see chapter 14, if seeing someone in pain activates your SNS, you're likely to be preoccupied with your own distress instead of helping. Turn on the PNS and it's the opposite. Given that the SNS and PNS do opposite things, the PNS is obviously going to be releasing a different neurotransmitter from its axon terminals, acetylcholine. There is a second, equally important way in which emotion influences the body. Specifically, the hypothalamus also regulates the release of many hormones. This is covered in chapter 4. So the limbic system indirectly regulates autonomic function and hormone release. What does this have to do with behavior? Plenty, because the autonomic and hormonal states of the body feed back to the brain, influencing behavior, typically unconsciously. Stay tuned for more in chapters 3 and 4. The interface between the limbic system and the cortex. Time to add the cortex. As noted, this is the brain's upper surface. Its name comes from the Latin cortic, meaning tree bark, and is the newest part of the brain. The cortex is the gleaming, logical, analytical crown jewel of layer 3. Most sensory information flows there to be decoded. It's where muscles are commanded to move, where language is comprehended and produced, where memories are stored, where spatial and mathematical skills reside, where executive decisions are made. It floats above the limbic system, supporting philosophers since at least Descartes who have emphasized the dichotomy between thought and emotion. Of course, that's all wrong as shown by the temperature of a cup, something processed in the hypothalamus altering assessment of the coldness of someone's personality. Emotions filter the nature and accuracy of what is remembered. Stroke damage to certain cortical regions blocks the ability to speak. Some sufferers reroute the cerebral world of speech through emotive, limbic detours. They can sing what they want to say. The cortex and limbic system are not separate, as scads of axonal projections course between the two. Crucially, those projections are bidirectional. The limbic system talks to the cortex, rather than merely being reined in by it. The false dichotomy between thought and feeling is presented in the classic Descartes error by the neurologist Antonio Damasio of the University of Southern California. His work is discussed later. Point 2. While the hypothalamus dwells at the interface of layers 1 and 2, it is the incredibly interesting frontal cortex that is the interface between layers 2 and 3. Key insight into the frontal cortex was provided in the 1960s by a giant of neuroscience, Wal Nada of MIT. Nada studied what brain regions sent axons to the frontal cortex and what regions got axons from it. and the frontal cortex was bidirectionally enmeshed with the limbic system, leading him to propose that the frontal cortex is a quasi-member of the limbic system. Naturally, everyone thought him daft. The frontal cortex was the most recently evolved part of the very highbrow cortex. The only reason why the frontal cortex would ever go slumming into the limbic system would be to preach honest labor and Christian temperance to the urchins there. Naturally, Nata was right. In different circumstances, the frontal cortex and limbic system stimulate or inhibit each other, collaborate and coordinate, or bicker and work at cross purposes. It really is an honorary member of the limbic system. And the interactions between the frontal cortex and other limbic structures are at the core of much of this book. 2 more details. First, the cortex is not a smooth surface, but instead is folded into convolutions. The convolutions form a superstructure of 4 separate lobes, the temporal, parietal, occipital, and frontal, each with different functions. Brain lateralization. Analytical thought, detail-oriented perception. Ordered. Sequencing. Rational thought, verbal. Cautious. Planning. Math/science logic. Right field vision, right side motor skills. Intuitive thought, holistic perception, random. Sequencing. Emotional thought, nonverbal. Adventurous impulse. Creative. Writing/art. Imagination. Left field vision, left side motor skills. Second, brains obviously have left and right sides, or hemispheres, that roughly mirror each other. Thus, except for the relatively few midline structures, brain regions come in pairs, a left and right amygdala, hippocampus, temporal lobe, and so on. Functions are often lateralized, such that the left and right hippocampi, for example, have different but related functions. The greatest lateralization occurs in the cortex. The left hemisphere is analytical, the right more involved in intuition and creativity. These contrasts have caught the public fancy, with cortical lateralization exaggerated by many to an absurd extent, where left brain edness has the connotation of anal, retentive bean counting, and right brain edness is about making mandalas or singing with whales. In fact, the functional differences between the hemispheres are generally subtle, and I'm mostly ignoring lateralization. We're now ready to examine the brain regions most central to this book, namely the amygdala, the frontal cortex, and the mesolimbic mesocortical dopamine system. Discussion of other bit player regions will be subsumed under the headings for these 3. We start with the one arguably most central to our worst behaviors. The amygdala. The amygdala is the archetypal limbic structure, sitting under the cortex in the temporal lobe. It is central to mediating aggression, along with other behaviors that tell us tons about aggression. A first pass at the amygdala and aggression. The evidence for the amygdala's role in aggression is extensive, based on research approaches that will become familiar. First, there's the correlative recording approach. Stick recording electrodes into numerous species amygdala and see when neurons there have action potentials. This turns out to be when the animal is being aggressive. In a related approach, determine which brain regions consume extra oxygen or glucose, or synthesize certain activity-related proteins during aggression. The amygdala tops the list. Moving beyond mere correlation, if you lesion the amygdala in an animal, rates of aggression decline. The same occurs transiently when you temporarily silence the amygdala by injecting Novocaine into it. Conversely, implanting electrodes that stimulate neurons there or spritzing in excitatory neurotransmitters, stay tuned, triggers aggression. Point 4. Show human subjects pictures that provoke anger, and the amygdala activates as shown with neuroimaging. Sticking an electrode in someone's amygdala and stimulating it, as is done before certain types of neurosurgery, produces rage. The most convincing data concern rare humans with damage restricted to the amygdala, either due to a type of encephalitis or a congenital disorder, called Urbach-Witt disease, or where the amygdala was surgically destroyed to control severe, drug-resistant seizures originating there. Point 5. Such individuals are impaired in detecting angry facial expressions, while being fine at recognizing other emotional states. Stay tuned. And what does amygdala damage do to aggressive behavior? This was studied in humans where amygdalotomies were done, not to control seizures but to control aggression. Such psychosurgery provoked fiery controversy in the 1970s. And I don't mean scientists not saying hello to each other at conferences. I mean a major public shitstorm. The issue raised bioethical lightning rods: what counted as pathological aggression? Who decided? What other interventions had been tried unsuccessfully? Were some types of hyperaggressive individuals more likely to go under the knife than others? What constituted a cure 6? Most of these cases concerned rare epileptics where seizure onset was associated with uncontrollable aggression, and where the goal was to contain that behavior. These papers had titles, such as Clinical and Physiological Effects of Stereotaxic Bilateral Amygdalotomy for Intractable Aggression. The fecal hurricane concerned the involuntary lopping out of the amygdala in people without epilepsy, but with a history of severe aggression. Well, doing this could be profoundly helpful. Or Orwellian. This is a long, dark story, and I will save it for another time. Did destruction of the human amygdala lessen aggression? Pretty clearly so, when violence was a reflexive, inchoate outburst preceding a seizure. But with surgery done solely to control behavior, the answer is, maybe. The heterogeneity of patients and surgical approaches, the lack of modern neuroimaging to pinpoint exactly which parts of the amygdala were destroyed in each individual, and the imprecision in the behavioral data, with papers reporting from 33 to 100% success rates, make things inconclusive. The procedure has almost entirely fallen out of practice. The amygdala aggression link pops up in two notorious cases of violence. The first concerns Ulrich Meinhof, a founder in 1968 of the Red Army faction, aka the Baader-Meinhof Gang, a terrorist group responsible for bombings and bank robberies in West Germany. Meinhof had a conventional earlier life as a journalist before becoming violently radicalized. During her 1976 murder trial, she was found hanged in her jail cell, suicide or murder. Still unclear. In 1962, Meinhof had had a benign brain tumor surgically removed. The 1976 autopsy showed that remnants of the tumor and surgical scar tissue impinged on her amygdala. 7. A second case concerns Charles Whitman, the 1966 Texas Tower sniper who, after killing his wife and mother, opened fire atop a tower at the University of Texas in Austin, killing 16 and wounding 32, one of the first school massacres. Whitman was literally an Eagle Scout and childhood choir boy, a happily married engineering major with an IQ in the 99th percentile. In the prior year, he had seen doctors complaining of severe headaches and violent impulses, e.g., to shoot people from the campus tower. He left notes by the bodies of his wife and his mother, proclaiming love and puzzlement at his actions. I cannot rationally pinpoint any specific reason for killing her, and let there be no doubt in your mind that I love this woman with all my heart. His suicide note requested an autopsy of his brain, and that any money he had be given to a mental health foundation. The autopsy proved his intuition correct. Whitman had a glioblastoma tumor, pressing on his amygdala. Did Whitman's tumor cause his violence? Probably not in a strict amygdaloid tumor equals murderer sense, as he had risk factors that interacted with his neurological issues. Whitman grew up being beaten by his father and watching his mother and siblings experience the same. This choir boy Eagle Scout had repeatedly physically abused his wife and had been court-martialed as a Marine for physically threatening another soldier, and, perhaps indicative of a thread running through the family, his brother was murdered at age 24 during a bar fight. 8. A whole other domain of amygdaloid function to the center stage. Thus considerable evidence implicates the amygdala in aggression. But if you asked amygdala experts what behavior their favorite brain structure brings to mind, aggression wouldn't top their list. It would be fear and anxiety. 9. Crucially, the brain region most involved in feeling afraid and anxious is most involved in generating aggression. The amygdala fear link is based on evidence similar to that supporting the amygdala aggression link. 10. In lab animals, this has involved lesioning the structure, detecting activity in its neurons with recording electrodes, electrically stimulating it, or manipulating genes in it. All suggest a key role for the amygdala in perceiving fear-provoking stimuli and in expressing fear. Moreover, fear activates the amygdala in humans, with more activation predicting more behavioral signs of fear. In one study, subjects in a brain scanner played a Ms. Pac-Man from Hell video game where they were pursued in a maze by a dot. If caught, they'd be shocked. 11. When people were evading the dot, the amygdala was silent. However, its activity increased as the dot approached. The stronger the shocks, the farther away the dot would be when first activating the amygdala, the stronger the activation, and the larger the self-reported feeling of panic. In another study, subjects waited an unknown length of time to receive a shock. 12. This lack of predictability and control was so aversive that many chose to receive a stronger shock immediately. And in the others, the period of anticipatory dread increasingly activated the amygdala. Thus the human amygdala preferentially responds to fear-evoking stimuli, even stimuli so fleeting as to be below conscious detection. Powerful support for an amygdaloid role in fear processing comes from post-traumatic stress disorder, PTSD. In PTSD sufferers, the amygdala is overreactive to mildly fearful stimuli, and is slow in calming down after being activated. 13. Moreover, the amygdala expands in size with long-term PTSD. This role of stress in this expansion will be covered in Chapter 4. The amygdala is also involved in the expression of anxiety. 14. Take a deck of cards, half are black, half are red. How much would you wager that the top card is red? That's about risk. Here's a deck of cards, at least one is black, at least one is red. How much would you wager that the top card is red? That's about ambiguity. The circumstances carry identical probabilities, but people are made more anxious by the second scenario and activate the amygdala more. The amygdala is particularly sensitive to unsettling circumstances that are social. A high-ranking male rhesus monkey is in a sexual consortship with a female. In one condition, the female is placed in another room where the male can see her. In the second, she's in the other room, along with a rival of the male. No surprise, that situation activates the amygdala. Is that about aggression or anxiety? Seemingly the latter. The extent of activation did not correlate with the amount of aggressive behaviors and vocalizations the male made, or the amount of testosterone secreted. Instead, it correlated with the extent of anxiety displayed, e.g., teeth chattering or self-scratching. The amygdala is linked to social uncertainty in other ways. In one neuroimaging study, a subject would participate in a competitive game against a group of other players. Outcomes were rigged so that the subject would wind up in the middle of the rankings. 15. Experimenters then manipulated game outcomes so that subjects' rankings either remained stable or fluctuated wildly. Stable rankings activated parts of the frontal cortex that we'll soon consider. Instability activated the frontal cortex plus the amygdala. Being unsure of your place is unsettling. Another study explored the neurobiology of conforming. 16. To simplify, a subject is part of a group where, secretly, the rest are confederates. They are shown X, then asked, "What did you see?" Everyone else says Y. Does the subject lie and say Y also? Often. Subjects who stuck to their guns with X showed amygdala activation. Finally, activating specific circuits within the amygdala in mice turns anxiety on and off. Activating others made mice unable to distinguish between safe and anxiety-producing settings. 17. The amygdala also helps mediate both innate and learned fear. 18. The core of innate fear, aka phobia, is that you don't have to learn by trial and error that something is aversive. For example, a rat born in a lab who has interacted only with other rats and grad students instinctually fears and avoids the smell of cats. While different phobias activate somewhat different brain circuitry, for example, dentist phobia involves the cortex more than does snake phobia, they all activate the amygdala. Such innate fear contrasts with things we learn to fear: a bad neighborhood, a letter from the IRS. The dichotomy between innate and learned fear is actually a bit fuzzy. 19. Everyone knows that humans are innately afraid of snakes and spiders. But some people keep them as pets, give them cute names instead of inevitable fear. We show prepared learning to be afraid of snakes and spiders more readily than of pandas or beagles. The same occurs in other primates. For example, lab monkeys who have never encountered snakes or artificial flowers can be conditioned to fear the former more readily than the latter. As we'll see in the next chapter, humans show prepared learning, being predisposed to be conditioned to fear people with a certain type of appearance. The fuzzy distinction between innate and learned fear maps nicely onto the amygdala's structure. The evolutionarily ancient central amygdala plays a key role in innate fears. Surrounding it is the basilateral amygdala, BLA, which is more recently evolved and somewhat resembles the fancy, modern cortex, It's the blah that learns fear and then sends the news to the central amygdala. Joseph Ledoux at New York University has shown how the blah learns fear. 20 Expose a rat to an innate trigger of fear, a shock. When this unconditioned stimulus occurs, the central amygdala activates, stress hormones are secreted, the sympathetic nervous system mobilizes, and as a clear endpoint, the rat freezes in place. What was that? What do I do? Now do some conditioning. Before each shock, expose the rat to a stimulus that normally does not evoke fear, such as a tone. And with repeated coupling of the tone, the conditioned stimulus with the shock, the unconditioned one, fear conditioning occurs, the sound of the tone alone elicits freezing, stress hormone release, and so on. Ledoux and others have shown how auditory information about the tone stimulates blah neurons. At first, activation of those neurons is irrelevant to the central amygdala, whose neurons are destined to activate following the shock. But with repeated coupling of tone with shock, there is remapping and those blah neurons acquire the means to activate the central amygdala. Blah neurons that respond to the tone only once conditioning has occurred would also have responded if conditioning instead had been to a light. In other words, these neurons respond to the meaning of the stimulus, rather than to its specific modality. Moreover, if you electrically stimulate them, rats are easier to fear condition. You've lowered the threshold for this association to be made. And if you electrically stimulate the auditory sensory input at the same time as shocks, i.e., there's no tone, just activation of the pathway that normally carries news of the tone to the amygdala, you cause fear conditioning to a tone. You've engineered the learning of a false fear. There are synaptic changes as well. Once conditioning to a tone has occurred, the synapses coupling the blah and central nucleus neurons have become more excitable. How this occurs is understood at the level of changes in the amount of receptors for excitatory neurotransmitters in dendritic spines in these circuits. Furthermore, conditioning increases levels of growth factors, which prompt the growth of new connections between blah and central amygdala neurons. Some of the genes involved have even been identified. We've now got learning to be afraid under our belts. 21 Now conditions change. The tone still occurs now and then, but no more shock. Gradually the conditioned fear response abates. How does fear extinction occur? How do we learn that this person wasn't so scary after all? That different doesn't necessarily equal frightening. Recall how a subset of blah neurons respond to the tone only once conditioning has occurred. Another population does the opposite, responding to the tone, once it's no longer signaling shock. Logically, the two populations of neurons inhibit each other. Where do these the tone isn't scary anymore, neurons get inputs from? The frontal cortex. When we stop fearing something, it isn't because some amygdaloid neurons have lost their excitability. We don't passively forget that something is scary. We actively learn that it isn't anymore. The amygdala also plays a logical role in social and emotional decision-making. In the Ultimatum Game, an economic game involving two players, the first makes an offer as to how to divide a pot of money, which the other player either accepts or rejects. 22. If the latter, neither gets anything. Research shows that rejecting an offer is an emotional decision, triggered by anger at a lousy offer and the desire to punish. The more the amygdala activation in the second player after an offer, the more likely the rejection. People with damaged amygdala are atypically generous in the Ultimatum Game and don't increase rejection rates if they start receiving unfair offers. Why? These individuals understand the rules and can give sound, strategic advice to other players. Moreover, they use the same strategies as control subjects in a non-social version of the game when believing the other player is a computer. And they don't have a particularly long view, undistracted by the amygdala's emotional tumult, reasoning that their non-contingent generosity will induce reciprocity and pay off in the long run. When asked, they anticipate the same levels of reciprocity as do controls. Instead, these findings suggest that the amygdala injects implicit distrust and vigilance into social decision-making. 23. All thanks to learning. In the words of the authors of the study, the generosity in the trust game of our blah damaged subjects might be considered pathological altruism in the sense that inborn altruistic behaviors have not, due to BLA damage, been unlearned through negative social experience. In other words, the default state is to trust, and what the amygdala does is learn vigilance and distrust. Unexpectedly, The amygdala and one of its hypothalamic targets also play a role in male sexual motivation. Other hypothalamic nuclei are central to male sexual performance but not female. What's that about? One neuroimaging study shed some light. Young heterosexual men looked at pictures of attractive women versus as a control of attractive men. Passively observing the pictures activated the reward circuitry just alluded to. In contrast, working to see the pictures by repeatedly pressing a button also activated the amygdala. Similarly, other studies show that the amygdala is most responsive to positive stimuli when the value of the reward is shifting. Moreover, some BLA neurons that respond in that circumstance also respond when the severity of something aversive is shifting. These neurons are paying attention to change independent of direction. For them, the amount of reward is changing, and the amount of punishment is changing are the same. Studies like these clarify that the amygdala isn't about the pleasure of experiencing pleasure. It's about the uncertain, unsettled yearning for a potential pleasure, the anxiety and fear and anger that the reward may be smaller than anticipated, or may not even happen. It's about how many of our pleasures and our pursuits of them contain a corrosive vein of disease. 24. The amygdala as part of networks in the brain. Now that we know about the subparts of the amygdala, it's informative to consider its extrinsic connections, i.e., what parts of the brain send projection to it, and what parts does it project to. 25. Some inputs to the amygdala. Sensory inputs. For starters, the amygdala, specifically the BLA, gets projections from all the sensory systems. 26. How else can you get terrified by the shark's theme music in Jaws? Normally, sensory information from various modalities, eyes, ears, skin, courses into the brain, reaching the appropriate cortical region, visual cortex, auditory cortex, tactile cortex, for processing. For example, the visual cortex would engage layers and layers of neurons to turn pixels of retinal stimulation into recognizable images before it can scream to the amygdala, "It's a gun." Importantly, some sensory information entering the brain takes a shortcut, bypassing the cortex and going directly to the amygdala. Thus the amygdala can be informed about something scary before the cortex has a clue. Moreover, thanks to the extreme excitability of this pathway, the amygdala can respond to stimuli that are too fleeting or faint for the cortex to note. Additionally, the shortcut projections form stronger, more excitable synapses in the BLA than do the ones from the sensory cortex. Emotional arousal enhances fear conditioning through this pathway. This shortcut's power is shown in the case of a man with stroke damage to his visual cortex, producing cortical blindness. While unable to process most visual information, he still recognized emotional facial expressions via the shortcut. Crucially, while sensory information reaches the amygdala rapidly by this shortcut, it isn't terribly accurate, since, after all, accuracy is what the cortex supplies. As we'll see in the next chapter, this produces tragic circumstances where, say, the amygdala decides it's seeing a handgun before the visual cortex can report that it's actually a cell phone. Information about pain. The amygdala receives news of that reliable trigger of fear and aggression, namely pain. 27. This is mediated by projections from an ancient, core brain structure, the periaqueductal gray PAG. Stimulation of the PAG can evoke panic attacks, and it is enlarged in people with chronic panic attacks. Reflecting the amygdala's roles in vigilance, uncertainty, anxiety, and fear, it's unpredictable pain, rather than pain itself, that activates the amygdala. Pain, and the amygdala's response to it, is all about context. Disgust of all stripes. The amygdala also receives a hugely interesting projection from the insular cortex, an honorary part of the prefrontal cortex, which we will consider at length in later chapters. 28. If you, or any other mammal, bite into rancid food, the insular cortex lights up, causing you to spit it out, gag, feel nauseated, make a revolted facial expression. The insular cortex processes gustatory disgust. Ditto for disgusting smells. Remarkably, humans also activate it by thinking about something morally disgusting, social norm violations, or individuals who are typically stigmatized in society. And in that circumstance its activation drives that of the amygdala. Someone does something lousy and selfish to you in a game, and the extent of insular and amygdaloid activation predicts how much outrage you feel and how much revenge you take. This is all about sociality. The insula and amygdala don't activate if it's a computer that has stabbed you in the back. The insula activates when we eat a cockroach or imagine doing so. And the insula and amygdala activate when we think of the neighboring tribe as loathsome cockroaches. As we'll see, this is central to how our brains process us and them. And finally, the amygdala gets tons of inputs from the frontal cortex. Much more to come. Some outputs from the amygdala. Bi-directional connections. As we'll see, the amygdala talks to many of the regions that talk to it, including the frontal cortex, insula, periaqueductal gray, and sensory projections, modulating their sensitivity. The amygdala-hippocampus interface. Naturally, the amygdala talks to other limbic structures, including the hippocampus. As reviewed, typically the amygdala learns fear and the hippocampus learns detached, dispassionate facts. But at times of extreme fear, the amygdala pulls the hippocampus into a type of fear, learning. 29. Back to the rat undergoing fear conditioning. When it's in cage A, a tone is followed by a shock. But in cage B, the tone isn't. This produces context-dependent conditioning. The tone causes fearful, freezing in cage A but not in cage B. The amygdala learns the stimulus cue, the tone, while the hippocampus learns about the contexts of cage A versus B. The coupled learning between amygdala and hippocampus is very focalized. We all remember the view of the plane hitting the Second World Trade Center tower, but not whether there were clouds in the background. The hippocampus decides whether a factoid is worth filing away, depending on whether the amygdala has gotten worked up over it. Moreover, the coupling can rescale. Suppose someone robs you at gunpoint in an alley in a bad part of town. Afterward, depending on the circumstance, the gun can be the cue and the alley the context, or the alley is the cue and the bad part of town, the context. Motor outputs. There's a second shortcut, regarding the amygdala, specifically when it's talking to motor neurons that command movement. 30. Logically, when the amygdala wants to mobilize a behavior, say, fleeing, it talks to the frontal cortex, seeking its executive approval. But if sufficiently aroused, the amygdala talks directly to subcortical, reflexive motor pathways. Again, there's a trade-off: increased speed by bypassing the cortex, but decreased accuracy. Thus the input shortcut may prompt you to see the cell phone as a gun. And the output shortcut may prompt you to pull a trigger before you consciously mean to. Arousal. Ultimately, amygdala outputs are mostly about setting off alarms throughout the brain and body. As we saw, the core of the amygdala is the central amygdala. 31. Axonal projections from there go to an amygdala-ish structure nearby called the bed nucleus of the striatum terminalis, BNST. The BNST, in turn, projects to parts of the hypothalamus that initiate the hormonal stress response, see Chapter 4, as well as to midbrain and brainstem sites that activate the sympathetic nervous system and inhibit the parasympathetic nervous system. Something emotionally arousing occurs. Layer 2 limbic amygdala signals, Layer 1 regions, and heart rate and blood pressure soar. The amygdala also activates a brainstem structure called the locus coeruleus, akin to the brain's own sympathetic nervous system. 32. It sends norepinephrine-releasing projections throughout the brain, particularly the cortex. If the locus coeruleus is drowsy and silent, so are you. If it's moderately activated, you're alert. And if it's firing like gangbusters, thanks to inputs from an aroused amygdala, all neuronal hands are on deck. The amygdala's projection pattern raises an important point. 33. When is the sympathetic nervous system going full blast? During fear, flight, fight, and sex. Or if you've won the lottery, are happily sprinting down a soccer field, or have just solved Fermat's theorem, if you're that kind of person. Reflecting this, about a quarter of neurons in one hypothalamic nucleus are involved in both sexual behavior and, when stimulated at a higher intensity, aggressive behavior in male mice. This has two implications. Both sex and aggression activate the sympathetic nervous system, which in turn can influence behavior. People feel differently about things if, say, their heart is racing versus beating slowly. Does this mean that the pattern of your autonomic arousal influences what you feel? Not really. But autonomic feedback influences the intensity of what is felt. More on this in the next chapter. The second consequence reflects a core idea of this book. Your heart does roughly the same thing whether you are in a murderous rage or having an orgasm. Again, the opposite of love is not hate, it's indifference. This concludes our overview of the amygdala. Amid the jargon and complexity, the most important theme is the amygdala's dual role in both aggression and facets of fear and anxiety. Fear and aggression are not inevitably intertwined, not all fear causes aggression, and not all aggression is rooted in fear. Fear typically increases aggression only in those already prone to it. Among the subordinate who lack the option of expressing aggression safely, fear does the opposite. The dissociation between fear and aggression is evident in violent psychopaths, who are the antithesis of fearful. Both physiologically and subjectively they are less reactive to pain. Their amygdala are relatively unresponsive to typical fear-evoking stimuli and are smaller than normal. 34. This fits with the picture of psychopathic violence. It is not done in aroused reaction to provocation. Instead, it is purely instrumental, using others as a means to an end with emotionless, remorseless, reptilian indifference. Thus, fear and violence are not always connected at the hip. But a connection is likely when the aggression evoked is reactive, frenzied, and flecked with spittle. In a world in which no amygdaloid neuron need be afraid and instead can sit under its vine and fig tree, the world is very likely to be a more peaceful place. We now move to the second of the three brain regions we're considering in detail. The frontal cortex. They spent decades studying the hippocampus. It's been good to me. I'd like to think I've been the same in return. Yet I think I might have made the wrong choice back then. Maybe I should have studied the frontal cortex all these years. Because it's the most interesting part of the brain. What does the frontal cortex do? Its list of expertise includes working memory, executive function, organizing knowledge strategically, and then initiating an action based on an executive decision, gratification postponement, long-term planning, regulation of emotions, and reining in impulsivity. 35. This is a sprawling portfolio. I will group these varied functions under a single definition, pertinent to every page of this book. The frontal cortex makes you do the harder thing when it's the right thing to do. To start, here are some important features of the frontal cortex. It's the most recently evolved brain region, not approaching full splendor until the emergence of primates. A disproportionate percentage of genes unique to primates are active in the frontal cortex. Moreover, such gene expression patterns are highly individuated with greater inter-individual variability than average levels of whole brain differences between humans and chimps. The human frontal cortex is more complexly wired than in other apes and, by some definitions as to its boundaries, proportionately bigger as well. 36. The frontal cortex is the last brain region to fully mature, with the most evolutionarily recent subparts the very last. Amazingly, it's not fully online until people are in their mid-20s. You'd better bet this factoid will be relevant to the chapter about adolescence. Finally, the frontal cortex has a unique cell type. In general, the human brain isn't unique because we've evolved unique types of neurons, neurotransmitters, enzymes, and so on. Human and fly neurons are remarkably similar. The uniqueness is quantitative. For every fly neuron, we have a gazillion more neurons, and a bazillion more connections. 37. The sole exception is an obscure type of neuron with a distinctive shape and pattern of wiring, called von Economo neurons, aka spindle neurons. At first they seem to be unique to humans, but we've now found them in other primates, whales, dolphins, and elephants. That's an all-star team of socially complex species. Moreover, the few von Economo neurons occur only in two subregions of the frontal cortex, as shown by John Allman at Caltech. One we've heard about already, the insula with its role in gustatory and moral disgust. The second is an equally interesting area called the anterior cingulate. To give a hint, with more to come, it's central to empathy. So from the standpoint of evolution, size, complexity, development, genetics, and neuron type, the frontal cortex is distinctive, with the human version the most unique. The subregions of the frontal cortex. Frontal cortical anatomy is hellishly complicated, and there are debates as to whether some parts of the primate frontal cortex even exist in simpler species. Nonetheless, there are some useful broad themes. In the very front is the prefrontal cortex, PFC, the newest part of the frontal cortex. As noted, the frontal cortex is central to executive function. To quote George W. Bush, within the frontal cortex, it's the PFC that is the decider. Most broadly, the PFC chooses between conflicting options, Coke or Pepsi, blurting out what you really think, or restraining yourself, pulling the trigger or not. and often the conflict being resolved is between a decision heavily driven by cognition and one driven by emotions. Once it has decided, the PFC sends orders via projections to the rest of the frontal cortex, sitting just behind it. Those neurons then talk to the premotor cortex, sitting just behind it, which then passes it to the motor cortex, which talks to your muscles. And a behavior ensues. Before considering how the frontal cortex influences social behavior, let's start with a simpler domain of its function. The frontal cortex and cognition. What does doing the harder thing when it's the right thing to do look like in the realm of cognition, defined by Princeton's Jonathan Cohen as the ability to orchestrate thought and action in accordance with internal goals? 38. Suppose you've looked up a phone number in a city where you once lived. The frontal cortex not only remembers it long enough to dial, but also considers it strategically. Just before dialing, you consciously recall that it is in that other city and retrieve your memory of the city's area code. And then you remember to dial 1 inch before the area code. The frontal cortex is also concerned with focusing on a task. If you step off the curb planning to jaywalk, you look at traffic, paying attention to motion, calculating whether you can cross safely. If you step off looking for a taxi, you pay attention to whether a car has one of those lit taxicab thingies on top. In a great study, monkeys were trained to look at a screen of dots of various colors, moving in particular directions. Depending on a signal, a monkey had to pay attention to either color or movement. Each signal indicating a shift in tasks triggered a burst of PFC activity and, coupled with that, suppression of the stream of information, color or movement, that was now irrelevant. This is the PFC getting you to do the harder thing. Remembering that the rule has changed, don't do the previous habitual response. 39. The frontal cortex also mediates executive function, considering bits of information, looking for patterns, and then choosing a strategic action. 40. Consider this truly frontally demanding test. The experimenter tells a masochistic volunteer, "I'm going to the market and I'm going to buy peaches, cornflakes, laundry detergent, cinnamon." 16 items recited. The volunteer is asked to repeat the list. Maybe they correctly recall the first few, the last few, list some near misses, say, nutmeg instead of cinnamon. Then the experimenter repeats the same list. This time the volunteer remembers a few more, avoids repeating the nutmeg incident. Now do it again and again. This is more than a simple memory test. With repetition, subjects notice that 4 of the items are fruits, 4 for cleaning, 4 spices, 4 carbs. They come in categories. And this changes subjects' encoding strategy as they start clumping by semantic group: peaches, apples, blueberries, no, I mean blackberries. There was another fruit, can't remember what. Okay, cornflakes, bread, donuts, muffins. Cumin, nutmeg, arg, again, I mean cinnamon, oregano. And throughout, the PFC imposes an overarching executive strategy for remembering these 16 factoids. The PFC is essential for categorical thinking, for organizing and thinking about bits of information with different labels. The PFC groups apples and peaches as closer to each other, in a conceptual map, than are apples and toilet plungers. In a relevant study, monkeys were trained to differentiate between pictures of a dog and of a cat. The PFC contained individual neurons that responded to dog and others that responded to cat. Now the scientists morphed the pictures together, creating hybrids with varying percentages of dog and cat. Dog PFC neurons responded about as much to hybrids that were 80% dog and 20% cat, or 60-40, as to 100% dog. But not to 40-60 cat neurons would kick in their 0.41. The frontal cortex aids the underdog outcome, fueled by thoughts supplied from influences that fill the rest of this book. Stop, those aren't your cookies, you'll go to hell, self-discipline is good, you're happier when you're thinner, all giving some lone inhibitory motor neuron more of a fighting chance. Frontal metabolism and an implicit vulnerability. This raises an important point, pertinent to the social as well as cognitive functions of the frontal cortex. 42. All this I wouldn't do that if I were you. Eating by the frontal cortex is taxing. Other brain regions respond to instances of some contingency. The frontal cortex tracks rules. Just think how around age 3 our frontal cortices learned a rule followed for the rest of our lives: don't pee whenever you feel like it, and gain the means to enact that rule by increasing their influence over neurons regulating the bladder. Moreover, the frontal mantra of self-discipline is good when cookies beckon is also invoked when economizing to increase retirement savings. Frontal cortical neurons are generalists with broad patterns of projections, which makes for more work. 43. All this takes energy, and when it is working hard, the frontal cortex has an extremely high metabolic rate and rates of activation of genes related to energy production. 44. Willpower is more than just a metaphor. Self-control is a finite resource. Frontal neurons are expensive cells, and expensive cells are vulnerable cells. Consistent with that, the frontal cortex is atypically vulnerable to various neurological insults. Pertinent to this is the concept of cognitive load. Make the frontal cortex work hard, a tough working memory task, regulating social behavior, or making numerous decisions while shopping. Immediately afterward, performance on a different frontally dependent task declines. 45. Likewise, during multitasking, where PFC neurons simultaneously participate in multiple activated circuits. Importantly, increase cognitive load on the frontal cortex, and afterward subjects become less prosocial, less charitable or helpful, more likely to lie. 46. Or increase cognitive load with a task requiring difficult emotional regulation, and subjects cheat more on their diets afterward. 47. So the frontal cortex is awash in Calvinist self-discipline, a superego with its nose to the grindstone. 48. But as an important qualifier, soon after we're potty trained, doing the harder thing with our bladder muscles becomes automatic. Likewise with other initially demanding frontal tasks. For example, you're learning a piece of music on the piano, there's a difficult trill, and each time as you approach it, you think, here it comes. Remember, tuck my elbow in, lead with my thumb. A classic working memory task. And then one day you realize that you're 5 measures past the trill, it went fine, and you didn't have to think about it. And that's when doing the trill is transferred from the frontal cortex to more reflexive brain regions, e.g., the cerebellum. This transition to automaticity also happens when you get good at a sport, when metaphorically your body knows what to do without your thinking about it. The chapter on morality considers automaticity in a more important realm. Is resisting lying a demanding task for your frontal cortex, or is it effortless habit? As we'll see, honesty often comes more easily thanks to automaticity. This helps explain the answer typically given after someone has been profoundly brave. What were you thinking when you dove into the river to save that drowning child? I wasn't thinking. Before I knew it, I had jumped in. Often the neurobiology of automaticity mediates doing the hardest moral acts, while the neurobiology of the frontal cortex mediates working hard on a term paper about the subject. The frontal cortex and social behavior. Things get interesting when the frontal cortex has to add social factors to a cognitive mix. For example, one part of the monkey PFC contains neurons that activate when the monkey makes a mistake on a cognitive task or observes another monkey doing so. Some activate only when it's a particular animal who made the mistake. In a neuroimaging study, humans had to choose something, balancing feedback obtained from their own prior choices with advice from another person. Different PFC circuits tracked reward-driven and advice-driven cogitating. 49. Findings like these segue into the central role of the frontal cortex in social behavior. 50. This is appreciated when comparing various primates. Across primate species, the bigger the size of the average social group, the larger the relative size of the frontal cortex. This is particularly so with fission-fusion species, where there are times when subgroups split up and function independently for a while before regrouping. Such a social structure is demanding, requiring the scaling of appropriate behavior to subgroup size and composition. Logically, primates from fission-fusion species, chimps, bonobos, orangutans, spider monkeys, have better frontocortical inhibitory control over behavior than do non-fission-fusion primates, gorillas, capuchins, macaques. Among humans, the larger someone's social network, measured by number of different people texted, the larger a particular PFC subregion. Stay tuned. 51. That's cool, but we can't tell if the big brain region causes the sociality or the reverse, assuming there's causality. Another study resolves this: if rhesus monkeys are randomly placed into social groups over the subsequent 15 months, the bigger the group, the larger the PFC becomes. Social complexity expands the frontal cortex. We utilize the frontal cortex to do the harder thing in social contexts. We praise the hosts for the inedible dinner, refrain from hitting the infuriating coworker, don't make sexual advances to someone, despite our fantasies, don't belch loudly during the eulogy. A great way to appreciate the frontal cortex is to consider what happens when it is damaged. The first frontal patient, the famous Phineas Gage, was identified in 1848 in Vermont. Gage, the foreman on a railroad construction crew, was injured when an accident with blasting powder blew a 13-pound iron tamping rod through the left side of his face and out the top front of his skull. It landed 80 feet away, along with much of his left frontal cortex. 52. The two known pictures of Gage, along with the tamping rod. Remarkably, he survived and recovered his health. But the respected, even-keeled Gage was transformed. In the words of the doctor who followed him over the years, the equilibrium or balance, so to speak, between his intellectual faculties and animal propensities, seems to have been destroyed. He is fitful, irreverent, indulging at times in the grossest profanity, which was not previously his custom, manifesting but little deference for his fellows, impatient of restraint or advice when it conflicts with his desires, at times pertinaciously obstinate, yet capricious and vacillating, devising many plans of future operations, which are no sooner arranged than they are abandoned in turn for others appearing more feasible. Gage was described by friends as no longer Gage, was incapable of resuming his job and was reduced to appearing with his rod as an exhibit displayed by P.T. Barnum. Poignant as hell. Amazingly, Gage got better. Within a few years of his injury, he could resume work, mostly as a stagecoach driver, and was described as being broadly appropriate in his behavior. His remaining right frontal cortical tissue had taken on some of the functions lost in the injury. Such malleability of the brain is the focus of Chapter 5. Another example of what happens when the frontal cortex is damaged is observed in frontotemporal dementia (FTD), which starts by damaging the frontal cortex. Intriguingly, the first neurons killed are those mysterious von Economo neurons that are unique to primates, elephants, and cetaceans. 53. What are people with FTD like? They exhibit behavioral disinhibition and socially inappropriate behaviors. There's also an apathy and lack of initiating behavior that reflects the fact that the decider is being destroyed. Something similar is seen in Huntington's disease, a horrific disorder due to a thoroughly weird mutation. Subcortical circuits that coordinate signaling to muscles are destroyed and the sufferer is progressively incapacitated by involuntary writhing movements. Except that it turns out that there is frontal damage as well, often before the subcortical damage. In about half the patients, there's also behavioral disinhibition, stealing, aggressiveness, hypersexuality, bursts of compulsive, inexplicable gambling. Social and behavioral disinhibition also occur in individuals with stroke damage in the frontal cortex, for example, sexually assaultive behavior in an octogenarian. There's another circumstance where the frontal cortex is hypofunctional, producing similar behavioral manifestations, hypersexuality, outbursts of emotion, flamboyantly illogical acts. 54. What disease is this? It isn't. You're dreaming. During REM sleep, when dreaming occurs, the frontal cortex goes offline, and dream scriptwriters run wild. Moreover, if the frontal cortex is stimulated while people are dreaming, the dreams become less dreamlike, with more self-awareness. And there's another non-pathological circumstance where the PFC silences, producing emotional tsunamis during orgasm. One last realm of frontal damage. Adrian Rain of the University of Pennsylvania and Kent Keel of the University of New Mexico report that criminal psychopaths have decreased activity in the frontal cortex and less coupling of the PFC to other brain regions, compared with non-psychopathic criminals and non-criminal controls. Moreover, a shockingly large percentage of people incarcerated for violent crimes have a history of concussive trauma to the frontal cortex. 55. More to come in Chapter 16. The obligatory declaration of the falseness of the dichotomy between cognition and emotion. The PFC consists of various parts, subparts, and sub-subparts, enough to keep neuroanatomists off the dole. Two regions are crucial. First, there is the dorsal part of the PFC, especially the dorsolateral PFC, DLPFC, Don't worry about dorsal or dorsolateral, it's just jargon. The DLPFC is the decider of deciders, the most rational, cognitive, utilitarian, unsentimental part of the PFC. It's the most recently evolved part of the PFC and the last part to fully mature. It mostly hears from and talks to other cortical regions. In contrast to the DLPFC, there's the ventral part of the PFC, particularly the ventromedial PFC, VMPFC. This is the frontocortical region that the visionary neuroanatomist Nata made an honorary member of the limbic system because of its interconnections with it. Logically, the VMPFC is all about the impact of emotion on decision-making. And many of our best and worst behaviors involve interactions of the VMPFC with the limbic system and DLPFC. The functions of the cognitive DLPFC are the essence of doing the harder thing. 56. It's the most active frontocortical region when someone forgoes an immediate reward for a bigger one later. Consider a classic moral quandary: is it okay to kill one innocent person to save 5? When people ponder the question, greater DLPFC activation predicts a greater likelihood of answering yes, but as we'll see in Chapter 13, it also depends on how you ask the question. Monkeys with DLPFC lesions can't switch strategies in a task when the rewards given for each strategy shift. They perseverate with the strategy offering the most immediate reward. 57. Similarly, humans with DLPFC damage are impaired in planning or gratification postponement, perseverate on strategies that offer immediate reward, and show poor executive control over their behavior. Remarkably, the technique of transcranial magnetic stimulation can temporarily silence part of someone's cortex, as was done in a fascinating study by Ernst Farah of the University of Zurich. 58. When the DLPFC was silenced, subjects playing an economic game impulsively accepted lousy offers that they'd normally reject in the hopes of getting better offers in the future. Crucially, this was about sociality. Silencing the DLPFC had no effect if subjects thought the other player was a computer. Moreover, controls and subjects with silenced DLPFCs rated lousy offers as being equally unfair. Thus, as concluded by the authors, subjects with the silenced DLPFC behave as if they can no longer implement their fairness goals. What are the functions of the emotional VMPFC? 59. What you'd expect, given its inputs from limbic structures. It activates if the person you're rooting for wins a game, or if you listen to pleasant versus dissonant music, particularly if the music provokes a shiver down the spine moment. What are the effects of VMPFC damage? 60. Lots of things remain normal: intelligence, working memory, making estimates. Individuals can do the harder thing with purely cognitive frontal tasks, e.g., puzzles where you have to give up a step of progress in order to gain 2 more. The differences appear when it comes to making social emotional decisions. VMPFC patients just can't decide. They understand the options and can sagely advise someone else in similar circumstances. But the closer to home and the more emotional the scenario, the more they have problems. Damasio has produced an influential theory about emotion, latent decision-making, rooted in the philosophies of Hume and William James. This will soon be discussed. 61. Briefly, the frontal cortex runs as if experiments of gut feelings. How would I feel if this outcome occurred? And makes choices with the answer in mind. Damaging the VMPFC, thus removing limbic input to the PFC, eliminates gut feelings, making decisions harder. Moreover, eventual decisions are highly utilitarian. VMPFC patients are atypically willing to sacrifice one person, including a family member, to save 5 strangers. 62. They're more interested in outcomes than in their underlying emotional motives, punishing someone who accidentally kills, but not one who tried to kill but failed, because, after all, no one died in the second case. It's Mr. Spock, running on only the DLPFC. Now for a crucial point. People who dichotomize between thought and emotion often prefer the former, viewing emotion as suspect. It gums up decision-making by getting sentimental, sings too loudly, dresses flamboyantly, has unsettling amounts of armpit hair. In this view, get rid of the VMPFC, and we'd be more rational and function better. But that's not the case, as emphasized eloquently by Damasio. People with VMPFC damage not only have trouble making decisions but also make bad ones. 63. They show poor judgment in choosing friends and partners and don't shift behavior based on negative feedback. For example, consider a gambling task where reward rates for various strategies change without subjects knowing it, and subjects can shift their play strategy. Control subjects shift optimally, even if they can't verbalize how reward rates have changed. Those with VMPFC damage don't, even when they can verbalize. Without a VMPFC, you may know the meaning of negative feedback, but you don't know the feeling of it in your gut and thus don't shift behavior. As we saw, without the DLPFC, the metaphorical superego is gone, resulting in individuals who are now hyperaggressive, hypersexualids. But without a VMPFC, behavior is inappropriate in a detached way. This is the person who, encountering someone after a long time, says, "Hello, I see you've put on some weight." And when castigated later by their mortified spouse, they will say with calm puzzlement, "But it's true." The VMPFC is not the vestigial appendix of the frontal cortex, where emotion is something akin to appendicitis inflaming a sensible brain. Instead, it's essential. 64. It wouldn't be if we had evolved into Vulcans. But as long as the world is filled with humans, evolution would never have made us that way. Activation of the DLPFC and VMPFC can be inversely correlated. In an inspired study where a keyboard was provided to jazz pianists inside a brain scanner, the VMPFC became more active and the DLPFC less so when subjects improvised. In another study, subjects judged hypothetical harmful acts. Pondering perpetrators' responsibility activated the DLPFC, deciding the amount of punishment activated the VMPFC when subjects did a gambling task where reward probabilities for various strategies shifted and they could always change strategies. Decision-making reflected two factors: the outcome of their most recent action, the better that had turned out, the more VMPFC activation, and b. reward rates from all the previous rounds, something requiring a long retrospective view, the better the long-term rewards. The more DLPFC activation. Relative activation between the two regions predicted the decision subjects made. 65. A simplistic view is that the VMPFC and DLPFC perpetually battle for domination by emotion versus cognition. But while emotion and cognition can be somewhat separable, they're rarely in opposition. Instead, they are intertwined in a collaborative relationship needed for normal function, and as tasks with both emotive and cognitive components become more difficult, making an increasingly complex economic decision in a setting that is increasingly unfair, activity in the two structures becomes more synchronized. The frontal cortex and its relationship with the limbic system. We now have a sense of what different subdivisions of the PFC do and how cognition and emotion interact neurobiologically. This leads us to consider how the frontal cortex and limbic system interact. In landmark studies, Joshua Green of Harvard and Princeton's Cohen showed how the emotional and cognitive parts of the brain can somewhat dissociate. 66. They used philosophy's famous runaway trolley problem, where a trolley is bearing down on 5 people and you must decide if it's okay to kill one person to save the 5. Framing of the problem is key. In one version, you pull a lever, diverting the trolley onto a sidetrack. This saves the 5, but the trolley kills someone who happened to be on this other track. 70 to 90% of people say they would do this. In the second scenario, you push the person in front of the trolley with your own hands. This stops the trolley, but the person is killed. 70 to 90% say no way. The same numerical trade-off, but utterly different decisions. Green and Cohen gave subjects the two versions while neuroimaging them. Contemplating intentionally killing someone with your own hands activates the decider DLPFC, along with emotion-related regions that respond to aversive stimuli, including a cortical region activated by emotionally laden words, the amygdala, and the VMPFC. The more amygdaloid activation and the more negative emotions the participant reported in deciding, the less likely they were to push. And when people contemplate detachedly pulling a lever that inadvertently kills someone, the DLPFC alone activates. As purely cerebral a decision as choosing which wrench to use to fix a widget. A great study. Other studies have examined interactions between cognitive and emotional parts of the brain. A few examples. Chapter 3 discusses some unsettling research. Stick your average person in a brain scanner, and show him a picture of someone of another race for only 1/10 of a second. This is too fast for him to be aware of what he saw. But thanks to that anatomical shortcut, the amygdala knows and activates. In contrast, show the picture for a longer time. Again, the amygdala activates, but then the cognitive DLPFC does as well, inhibiting the amygdala, the effort to control what is for most people an unpalatable initial response. Chapter 6 discusses experiments where a subject plays a game with two other people and is manipulated into feeling that she is being left out. This activates her amygdala, periaqueductal gray, that ancient brain region that helps process physical pain, anterior cingulate and insula, the anatomical picture of anger, anxiety, pain, disgust, sadness. Soon afterward her PFC activates as rationalizations kick in. This is just a stupid game, I have friends, my dog loves me. And the amygdala et al. Quiet down. And what if you do the same to someone whose frontal cortex is not fully functional? The amygdala is increasingly activated, the person feels increasingly distressed. What neurological disease is involved? None. This is a typical teenager. Finally, the PFC mediates fear extinction. Yesterday the rat learned that tone is followed by a shock, so the sound of the tone began to trigger freezing. Today there are no shocks, and the rat has acquired another truth that takes precedence, but not today. The first truth is still there as proof. Start coupling tone with shock again, and freezing to tone is reinstated faster than the association was initially learned. Whereas but not today, consolidated. In the PFC, after receiving information from the hippocampus. 67. The medial PFC activates inhibitory circuits in the blah, and the rat stops freezing to the tone. In a similar vein but reflecting cognition specific to humans, condition people to associate a blue square on a screen with a shock, and the amygdala will activate when seeing that square, but less so in subjects who reappraise the situation, activating the medial PFC by thinking of, say, a beautiful blue sky. This segues into the subject of regulating emotion through thought. 68. It's hard to regulate thought. Try not thinking about a hippo, but even tougher with emotion. Research by my Stanford colleague and close friend James Gross has explored this. First off, thinking differently about something emotional differs from simply suppressing the expression of the emotions. For example, show someone graphic footage of, say, an amputation. Subjects cringe, activate the amygdala and sympathetic nervous system. Now one group is instructed to hide their emotions. I'm going to show you another film clip, and I want you to hide your emotional reactions. How to do so most effectively? Gross distinguishes between antecedent and response-focused strategies. Response-focused is dragging the emotional horse back to the barn after it's fled. You're watching the next horrific footage, feeling queasy, and you think, okay, sit still, breathe slowly. Typically this causes even greater activation of the amygdala and sympathetic nervous system. Antecedent strategies generally work better as they keep the barn door closed from the start. These are about thinking feeling about something else, e.g., that great vacation, or thinking feeling differently about what you're seeing. Reappraisals such as that isn't real, those are just actors. And when done right, the PFC, particularly the DLPFC, activates, the amygdala and sympathetic nervous system are damped, and subjective distress decreases. Antecedent reappraisal is why placebos work. 69. Thinking, my finger is about to be pricked by a pin, activates the amygdala along with a circuit of pain, responsive brain regions, and the pin hurts. Be told beforehand that the hand cream being slathered on your finger is a powerful analgesic cream, and you think, my finger is about to be pricked by a pin, but this cream will block the pain. The PFC activates, blunting activity in the amygdala and pain circuitry, as well as pain perception. Thought processes like these, writ large, are the core of a particularly effective type of psychotherapy, cognitive behavioral therapy, CBT, for the treatment of disorders of emotion, regulation. 70. Consider someone with a social anxiety disorder, caused by a horrible early experience with trauma. To simplify, CBT is about providing the tools to reappraise circumstances that evoke the anxiety. Remember that in this social situation, those awful feelings you're having are about what happened back then, not what is happening now. Controlling emotional responses with thought like this is very top-down. The frontal cortex calms the overwrought amygdala. But the PFC-limbic relationship can be bottom-up as well, when a decision involves a gut feeling. This is the backbone of Damasio's somatic marker hypothesis. Choosing among options can involve a cerebral cost-benefit analysis. But it also involves somatic markers, internal simulations of what each outcome would feel like, run in the limbic system, and reported to the VMPFC. The process is not a thought experiment, it's an emotion experiment, in effect an emotional memory of a possible future. A mild somatic marker activates only the limbic system. 71. Should I do behavior A? Maybe not, the possibility of outcome B feels scary. A more vivid somatic marker activates the sympathetic nervous system as well. Should I do behavior A? Definitely not, I can feel my skin getting clammy at the possibility of outcome B. Experimentally boosting the strength of that sympathetic signal strengthens the aversion. This is a picture of normal collaboration between the limbic system and frontal cortex. 72. Naturally, things are not always balanced. Anger, for example, makes people less analytical and more reflexive in decisions about punishment. Stressed people often make hideously bad decisions, marinated in emotion. Chapter 4 examines what stress does to the amygdala and frontal cortex. The effects of stress on the frontal cortex are dissected by the late Harvard psychologist, Daniel Wegner, in an aptly titled paper, How to Think, Say or Do Precisely the Worst Thing on Any Occasion. 73. He considers what Edgar Allan Poe called the imp of the perverse. We see a rut coming up in the road ahead and proceed to steer our bike right into it. We make a mental note not to mention a sore point in conversation and then cringe in horror as we blurt out exactly that thing. We carefully cradle the glass of red wine as we cross the room, all the while thinking don't spill, and then juggle it onto the carpet under the gaze of our host. Wegner demonstrated a two-step process of frontocortical regulation. One stream identifies X as being very important, B, the other stream tracks whether the conclusion is do X, or never do X. And during stress, distraction, or heavy cognitive load, the two streams can dissociate. The A stream exerts its presence, without the B stream saying which fork in the road to take. The chance that you will do precisely the wrong thing rises not despite your best efforts but because of a stress-boggled version of them. This concludes our overview of the frontal cortex. The mantra is that it makes you do the harder thing when that is the right thing. 5 final points. Doing the harder thing, effectively, is not an argument for valuing either emotion or cognition more than the other. For example, as discussed in Chapter 11, we are our most prosocial concerning in group morality, when our rapid, implicit emotions and intuitions dominate, but are most prosocial concerning out group morality when cognition holds sway. It's easy to conclude that the PFC is about preventing imprudent behaviors. Don't do it, you'll regret it. But that isn't always the case. For example, in Chapter 17 we'll consider the surprising amount of frontocortical effort it can take to pull a trigger. Like everything about the brain, the structure and function of the frontal cortex vary enormously among individuals. For example, resting metabolic rate in the PFC varies approximately 30-fold among people. What causes such individual differences? See the rest of this book. 74. Doing the harder thing when it's the right thing to do. Right in this case is used in a neurobiological and instrumental sense, rather than a moral one. Consider lying. Obviously, the frontal cortex aids the hard job of resisting the temptation. But it is also a major frontocortical task, particularly a DLPFC task, to lie competently, to control the emotional content of a signal, to generate an abstract distance between message and meaning. Interestingly, pathological liars have atypically large amounts of white matter in the PFC, indicating more complex wiring. 75. But again, the right thing in the setting of the frontal cortically assisted lying is amoral. An actor lies to an audience about having the feelings of a morose Danish prince. A situationally ethical child lies, telling grandma how excited she is about her present, concealing the fact that she already has that toy. A leader tells bold-faced lies, starting a war. A financier with Ponzi in his blood defrauds investors. A peasant woman lies to a uniformed thug, telling him she does not know the whereabouts of the refugees she knows are hiding in her attic. As with much about the frontal cortex, it's context, context, context. Where does the frontal cortex get the metaphorical motivation to do the harder thing? For this we now look at our final branch, the dopaminergic reward system in the brain. R. The mesolimbic mesocortical dopamine system. Reward, pleasure, and happiness are complex, and the motivated pursuit of them occurs in at least a rudimentary form in many species. The neurotransmitter dopamine is central to understanding this. Nuclei, inputs, and outputs. Dopamine is synthesized in multiple brain regions. One such region helps initiate movement, damage there produces Parkinson's disease. Another regulates the release of a pituitary hormone. But the dopaminergic system that concerns us arises from an ancient, evolutionarily conserved region near the brainstem called the ventral tegmental area, henceforth the tegmentum. A key target of these dopaminergic neurons is the last multisyllabic brain region to be introduced in this chapter, the Nucleus Accumbens, henceforth the accumbens. There's debate as to whether the accumbens should count as part of the limbic system, but at the least it's highly limbic-ish. Here's our first pass at the organization of this circuitry 7-6. A. The tegmentum sends projections to the accumbens and other limbic areas such as the amygdala and hippocampus. This is collectively called the mesolimbic dopamine pathway. B. The tegmentum also projects to the PFC, but, significantly, not other cortical areas. This is called the mesocortical dopamine pathway. I'll be lumping the mesolimbic plus mesocortical pathways together as the dopaminergic system, ignoring they're not always being activated simultaneously. C. The accumbens projects to regions associated with movement. D. Naturally, most areas getting projections from the tegmentum and/or accumbens project back to them. Most interesting will be the projections from the amygdala and PFC. Reward. As a first pass, the dopaminergic system is about reward. Various pleasurable stimuli activate tegmental neurons, triggering their release of dopamine. 77. Some supporting evidence, drugs like cocaine, heroin, and alcohol release dopamine in the accumbens. B. If tegmental release of dopamine is blocked, previously rewarding stimuli become aversive. C. Chronic stress or pain depletes dopamine and decreases the sensitivity of dopamine neurons to stimulation, producing the defining symptom of depression: anhedonia, the inability to feel pleasure. Some rewards, such as sex, release dopamine in every species examined. 78. For humans, just thinking about sex suffices. 79. Food evokes dopamine release in hungry individuals of all species, with an added twist in humans. Show a picture of a milkshake to someone after they've consumed one, and there's rarely dopaminergic activation, there's satiation. But with subjects who have been dieting, there's further activation. If you're working to restrict your food intake, a milkshake just makes you want another one. The mesolimbic dopamine system also responds to pleasurable aesthetics. 80. In one study, people listened to new music. The more accumbens activation, the more likely subjects were to buy the music afterward. And then there is dopaminergic activation for artificial cultural inventions. For example, when typical males look at pictures of sports cars. Patterns of dopamine release are most interesting when concerning social interactions. 81. Some findings are downright heartwarming. In one study, a subject would play an economic game with someone, where a player is rewarded under two circumstances: A. If both players cooperate, each receives a moderate reward, and B. Stabbing the other person in the back gets the subject a big reward, while the other person gets nothing. While both outcomes increased dopaminergic activity, the bigger increase occurred after cooperation. Other research examined the economic behavior of punishing jerks. 82. In one study, subjects played a game where player B could screw over player A for a profit. Depending on the round, player A could either A. Do nothing, B. Punish player B by having some of player B's money taken at no cost to player B, or C. Pay 1 unit of money to have 2 units taken from player B. Punishment activated the dopamine system, especially when subjects had to pay to punish. The greater the dopamine increase during no-cost punishment, the more willing someone was to pay to punish. Punishing norm violations is satisfying. Another great study, carried out by Elizabeth Phelps of New York University, concerns overbidding in auctions, where people bid more money than anticipated. 83. This is interpreted as reflecting the additional reward of besting someone in the competitive aspect of bidding. Thus, winning an auction is intrinsically socially competitive, unlike winning a lottery. Winning a lottery and winning a bid both activated dopaminergic signaling in subjects. Losing a lottery had no effect, while losing a bidding war inhibited dopamine release. Not winning the lottery is bad luck, not winning an auction is social subordination. This raises the specter of envy. In one neuroimaging study, subjects read about a hypothetical person's academic record, popularity, attractiveness, and wealth. 84. Descriptions that evoke self-reported envy activated cortical regions involved in pain perception. Then the hypothetical individual was described as experiencing a misfortune, e.g., they were demoted. More activation of pain pathways at the news of the person's good fortune predicted more dopaminergic activation after learning of their misfortune. Thus there's dopaminergic activation during schadenfreude, gloating over an envied person's fall from grace. The dopamine system gives insights into jealousy, resentment, and invidiousness, leading to another depressing finding. 85. A monkey has learned that when he presses a lever 10 times, he gets a raisin as a reward. That's just happened, and as a result, 10 units of dopamine are released in the accumbens. Now, surprise, the monkey presses the lever 10 times and gets 2 raisins. Whoa, 20 units of dopamine are released. And as the monkey continues to get paychecks of 2 raisins, the size of the dopamine response returns to 10 units. Now reward the monkey with only a single raisin, and dopamine levels decline. Why? This is our world of habituation, where nothing is ever as good as that first time. Unfortunately, things have to work this way because of our range of rewards. 86. After all, reward coding must accommodate the rewarding properties of both solving a math problem and having an orgasm. Dopaminergic responses to reward, rather than being absolute, are relative to the reward value of alternative outcomes. In order to accommodate the pleasures of both mathematics and orgasms, the system must constantly rescale to accommodate the range of intensity offered by particular stimuli. The response to any reward must habituate with repetition, so that the system can respond over its full range to the next new thing. This was shown in a beautiful study by Wolfram Schultz of Cambridge University. 87. Depending on the circumstance, monkeys were trained to expect either 2 or 20 units of reward. If they unexpectedly got either 4 or 40 units, respectively, there'd be an identical burst of dopamine release, giving 1 or 10 units, produced an identical decrease. It was the relative, not absolute, size of the surprise that mattered over a tenfold range of reward. These studies show that the dopamine system is bidirectional. 88. It responds with scale-free increases for unexpected good news and decreases for bad. Schultz demonstrated that following a reward, the dopamine system codes for discrepancy from expectation. Get what you expected, and there's a steady-state dribble of dopamine. Get more reward or get it sooner than expected, and there's a big burst, lessen or later a decrease. Some tegmental neurons respond to positive discrepancy from expectation, others to negative, appropriately. The latter are local neurons that release the inhibitory neurotransmitter GABA. Those same neurons participate in habituation, where the reward that once elicited a big dopamine response becomes less exciting. Logically, these different types of coding neurons in the tegmentum, as well as the accumbens, get projections from the frontal cortex. That's where all the expectancy discrepancy calculations take place. Okay, I thought I was going to get 5.0, but got 4.9. How big of a bummer is that? Additional cortical regions weigh in. In one study, subjects were shown an item to purchase, with the degree of accumbens activation predicting how much a person would pay. 89. Then they were told the price. If it was less than what they were willing to spend, there was activation of the emotional VMPFC, more expensive, and there'd be activation of that disgust-related insular cortex. Combine all the neuroimaging data, and you could predict whether the person would buy the item. Thus, in typical mammals, the dopamine system codes in a scale-free manner over a wide range of experience for both good and bad surprises and is constantly habituating to yesterday's news. But humans have something in addition, namely that we invent pleasures far more intense than anything offered by the natural world. Once, during a concert of cathedral organ music, as I sat getting gooseflesh amid that tsunami of sound, I was struck with a thought. For a medieval peasant, this must have been the loudest human-made sound they ever experienced, awe-inspiring in now unimaginable ways. No wonder they signed up for the religion being proffered. And now we are constantly pummeled with sounds that dwarf quaint organs. Once, hunter-gatherers might chance upon honey from a beehive and thus briefly satisfy a hardwired food craving. And now we have hundreds of carefully designed commercial foods that supply a burst of sensation unmatched by some lowly natural food. Once, we had lives that, amid considerable privation, also offered numerous subtle, hard-won pleasures. And now we have drugs that cause spasms of pleasure and dopamine release 1,000-fold higher than anything stimulated in our old drug-free world. An emptiness comes from this combination of over-the-top non-natural sources of reward and the inevitability of habituation. This is because unnaturally strong explosions of synthetic experience and sensation and pleasure evoke unnaturally strong degrees of habituation. 90. This has two consequences. First, soon we barely notice the fleeting whispers of pleasure caused by leaves in autumn, or by the lingering glance of the right person, or by the promise of reward, following a difficult, worry task. And the other consequence is that we eventually habituate to even those artificial deluges of intensity. If we were designed by engineers as we consumed more, we desire less. But our frequent human tragedy is that the more we consume, the hungrier we get. More and faster and stronger. What was an unexpected pleasure yesterday is what we feel entitled to today, and what won't be enough tomorrow. The anticipation of reward. Thus, dopamine is about invidious, rapidly habituating reward. But dopamine is more interesting than that. Back to our well-trained monkey working for a reward. A light comes on in his room, signaling the start of a reward trial. He goes over to the lever, presses 10 times, and gets the raisin reward. This has happened often enough that there's only a small increase in dopamine with each raisin. However, importantly, lots of dopamine is released when the light first comes on, signaling the start of the reward trial, before the monkey starts lever pressing. Visit bit.ly/2ofjngg for a larger version of this graph. In other words, once reward contingencies are learned, dopamine is less about reward than about its anticipation. Similarly, work by my Stanford colleague Brian Knutson has shown dopamine pathway activation in people in anticipation of a monetary reward. 91. Dopamine is about mastery and expectation and confidence. It's I know how things work, this is going to be great. In other words, the pleasure is in the anticipation of reward, and the reward itself is nearly an afterthought, unless, of course, the reward fails to arrive, in which case it's the most important thing in the world. If you know your appetite will be sated, pleasure is more about the appetite than about the sating. This is hugely important. Anticipation requires learning. 92. Learn Warren G. Harding's middle name, and synapses in the hippocampus become more excitable. Learn that when the light comes on, it's reward time, and it's hippocampal amygdaloid and frontal cortical neurons, projecting to dopamine neurons that become more excitable. This explains context-dependent craving in addiction. 93. Suppose an alcoholic has been clean and sober for years. Return him to where the alcohol consumption used to occur, e.g. that rundown street corner, that fancy men's club, and those potentiated synapses, those cues that were learned to be associated with alcohol, come roaring back into action. Dopamine surges with anticipation, and the craving inundates. Can a reliable cue of an impending reward eventually become rewarding itself? This has been shown by Huda Akil of the University of Michigan. A light in the left side of a rat's cage signals that lever pressing will produce a reward from a food chute on the right side. Remarkably, rats eventually will work for the chance to hang around on the left side of the cage, just because it feels so nice to be there. The signal has gained the dopaminergic power of what is being signaled. Similarly, rats will work to be exposed to a cue that signals that some kind of reward is likely, without knowing what or when. This is what fetishes are in both the anthropological and sexual sense. 94. Schultz group has shown that the magnitude of an anticipatory dopamine rise reflects two variables. First is the size of the anticipated reward. A monkey has learned that a light means that 10 lever presses earns 1 unit of reward, while a tone means 10 presses earns 10 units. And soon a tone provokes more anticipatory dopamine than does a light. It's this is going to be great versus this is going to be great. The second variable is extraordinary. The rule is that the light comes on, you press the lever, you get the reward. Now things change. Light comes on, press the lever, get the reward. Only 50% of the time. Remarkably, once that new scenario is learned, far more dopamine is released. Why? Because nothing fuels dopamine release like the maybe of intermittent reinforcement. 95. This additional dopamine is released at a distinctive time. The light comes on in the 50% scenario, producing the usual anticipatory dopamine rise before the lever pressing starts. Back in the predictable days, when lever pressing always earned a reward, once the pressing was finished, dopamine levels remained low until the reward arrived, followed by a little dopamine blip. But in this 50% scenario, once the pressing is finished, dopamine levels start rising, driven by the uncertainty of maybe yes, maybe no. Visit bit.ly/203zvcq for a larger version of this graph. Modify things further, reward now occurs 25 or 75% of the time. A shift from 50 to 25% and a shift from 50 to 75% are exactly opposite in terms of the likelihood of reward, and work from Knudsen's group shows that the greater the probability of reward, the more activation in the medial PFC. 0.96. But switches from 50 to 25% and from 50 to 75% both reduce the magnitude of uncertainty. And the secondary rise of dopamine for a 25 or 75% likelihood of reward is smaller than for 50%. Thus, anticipatory dopamine release peaks with the greatest uncertainty as to whether a reward will occur. Interestingly, in circumstances of uncertainty, enhanced anticipatory dopamine release is mostly in the mesocortical rather than mesolimbic pathway, implying that uncertainty is a more cognitively complex state than is anticipation of predictable reward. None of this is news to the honorary psychologists running Las Vegas. Logically, gambling shouldn't evoke much anticipatory dopamine, given the astronomical odds against winning. But the behavioral engineering, the 24/7 activity and lack of time cues, the cheap alcohol pickling frontocortical judgment, the manipulations to make you feel like today is your lucky day, distorts and shifts the perception of the odds into a range where dopamine pours out and, oh, why not, let's try again. The interaction between maybe and the propensity for addictive gambling is seen in a study of near misses when 2 out of 3 reels line up in a slot machine. In control subjects there was minimal dopaminergic activation after misses of any sort. Among pathological gamblers, a near miss activated the dopamine system like crazy. Another study concerned 2 betting situations with identical probabilities of reward but different levels of information about reward contingencies. The circumstance with less information, i.e., that was more about ambiguity than risk, activated the amygdala and silenced dopaminergic signaling. What is perceived to be well-calibrated risk is addictive, while ambiguity is just agitating. 0.97. Pursuit. So dopamine is more about anticipation of reward than about reward itself. Time for one more piece of the picture. Consider that monkey trained to respond to the light cue with lever pressing, and out comes the reward. As we now know, once that relationship is established, most dopamine release is anticipatory, occurring right after the cue. What happens if the post-light cue release of dopamine doesn't occur? 98. Crucially, the monkey doesn't press the lever. Similarly, if you destroy its accumbens, a rat makes impulsive choices, instead of holding out for a delayed larger reward. Conversely, back to the monkey, if instead of flashing the light cue, you electrically stimulate the tegmentum to release dopamine, the monkey presses the lever. Dopamine is not just about reward anticipation. It fuels the goal-directed behavior needed to gain that reward. Dopamine binds the value of a reward to the resulting work. It's about the motivation arising from those dopaminergic projections to the PFC that is needed to do the harder thing, i.e., to work. In other words, dopamine is not about the happiness of reward. It's about the happiness of pursuit of reward that has a decent chance of occurring. 99. This is central to understanding the nature of motivation, as well as its failures, e.g., during depression, where there is inhibition of dopamine signaling thanks to stress, or in anxiety, where such inhibition is caused by projections from the amygdala. 100. It also tells us about the source of the frontocortical power behind willpower. In a task where one chooses between an immediate and a larger, delayed reward, contemplating the immediate reward activates limbic targets of dopamine, i.e., the mesolimbic pathway, whereas contemplating the delayed reward activates frontocortical targets, i.e., the mesocortical pathway. The greater the activation of the latter, the more likely there'll be gratification postponement. These studies involve scenarios of a short burst of work soon followed by reward. 101. What about when the work required is prolonged, and reward is substantially delayed? In that scenario there is a secondary rise of dopamine, a gradual increase that fuels the sustained work. The extent of the dopamine ramp-up is a function of the length of the delay and the anticipated size of the reward. Visit bit.ly/2ngtc7v for a larger version of this graph. This reveals how dopamine fuels delayed gratification. If waiting X amount of time for a reward has value Z, waiting 2X should logically have value 1/2 Z. Instead we temporally discount. The value is smaller, e.g., 1/4 Z. We don't like waiting. Dopamine and the frontal cortex are in the thick of this phenomenon. Discounting curves, a value of 1/4 Z instead of 1/2 Z, are coded in the accumbens, while DLPFC and VMPFC neurons code for time, delay. 102. This generates some complex interactions. For example, activate the VMPFC or inactivate the DLPFC, and short-term reward becomes more alluring. And a cool neuroimaging study of Knutson's gives insight into impatient people with steep temporal discounting curves. Their accumbens in effect underestimates the magnitude of the delayed reward, and their DLPFC overestimates the length of the delay. 103. Collectively these studies show that our dopaminergic system, frontal cortex, amygdala insula, and other members of the chorus code for differing aspects of reward magnitude, delay, and probability with varying degrees of accuracy, all influencing whether we manage to do the harder, more correct thing. 104. Individual differences among people in the capacity for gratification, postponement arise from variation in the volume of these individual neural voices. 105. For example, there are abnormalities in dopamine response profiles during temporal discounting tasks in people with the maladaptive impulsiveness of attention deficit/hyperactivity disorder, ADHD. Similarly, addictive drugs bias the dopamine system toward impulsiveness. Few. One more complication, these studies of temporal discounting typically involve delays on the order of seconds. Though the dopamine system is similar across numerous species, humans do something utterly novel: we delay gratification for insanely long times. No warthog restricts calories to look good in a bathing suit next summer. No gerbil works hard at school to get good SAT scores, to get into a good college, to get into a good grad school, to get a good job, to get into a good nursing home. We do something even beyond this unprecedented gratification delay. We use the dopaminergic power of the happiness of pursuit to motivate us to work for rewards that come after we are dead. Depending on your culture, this can be knowing that your nation is closer to winning a war because you've sacrificed yourself in battle, that your kids will inherit money because of your financial sacrifices, or that you will spend eternity in paradise. It is extraordinary neural circuitry that bucks temporal discounting enough to allow some of us to care about the temperature of the planet that our great-grandchildren will inherit. Basically, it's unknown how we humans do this. We may merely be a type of animal, mammal, primate, and ape, but we're a profoundly unique one. A final small topic, serotonin. This lengthy section has concerned dopamine, but an additional neurotransmitter, serotonin, plays a clear role in some behaviors that concern us. Starting with a 1979 study, low levels of serotonin in the brain were shown to be associated with elevated levels of human aggression, with endpoints ranging from psychological measures of hostility to overt violence. 106. A similar serotonin aggression relationship was observed in other mammals and, remarkably, even crickets, mollusks, and crustaceans. As work continued, an important qualifier emerged. Low serotonin didn't predict premeditated, instrumental violence. It predicted impulsive aggression, as well as cognitive impulsivity, e.g., steep temporal discounting or trouble inhibiting a habitual response. Other studies linked low serotonin to impulsive suicide, independent of severity of the associated psychiatric illness. 107. Moreover, in both animals and humans, pharmacologically decreasing serotonin signaling increases behavioral and cognitive impulsivity, e.g., impulsively torpedoing a stable, cooperative relationship with a player in an economic game. 108. Importantly, while increasing serotonin signaling did not lessen impulsiveness in normal subjects, it did in subjects prone toward impulsivity, such as adolescents with conduct disorder. How does serotonin do this? Nearly all serotonin is synthesized in one brain region, which projects to the usual suspects, the tegmentum, accumbens, PFC, and amygdala, where serotonin enhances dopamine's effects on goal-directed behavior. 109. This is as dependable a finding as you get in this business. 110. Until we get to Chapter 8 and look at genes related to serotonin, at which point everything becomes a completely contradictory mess. Just as a hint of what's to come, one gene variant has even been referred to, straight-faced, by some scientists as the warrior gene, and its presence has been used successfully in some courtrooms to lessen sentences for impulsive murders. T. Conclusions. This completes our introduction to the nervous system and its role in pro- and antisocial behaviors. It was organized around three themes: the hub of fear, aggression, and arousal centered in the amygdala, the hub of reward, anticipation, and motivation of the dopaminergic system, and the hub of frontal cortical regulation and restraint of behavior. Additional brain regions and neurotransmitters will be introduced in subsequent chapters. Amid this mountain of information, be assured that the key brain regions, circuits, and neurotransmitters will become familiar as the book progresses. Hang on. So what does this all mean? It's useful to start with three things that this information doesn't mean. 1. First, there's the lure of needing neurobiology to confirm the obvious. Someone claims that, for example, their crappy, violent neighborhood leaves them so anxious that they can't function effectively. Toss them in a brain scanner and flash pictures of various neighborhoods. When their own appears, the amygdala explodes into activity. It is tempting to conclude, we've now proven that the person really does feel frightened. It shouldn't require neuroscience to validate someone's internal state. An example of this fallacy was reports of atrophy of the hippocampus in combat vets suffering from PTSD. This was in accord with basic research, including from my lab, showing that stress can damage the hippocampus. The hippocampal atrophy in PTSD got a lot of play in Washington, helping to convince skeptics that PTSD is an organic disorder rather than neurotic malingering. It struck me that if it took brain scans to convince legislators that there's something tragically, organically damaged in combat vets with PTSD, then these legislators have some neurological problems of their own. Yet it required precisely this to prove to many that PTSD was an organic brain disorder. The notion that if a neuroscientist can demonstrate it, we know that the person's problem is for real, has a corollary: the fancier the neurobiology utilized, the more reliable the verification. That's simply not true. For example, a good neuropsychologist can discern more of what's happening to someone with subtle, but pervasive memory problems than can a gazillion-dollar brain scanner. It shouldn't take neuroscience to prove what we think and feel. 2. There's been a proliferation of neuro- fields. Some, like neuroendocrinology and neuroimmunology, are stodgy old institutions by now. Others are relatively new: neuroeconomics, neuromarketing, neuroethics, and, I kid you not, neuroliterature and neuroexistentialism. In other words, a hegemonic neuroscientist might conclude that their field explains everything. And with that comes the danger, raised by the New Yorker writer Adam Gopnik under the sardonic banner of neuroskepticism, that explaining everything leads to forgiving everything. 111. This premise is at the heart of debates in the new field of neurology. In Chapter 16 I will argue that it is wrong to think that understanding must lead to forgiveness, mainly because I think that a term like forgiveness, and others related to criminal justice, e.g., evil, soul, volition, and blame, are incompatible with science and should be discarded. 3. Finally, there is the danger of thinking that neuroscience supports a tacit sort of dualism. A guy does something impulsive and awful, and neuroimaging reveals that, unexpectedly, he's missing all his PFC neurons. There's a dualist temptation now to view his behavior as more biological or organic in some nebulous manner than if he had committed the same act with a normal PFC. However, the guy's awful, impulsive act is equally biological with or without a PFC. The sole difference is that the workings of the PFC-less brain are easier to understand with our primitive research tools. So what does all of this tell us? Sometimes these studies tell us what different brain regions do. They are getting fancier, telling us about circuits, thanks to the growing time resolution of neuroimaging, transitioning from this stimulus activates brain regions A, B, C, to this stimulus activates both A and B, and then C, and C activates only if B does. And identifying what specific regions, circuits do gets harder as studies become subtler. Consider, for example, the fusiform face area. As discussed in the next chapter, it is a cortical region that responds to faces in humans and other primates. We primates sure are social creatures. But work by Isabelle Gaudier of Vanderbilt University demonstrates something more complicated. Show pictures of different cars, and the fusiform activates in automobile aficionados. 112. Show pictures of birds, and ditto among bird watchers. The fusiform isn't about faces, It's about recognizing examples of things from categories that are emotionally salient to each individual. Thus, studying behavior is useful for understanding the nature of the brain. Isn't it interesting that behavior arises from the coupling of brain regions X and Y, and sometimes studying the brain is useful for understanding the nature of behavior. Isn't it interesting that brain region A is central to both behavior X and behavior Y? For example, to me the most interesting thing about the amygdala is its dual involvement in both aggression and fear. You can't understand the former without recognizing the relevance of the latter. A final point related to the core of this book: while this neurobiology is mighty impressive, the brain is not where a behavior begins. It's merely the final common pathway by which all the factors in the chapters to come converge and create behavior. N. 3 seconds to minutes before a thing comes from nothing. No brain is an island. Thanks to messages bouncing around your brain, a command has been sent to your muscles to pull that trigger or touch that arm. Odds are that a short time earlier, something outside your brain prompted this to happen, raising this chapter's key questions. What outside stimulus, acting through what sensory channel, and targeting which parts of the brain, prompted this? B. Were you aware of that environmental stimulus? C. What stimuli had your brain made you particularly sensitive to? And, of course, D. What does this tell us about our best and worst behaviors? Varied sensory information can prompt the brain into action. This can be appreciated by considering this variety in other species. Often we're clueless about this because animals can sense things in ranges that we can't, or with sensory modalities we didn't know exist. Thus, you must think like the animal to learn what is happening. We'll begin by seeing how this pertains to the field of ethology, the science of interviewing an animal in its own language. E. Universal rules versus knobby knees. Ethology formed in Europe in the early 20th century in response to an American brand of psychology, behaviorism. Behaviorism descended from the introductions John Watson, the field's famed champion, was B.F. Skinner. Behaviorists cared about universalities of behavior across species. They worshipped a doozy of a seeming universal concerning stimulus and response. Rewarding an organism for a behavior makes the organism more likely to repeat that behavior, while failure to get rewarded, or, worse, punishment for it, makes the organism less likely to repeat it. Any behavior can be made more or less common through operant conditioning, a term Skinner coined, the process of controlling the rewards and punishments in the organism's environment. Thus, for behaviorists, or Skinnerians, a term Skinner labored to make synonymous, virtually any behavior could be shaped into greater or lesser frequency, or even extinguished entirely. If all behaving organisms obeyed these universal rules, you might as well study a convenient species. Most behaviorist research was done on rats, or, Skinner's favorite, pigeons. Behaviorists loved data, no-nonsense hard numbers. These were generated by animals pressing or pecking away at levers in operant conditioning boxes, aka Skinner boxes. And anything discovered applied to any species. A pigeon is a rat is a boy, Skinner preached. Soulless droid. Behaviorists were often right about behavior, but wrong in really important ways, as many interesting behaviors don't follow behaviorist rules. One, raise an infant, rat or monkey with an abusive mother, and it becomes more attached to her. And behaviorist rules have failed when humans love the wrong abusive person. Meanwhile, ethology was emerging in Europe. In contrast with behaviorism's obsession with uniformity and universality of behavior, ethologists loved behavioral variety. They'd emphasize how every species evolves unique behaviors in response to unique demands, and how one had to open-mindedly observe animals in their natural habitats to understand them. Studying rat social behavior in a cage is like studying dolphin swimming behavior in a bathtub, is an ethology adage. They'd ask, what, objectively, is the behavior? What triggered it? Did it have to be learned? How did it evolve? What is the behavior's adaptive value? 19th century Parsons went into nature to collect butterflies, revel in the variety of wing colors, and marvel at what God had wrought. 20th century ethologists went into nature to collect behavior, revel in its variety, and marvel at what evolution had wrought. In contrast to lab coat-clad behaviorists, ethologists tromped around fields in hiking shoes and had fetching knobby knees. Sensory triggers of behavior in some other species. Using an ethological framework, we now consider sensory triggers of behavior in animals. First, there's the auditory channel. Animals vocalize to intimidate, proclaim, and seduce. Birds sing, stags roar, howler monkeys howl, orangutans give territorial calls audible for miles. As a subtle example of information being communicated, when female pandas ovulate, their vocalizations get higher, something preferred by males. Remarkably, the same shift and preference happens in humans. There are also visual triggers of behavior. Dogs crouch to invite play, birds strut their plumage, monkeys display their canines menacingly with threat yawns. And there are visual cues of cute babyness, big eyes, shortened muzzle, round forehead, that drive mammals crazy, motivating them to care for the kid. Stephen Jay Gould noted that the unsung ethologist, Walt Disney, understood exactly what alterations transformed rodents into Mickey and Minnie 3. Then there are animals, signaling in ways we can't detect, requiring creativity to interview an animal in its own language. 4. Scads of mammals scent mark with pheromones, odors that carry information about sex, age, reproductive status, health, and genetic makeup. Some snakes see in infrared, electric eels court with electric songs, bats compete by jamming one another's feeding echolocation signals, and spiders identify intruders by vibration patterns on their webs. How about this? Tickle a rat and it chirps ultrasonically as its mesolimbic dopamine system is activated. Back to the rhinencephalon limbic system war and the resolution ethologists already knew, for a rodent, emotion is typically triggered by olfaction. Across species, the dominant sensory modality, vision, sounds, whichever, has the most direct access to the limbic system. Under the radar, subliminal and unconscious quing. It's easy to see how the sight of a knife, the sound of a voice, calling your name, a touch on your hand can rapidly alter your brain. 5. But crucially, tons of subliminal sensory triggers occur, so fleeting or minimal that we don't consciously note them, or of a type that, even if noted, seems irrelevant to a subsequent behavior. Subliminal quing and unconscious priming influence numerous behaviors unrelated to this book. People think potato chips taste better when hearing crunching sounds. We like a neutral stimulus more if, just before seeing it, a picture of a smiling face is flashed for a 20th of a second. The more expensive a supposed placebo painkiller, the more effective people report the placebo to be. Ask subjects their favorite detergent. If they've just read a paragraph containing the word ocean, they're more likely to choose Tide, and then explain its cleaning virtues. 6. Thus, over the course of seconds, sensory cues can shape your behavior unconsciously. A hugely unsettling sensory cue concerns race. 7. Our brains are incredibly attuned to skin color. Flash of face for less than a tenth of a second, 100 milliseconds, so short a time that people aren't even sure they've seen something. Have them guess the race of the pictured face, and there's a better than even chance of accuracy. We may claim to judge someone by the content of their character rather than by the color of their skin. But our brains sure as hell note the color, real fast. By 100 milliseconds, brain function already differs in two depressing ways, depending on the race of the face as shown with neuroimaging. First, in a widely replicated finding, the amygdala activates. Moreover, the more racist someone is in an implicit test of race bias, stay tuned, the more activation there is. 8. Similarly, repeatedly show subjects a picture of a face accompanied by a shock, soon, seeing the face alone activates the amygdala. 9. As shown by Elizabeth Phelps of NYU, such fear conditioning occurs faster for other race than same race faces. Amygdala are prepared to learn to associate something bad with them. Moreover, people judge neutral other race faces as angrier than neutral same race faces. So if whites see a black face shown at a subliminal speed, the amygdala activates. 10. But if the face is shown long enough for conscious processing, the anterior cingulate and the cognitive DLPFC then activate and inhibit the amygdala. It's the frontal cortex exerting executive control over the deeper, darker amygdaloid response. Second depressing finding, subliminal signaling of race also affects the fusiform face area, the cortical region that specializes in facial recognition. 11. Damaging the fusiform, for example, selectively produces face blindness aka prosopagnosia, an inability to recognize faces. Work by John Gabrielli at MIT demonstrates less fusiform activation for other race faces with the effect strongest in the most implicitly racist subjects. This isn't about novelty, Show a face with purple skin and the fusiform responds as if it's same race. The fusiform isn't fooled that's not an other, it's just a normal photoshopped face. In accord with that, white Americans remember white better than black faces. Moreover, mixed-race faces are remembered better if described as being of a white rather than a black person. Remarkably, if mixed-race subjects are told they've been assigned to one of the two races for the study, they show less fusiform response to faces of the arbitrarily designated other race. 12. Our attunement to race is shown in another way. 2.13. Show a video of someone's hand being poked with a needle and subjects have an isomorphic sensory motor response, hands tense in empathy. Among both whites and blacks, the response is blunted for other race hands. The more the implicit racism, the more blunting. Similarly, among subjects of both races, there's more activation of the emotional, medial PFC when considering misfortune befalling a member of their own race than of another race. This has major implications. In work by Joshua Curl at the University of Colorado, subjects were rapidly shown pictures of people holding either a gun or a cell phone and were told to shoot only gun toters. This is painfully reminiscent of the 1999 killing of Amadou Diallo. Diallo, a West African immigrant in New York, matched a description of a rapist. 4 white officers questioned him, and when the unarmed Diallo started to pull out his wallet, they decided it was a gun and fired 41 shots. The underlying neurobiology concerns event-related potentials (ERPs), which are stimulus-induced changes in electrical activity of the brain, as assessed by EEG, electroencephalography. Threatening faces produce a distinctive change, called the P200 component, in the ERP waveform in under 200 milliseconds. Among white subjects, viewing someone black evokes a stronger P200 waveform than viewing someone white, regardless of whether the person is armed. Then, a few milliseconds later, a second inhibitory waveform, the N200 component, appears, originating from the frontal cortex. Let's think a sec about what we're seeing before we shoot. Viewing a black individual evokes less of an N200 waveform than does seeing someone white. The greater the P200/N200 ratio, i.e., the greater the ratio of I'm feeling threatened to hold on a sec, the greater the likelihood of shooting an unarmed black individual. In another study, subjects had to identify fragmented pictures of objects. Priming white subjects with subliminal views of black, but not white, faces made them better at detecting pictures of weapons, but not cameras or books. 0.14. Finally, for the same criminal conviction, the more stereotypically African a black individual's facial features, the longer the sentence. 0.15. A wonderfully complicating piece of the story. We've now seen how sensory and interoceptive information influence the brain to produce a behavior within seconds to minutes. But as a complication, the brain can alter the sensitivity of those sensory modalities, making some stimuli more influential. As an obvious one, dogs prick up their ears when they're alert. The brain has stimulated ear muscles in a way that enables the ears to more easily detect sounds, which then influences the brain. 0.40. During acute stress, all of our sensory systems become more sensitive. More selectively, if you're hungry, you become more sensitive to the smell of food. How does something like this work? A priori, it seems as if all sensory roads lead to the brain. But the brain also sends neuronal projections to sensory organs. For example, low blood sugar might activate particular hypothalamic neurons. These, in turn, project to and stimulate receptor neurons in the nose that respond to food smells. The stimulation isn't enough to give those receptor neurons action potentials, but it now takes fewer food odorant molecules to trigger one. Something along these lines explains how the brain alters the selective sensitivity of sensory systems. This certainly applies to the behaviors that fill this book. Recall how eyes carry lots of information about emotional state. It turns out that the brain biases us toward preferentially looking at eyes. This was shown by Damasio, studying a patient with Urbach-Witt disease, which selectively destroys the amygdala. As expected, she was poor at accurately detecting fearful faces. But in addition, while control subjects spent about half their face-gazing time looking at eyes, she spent half that. When instructed to focus on the eyes, she improved at recognizing fearful expressions. Thus, not only does the amygdala detect fearful faces, but it also biases us toward obtaining information about fearful faces. 0.41. Psychopaths are typically poor at recognizing fearful expressions, though they accurately recognize other types. 42. They also look less at eyes than normal and improve at fear recognition when directed to focus on eyes. This makes sense, given the amygdaloid abnormalities in psychopaths noted in Chapter 2. Now an example foreshadowing Chapter 9's focus on culture. Show subjects a picture of an object embedded in a complex background. Within seconds, people from collectivist cultures, e.g., China, tend to look more at and remember better the surrounding contextual information, while people from individualistic cultures, e.g., the United States, do the same with the focal object. Instruct subjects to focus on the domain that their culture doesn't gravitate toward, and there's frontal cortical activation. This is a difficult perceptual task. Thus, culture literally shapes how and where you look at the world. 43. N. Conclusions. Brain operates in a vacuum, and over the course of seconds to minutes, the wealth of information streaming into the brain influences the likelihood of prior antisocial acts. As we've seen, pertinent information ranges from something as simple and unidimensional as shirt color to things as complex and subtle as cues about ideology. Moreover, the brain also constantly receives interoceptive information. And most important, much of these varied types of information is subliminal. Ultimately, the most important point of this chapter is that in the moments just before we decide upon some of our most consequential acts, we are less rational and autonomous decision makers than we like to think. 4 hours to days before. Now take the next step back in our chronology, considering events from hours to days before a behavior occurs. To do so, we enter the realm of hormones. What are the effects of hormones on the brain and sensory systems that filled the last 2 chapters? How do hormones influence our best and worst behaviors? While this chapter examines various hormones, the most attention is paid to one inextricably tied to aggression, namely testosterone. And as the punchline, testosterone is far less relevant to aggression than usually assumed. At the other end of the spectrum, the chapter also considers a hormone with cult status for fostering warm, fuzzy prosociality, namely oxytocin. As we'll see, it's not quite as groovy as assumed. Those who are unfamiliar with hormones and endocrinology, please see the primer in Appendix 2. Testosterone's bum rap. Testosterone is secreted by the testes as the final step in the hypothalamic/pituitary/testicular axis. It has effects on cells throughout the body, including neurons, of course. And testosterone is everyone's usual suspect when it comes to the hormonal causes of aggression. Correlation and causality. Why is it that throughout the animal kingdom, and in every human culture, males account for most aggression and violence? Well, what about testosterone and some related hormones, collectively called androgens, a term that, unless otherwise noted, I will use simplistically as synonymous with testosterone? In nearly all species, males have more circulating testosterone than do females, who secrete small amounts of androgens from the adrenal glands. Moreover, male aggression is most prevalent when testosterone levels are highest adolescence and during mating season in seasonal breeders. Thus, testosterone and aggression are linked. Furthermore, there are particularly high levels of testosterone receptors in the amygdala in the way station, by which it projects to the rest of the brain, the bed nucleus of the stria terminalis, and in its major targets, the hypothalamus, the central gray of the midbrain, and the frontal cortex. But these are merely correlative data. Showing that testosterone causes aggression requires a subtraction plus a replacement experiment. Subtraction, castrate a male. Do levels of aggression decrease? Yes, including in humans. This shows that something coming from the testes causes aggression. Is it testosterone? Replacement. Give that castrated individual replacement testosterone. Do pre-castration levels of aggression return? Yes, including in humans. Thus, testosterone causes aggression. Time to see how wrong that is. The first hint of a complication comes after castration, when average levels of aggression plummet in every species. But, crucially, not to zero. Well, maybe the castration wasn't perfect, you missed some bits of testes. Or maybe enough of the minor adrenal androgens are secreted to maintain the aggression. But no, even when testosterone and androgens are completely eliminated, some aggression remains. Thus, some male aggression is testosterone independent. This point is driven home by castration of some sexual offenders, a legal procedure in a few states. Point 1. This is accomplished with chemical castration, administration of drugs that either inhibit testosterone production or block testosterone receptors. Castration decreases sexual urges in the subset of sex offenders with intense, obsessive, and pathological urges. But otherwise, castration doesn't decrease recidivism rates. As stated in one meta-analysis, hostile rapists and those who commit sex crimes motivated by power or anger are not amenable to treatment with the anti-androgenic drugs. This leads to a hugely informative point. The more experience a male had being aggressive prior to castration, the more aggression continues afterward. In other words, the less his being aggressive in the future requires testosterone, and the more, it's a function of social learning. On to the next issue that lessens the primacy of testosterone. What do individual levels of testosterone have to do with aggression? If one person has higher testosterone levels than another, or higher levels this week than last, are they more likely to be aggressive? Initially, the answer seemed to be yes, as studies showed correlation between individual differences in testosterone levels and levels of aggression. In a typical study, higher testosterone levels would be observed in those male prisoners with higher rates of aggression. But being aggressive stimulates testosterone secretion. No wonder more aggressive individuals had higher levels. Such studies couldn't disentangle chickens and eggs. Thus, a better question is whether differences in testosterone levels among individuals predict who will be aggressive. And among birds, fish, mammals, and especially other primates, the answer is generally no. This has been studied extensively in humans, examining a variety of measures of aggression. And the answer is clear. To quote the British endocrinologist John Archer in a definitive 2006 review, there is a weak and inconsistent association between testosterone levels and aggression in human adults, and administration of testosterone to volunteers typically does not increase their aggression. The brain doesn't pay attention to fluctuations of testosterone levels within the normal range. Point 2. Things differ when levels are made superphysiological, higher than the body normally generates. This is the world of athletes and bodybuilders abusing high-dose testosterone like anabolic steroids. In that situation, risk of aggression does increase. 2 complications. It's not random who would choose to take these drugs, and abusers are often already predisposed toward aggression. Superphysiological levels of androgens generate anxiety and paranoia, and increased aggression may be secondary to that. 3. Thus, aggression is typically more about social learning than about testosterone, and differing levels of testosterone generally can't explain why some individuals are more aggressive than others. So what does testosterone actually do to behavior? Subtleties of testosterone effects. When looking at faces expressing strong emotions, we tend to make micro-expressions that mimic them. Testosterone decreases such empathic mimicry. 4. Moreover, testosterone makes people less adept at identifying emotions by looking at people's eyes, and faces of strangers activate the amygdala more than familiar ones and are rated as less trustworthy. Testosterone also increases confidence and optimism while decreasing fear and anxiety. Point 5. This explains the winner effect in lab animals, where winning a fight increases an animal's willingness to participate in, and its success in, another such interaction. Part of the increased success probably reflects the fact that winning stimulates testosterone secretion, which increases glucose delivery and metabolism in the animal's muscles and makes his pheromone smell scarier. Moreover, winning increases the number of testosterone receptors in the bed nucleus of the stria terminalis, the way station through which the amygdala communicates with the rest of the brain, increasing its sensitivity to the hormone. Success in everything from athletics to chess to the stock market boosts testosterone levels. Confident and optimistic. Well, endless self-help books urge us to be precisely that. But testosterone makes people overconfident and overly optimistic, with bad consequences. In one study, pairs of subjects could consult each other before making individual choices in a task. Testosterone made subjects more likely to think their opinion was correct and to ignore input from their partner. Testosterone makes people cocky, egocentric, and narcissistic. Point 6. Testosterone boosts impulsivity and risk-taking, making people do the easier thing when it's the dumbass thing to do. Point 7. Testosterone does this by decreasing activity in the prefrontal cortex and its functional coupling to the amygdala and increasing amygdaloid coupling with the thalamus, the source of that shortcut path of sensory information into the amygdala. Thus, more influence by split-second, low-accuracy inputs and less by the let's stop and think about this frontal cortex. Being fearless, overconfident, and delusionally optimistic sure feels good. No surprise, then, that testosterone can be pleasurable. Rats will work by pressing levers to be infused with testosterone and show conditioned place preference, returning to a random corner of the cage where infusions occur. I don't know why, but I feel good whenever I stand there. 8-9. The underlying neurobiology fits perfectly. Dopamine is needed for place preference conditioning to occur, and testosterone increases activity in the ventral tegmentum, the source of those mesolimbic and mesocortical dopamine projections. Moreover, conditioned place preference is induced when testosterone is infused directly into the Nucleus Accumbens, the ventral tegmentum's main projection target. When a rat wins a fight, the number of testosterone receptors increases in the ventral tegmentum and accumbens, increasing sensitivity to the hormone's feel-good effects. 10. So testosterone does subtle things to behavior. Nonetheless, this doesn't tell us much because everything can be interpreted every which way. Testosterone increases anxiety, you feel threatened and become more reactively aggressive. Testosterone decreases anxiety, you feel cocky and overconfident, become more preemptively aggressive. Testosterone increases risk-taking, hey, let's gamble and invade. Testosterone increases risk-taking, hey, let's gamble and make a peace offer. Testosterone makes you feel good, let's start another fight, since the last one went swell. Testosterone makes you feel good, let's all hold hands. It's a crucial unifying concept that testosterone's effects are hugely context-dependent. Contingent testosterone effects. This context dependency means that rather than causing X, testosterone amplifies the power of something else to cause X. A classic example comes from a 1977 study of groups of male Talapoin monkeys. 11. Testosterone was administered to the middle-ranking male in each group, say, rank number 3 out of 5, increasing their levels of aggression. Does this mean that these guys, stoked on roids, started challenging numbers 1 and 2 in the hierarchy? No. They became aggressive jerks to poor numbers 4 and 5. Testosterone did not create new social patterns of aggression, it exaggerated pre-existing ones. In human studies, testosterone didn't raise baseline activity in the amygdala, it boosted the amygdala's response and heart rate reactivity to angry faces, but not to happy or neutral ones. Similarly, testosterone did not make subjects more selfish and uncooperative in an economic game, it made them more punitive when provoked by being treated poorly, enhancing vengeful reactive aggression. 12. The context dependency also occurs on the neurobiological level, in that the hormone shortens the refractory period of neurons in the amygdala and amygdaloid targets in the hypothalamus. 13. Recall that the refractory period comes in neurons after action potentials. This is when the neuron's resting potential is hyperpolarized, i.e., when it is more negatively charged than usual, making the neuron less excitable, producing a period of silence after the action potential. Thus, shorter refractory periods mean a higher rate of action potentials. So is testosterone causing action potentials in these neurons? No. It's causing them to fire at a faster rate if they are stimulated by something else. Similarly, testosterone increases amygdala response to angry faces, but not to other sorts. Thus, if the amygdala is already responding to some realm of social learning, testosterone ups the volume. A key synthesis, the challenge hypothesis. Thus, testosterone's actions are contingent and amplifying, exacerbating pre-existing tendencies toward aggression rather than creating aggression out of thin air. This picture inspired the challenge hypothesis, a wonderfully unifying conceptualization of testosterone's actions. 14. As proposed in 1990 by the superb behavioral endocrinologist, John Wingfield of the University of California at Davis and colleagues, the idea is that rising testosterone levels increase aggression only at the time of a challenge. Which is precisely how things work. He explains why basal levels of testosterone have little to do with subsequent aggression, and why increases in testosterone due to puberty, sexual stimulation, or the start of mating season don't increase aggression either. 15. But things are different during challenges. Point 16. Among various primates, testosterone levels rise when a dominance hierarchy first forms or undergoes reorganization. Testosterone rises in humans in both individual and team sports competition, including basketball, wrestling, tennis, rugby, and judo. There's generally a rise in anticipation of the event and a larger one afterward, especially among winners. Remarkably, watching your favorite team win raises testosterone levels, showing that the rise is less about muscle activity than about the psychology of dominance, identification, and self-esteem. Most important, the rise in testosterone after a challenge makes aggression more likely. Point 17. Think about this. Testosterone levels rise, reaching the brain. If this occurs because someone is challenging you, you head in the direction of aggression. If an identical rise occurs because days are lengthening and mating season is approaching, you decide to fly 1,000 miles to your breeding grounds. And if the same occurs because of puberty, you get stupid and giggly around that girl who plays clarinet in the band. The context dependency is remarkable. 18. The challenge hypothesis has a second part to it. When testosterone rises after a challenge, it doesn't prompt aggression. Instead, it prompts whatever behaviors are needed to maintain status. This changes things enormously. Well, maybe not, since maintaining status for, say, male primates consists mostly of aggression or threats of it, from slashing your opponent to giving a you have no idea who you're screwing with stare. Point 19. And now for some flabbergastingly important research. What happens if defending your status requires you to be nice? This was explored in a study by Christoph Eisniger and Ernst Farah of the University of Zurich. Point 20. Participants played the Ultimatum Game introduced in Chapter 2, where you decide how to split money between you and another player. The other person can accept the split or reject it, in which case neither of you gets anything. Prior research had shown that, when someone's offer is rejected, they feel dissed, subordinated, especially if news of that carries into future rounds with other players. In other words, in this scenario, status and reputation rest on being fair. And what happens when subjects were given testosterone beforehand? People made more generous offers. What the hormone makes you do depends on what counts as being studly. This requires some fancy neuroendocrine wiring that is sensitive to social learning. You couldn't ask for a finding more counter to testosterone's reputation. The study contained a slick additional finding that further separated testosterone myth from reality. As per usual, subjects got either testosterone or saline, without knowing which. Subjects who believed it was testosterone, independent of whether it actually was, made less generous offers. In other words, testosterone doesn't necessarily make you behave in a crappy manner, but believing that it does and that you're drowning in the stuff makes you behave in a crappy manner. Additional studies show that testosterone promotes prosociality in the right setting. In one, under circumstances where someone's sense of pride rides on honesty, testosterone decreased men's cheating in a game. In another, subjects decided how much of a sum of money they would keep and how much they would publicly contribute to a common pool shared by all the players. Testosterone made most subjects more prosocial. Point 21. What does this mean? Testosterone makes us more willing to do what it takes to attain and maintain status. And the key point is what it takes. Engineer social circumstances right, and boosting testosterone levels during a challenge would make people compete like crazy to do the most acts of random kindness. In our world riddled with male violence, the problem isn't that testosterone can increase levels of aggression. The problem is the frequency with which we reward aggression. Oxytocin and vasopressin, a marketing dream. The point of the preceding section is that testosterone has gotten a bum rap. The point of this one is that oxytocin, and the closely related vasopressin, is coasting in a Teflon presidency. According to lore, oxytocin makes organisms less aggressive, more socially attuned, trusting, and empathic. Individuals treated with oxytocin become more faithful partners and more attentive parents. It makes lab rats more charitable and better listeners, makes fruit flies sing like Joan Baez. Naturally, things are more complicated, and oxytocin has an informative dark side. Basics. Oxytocin and vasopressin are chemically similar hormones. The DNA sequences that constitute their genes are similar, and the two genes occur close to each other on the same chromosome. There was a single ancestral gene that, a few 100 million years ago, was accidentally duplicated in the genome, and the DNA sequences in the two copies of the gene drifted independently, evolving into two closely related genes. Stay tuned for more in Chapter 8. This gene duplication occurred as mammals were emerging. Other vertebrates have only the ancestral version, called oxytocin, which is structurally between the two separate mammalian hormones. For 20th century neurobiologists, oxytocin and vasopressin were pretty boring. They were made in hypothalamic neurons that sent axons to the posterior pituitary. There they would be released into circulation, thereby attaining hormone status, and have nothing to do with the brain ever again. Oxytocin stimulated uterine contraction during labor and milk letdown afterward. Vasopressin, aka antidiuretic hormone, regulated water retention in the kidneys. And reflecting their similar structures, each also had mild versions of the other one's effects. End of story. Neurobiologists take notice. Things became interesting with the discovery that those hypothalamic neurons that made oxytocin and vasopressin also sent projections throughout the brain, including the dopamine-related ventral tegmentum and nucleus accumbens, hippocampus, amygdala, and frontal cortex, all regions with ample levels of receptors for the hormones. Moreover, oxytocin and vasopressin turned out to be synthesized and secreted elsewhere in the brain. These two boring, classical peripheral hormones affected brain function and behavior. They started being called neuropeptides, neuroactive messengers with a peptide structure, which is a fancy way of saying they are small proteins, and to avoid writing oxytocin and vasopressin endlessly, I will refer to them as neuropeptides. Note though that there are other neuropeptides. The initial findings about their behavioral effects made sense. 22. Oxytocin prepares the body of a female mammal for birth and lactation. Logically, oxytocin also facilitates maternal behavior. The brain boosts oxytocin production when a female rat gives birth, thanks to a hypothalamic circuit with markedly different functions in females and males. Moreover, the ventral tegmentum increases its sensitivity to the neuropeptide by increasing levels of oxytocin receptors. Infuse oxytocin into the brain of a virgin rat, and she'll act maternally, retrieving, grooming, and licking pups. Block the actions of oxytocin in a rodent mother, 23, and she'll stop maternal behaviors, including nursing. Oxytocin works in the olfactory system, helping a new mom learn the smell of her offspring. Meanwhile, vasopressin has similar but milder effects. Soon other species were heard from. Oxytocin lets sheep learn the smell of their offspring and facilitates female monkeys grooming their offspring. Spray oxytocin up a woman's nose, a way to get the neuropeptide past the blood-brain barrier and into the brain, and she'll find babies to look more appealing. Moreover, women with variants of genes that produce higher levels of oxytocin or oxytocin receptors average higher levels of touching their infants and more synchronized gazing with them. So oxytocin is central to female mammals nursing, wanting to nurse their child, and remembering which one is their child. Males then got into the act as vasopressin plays a role in paternal behavior. A female rodent giving birth increases vasopressin and vasopressin receptor levels throughout the body, including the brain of the nearby father. Among monkeys, experienced fathers have more dendrites in frontal cortical neurons containing vasopressin receptors. Moreover, administering vasopressin enhances paternal behaviors. However, an ethological caveat, this occurs only in species where males are paternal, e.g., prairie voles and marmoset monkeys. 24. Then, dozens of millions of years ago, some rodent and primate species independently evolved monogamous pair bonding, along with the neuropeptides central to the process. 25. Among marmoset and titty monkeys, which both pair bond, oxytocin strengthens the bond, increasing a monkey's preference for huddling with her partner over huddling with a stranger. Then there was a study that is embarrassingly similar to stereotypical human couples. Among pair bonding tamarin monkeys, lots of grooming and physical contact predicted high oxytocin levels in female members of a pair. What predicted high levels of oxytocin in males? Lots of sex. Beautiful, pioneering work by Thomas Insel of the National Institute of Mental Health, Larry Young of Emory University, and Sue Carter of the University of Illinois has made a species of vole, arguably the most celebrated rodent on Earth. 26. Most voles, e.g., montane voles, are polygamous. In contrast, prairie voles, in a salute to Garrison Kaler, form monogamous mating pairs for life. Naturally, this isn't quite the case. While they are social pair bonders with their permanent relationships, they're not quite perfect, sexual pair bonders, as males might mess around on the side. Nonetheless, prairie voles pair bond more than other voles, prompting Insel, Young, and Carter to figure out why. First finding, sex releases oxytocin and vasopressin in the nucleus accumbens of female and male voles, respectively. Obvious theory, prairie voles release more of the stuff during sex than do polygamous voles, causing a more rewarding buzz, encouraging the individuals to stick with their partner. But prairie voles don't release more neuropeptides than montane voles. Instead, prairie voles have more of the pertinent receptors in the nucleus accumbens than do polygamous voles. Moreover, male prairie voles with a variant of the vasopressin receptor gene that produced more receptors in the nucleus accumbens were stronger pair bonders. Then the scientists conducted two tour de force studies. First, they engineered the brains of male mice to express the prairie vole version of the vasopressin receptor in their brains, and they groomed and huddled more with familiar females, but not with strangers. Then the scientists engineered the brains of male montane voles to have more vasopressin receptors in the nucleus accumbens. The males became more socially affiliative with individual females. What about versions of vasopressin receptor genes in other species? When compared with chimps, bonobos have a variant associated with more receptor expression and far more social bonding between females and males, although in contrast to prairie voles, bonobos are anything but monogamous. 27. How about humans? This is tough to study, because you can't measure these neuropeptides in tiny, brain regions in humans and instead have to examine levels in the circulation, a fairly indirect measure. Nevertheless, these neuropeptides appear to play a role in human pair bonding. 28. For starters, circulating oxytocin levels are elevated in couples when they've first hooked up. Furthermore, the higher the levels, the more physical affection, the more behaviors are synchronized, the more long-lasting the relationship, and the happier interviewers rate couples to be. Even more interesting were studies where oxytocin, or a control spray, was administered intranasally. In one fun study, couples had to discuss one of their conflicts, oxytocin up their noses, and they'd be rated as communicating more positively and would secrete less stress hormones. Another study suggests that oxytocin unconsciously strengthens the pair bond. Heterosexual male volunteers, with or without an oxytocin spritz, interacted with an attractive female researcher doing some nonsense task. Among men in stable relationships, oxytocin increased their distance from the woman an average of 4 to 6 inches. Single guys, no effect. Why didn't oxytocin make them stand closer? The researchers indicated that they were already about as close as one could get away with. If the experimenter was male, no effect. Moreover, oxytocin caused males in relationships to spend less time looking at pictures of attractive women. Importantly, oxytocin didn't make men rate these women as less attractive. They were simply less interested. 29. Thus, oxytocin and vasopressin facilitate bonding between parent and child and between couples. Now for something truly charming that evolution has cooked up recently. Sometime in the last 50,000 years, i.e., less than 0.1% of the time that oxytocin has existed, the brains of humans and domesticated wolves evolved a new response to oxytocin. When a dog and its owner, but not a stranger, interact, they secrete oxytocin. 30. The more of that time is spent gazing at each other, the bigger the rise. Give dogs oxytocin, and they gaze longer at their humans, which raises the human's oxytocin levels. So a hormone that evolved for mother-infant bonding plays a role in this bizarre, unprecedented form of bonding between species. In line with its effects on bonding, oxytocin inhibits the central amygdala, suppresses fear and anxiety, and activates the calm, vegetative, parasympathetic nervous system. Moreover, people with an oxytocin receptor gene variant associated with more sensitive parenting also have less of a cardiovascular startle response. In the words of Sue Carter, exposure to oxytocin is a physiological metaphor for safety. Furthermore, oxytocin reduces aggression in rodents and mice whose oxytocin system was silenced by deleting the gene for oxytocin, or its receptor, were abnormally aggressive. 31. Other studies showed that people rate faces as more trustworthy, and are more trusting in economic games when given oxytocin. Oxytocin had no effect when someone thought they were playing with a computer, showing that this was about social behavior. 32. This increased trust was interesting. Normally, if the other player does something duplicitous in the game, subjects are less trusting in subsequent rounds. In contrast, oxytocin-treated investors didn't modify their behavior in this way. Stated scientifically, oxytocin inoculated betrayal aversion among investors. Stated caustically, oxytocin makes people irrational dupes. Stated more angelically, oxytocin makes people turn the other cheek. More prosocial effects of oxytocin emerged. It made people better at detecting happy versus angry, fearful or neutral faces or words with positive versus negative social connotations when these were displayed briefly. Moreover, oxytocin made people more charitable. People with the version of the oxytocin receptor gene associated with more sensitive parenting were rated by observers as more prosocial when discussing a time of personal suffering, as well as more sensitive to social approval. And the neuropeptide made people more responsive to social reinforcement, enhancing performance in a task where correct or wrong answers elicited a smile or frown, respectively, while having no effect when right and wrong answers elicited different colored lights. 33. So oxytocin elicits prosocial behavior, and oxytocin is released when we experience prosocial behavior, being trusted in a game, receiving a warm touch, and so on. In other words, a warm and fuzzy positive feedback loop. 34. Obviously, oxytocin and vasopressin are the grooviest hormones in the universe. Pour them into the water supply, and people will be more charitable, trusting, and empathic. We'd be better parents and would make love, not war, mostly platonic love, though, since people in relationships would give wide berths to everyone else. Best of all, we'd buy all sorts of useless crap, trusting the promotional banners in stores once oxytocin starts spraying out of the ventilation system. Okay, time to settle down a bit. Prosociality versus sociality. Are oxytocin and vasopressin about prosociality or social competence? Do these hormones make us see happy faces everywhere or become more interested in gathering accurate, social information about faces? The latter isn't necessarily prosocial. After all, accurate information about someone's emotions makes them easier to manipulate. The groovy neuropeptide school supports the idea of ubiquitous prosociality. 35. But the neuropeptides also foster social interest and competence. They make people look at eyes longer, increasing accuracy in reading emotions. Moreover, oxytocin enhances activity in the temporoparietal juncture, that region involved in theory of mind, when people do a social recognition task. The hormone increases the accuracy of assessments of other people's thoughts. With a gender twist, women improve at detecting kinship relations, while men improve at detecting dominance relations. In addition, oxytocin increases accuracy in remembering faces and their emotional expressions, and people with a sensitive parenting, oxytocin receptor gene variant are particularly adept at assessing emotions. Similarly, the hormones facilitate rodents' learning of an individual's smell, but not non-social odors. Neuroimaging research shows that these neuropeptides are about social competence, as well as prosociality. 36. For example, variants of a gene related to oxytocin signaling are associated with differing degrees of activation of the fusiform face area when looking at faces. Findings like these suggest that abnormalities in these neuropeptides increase the risk of disorders of impaired sociality, namely autism spectrum disorders, ASD. Strikingly, people with ASD show blunted fusiform responses to faces. 37. Remarkably, ASD has been linked to gene variants related to oxytocin and vasopressin, to non-genetic mechanisms for silencing the oxytocin receptor gene, and to lower levels of the receptor itself. Moreover, the neuropeptides improve social skills in some individuals with ASD, e.g. enhancing eye contact. Thus, sometimes oxytocin and vasopressin make us more prosocial, but sometimes they make us more avid and accurate social information gatherers. Nonetheless, there is a happy face bias, since accuracy is most enhanced for positive emotions. 38. Time for more complications. Contingent effects of oxytocin and vasopressin. Recall testosterone's contingent effects, e.g., making a monkey more aggressive, but only toward individuals he already dominates. Naturally, these neuropeptides' effects are also contingent. 39. One factor already mentioned is gender. Oxytocin enhances different aspects of social competence in women and men. Moreover, oxytocin's calming effects on the amygdala are more consistent in men than in women. Predictably, neurons that make these neuropeptides are regulated by both estrogen and testosterone. 40. As a really interesting contingent effect, oxytocin enhances charitability, but only in people who are already so. This mirrors testosterone's only raising aggression in aggression-prone people. Hormones rarely act outside the context of the individual and his or her environment. 41. Finally, a fascinating study shows cultural contingencies in oxytocin's actions. 42. During stress, Americans seek emotional support, e.g., telling a friend about their problem, more readily than do East Asians. In one study, oxytocin receptor gene variants were identified in American and Korean subjects. Under unstressful circumstances, neither cultural background nor receptor variant affected support-seeking behavior. During stressful periods, support-seeking rose among subjects with the receptor variant associated with enhanced sensitivity to social feedback and approval, but only among the Americans, including Korean Americans. What does oxytocin do to support-seeking behavior? It depends on whether you're stressed, and on the genetic variant of your oxytocin receptor, and on your culture. More to come in chapters 8 and 9. And the dark side of these neuropeptides. As we saw, oxytocin and vasopressin decreases aggression in rodent females. Except for aggression in defense of one's pups, which the neuropeptide increases via effects in the central amygdala with its involvement in instinctual fear. 43. This readily fits with these neuropeptides enhancing maternalism, including snarling don't get one step closer maternalism. Similarly, vasopressin enhances aggression in paternal, prairie vole males. This finding comes with a familiar additional contingency. The more aggressive the male prairie vole, the less that aggression decreases after blocking of his vasopressin system. Just as in the case of testosterone, with increased experience, aggression is maintained by social learning rather than by a hormone/neuropeptide. Moreover, vasopressin increases aggression most in male rodents who are already aggressive, yet another biological effect, depending on individual and social context. 44. And now to really upend our view of these feel-good neuropeptides. For starters, back to oxytocin enhancing trust and cooperation in an economic game, but not if the other player is anonymous and in a different room. When playing against strangers, oxytocin decreases cooperation, enhances envy when luck is bad, and enhances gloating when it's good. 45. Finally, beautiful studies by Karsten de Drew of the University of Amsterdam showed just how unwarm and unfuzzy oxytocin can be. 46. In the first, male subjects formed two teams. Each subject chose how much of his money to put into a pot shared with teammates. As usual, oxytocin increased such generosity. Then participants played the prisoner's dilemma with someone from the other team when financial stakes were high, making subjects more motivated. Oxytocin made them more likely to preemptively stab the other player in the back. Thus, oxytocin makes you more prosocial to people like you, i.e., your teammates, but spontaneously lousy to others who are a threat. As emphasized by de Drew, perhaps oxytocin evolved to enhance social competence to make us better at identifying who is in us. In de Drew's second study, Dutch student subjects took the implicit association test of unconscious bias and oxytocin exaggerated biases against two outgroups, namely Middle Easterners and Germans. 47. Then came the study's truly revealing second part. Subjects had to decide whether it was okay to kill one person in order to save 5. In the scenario, the potential sacrificial lamb's name was either stereotypically Dutch, Dirk or Peter, German, Marcus or Helmut, or Middle Eastern, Ahmed or Yusuf. The 5 people in danger were unnamed. Remarkably, oxytocin made subjects less likely to sacrifice good old Dirk or Peter, rather than Helmut or Ahmed. Oxytocin, the love hormone, makes us more prosocial to us and worse to everyone else. That's not generic prosociality. That's ethnocentrism and xenophobia. In other words, the actions of these neuropeptides depend dramatically on context, who you are, your environment, and who that person is. As we will see in Chapter 8, the same applies to the regulation of genes relevant to these neuropeptides. H. The endocrinology of aggression in females. ELP. This topic confuses me. Here's why. This is a domain where the ratios of two hormones can matter more than their absolute levels, where the brain responds the same way to 2 units of estrogen plus 1 unit of progesterone and 2 gazillion units of estrogen plus 1 gazillion units of progesterone. This requires some complex neurobiology. Hormone levels are extremely dynamic, with hundredfold changes in some within hours. No male's testes ever had to navigate the endocrinology of ovulation or childbirth. Among other things, recreating such endocrine fluctuations in lab animals is tough. There's dizzying variability across species. Some breed year-round, others only in particular seasons. Nursing inhibits ovulation in some, stimulates it in others. Progesterone rarely works in the brain as itself. Instead, it's usually converted into various neurosteroids with differing actions in different brain regions. And estrogen describes a soup of related hormones, none of which work identically. Finally, one must debunk the myth that females are always nice and affiliative, unless, of course, they're aggressively protecting their babies, which is cool and inspirational. Maternal aggression. Levels of aggression rise in rodents during pregnancy, peaking around parturition 48 appropriately. The highest levels occur in species and breeds with the greatest threat of infanticide. 49. During late pregnancy, estrogen and progesterone increase maternal aggression by increasing oxytocin release in certain brain regions, bringing us back to oxytocin promoting maternal aggression. 50. Two complications illustrate some endocrine principles. Estrogen contributes to maternal aggression. But estrogen can also reduce aggression and enhance empathy and emotional recognition. It turns out there are two different types of receptors for estrogen in the brain, mediating these opposing effects, and with their levels independently regulated. Thus, same hormone, same levels, different outcome if the brain is set up to respond differently. 51. The other complication, as noted, progesterone, working with estrogen, promotes maternal aggression. However, on its own it decreases aggression and anxiety. Same hormone, same levels, diametrically opposite outcomes depending on the presence of a second hormone. 52. Progesterone decreases anxiety through a thoroughly cool route. When it enters neurons, it is converted to another steroid. This binds to GABA receptors, making them more sensitive to the inhibitory effects of GABA, thereby calming the brain. Thus, direct crosstalk between hormones and neurotransmitters. Bare-knuckled female aggression. The traditional view is that other than maternal aggression, any female-male competition is passive, cover. As noted by the pioneering primatologist Sarah Blaffer-Hurry of the University of California at Davis, before the 1970s hardly anyone even researched competition among females. 53. Nevertheless, there is plenty of female-female aggression. This is often dismissed with a psychopathology argument. If, say, a female chimp is murderous, it's because, well, she's crazy. Or female aggression is viewed as endocrine spillover. 54. Females synthesize small amounts of androgens in the adrenals and ovaries. In the spillover view, the process of synthesizing real female steroid hormones is somewhat sloppy, and some androgenic steroids are inadvertently produced. Since evolution is lazy and hasn't eliminated androgen receptors in female brains, there's some androgen-driven aggression. These views are wrong for a number of reasons. Female brains don't contain androgen receptors simply because they come from a similar blueprint as male brains. Instead, androgen receptors are distributed differently in the brains of females and males, with higher levels in some regions in females. There has been active selection for androgen effects in females. 55. Even more important, female aggression makes sense. Females can increase their evolutionary fitness with strategic, instrumental aggression. 56. Depending on the species, females compete aggressively for resources, e.g., food or nesting places, harass lower-ranking reproductive competitors into stress-induced infertility, or kill each other's infants as in chimps. And in the bird and rare primate species where males are actually paternal, females compete aggressively for such princes. Remarkably, there are even species, primates, bonobos, lemurs, marmosets, and tamarins, rock hyraxes, and rodents, the California mouse, Syrian golden hamsters, and naked mole rats, where females are socially dominant and more aggressive, and often more muscular, than males. 57. The most celebrated example of a sex reversal system is the spotted hyena, shown by Lawrence Frank of UC Berkeley and colleagues. Among typical social carnivores, e.g., lions, females do most of the hunting, after which males show up and eat first. Among hyenas it's the socially subordinate males who hunt, they are then booted off the kill by females so that the kids eat first. Get this, in many mammals erections are a sign of dominance, of a guy strutting his stuff. Among hyenas it's reversed, when a female is about to terrorize a male. He gets an erection. Please don't hurt me. Look, I'm just a non-threatening male. What explains female competitive aggression in sex reversal species or normal animals? Those androgens in females are obvious suspects, and in some sex reversal species females have androgen levels that equal or even trump those in males. 0.58. Among hyenas where this occurs, spending fetal life awash in mom's plentiful androgens produces a pseudo-hermaphrodite. Female hyenas have a fake scrotal sac, no external vagina, and a clitoris that is as large as a penis and gets erect as well. Moreover, some of the sex differences in the brain seen in most mammals don't occur in hyenas or naked mole rats, reflecting their fetal androgenization. This suggests that elevated female aggression in sex reversal species arises from the elevated androgen exposure and, by extension, that the diminished aggression among females of other species comes from their low androgen levels. But complications emerge. For starters, there are species, e.g., Brazilian guinea pigs, where females have high androgen levels, but aren't particularly aggressive or dominant toward males. Conversely, there are sex reversal bird species without elevated androgen levels in females. Moreover, as with males, individual levels of androgens in females, whether in conventional or sex reversal species, do not predict individual levels of aggression. And most broadly, androgen levels don't tend to rise around periods of female aggression. 0.59. This makes sense. Female aggression is mostly related to reproduction and infant survival, maternal aggression obviously, but also female competition for mates, nesting places, and much-needed food during pregnancy or lactation. Androgens disrupt aspects of reproduction and maternal behavior in females. As emphasized by Hurry, females must balance the progression advantages of androgens with their anti-reproductive disadvantages. Ideally, then androgens in females should affect the aggression parts of the brain but not the reproduction/maternalism parts. Which is precisely what has evolved, as it turns out. 60. Perimenstrual aggression and irritability. Inevitably we turn to premenstrual syndrome, PMS, the symptoms of negative mood and irritability that come around the time of menstruation, along with the bloating of water retention, cramps, acne. There's a lot of baggage and misconceptions about PMS, along with PMDD, premenstrual dysphoric disorder, where symptoms are severe enough to impair normal functioning. It affects 2 to 5% of women. 0.61. The topic is mired in two controversies: what causes PMS/PMDD, and how is it relevant to aggression? The first is a doozy. Is PMS/PMDD a biological disease or a social construct? In the extreme, it's just a social construct. School, PMS is entirely culture-specific, meaning it occurs only in certain societies. Margaret Mead started this by asserting in 1928 in Coming of Age in Samoa that Samoan women don't have mood or behavioral changes when menstruating. Since the Samoans were enshrined by Mead as the coolest, most peaceful and sexually free primates east of bonobos, this started trendy anthropological claims that women in other hip, minimal clothing cultures had no PMS either, and naturally, cultures with rampant PMS, e.g., American primates, were anti-Samoans, where symptoms arose from mistreatment and sexual repression of women. This view even had room for a socioeconomic critique, with howlers like PMS is a mode for the expression of women's anger, resulting from her oppressed position in American capitalist society. 62. An offshoot of this view is the idea that in such repressive societies, it's the most repressed women who have the worst PMS. Thus, depending on the paper, women with bad PMS must be anxious, depressed, neurotic, hypochondriacal, sexually repressed, toadies of religious repression, or more compliant with gender stereotypes, and must respond to challenge by withdrawing, rather than by tackling things head-on. In other words, not a single cool Samoan among them. Fortunately, this has mostly subsided. Numerous studies show normal shifts in the brain and behavior over the course of the reproductive cycle, with as many behavioral correlates of ovulation as of menses. 63. PMS, then, is simply a disruptively extreme version of those shifts. While PMS is real, symptoms vary by culture. For example, perimenstrual women in China report less negative effect than do Western women, raising the issue of whether they experience less and/or report less. Given the more than 100 symptoms linked to PMS, it's not surprising if different symptoms predominate in different populations. As strong evidence that perimenstrual mood and behavioral changes are biological, they occur in other primates. 0.64. Both female baboons and female vervet monkeys become more aggressive and less social before their menses, without, to my knowledge, having issues with American capitalism. Interestingly, the baboon study showed increased aggressiveness only in dominant females. Presumably, subordinate females simply couldn't express increased aggressiveness. All these findings suggest that the mood and behavioral shifts are biologically based. What is a social construct is medicalizing and pathologizing these shifts as symptoms, a syndrome, or disorder. Thus, what is the underlying biology? A leading theory points to the plunging levels of progesterone as menses approaches, and thus the loss of its anxiolytic and sedating effects. In this view, PMS arises from too extreme of a decline. However, there's not much actual support for this idea. Another theory, backed by some evidence, concerns the hormone beta-endorphin, famed for being secreted during exercise and inducing a gauzy, euphoric runner's high. In this model, PMS is about abnormally low levels of beta-endorphin. There are plenty more theories, but very little certainty. Now for the question of how much PMS is associated with aggression. In the 1960s, studies by Katharina Dalton, who coined the term premenstrual syndrome in 1953, reported that female criminals committed their crimes disproportionately during their perimenstrual period, which may tell less about committing a crime than about getting caught. 65. Other studies of a boarding school showed a disproportionate share of bad marks for behavioral offenses going to perimenstrual students. However, the prison studies didn't distinguish between violent and non-violent crimes, and the school study didn't distinguish between aggressive acts and infractions like tardiness. Collectively, there is little evidence that women tend toward aggression around their menses, or that violent women are more likely to have committed their acts around their menses. Nevertheless, defense pleas of PMS-related diminished responsibility have been successful in courtrooms. 66. A notable 1980 case concerns Sandy Craddock, who murdered a coworker and had a long rap sheet with more than 30 convictions for theft, arson, and assault. Incongruously but fortuitously, Craddock was a meticulous diarist, having years of records of not just when she was having her period, but also when she was out about town on a criminal spree. Her criminal acts and times of menses matched so closely that she was put on probation plus progesterone treatment. And making the case stranger, Craddock's doctor later reduced her progesterone dose. By her next period, she had been arrested for attempting to knife someone. Probation again, plus a wee bit more progesterone. These studies suggest that a small number of women do show perimenstrual behavior that qualifies as psychotic and should be mitigating in a courtroom. Nevertheless, normal garden variety perimenstrual shifts in mood and behavior are not particularly associated with increased aggression. Stress and imprudent brain function. Time before some of our most important, consequential behaviors can be filled with stress. Which is too bad, since stress influences the decisions we make, rarely for the better. The basic dichotomy of the acute and the chronic stress response. We begin with a long-forgotten term from 9th grade biology. Remember homeostasis? It means having an ideal body temperature, heart rate, glucose level, and so on. A stressor is anything that disrupts homeostatic balance, say, being chased by a lion if you're a zebra, or chasing after a zebra if you're a hungry lion. The stress response is the array of neural and endocrine changes that occur in that zebra or lion, designed to get them through that crisis and re-establish homeostasis. 67. Critical events in the brain mediate the start of the stress response. Warning, the next two paragraphs are technical and non-essential. The sight of the lion activates the amygdala. Amygdaloid neurons stimulate brain stem neurons, which then inhibit the parasympathetic nervous system and mobilize the sympathetic nervous system, releasing epinephrine and norepinephrine throughout the body. The amygdala also mediates the other main branch of the stress response, activating the paraventricular nucleus, PVN, in the hypothalamus. And the PVN sends projections to the base of the hypothalamus where it secretes corticotropin-releasing hormone, CRH. This triggers the pituitary to release adrenocorticotropic hormone, ACTH, which stimulates glucocorticoid secretion from the adrenals. Glucocorticoids plus the sympathetic nervous system enable an organism to survive a physical stressor by activating the classical fight or flight response. Whether you are that zebra or that lion, you'll need energy for your muscles, and the stress response rapidly mobilizes energy into circulation from storage sites in your body. Furthermore, heart rate and blood pressure increase, delivering that circulating energy to exercising muscles faster. Moreover, during stress, long-term building projects, growth, tissue repair, and reproduction are postponed until after the crisis. After all, if the lion is chasing you, you have better things to do with your energy than, say, thicken your uterine walls. Beta endorphin is secreted, the immune system is stimulated, and blood clotting is enhanced, all useful following painful injury. Moreover, glucocorticoids reach the brain, rapidly enhancing aspects of cognition and sensory acuity. This is wonderfully adaptive for the zebra or lion. Try sprinting without epinephrine and glucocorticoids, and you'll soon be dead. Reflecting its importance, this basic stress response is ancient physiology, found in mammals, birds, fish, and reptiles. What is not ancient is how stress works in smart, socially sophisticated, recently evolved primates. For primates, the definition of a stressor expands beyond merely a physical challenge to homeostasis. In addition, it includes thinking you're going to be thrown out of homeostasis. An anticipatory stress response is adaptive if there really is a physical challenge coming. However, if you're constantly but incorrectly convinced that you're about to be thrown out of balance, you're being an anxious, neurotic, paranoid, or hostile primate who is psychologically stressed. And the stress response did not evolve for dealing with this recent mammalian innovation. Mobilizing energy while sprinting for your life helps save you. Do the same thing chronically, because of a stressful, 30-year mortgage, and you're at risk for various metabolic problems, including adult-onset diabetes. Likewise with blood pressure, increase it to sprint across the savannah, good thing. Increase it because of chronic psychological stress, and you've got stress-induced hypertension. Chronically impair growth and tissue repair, and you'll pay the price. Ditto for chronically inhibiting reproductive physiology. You'll disrupt ovulatory cycles in women and cause plummeting, erections, and testosterone levels in men. Finally, while the acute stress response involves enhanced immunity, chronic stress suppresses immunity, increasing vulnerability to some infectious diseases. We have a dichotomy: if you're stressed like a normal mammal in an acute physical crisis, the stress response is life-saving. But if instead you chronically activate the stress response for reasons of psychological stress, your health suffers. It is a rare human who sickens because they can't activate the stress response when it is needed. Instead, we get sick from activating the stress response too often, too long, and for purely psychological reasons. Crucially, the beneficial effects of the stress response for sprinting, zebras and lions play out over the course of seconds to minutes. But once you take stress to the time course of this chapter, henceforth referred to as sustained stress, you'll be dealing with adverse consequences, including some unwelcome effects on the behaviors that fill this book. A brief digression: stress that we love. Either running from a lion or dealing with years of traffic jams is a drag, which contrasts with stress that we love. 68. We love stress that is mild and transient and occurs in a benevolent context. The stressful menace of a roller coaster ride is that it will make us queasy, not that it will decapitate us. It lasts for 3 minutes, not 3 days. We love that kind of stress, clamor for it, pay to experience it. What do we call that optimal amount of stress? Being engaged, engrossed, and challenged. Being stimulated. Playing. The core of psychological stress is loss of control and predictability. But in benevolent settings, we happily relinquish control and predictability to be challenged by the unexpected: a dip in the roller coaster tracks, a plot twist, a difficult line drive heading our way, an opponent's unexpected chess move. Surprise me, this is fun. This brings up a key concept, namely the inverted U. The complete absence of stress is aversively boring. Moderate. Transient stress is wonderful, various aspects of brain function are enhanced, glucocorticoid levels in that range enhance dopamine release. Rats work at pressing levers in order to be infused with just the right amount of glucocorticoids. And as stress becomes more severe and prolonged, those good effects disappear, with, of course, dramatic individual differences as to where the transition from stress as stimulatory to overstimulatory occurs. One person's nightmare is another's hobby. Visit bit.ly/2ngw6bq for a larger version of this graph. We love the right amount of stress, would wither without it. But back now to sustained stress and the right side of the inverted U. Sustained stress and the neurobiology of fear. For starters, sustained stress makes people implicitly, i.e., not consciously, look more at angry faces. Moreover, during stress, that sensory shortcut from the thalamus to the amygdala becomes more active, with more excitable synapses. We know the resulting trade-off between speed and accuracy. Compounding things further, glucocorticoids decrease activation of the cognitive, medial PFC during processing of emotional phases. Collectively, stress or glucocorticoid administration decreases accuracy when rapidly assessing emotions of phases. 0.69. Meanwhile, during stress things aren't going great in the amygdala. The region is highly sensitive to glucocorticoids, with lots of glucocorticoid receptors. Stress and glucocorticoids increase excitability of amygdaloid neurons, particularly in the basolateral amygdala, the blob, with its role in learning fear. Thus, this is another contingent hormone action. Glucocorticoids don't cause action potentials in amygdaloid neurons, don't invent excitation. Instead they amplify pre-existing excitation. Stress and glucocorticoids also increase levels of CRH in the blah, and of a growth factor that builds new dendrites and synapses, brain-derived neurotrophic factor, or BDNF. 0.70. Recall from Chapter 2 how during a fearful situation, the amygdala recruits the hippocampus into remembering contextual information about the event, e.g., the amygdala remembers the thief's knife, whereas the hippocampus remembers where the robbery occurred. 0.71. Stress strengthens this recruitment, making the hippocampus a temporary fear-laden suburb of the amygdala. Thanks to these glucocorticoid actions in the amygdala, stress makes it easier to learn a fear association and to consolidate it into a long-term memory. This sets us up for a positive feedback loop. As noted, with the onset of stress, the amygdala indirectly activates the glucocorticoid stress response. And in turn, glucocorticoids increase amygdala excitability. Stress also makes it harder to unlearn fear, to extinguish a conditioned fear association. This involves the prefrontal cortex, which causes fear extinction. By inhibiting the blah, as covered in Chapter 2, stress weakens the PFC's hold over the amygdala. 0.72. Recall what fear extinction is about. You've learned to fearfully associate a light with a shock, but today the light keeps coming on with no shock. Extinction is not passively forgetting that light equals shock. It is the blah actively learning that light no longer equals shock. Thus stress facilitates learning fear associations but impairs learning fear extinction. Sustained stress, executive function, and judgment. Stress compromises other aspects of frontal cortical function. Working memory is disrupted. In one study, prolonged administration of high glucocorticoid levels to healthy subjects impaired working memory into the range seen after frontal cortical damage. Glucocorticoids accomplish this by enhancing norepinephrine signaling in the PFC so much that, instead of causing aroused focus, it induces chicken with its head, cut off cognitive tumult, and by enhancing disruptive signaling from the amygdala to the PFC. Stress also desynchronizes activation in different frontocortical regions, which impairs the ability to shift attention between tasks. 0.73. These stress effects on frontal function also make us perseverative, in a rut, set in our ways, running on automatic, being habitual. We all know this. What do we typically do during a stressful time when something isn't working? The same thing again, many more times, faster and more intensely. It becomes unimaginable that the usual isn't working. This is precisely where the frontal cortex makes you do the harder, but more correct thing: recognize that it's time for a change. Except for a stressed frontal cortex, or one that's been exposed to a lot of glucocorticoids. In rats, monkeys, and humans, stress weakens frontal connections with the hippocampus, essential for incorporating the new information that should prompt shifting to a new strategy, while strengthening frontal connections with more habitual brain circuits. 0.74. Finally, the decreased frontal function and increased amygdaloid function during stress alter risk-taking behavior. For example, the stress of sleep deprivation, or of public speaking, or the administration of high glucocorticoid levels, shifts people from protecting against losses to seeking bigger gains when gambling. This involves an interesting gender difference. In general, major stressors make people of both genders more risk-taking. But moderate stressors bias men toward, and women away from, risk-taking. In the absence of stress, men tend toward more risk-taking than women. Thus, once again, hormones enhance a pre-existing tendency. 0.75. Whether one becomes irrationally risk-taking, failing to shift strategy in response to a declining reward rate, or risk-averse, failing to respond to the opposite, one is incorporating new information poorly. Stated most broadly, sustained stress impairs risk assessment. 0.76. Sustained stress and pro and antisociality. During sustained stress, the amygdala processes emotional sensory information more rapidly, and less accurately, dominates hippocampal function, and disrupts frontocortical function. We're more fearful, our thinking is muddled, and we assess risks poorly and act impulsively out of habit, rather than incorporating new data. 77. This is a prescription for rapid, reactive aggression. Stress and acute administration of glucocorticoids increase such aggression in both rodents and humans. We have two familiar qualifications. A. Rather than creating aggression, stress and glucocorticoids increase sensitivity to social triggers of aggression. B. This occurs most readily in individuals already predisposed toward aggression. As we will see in the next chapter, stress over the course of weeks to months produces a less nuanced picture. There's an additional depressing reason why stress fosters aggression, because it reduces stress. Shock a rat and its glucocorticoid levels and blood pressure rise. With enough shocks, it's at risk for a stress ulcer. Various things can buffer the rat during shocks: running on a running wheel, eating, gnawing on wood in frustration. But a particularly effective buffer is for the rat to bite another rat. Stress-induced, aka frustration-induced, displacement aggression is ubiquitous in various species. Among baboons, for example, nearly half of aggression is this type. A high-ranking male loses a fight and chases a subadult male, who promptly bites a female, who then lunges at an infant. My research shows that within the same dominance rank, the more a baboon tends to displace aggression after losing a fight, the lower his glucocorticoid levels. 78. Humans excel at stress-induced displacement aggression. Consider how economic downturns increase rates of spousal and child abuse. Or consider a study of family violence and pro football. If the local team unexpectedly loses, spousal/partner violence by men increases 10% soon afterward, with no increase when the team won or was expected to lose. And as the stakes get higher, the pattern is exacerbated: a 13% increase after upsets when the team was in playoff contention, a 20% increase when the upset is by a rival. 79. Little is known concerning the neurobiology of displacement aggression, blunting the stress response. I'd guess that lashing out activates dopaminergic reward pathways, a surefire way to inhibit CRH release. 80. Far too often, giving an ulcer helps avoid getting one. More bad news: stress biases us toward selfishness. In one study, subjects answered questions about moral decision-making scenarios after either a social stressor or a neutral situation. Some scenarios were of low emotional intensity: in the supermarket you wait at the meat counter and an elderly man pushes to the front. Would you complain? Others: high intensity, you meet the love of your life, but you are married and have children. Would you leave your family? Stress made people give more egoistic answers about emotionally intense moral decisions, but not milder ones. The more glucocorticoid levels rose, the more egoistic the answers. Moreover, in the same paradigm, stress lessened how altruistic people claimed they'd be concerning personal, but not impersonal, moral decisions. 81. We have another contingent endocrine effect. Stress makes people more egoistic, but only in the most emotionally intense and personal circumstances. This resembles another circumstance of poor frontal function. Recall from Chapter 2 how individuals with frontal cortical damage make reasonable judgments about someone else's issues, but the more personal and emotionally potent the issue, the more they are impaired. Feeling better by abusing someone innocent, or thinking more about your own needs, is not compatible with feeling empathy. Does stress decrease empathy? Seemingly yes, in both mice and humans. A remarkable 2006 paper in Science by Jeffrey Mogul of McGill University showed the rudiments of mouse empathy. A mouse's pain threshold is lowered when it is near another mouse in pain, but only if the other mouse is its cagemate. 82. This prompted a follow-up study that I did with Mogul's group involving the same paradigm. The presence of a strange mouse triggers a stress response. But when glucocorticoid secretion is temporarily blocked, mice show the same pain empathy for a strange mouse as for a cagemate. In other words, to personify mice, glucocorticoids narrow who counts as enough of an US to evoke empathy. Likewise in humans, pain empathy was not evoked for a stranger unless glucocorticoid secretion was blocked, either after administration of a short-acting drug or after the subject and stranger interacted socially. Recall from Chapter 2 the involvement of the anterior cingulate cortex in pain empathy. I bet that glucocorticoids do some disabling, atrophying things to neurons there. Thus, sustained stress has some pretty unappealing behavioral effects. Nonetheless, there are circumstances where stress brings out the magnificent, best in some people. Work by Shelley Taylor of UCLA shows that fight or flight is the typical response to stress in males, and naturally, the stress literature is predominantly studies of males by males. 83. Things often differ in females. Showing that she can match the good old boys when it comes to snappy sound bites, Taylor framed the female stress response as being more about tend and befriend, caring for your young and seeking social affiliation. This fits with striking sex differences in stress management styles, and tend and befriend most likely reflects the female stress response involving a stronger component of oxytocin secretion. Naturally, things are subtler than male equals fight/flight and female equals tend/befriend. There are frequent counterexamples to each. Stress elicits prosociality in more males than just pair-bonded male marmosets, and we saw that females are plenty capable of aggression. Then there's Mahatma Gandhi and Sarah Palin. Why are some people exceptions to these gender stereotypes? That's part of what the rest of this book is about. Stress can disrupt cognition, impulse control, emotional regulation, decision-making, empathy, and prosociality. One final point. Recall from Chapter 2 how the frontal cortex, making you do the harder thing when it's the right thing, is value-free. Right thing is purely instrumental. Same with stress. Its effects on decision-making are adverse only in a neurobiological sense. During a stressful crisis, an EMT may become perseverative, making her ineffectual at saving lives. A bad thing. During a stressful crisis, a sociopathic warlord may become perseverative, making him ineffectual at ethnically cleansing a village. Not a bad thing. Some important debunking. Alcohol. Review of the biological events in the minutes to hours prior to a behavior can omit alcohol. As everyone knows, alcohol lessens inhibitions, making people more aggressive. Wrong, and in a familiar way, alcohol only evokes aggression only in individuals prone to aggression. For example, mice with lower levels of serotonin signaling in the frontal cortex and men with the oxytocin receptor gene variant less responsive to oxytocin are preferentially made aggressive by alcohol. And B, those who believe that alcohol makes you more aggressive, once more showing the power of social learning to shape biology. 84. Alcohol works differently in everyone else. For example, drunken stupor has caused many a quickie Vegas wedding that doesn't seem like a great idea with the next day's sunrise. Summary and some conclusions. Hormones are great. They run circles around neurotransmitters in terms of the versatility and duration of their effects. And this includes affecting the behaviors pertinent to this book. Testosterone has far less to do with aggression than most assume. Within the normal range, individual differences in testosterone levels don't predict who will be aggressive. Moreover, the more an organism has been aggressive, the less testosterone is needed for future aggression. When testosterone does play a role, it's facilitatory. Testosterone does not invent aggression. It makes us more sensitive to triggers of aggression, particularly in those most prone to aggression. Also, rising testosterone levels foster aggression only during challenges to status. Finally, crucially, the rise in testosterone during a status challenge does not necessarily increase aggression. It increases whatever is needed to maintain status. In a world in which status is awarded for the best of our behaviors, testosterone would be the most prosocial hormone in existence. Oxytocin and vasopressin facilitate mother-infant bond formation and monogamous pair bonding, decrease anxiety and stress, enhance trust and social affiliation, and make people more cooperative and generous. But this comes with a huge caveat: these hormones increase prosociality only toward in us. When dealing with thems, they make us more ethnocentric and xenophobic. Oxytocin is not a universal love hormone. It's a parochial one. Female aggression in defense of offspring is typically adaptive and is facilitated by estrogen, progesterone, and oxytocin. Importantly, females are aggressive in many other evolutionarily adaptive circumstances. Such aggression is facilitated by the presence of androgens in females and by complex, neuroendocrine tricks for generating androgenic signals in aggressive, but not maternal or affiliative, parts of the female brain. Mood and behavioral changes around the time of menses are a biological reality, albeit poorly understood on a nuts and bolts level. In contrast, pathologizing these shifts is a social construct. Finally, except for rare, extreme cases, the link between PMS and aggression is minimal. Sustained stress has numerous adverse effects. The amygdala becomes overactive and more coupled to pathways of habitual behavior. It is easier to learn fear and harder to unlearn it. We process emotionally salient information more rapidly and automatically, but with less accuracy. Frontal function, working memory, impulse control, executive decision-making, risk assessment, and task shifting is impaired, and the frontal cortex has less control over the amygdala. And we become less empathic and prosocial. Reducing sustained stress is a win-win for us and those stuck around us. I'd been drinking is no excuse for aggression. Over the course of minutes to hours, hormonal effects are predominantly contingent and facilitative. Hormones don't determine, command, cause, or invent behaviors. Instead, they make us more sensitive to the social triggers of emotionally laden behaviors and exaggerate our pre-existing tendencies in those domains. And where do those pre-existing tendencies come from? From the contents of the chapters ahead of us. 5 days to months before your act has occurred, the pulling of a trigger, or the touching of an arm that can mean such different things in different contexts. Why did that just happen? We've seen how, seconds before, that behavior was the product of the nervous system, whose actions were shaped by sensory cues minutes to hours before, and how the brain's sensitivity to those cues was shaped by hormonal exposure in the preceding hours to days. What events in the prior days to months shaped that outcome? Chapter 2 introduced the plasticity of neurons, the fact that things alter in them. The strength of a dendritic input, the axon hillock set point for initiating an action potential, the duration of the refractory period. The previous chapter showed that, for example, testosterone increases the excitability of amygdaloid neurons and glucocorticoids decrease excitability of prefrontal cortical neurons. We even saw how progesterone boosts the efficacy with which GABAergic neurons decrease the excitability of other neurons. Those versions of neural plasticity occur over hours. We now examine more dramatic plasticity occurring over days to months. A few months is enough time for an Arab Spring, for a discontented winter, or for STDs to spread a lot during a summer of love. As we'll see, this is also sufficient time for enormous changes in the brain's structure. Nonlinear excitation. Start small. How can events from months ago produce a synapse with altered excitability today? How do synapses remember? When neuroscientists first approached the mystery of memory at the start of the 20th century, they asked that question on a more macro level. How does a brain remember? Obviously, a memory was stored in a single neuron, and a new memory required a new neuron. The discovery that adult brains don't make new neurons trashed that idea. Better microscopes revealed neuronal arborization, the breathtaking complexity of branches of dendrites and axon terminals. Maybe a new memory requires a neuron to grow a new axonal or dendritic branch. Knowledge emerged about synapses, neurotransmitterology was born, and this idea was modified. A new memory requires the formation of a new synapse, a new connection between an axon terminal and a dendritic spine. These speculations were tossed on the ash heap of history in 1949 because of the work of the Canadian neurobiologist Donald Hebb, a man so visionary that even now, nearly 70 years later, neuroscientists still own bobblehead dolls of him. In his seminal book, The Organization of Behavior, Hebb proposed what became the dominant paradigm. Forming memories doesn't require new synapses, let alone new branches or neurons. It requires the strengthening of pre-existing synapses. Point 1. What does strengthening mean? In circuitry terms, if neuron A synapses onto neuron B, it means that an action potential in neuron A more readily triggers one in neuron B. They are more tightly coupled, they remember. Translated into cellular terms, strengthening means that the wave of excitation in a dendritic spine spreads farther, getting closer to the distant axon hillock. Extensive research shows that experience that causes repeated firing across a synapse strengthens it, with a key role played by the neurotransmitter glutamate. Recall from Chapter 2 how an excitatory neurotransmitter binds to its receptor in the postsynaptic dendritic spine, causing a sodium channel to open. Some sodium flows in, causing a blip of excitation, which then spreads. Glutamate signaling works in a fancier way that is essential to learning. Point 2. To simplify considerably, while dendritic spines typically contain only one type of receptor, those responsive to glutamate contain two. The first, The non-NMDA works in a conventional way. For every little smidgen of glutamate binding to these receptors, a smidgen of sodium flows in, causing a smidgen of excitation. The second, the NMDA, works in a nonlinear, threshold manner. It is usually unresponsive to glutamate. It's not until the non-NMDA has been stimulated over and over by a long train of glutamate release allowing enough sodium to flow in that this activates the NMDA receptor. It suddenly responds to all that glutamate, opening its channels, allowing an explosion of excitation. This is the essence of learning. The lecturer says something, and it goes in one ear and out the other. The factoid is repeated, same thing. It's repeated enough times and, aha, the light bulb goes on and suddenly, you get it. At a synaptic level, the axon terminal having to repeatedly release glutamate is the lecturer droning on repetitively, the moment when the postsynaptic threshold is passed and the NMDA receptors first activate is the dendritic spine finally getting it. B. Aha versus actually remembering. This has only gotten us to first base. The light bulb going on in the middle of the lecture doesn't mean it'll still be on in an hour, let alone during the final exam. How can we make that burst of excitation persist so that NMDA receptors are more easily activated in the future? How does the potentiated excitation become long-term? This is our cue to introduce the iconic concept of LTP, long-term potentiation. LTP, first demonstrated in 1966 by Tarje Lomo at the University of Oslo, is the process by which the first burst of NMDA receptor activation causes a prolonged increase in excitability of the synapse. Hundreds of productive careers have been spent figuring out how LTP works, and the key is that, when NMDA receptors finally activate and open their channels, it is calcium, rather than sodium, that flows in. This causes an array of changes. Here are a few. The calcium tidal wave causes more copies of glutamate receptors to be inserted into the dendritic spine's membrane, making the neuron more responsive to glutamate. Thereafter, the calcium also alters glutamate receptors that are already on the front lines of that dendritic spine. Each will now be more sensitive to glutamate signals. The calcium also causes the synthesis of peculiar neurotransmitters in the dendritic spine, which are released and travel backward across the synapse. There they increase the amount of glutamate released from the axon terminal after future action potentials. In other words, LTP arises from a combination of the presynaptic axon terminal yelling glutamate more loudly and the postsynaptic dendritic spine listening more attentively. As I said, additional mechanisms underlie LTP, and neuroscientists debate which is most important, the one they study, naturally, in neurons and organisms when they are actually learning. In general, the debate has been whether pre- or the postsynaptic changes are more crucial. After LTP came a discovery that suggests a universe in balance. This is LTD, long-term depression, experience-dependent, long-term decreases in synaptic excitability, and interestingly, the mechanisms underlying limited are not merely the opposite of LTP. Limited is not the functional opposite of LTP either. Rather than being the basis of generic forgetting, it sharpens a signal by erasing what's extraneous. A final point about LTP. There's long-term and there's long-term. As noted, one mechanism underlying LTP is an alteration in glutamate receptors so that they are more responsive to glutamate. That change might persist for the lifetime of the copies of that receptor that were in that synapse at the time of the LTPing. But that's typically only a few days until those copies accumulate bits of oxygen radical damage and are degraded and replaced with new copies. Similar updating of all proteins constantly occurs. Somehow LTP-induced changes in the receptor are transferred to the next generation of copies. How else can octogenarians remember kindergarten? The mechanism is elegant, but beyond the scope of this chapter. All this is cool, but LTP and LDP are what happens in the hippocampus when you learn explicit facts, like someone's phone number. But we're interested in other types of learning, how we learn to be afraid, to control our impulses, to feel empathy, or to feel nothing for someone else. Synapses utilizing glutamate occur throughout the nervous system, and LTP isn't exclusive to the hippocampus. This was a traumatic discovery for many LTP hippocampus researchers. After all, LTP is what occurred in Schopenhauer's hippocampus when he read Hegel, not what the spinal cord does to make you more coordinated at twerking. Nonetheless, LTP occurs throughout the nervous system. For example, Fear conditioning involves synapses LTPing in the basolateral amygdala. LTP underlies the frontal cortex learning to control the amygdala. It's how dopaminergic systems learn to associate a stimulus with a reward, for example, how addicts come to associate a location with a drug, feeling cravings when in that setting. Let's add hormones to this, translating some of our stress concepts into the language of neural plasticity. Moderate, transient stress, i.e., the good, stimulatory stress, promotes hippocampal LTP, while prolonged stress disrupts it and promotes LTD, one reason why cognition tanks at such times. This is the inverted U concept of stress-rich synaptic point 4. Moreover, sustained stress and glucocorticoid exposure enhance LTP and suppress limited in the amygdala, boosting fear conditioning, and suppress LTP in the frontal cortex. Combining these effects, more excitable synapses in the amygdala, fewer ones in the frontal cortex, helps explain stress-induced impulsivity and poor emotional regulation. Point 5. Rescued from the trash. The notion of memory resting on the strengthening of pre-existing synapses dominates the field. But ironically, the discarded idea that memory requires the formation of new synapses has been resuscitated. Techniques for counting all of a neuron's synapses show that housing rats in a rich, stimulatory environment increases their number of hippocampal synapses. Profoundly fancy techniques let you follow one dendritic branch of a neuron over time as a rat learns something. Astonishingly, over minutes to hours a new dendritic spine emerges, followed by an axon terminal hovering nearby. Over the next weeks, they form a functioning synapse that stabilizes the new memory, and in other circumstances, dendritic spines retract, eliminating synapses. Such activity-dependent synaptogenesis is coupled to LTP. When a synapse undergoes LTP, the tsunami of calcium rushing into the spine can diffuse and trigger the formation of a new spine in the adjacent stretch of the dendritic branch. New synapses form throughout the brain in motor cortex neurons when you learn a motoric task, or in the visual cortex after lots of visual stimulation. Stimulate a rat's whiskers a lot, and ditto in the whisker cortex. 6. Moreover, when enough new synapses form in a neuron, the length and number of branches in its dendritic tree often expand as well, increasing the strength and number of the neurons that can talk to it. Stress and glucocorticoids have inverted U effects here as well. Moderate, transient stress, or exposure to the equivalent glucocorticoid levels increases spine number in the hippocampus. Sustained stress or glucocorticoid exposure does the opposite. Point 7. Moreover, major depression or anxiety, two disorders associated with elevated glucocorticoid levels, can reduce hippocampal dendrite and spine number. This arises from decreased levels of that key growth factor mentioned earlier this chapter, BDNF. Sustained stress and glucocorticoids also cause dendritic retraction and synapse loss, lower levels of NCAM, a neural cell adhesion molecule that stabilizes synapses, and less glutamate release in the frontal cortex. The more of these changes, the more attentional and decision-making impairments. Point 8. Recall from Chapter 4 how acute stress strengthens connectivity between the frontal cortex and motoric areas, while weakening frontal hippocampal connections. The result is decision-making that is habitual, rather than incorporating new information. Similarly, chronic stress increases spine number in frontal motor connections and decreases it in frontal hippocampal ones. Point 9. Continuing the theme of the amygdala differing from the frontal cortex and hippocampus, sustained stress increases BDNF levels and expands dendrites in the blah, persistently increasing anxiety and fear conditioning. Point 10. The same occurs in that way station, by which the amygdala talks to the rest of the brain, the BNST, bed nucleus of the stria terminalis. Recall that while the blah mediates fear conditioning, the central amygdala is more involved in innate phobias. Interestingly, stress seems not to increase the force of phobias or spine number in the central amygdala. There's wonderful context dependency to these effects. When a rat secretes tons of glucocorticoids because it's terrified, dendrites atrophy in the hippocampus. However, if it secretes the same amount by voluntarily running on a running wheel, dendrites expand. Whether the amygdala is also activated seems to determine whether the hippocampus interprets the glucocorticoids as good or bad. Stress. Point 11. Spine number and branch length in the hippocampus and frontal cortex are also increased by estrogen. Point 12. Remarkably, the size of neurons' dendritic trees in the hippocampus expands and contracts like an accordion throughout a female rat's ovulatory cycle, with the size and her cognitive skills peaking when estrogen peaks. Thus, neurons can form new dendritic branches and spines, increasing the size of their dendritic tree, or, in other circumstances, do the opposite. Hormones frequently mediate these effects. Axonal plasticity. Meanwhile, there's plasticity at the other end of the neuron, where axons can sprout offshoots that head off in novel directions. As a spectacular example, when a blind person, adept at Braille, reads in it, there's the same activation of the tactile cortex as in anyone else, but amazingly, uniquely, there is also activation of the visual cortex. 13. In other words, neurons that normally send axons to the fingertip processing part of the cortex instead have gone miles off course, growing projections to the visual cortex. One extraordinary case concerned a congenitally blind woman, adept at Braille, who had a stroke in her visual cortex. And as a result, she lost the ability to read Braille. The bumps on the page felt flattened, imprecise, while other tactile functions remained. In another study, blind subjects were trained to associate letters with distinctive tones to the point where they could hear a sequence of tones as letters and words. When these individuals would read with sound, they'd activate the part of the visual cortex activated in sighted individuals when reading. Similarly, when a person who is deaf and adept at American Sign Language watches someone signing, there is activation of the part of their auditory cortex normally activated by speech. The injured nervous system can remap in similar ways. Suppose there is stroke damage to the part of your cortex that receives tactile information from your hand. The tactile receptors in your hand work fine, but have no neurons to talk to, thus you lose sensation in your hand. In the subsequent months to years, axons from those receptors can sprout off in new directions, shoehorning their way into neighboring parts of the cortex, forming new synapses there. An imprecise sense of touch may slowly return to the hand, along with a less precise sense of touch in the part of the body projecting to the cortical region that accommodated those refugee axon terminals. Suppose, instead, that tactile receptors in the hand are destroyed, no longer projecting to those sensory cortical neurons. Neurons abhor a vacuum, and tactile neurons in the wrist may sprout collateral axonal branches and expand their territory into that neglected cortical region. Consider blindness due to retinal degeneration, where the projections to the visual cortex are silenced. As described, fingertip tactile neurons involved in reading Braille sprout projections into the visual cortex, setting up camp there. Or suppose there is a pseudo-injury. After merely 5 days of subjects being blindfolded, auditory projections start to remap into the visual cortex and retract once the blindfolds come off. 14. Consider how fingertip tactile neurons carrying information about Braille remap to the visual cortex in someone blind. The sensory cortex and visual cortex are far away from each other. How do those tactile neurons know that there's vacant property in the visual cortex? B. That hooking up with those unoccupied neurons helps turn fingertip information into reading. And C. How to send axonal projections to this new cortical continent. All are matters of ongoing research. What happens in a blind person when auditory projection neurons expand their target range into the inactive visual cortex? More acute hearing, the brain can respond to deficits in one realm with compensations in another. So sensory projection neurons can remap. And once, say, visual cortex neurons are processing Braille in a blind person, those neurons need to remap where they project to, triggering further downstream remapping. Waves of plasticity. Remapping occurs regularly throughout the brain in the absence of injury. My favorite examples concern musicians, who have larger auditory cortical representation of musical sounds than do non-musicians, particularly for the sound of their own instrument as well as for detecting pitch in speech. The younger the person begins being a musician, the stronger the remapping. 15. Such remapping does not require decades of practice, as shown in beautiful work by Alvaro Pascual-León at Harvard. 16. Non-musician volunteers learned a 5-finger exercise on the piano, which they practiced for 2 hours a day. Within a few days the amount of motor cortex devoted to the movement of that hand expanded, but the expansion lasted less than a day without further practice. This expansion was probably Hebbian in nature, meaning pre-existing connections transiently strengthened after repeated use. However, if subjects did the daily exercise for a crazed 4 weeks, the remapping persisted for many days afterward. This expansion probably involved axonal sprouting and the formation of new connections. Remarkably, remapping also occurred in volunteers who spent 2 hours a day, imagining playing the finger exercise. As another example of remapping, after female rats give birth, there is expansion of the tactile map representing the skin around the nipples. As a rather different example, spend 3 months learning how to juggle, and there is expansion of the cortical map for visual processing of movement 17. Thus, experience alters the number and strength of synapses, the extent of dendritic arbor, and the projection targets of axons. Time for the biggest revolution in neuroscience in years. R. Digging deeper in the ash heap of history. He called the crude, Neanderthalish notion that new memories require new neurons, an idea discarded when Heb was in diapers. The adult brain does not make new neurons. You've got your maximal number of neurons around birth, and it's downhill from there, thanks to aging and imprudence. You see where we're heading. Adult brains, including aged human brains, do make new neurons. The finding is truly revolutionary, its discovery epic. In 1965, an untenured associate professor at MIT named Joseph Altman, along with a longtime collaborator, Gopal Das, found the first evidence for adult neurogenesis, using a then novel technique. A newly made cell contains newly made DNA. So, find a molecule unique to DNA. Get a test tube full of the stuff and attach a minuscule, radioactive tag to each molecule. Inject it into an adult rat, wait a while, and examine its brain. If any neurons contain that radioactive tag, it means they were born during the waiting period with the radioactive marker incorporated into the new DNA. This is what Altman saw in a series of studies. 18. As even he notes, the work was initially well received, being published in good journals, generating excitement. But within a few years something shifted, and Altman and his findings were rejected by leaders in the field. It couldn't be true. He failed to get tenure, spent his career at Purdue University, lost funding for his adult neurogenesis work. Silence reigned for a decade until an assistant professor at the University of New Mexico named Michael Kaplan extended Altman's findings with some new techniques. Again, this caused mostly crushing rejection by senior figures in the field, including one of the most established men in neuroscience, Pasco Rakich of Yale. 19. Rakich publicly rejected Kaplan's, and tacitly Altman's, work, saying he had looked for new neurons himself, they weren't there, and Kaplan was mistaking other cell types for neurons. At a conference he notoriously told Kaplan, "Those may look like neurons in New Mexico, but they don't in New Haven." Kaplan soon left research, and a quarter-century later, amid the excitement of the rediscovery of adult neurogenesis, wrote a short memoir entitled Environmental Complexity, Stimulates Visual Cortex Neurogenesis, Death of a Dogma and a Research Career. The field lay dormant for another decade until unexpected evidence of adult neurogenesis emerged from the lab of Fernando Nottebohm of Rockefeller University. Nottebohm, a highly accomplished and esteemed neuroscientist, as good an old boy as you get, studied the neuroethology of birdsong. He demonstrated something remarkable. Using new, more sensitive techniques, new neurons are made in the brains of birds that learn a new territorial song each year. The quality of the science and Nottebohm's prestige silenced those who doubted that neurogenesis occurred. Instead they questioned its relevance. "Oh, that's nice for Fernando and his birdies, but what about in real species, in mammals?" But this was soon, convincingly shown in rats, using newer, fancier techniques. Much of this was the work of two young scientists, Elizabeth Gould of Princeton, and Fred Rusty Gage of the Salk Institute. Soon lots of other people were finding adult neurogenesis with these new techniques, including, lo and behold, Rakich. 20. A new flavor of skepticism emerged, led by Rakich. Yes, the adult brain makes new neurons, but only a few, they don't live long, and it doesn't happen where it really counts, i.e., the cortex. Moreover, this has been shown only in rodents, not in primates. Soon it was shown in monkeys. 21. Yeah, said the skeptics, but not humans, and besides, there's no evidence that these new neurons are integrated into pre-existing circuits and actually function. All of that was eventually shown. There's considerable adult neurogenesis in the hippocampus, where roughly 3% of neurons are replaced each month, and lesser amounts in the cortex. 22. It happens in humans throughout adult life. Hippocampal neurogenesis, for example, is enhanced by learning, exercise, estrogen, antidepressants, environmental enrichment, and brain injury, and inhibited by various stressors. 23. Moreover, the new hippocampal neurons integrate into pre-existing circuits with the perky excitability of young neurons in the perinatal brain. Most important, new neurons are essential for integrating new information into pre-existing schemas, something called pattern separation. This is when you learn that two things you previously thought were the same are, in fact, different: dolphins and porpoises, baking soda and baking powder, Zooey Deschanel and Katy Perry. Adult neurogenesis is the trendiest topic in neuroscience. In the 5 years after Altman's 1965 paper was published, it was cited, a respectable, 29 times in the literature. In the last 5, more than 1,000. Current work examines how exercise stimulates the process, probably by increasing levels of certain growth factors in the brain, how new neurons know where to migrate, whether depression is caused by a failure of hippocampal neurogenesis, and whether the neurogenesis stimulated by antidepressants is required for such medications to work. 24. Why did it take so long for adult neurogenesis to be accepted? I've interacted with many of the principles and am struck by their differing takes. At one extreme is the view that while skeptics like Rakic were ham-handed, they provided quality control, and that, counter to how Path of the Hero epics go, some early work in the field was not all that solid. At the other extreme is the view that Rakic et al., having failed to find adult neurogenesis, couldn't accept that it existed. This psychohistorical view of the old guard clinging to dogma in the face of changing winds is weakened a bit by Altman's not having been a young anarchist, running amok in the archives. In fact, he is a bit older than Rakic and other principal skeptics. All of this needs to be adjudicated by historians, screenwriters, and soon, I hope, by the folks in Stockholm. Altman, who at the time of this writing is 89, published a 2011 memoir. Chapter 25. Parts of it have a plaintive, confused tone. Everyone was so excited at first. What happened? Maybe he spent too much time in the lab and too little marketing the discovery, he suggests. There's the ambivalence of someone who spent a long time as a scorned prophet, who at least got to be completely vindicated. He's philosophical about it. Hey, I'm a Hungarian Jew who escaped from a Nazi camp. You take things in stride after that. Some other domains of neuroplasticity. You've seen how an adult's experience can alter the number of synapses and dendritic branches, remap circuitry, and stimulate neurogenesis. 26. Collectively, these effects can be big enough to actually change the size of brain regions. For example, postmenopausal estrogen treatment increases the size of the hippocampus, probably through a combination of more dendritic branches and more neurons. Conversely, the hippocampus atrophies, producing cognitive problems in prolonged depression, probably reflecting its stressfulness and the typically elevated glucocorticoid levels of the disease. Memory problems and loss of hippocampal volume also occur in individuals with severe chronic pain syndromes, or with Cushing's syndrome, an array of disorders where a tumor causes extremely elevated glucocorticoid levels. Moreover, post-traumatic stress disorder is associated with increased volume, and as we know, hyperreactivity of the amygdala. In all of these instances, it is unclear how much the stress glucocorticoid effects are due to changes in neuron number or to changes in amounts of dendritic processes. One cool example of the size of a brain region changing with experience concerns the back part of the hippocampus, which plays a role in memory of spatial maps. Cab drivers use spatial maps for a living, and one renowned study showed enlargement of that part of the hippocampus in London taxi drivers. Moreover, a follow-up study imaged the hippocampus in people before and after the grueling multi-process of working and studying for the London cabbie license test, called the toughest test in the world by the New York Times. The hippocampus enlarged over the course of the process in those who passed the test. 27. Thus, experience, health, and hormone fluctuations can change the size of parts of the brain in a matter of months. Experience can also cause long-lasting changes in the numbers of receptors for neurotransmitters and hormones, in levels of ion channels, and in the state of on-off switches on genes in the brain, to be covered in Chapter 8.28. With chronic stress, the nucleus accumbens is depleted of dopamine, biasing rats toward social subordination, and biasing humans toward depression. As we saw in the last chapter, if a rodent wins a fight on his home territory, there are long-lasting increases in levels of testosterone receptors in the nucleus accumbens and ventral tegmentum, enhancing testosterone's pleasurable effects. There's even a parasite called Toxoplasma gondii that can infect the brain over the course of weeks to months. It makes rats less fearful of the smell of cats and makes humans less fearful and more impulsive in subtle ways. Basically, Most anything you can measure in the nervous system can change in response to a sustained stimulus. And importantly, these changes are often reversible in a different environment. Some conclusions. The discovery of adult neurogenesis is revolutionary, and the general topic of neuroplasticity in all its guises is immensely important, as is often the case when something the experts said couldn't be turns out to be 0.29. The subject is also fascinating because of the nature of the revisionism. Neuroplasticity radiates optimism. Books on the topic are entitled The Brain That Changes Itself, Train Your Mind, Change Your Brain, and Rewire Your Brain, Think Your Way to a Better Life, hinting at the new neurology, i.e., no more need for neurology once we can fully harness neuroplasticity. There's can-do Horatio Alger spirit every which way you look. Amid that, some cautionary points. One recalls caveats aired in other chapters. The ability of the brain to change in response to experience is value-free. Axonal remapping in blind or deaf individuals is great, exciting, and moving. It's cool that your hippocampus expands if you drive a London cab. Ditto about the size and specialization of the auditory cortex in the triangle player in the orchestra. But at the other end, it's disastrous that trauma enlarges the amygdala and atrophies the hippocampus, crippling those with PTSD. Similarly, expanding the amount of motor cortex devoted to finger dexterity is great in neurosurgeons, but probably not a societal plus in safe crackers. The extent of neuroplasticity is most definitely finite. Otherwise, grievously injured brains and severed spinal cords would ultimately heal. Moreover, the limits of neuroplasticity are quotidian. Malcolm Gladwell has explored how vastly skilled individuals have put in vast amounts of practice. 10,000 hours is his magic number. Nevertheless, the reverse doesn't hold. 10,000 hours of practice does not guarantee the neuroplasticity needed to make any of us a Yo-Yo Ma or LeBron James. Manipulating neuroplasticity for recovery of function does have enormous, exciting potential in neurology. But this domain is far from the concerns of this book. Despite neuroplasticity's potential, it's unlikely that we'll ever be able to, say, spritz neuronal growth factors up people's noses to make them more open-minded or empathic, or to target neuroplasticity with gene therapy to blunt some jerk's tendency to displace aggression. So what's the subject good for in the realm of this book? I think the benefits are mostly psychological. This recalls a point from Chapter 2 in the discussion of the neuroimaging studies demonstrating loss of volume in the hippocampus of people with PTSD, certainly an example of the adverse effects of neuroplasticity. I sniped that it was ridiculous that many legislators needed pictures of the brain to believe that there was something desperately, organically wrong with veterans with PTSD. Similarly, neuroplasticity makes the functional malleability of the brain tangible, makes it scientifically demonstrated that brains change. That people change. In the time span considered in this chapter, people throughout the Arab world went from being voiceless to toppling tyrants, Rosa Parks went from victim to catalyst, Sadat and Begin from enemies to architects of peace, Mandela from prisoner to statesman. And you'd better bet that changes along the lines of those presented in this chapter occurred in the brains of anyone transformed by these transformations. A different world makes for a different worldview, which means a different brain. And the more tangible and real the neurobiology underlying such change seems, the easier it is to imagine that it can happen again. Some important debunking: alcohol. Review of the biological events in the minutes to hours prior to a behavior can omit alcohol. As everyone knows, alcohol lessens inhibitions, making people more aggressive. Wrong, and in a familiar way alcohol only evokes aggression only in individuals prone to aggression, for example, mice with lower levels of serotonin signaling in the frontal cortex and men with the oxytocin receptor, gene variant less responsive to oxytocin are preferentially made aggressive by alcohol, and B, those who believe that alcohol makes you more aggressive, once more showing the power of social learning to shape biology. 84. Alcohol works differently in everyone else. For example, drunken stupor has caused many a quickie Vegas wedding that doesn't seem like a great idea with the next day's sunrise. Summary and some conclusions. Hormones are great, they run circles around neurotransmitters in terms of the versatility and duration of their effects. And this includes affecting the behaviors pertinent to this book. Testosterone has far less to do with aggression than most assume. Within the normal range, individual differences in testosterone levels don't predict who will be aggressive. Moreover, the more an organism has been aggressive, the less testosterone is needed for future aggression. When testosterone does play a role, it's facilitatory. Testosterone does not invent aggression. It makes us more sensitive to triggers of aggression, particularly in those most prone to aggression. Also, rising testosterone levels foster aggression only during challenges to status. Finally, crucially, the rise in testosterone during a status challenge does not necessarily increase aggression. It increases whatever is needed to maintain status. In a world in which status is awarded for the best of our behaviors, testosterone would be the most prosocial hormone in existence. Oxytocin and vasopressin facilitate mother-infant bond formation and monogamous pair bonding, decrease anxiety and stress, enhance trust and social affiliation, and make people more cooperative and generous. But this comes with a huge caveat: these hormones increase prosociality only toward in us. When dealing with thems, they make us more ethnocentric and xenophobic. Oxytocin is not a universal love hormone. It's a parochial one. Female aggression in defense of offspring is typically adaptive and is facilitated by estrogen, progesterone, and oxytocin. Importantly, females are aggressive in many other evolutionarily adaptive circumstances. Such aggression is facilitated by the presence of androgens in females and by complex, neuroendocrine tricks for generating androgenic signals in aggressive, but not maternal or affiliative, parts of the female brain. Mood and behavioral changes around the time of menses are a biological reality, albeit poorly understood on a nuts and bolts level. In contrast, pathologizing these shifts is a social construct. Finally, except for rare, extreme cases, the link between PMS and aggression is minimal. Sustained stress has numerous adverse effects. The amygdala becomes overactive and more coupled to pathways of habitual behavior. It is easier to learn fear and harder to unlearn it. We process emotionally salient information more rapidly and automatically, but with less accuracy. Frontal function, working memory, impulse control, executive decision-making, risk assessment, and task shifting is impaired, and the frontal cortex has less control over the amygdala. And we become less empathic and prosocial. Reducing sustained stress is a win-win for us and those stuck around us. I'd been drinking is no excuse for aggression. Over the course of minutes to hours, hormonal effects are predominantly contingent and facilitative. Hormones don't determine, command, cause, or invent behaviors. Instead, they make us more sensitive to the social triggers of emotionally laden behaviors and exaggerate our pre-existing tendencies in those domains. And where do those pre-existing tendencies come from? From the contents of the chapters ahead of us. 5 days to months before your act has occurred, the pulling of a trigger, or the touching of an arm that can mean such different things in different contexts. Why did that just happen? We've seen how, seconds before, that behavior was the product of the nervous system, whose actions were shaped by sensory cues minutes to hours before, and how the brain's sensitivity to those cues was shaped by hormonal exposure in the preceding hours to days. What events in the prior days to months shaped that outcome? Chapter 2 introduced the plasticity of neurons, the fact that things alter in them. The strength of a dendritic input, the axon-hillock set point for initiating an action potential, the duration of the refractory period. The previous chapter showed that, for example, testosterone increases the excitability of amygdaloid neurons and glucocorticoids decrease excitability of prefrontal cortical neurons. We even saw how progesterone boosts the efficacy with which GABAergic neurons decrease the excitability of other neurons. Those versions of neural plasticity occur over hours. We now examine more dramatic plasticity occurring over days to months. A few months is enough time for an Arab Spring, for a discontented winter, or for STDs to spread a lot during a summer of love. As we'll see, this is also sufficient time for enormous changes in the brain's structure. Nonlinear excitation. Start small. How can events from months ago produce a synapse with altered excitability today? How do synapses remember? When neuroscientists first approached the mystery of memory at the start of the 20th century, they asked that question on a more macro level. How does a brain remember? Obviously, a memory was stored in a single neuron, and a new memory required a new neuron. The discovery that adult brains don't make new neurons trashed that idea. Better microscopes revealed neuronal arborization, the breathtaking complexity of branches of dendrites and axon terminals. Maybe a new memory requires a neuron to grow a new axonal or dendritic branch. Knowledge emerged about synapses, neurotransmitterology was born, and this idea was modified. A new memory requires the formation of a new synapse, a new connection between an axon terminal and a dendritic spine. These speculations were tossed on the ash heap of history in 1949 because of the work of the Canadian neurobiologist Donald Hebb, a man so visionary that even now, nearly 70 years later, neuroscientists still own bobblehead dolls of him. In his seminal book, The Organization of Behavior, Hebb proposed what became the dominant paradigm. Forming memories doesn't require new synapses, let alone new branches or neurons. It requires the strengthening of pre-existing synapses. Point 1. What does strengthening mean? In circuitry terms, if neuron A synapses onto neuron B, it means that an action potential in neuron A more readily triggers one in neuron B. They are more tightly coupled, they remember. Translated into cellular terms, strengthening means that the wave of excitation in a dendritic spine spreads farther, getting closer to the distant axon hillock. Extensive research shows that experience that causes repeated firing across a synapse strengthens it, with a key role played by the neurotransmitter glutamate. Recall from Chapter 2 how an excitatory neurotransmitter binds to its receptor in the postsynaptic dendritic spine, causing a sodium channel to open. Some sodium flows in, causing a blip of excitation, which then spreads. Glutamate signaling works in a fancier way that is essential to learning. Point 2. To simplify considerably, while dendritic spines typically contain only one type of receptor, those responsive to glutamate contain two. The first, the non-NMDA, works in a conventional way. For every little smidgen of glutamate binding to these receptors, a smidgen of sodium flows in, causing a smidgen of excitation. The second, the NMDA, works in a nonlinear, threshold manner. It is usually unresponsive to glutamate. It's not until the non-NMDA has been stimulated over and over by a long train of glutamate release allowing enough sodium to flow in, that this activates the NMDA receptor. It suddenly responds to all that glutamate, opening its channels, allowing an explosion of excitation. This is the essence of learning. The lecturer says something, and it goes in one ear and out the other. The factoid is repeated, same thing. It's repeated enough times and, aha, the light bulb goes on and suddenly, you get it. At a synaptic level, the axon terminal having to repeatedly release glutamate is the lecturer droning on repetitively. The moment when the postsynaptic threshold is passed and the NMDA receptors first activate is the dendritic spine finally getting it. B. Aha versus actually remembering. This has only gotten us to first base. The light bulb going on in the middle of the lecture doesn't mean it'll still be on in an hour, let alone during the final exam. How can we make that burst of excitation persist, so that NMDA receptors are more easily activated in the future? How does the potentiated excitation become long-term? This is our cue to introduce the iconic concept of LTP, long-term potentiation. LTP, first demonstrated in 1966 by Tarje Lomo at the University of Oslo, is the process by which the first burst of NMDA receptor activation causes a prolonged increase in excitability of the synapse. Hundreds of productive careers have been spent figuring out how LTP works, and the key is that, when NMDA receptors finally activate and open their channels, it is calcium, rather than sodium, that flows in. This causes an array of changes. Here are a few. The calcium tidal wave causes more copies of glutamate receptors to be inserted into the dendritic spine's membrane, making the neuron more responsive to glutamate. Thereafter, the calcium also alters glutamate receptors that are already on the front lines of that dendritic spine. Each will now be more sensitive to glutamate signals. The calcium also causes the synthesis of peculiar neurotransmitters in the dendritic spine, which are released and travel backward across the synapse. There they increase the amount of glutamate released from the axon terminal after future action potentials. In other words, LTP arises from a combination of the presynaptic axon terminal yelling glutamate more loudly and the postsynaptic dendritic spine listening more attentively. As I said, additional mechanisms underlie LTP, and neuroscientists debate which is most important, the one they study, naturally, in neurons in organisms when they are actually learning. In general, the debate has been whether pre- or the postsynaptic changes are more crucial. After LTP came a discovery that suggests a universe in balance. This is LTD, long-term depression, experience-dependent, long-term decreases in synaptic excitability, and interestingly, the mechanisms underlying limited are not merely the opposite of LTP. Limited is not the functional opposite of LTP either. Rather than being the basis of generic forgetting, it sharpens a signal by erasing what's extraneous. A final point about LTP. There's long-term and there's long-term. As noted, one mechanism underlying LTP is an alteration in glutamate receptors so that they are more responsive to glutamate. That change might persist for the lifetime of the copies of that receptor that were in that synapse at the time of the LTPing. But that's typically only a few days until those copies accumulate bits of oxygen radical damage and are degraded and replaced with new copies. Similar updating of all proteins constantly occurs. Somehow LTP-induced changes in the receptor are transferred to the next generation of copies. How else can octogenarians remember kindergarten? The mechanism is elegant, but beyond the scope of this chapter. All this is cool, but LTP and LDP are what happens in the hippocampus when you learn explicit facts, like someone's phone number. But we're interested in other types of learning, how we learn to be afraid, to control our impulses, to feel empathy, or to feel nothing for someone else. Synapses utilizing glutamate occur throughout the nervous system, and LTP isn't exclusive to the hippocampus. This was a traumatic discovery for many LTP hippocampus researchers. After all, LTP is what occurred in Schopenhauer's hippocampus when he read Hegel, not what the spinal cord does to make you more coordinated at twerking. Nonetheless, LTP occurs throughout the nervous system. For example, fear conditioning involves synapses LTPing in the basilateral amygdala. LTP underlies the frontal cortex learning to control the amygdala. It's how dopaminergic systems learn to associate a stimulus with a reward, for example, how addicts come to associate a location with a drug, feeling cravings when in that setting. Let's add hormones to this, translating some of our stress concepts into the language of neural plasticity. Moderate, transient stress, i.e., the good, stimulatory stress, promotes hippocampal LTP, while prolonged stress disrupts it and promotes LTD, one reason why cognition tanks at such times. This is the inverted U concept of stress-rich synaptic point 4. Moreover, sustained stress and glucocorticoid exposure enhance LTP and suppress limited in the amygdala, boosting fear conditioning, and suppress LTP in the frontal cortex. Combining these effects, more excitable synapses in the amygdala, fewer ones in the frontal cortex, helps explain stress-induced impulsivity and poor emotional regulation. Point 5. Rescued from the trash. The notion of memory resting on the strengthening of pre-existing synapses dominates the field. But ironically, the discarded idea that memory requires the formation of new synapses has been resuscitated. Techniques for counting all of a neuron's synapses show that housing rats in a rich, stimulatory environment increases their number of hippocampal synapses. Profoundly fancy techniques let you follow one dendritic branch of a neuron over time as a rat learns something. Astonishingly, over minutes to hours a new dendritic spine emerges, followed by an axon terminal hovering nearby. Over the next weeks, they form a functioning synapse that stabilizes the new memory, and in other circumstances, dendritic spines retract, eliminating synapses. Such activity-dependent synaptogenesis is coupled to LTP. When a synapse undergoes LTP, the tsunami of calcium rushing into the spine can diffuse and trigger the formation of a new spine in the adjacent stretch of the dendritic branch. New synapses form throughout the brain in motor cortex neurons when you learn a motoric task, or in the visual cortex after lots of visual stimulation. Stimulate a rat's whiskers a lot, and ditto in the whisker cortex. 6. Moreover, when enough new synapses form in a neuron, the length and number of branches in its dendritic tree often expand as well, increasing the strength and number of the neurons that can talk to it. Stress and glucocorticoids have inverted U effects here as well. Moderate, transient stress, or exposure to the equivalent glucocorticoid levels increases spine number in the hippocampus. Sustained stress or glucocorticoid exposure does the opposite. Point 7. Moreover, major depression or anxiety, two disorders associated with elevated glucocorticoid levels, can reduce hippocampal dendrite and spine number. This arises from decreased levels of that key growth factor mentioned earlier this chapter, BDNF. Sustained stress and glucocorticoids also cause dendritic retraction and synapse loss, lower levels of NCAM, a neural cell adhesion molecule that stabilizes synapses, and less glutamate release in the frontal cortex. The more of these changes, the more attentional and decision-making impairments. Point 8. Recall from Chapter 4 how acute stress strengthens connectivity between the frontal cortex and motoric areas while weakening frontal hippocampal connections. The result is decision-making that is habitual, rather than incorporating new information. Similarly, chronic stress increases spine number in frontal motor connections and decreases it in frontal hippocampal ones. Point 9. Continuing the theme of the amygdala differing from the frontal cortex and hippocampus, sustained stress increases BDNF levels and expands dendrites in the blah, persistently increasing anxiety and fear conditioning. Point 10. The same occurs in that way station, by which the amygdala talks to the rest of the brain, the bed nucleus of the stria terminalis. Recall that while the blah mediates fear conditioning, the central amygdala is more involved in innate phobias. Interestingly, stress seems not to increase the force of phobias or spine number in the central amygdala. There's wonderful context dependency to these effects. When a rat secretes tons of glucocorticoids because it's terrified, dendrites atrophy in the hippocampus. However, if it secretes the same amount by voluntarily running on a running wheel, dendrites expand. Whether the amygdala is also activated seems to determine whether the hippocampus interprets the glucocorticoids as good or bad. Stress. Point 11. Spine number and branch length in the hippocampus and frontal cortex are also increased by estrogen. Point 12. Remarkably, the size of neurons' dendritic trees in the hippocampus expands and contracts like an accordion throughout a female rat's ovulatory cycle, with the size and her cognitive skills peaking when estrogen peaks. Thus, neurons can form new dendritic branches and spines, increasing the size of their dendritic tree, or, in other circumstances, do the opposite. Hormones frequently mediate these effects. Axonal plasticity. Meanwhile, there's plasticity at the other end of the neuron, where axons can sprout offshoots that head off in novel directions. As a spectacular example, when a blind person, adept at Braille, reads in it, there's the same activation of the tactile cortex as in anyone else, but amazingly, uniquely, there is also activation of the visual cortex. Point 13. In other words, neurons that normally send axons to the fingertip processing part of the cortex instead have gone miles off course, growing projections to the visual cortex. One extraordinary case concerned a congenitally blind woman, adept at Braille, who had a stroke in her visual cortex. And as a result, she lost the ability to read Braille. The bumps on the page felt flattened, imprecise, while other tactile functions remained. In another study, blind subjects were trained to associate letters with distinctive tones to the point where they could hear a sequence of tones as letters and words. When these individuals would read with sound, they'd activate the part of the visual cortex activated in sighted individuals when reading. Similarly, when a person who is deaf and adept at American Sign Language watches someone signing, there is activation of the part of their auditory cortex normally activated by speech. The injured nervous system can remap in similar ways. Suppose there is stroke damage to the part of your cortex that receives tactile information from your hand. The tactile receptors in your hand work fine, but have no neurons to talk to, thus you lose sensation in your hand. In the subsequent months to years, axons from those receptors can sprout off in new directions, shoehorning their way into neighboring parts of the cortex, forming new synapses there. An imprecise sense of touch may slowly return to the hand, along with a less precise sense of touch in the part of the body projecting to the cortical region that accommodated those refugee axon terminals. Suppose, instead, that tactile receptors in the hand are destroyed, no longer projecting to those sensory cortical neurons. Neurons abhor a vacuum, and tactile neurons in the wrist may sprout collateral axonal branches and expand their territory into that neglected cortical region. Consider blindness due to retinal degeneration, where the projections to the visual cortex are silenced. As described, fingertip tactile neurons involved in reading Braille sprout projections into the visual cortex, setting up camp there. Or suppose there is a pseudo-injury. After merely 5 days of subjects being blindfolded, auditory projections start to remap into the visual cortex and retract once the blindfolds come off. Point 14. Consider how fingertip tactile neurons carrying information about Braille remap to the visual cortex in someone blind. The sensory cortex and visual cortex are far away from each other. How do those tactile neurons know that there's vacant property in the visual cortex? B. That hooking up with those unoccupied neurons helps turn fingertip information into reading. And C. How to send axonal projections to this new cortical continent. All are matters of ongoing research. What happens in a blind person when auditory projection neurons expand their target range into the inactive visual cortex? More acute hearing, the brain can respond to deficits in one realm with compensations in another. So sensory projection neurons can remap. And once, say, visual cortex neurons are processing Braille in a blind person, those neurons need to remap where they project to, triggering further downstream remapping. Waves of plasticity. Remapping occurs regularly throughout the brain in the absence of injury. My favorite examples concern musicians, who have larger auditory cortical representation of musical sounds than do non-musicians, particularly for the sound of their own instrument as well as for detecting pitch in speech. The younger the person begins being a musician, the stronger the remapping. 0.15. Such remapping does not require decades of practice as shown in beautiful work by Alvaro Pascual-León at Harvard. 16. Non-musician volunteers learned a 5-finger exercise on the piano, which they practiced for 2 hours a day. Within a few days the amount of motor cortex devoted to the movement of that hand expanded, but the expansion lasted less than a day without further practice. This expansion was probably Hebbian in nature, meaning pre-existing connections transiently strengthened after repeated use. However, if subjects did the daily exercise for a crazed 4 weeks, the remapping persisted for many days afterward. This expansion probably involved axonal sprouting and the formation of new connections. Remarkably, remapping also occurred in volunteers who spent 2 hours a day, imagining playing the finger exercise. As another example of remapping, after female rats give birth, there is expansion of the tactile map representing the skin around the nipples. As a rather different example, spend 3 months learning how to juggle, and there is expansion of the cortical map for visual processing of movement. 17. Thus, experience alters the number and strength of synapses, the extent of dendritic arbor, and the projection targets of axons. Time for the biggest revolution in neuroscience in years. R. Digging deeper in the ash heap of history. He called the crude, Neanderthalish notion that new memories require new neurons, an idea discarded when Hebb was in diapers. The adult brain does not make new neurons. You've got your maximal number of neurons around birth, and it's downhill from there, thanks to aging and imprudence. You see where we're heading. Adult brains, including aged human brains, do make new neurons. The finding is truly revolutionary, its discovery epic. In 1965, an untenured associate professor at MIT named Joseph Altman, along with a longtime collaborator, Gopal Das, found the first evidence for adult neurogenesis, using a then novel technique. A newly made cell contains newly made DNA. So, find a molecule unique to DNA. Get a test tube full of the stuff and attach a minuscule, radioactive tag to each molecule. Inject it into an adult rat, wait a while, and examine its brain. If any neurons contain that radioactive tag, it means they were born during the waiting period with the radioactive marker incorporated into the new DNA. This is what Altman saw in a series of studies. 18. As even he notes, the work was initially well received, being published in good journals, generating excitement. But within a few years something shifted, and Altman and his findings were rejected by leaders in the field. It couldn't be true. He failed to get tenure, spent his career at Purdue University, lost funding for his adult neurogenesis work. Silence reigned for a decade until an assistant professor at the University of New Mexico named Michael Kaplan extended Altman's findings with some new techniques. Again, this caused mostly crushing rejection by senior figures in the field, including one of the most established men in neuroscience, Pasco Rakic of Yale. 19. Rakic publicly rejected Kaplan's, and tacitly Altman's, work, saying he had looked for new neurons himself, they weren't there, and Kaplan was mistaking other cell types for neurons. At a conference he notoriously told Kaplan, "Those may look like neurons in New Mexico, but they don't in New Haven." Kaplan soon left research, and a quarter-century later, amid the excitement of the rediscovery of adult neurogenesis, wrote a short memoir entitled Environmental Complexity, Stimulates Visual Cortex Neurogenesis, Death of a Dogma and a Research Career. The field lay dormant for another decade until unexpected evidence of adult neurogenesis emerged from the lab of Fernando Nottebohm of Rockefeller University. Nottebohm, a highly accomplished and esteemed neuroscientist, as good an old boy as you get, studied the neuroethology of birdsong. He demonstrated something remarkable. Using new, more sensitive techniques, new neurons are made in the brains of birds that learn a new territorial song each year. The quality of the science and Nottebohm's prestige silenced those who doubted that neurogenesis occurred. Instead, they questioned its relevance. Oh, that's nice for Fernando and his birdies, but what about in real species, in mammals? But this was soon convincingly shown in rats using newer, fancier techniques. Much of this was the work of two young scientists, Elizabeth Gould of Princeton and Fred Rusty Gage of the Salk Institute. Soon lots of other people were finding adult neurogenesis with these new techniques, including, lo and behold, Rakic .20. A new flavor of skepticism emerged, led by Rakic. Yes, the adult brain makes new neurons, but only a few. They don't live long, and it doesn't happen where it really counts, i.e., the cortex. Moreover, this has been shown only in rodents, not in primates. Soon it was shown in monkeys. 21 Yeah, said the skeptics, but not humans, and besides, there's no evidence that these new neurons are integrated into pre-existing circuits and actually function. All of that was eventually shown. There's considerable adult neurogenesis in the hippocampus, where roughly 3% of neurons are replaced each month and lesser amounts in the cortex. 22. It happens in humans throughout adult life. Hippocampal neurogenesis, for example, is enhanced by learning, exercise, estrogen, antidepressants, environmental enrichment, and brain injury, and inhibited by various stressors. 23 Moreover, the new hippocampal neurons integrate into pre-existing circuits with the perky excitability of young neurons in the perinatal brain. Most important, new neurons are essential for integrating new information into pre-existing schemas, something called pattern separation. This is when you learn that two things you previously thought were the same are, in fact, different. Dolphins and porpoises, baking soda and baking powder, Zooey Deschanel and Katy Perry. Adult neurogenesis is the trendiest topic in neuroscience. In the 5 years after Altman's 1965 paper was published, it was cited, a respectable, 29 times in the literature. In the last 5, more than 1,000. Current work examines how exercise stimulates the process, probably by increasing levels of certain growth factors in the brain, how new neurons know where to migrate, whether depression is caused by a failure of hippocampal neurogenesis, and whether the neurogenesis stimulated by antidepressants is required for such medications to work. 24. Why did it take so long for adult neurogenesis to be accepted? I've interacted with many of the principals and am struck by their differing takes. At one extreme is the view that while skeptics like Rakic were ham-handed, they provided quality control, and that, counter to how Path of the Hero epics go, some early work in the field was not all that solid. At the other extreme is the view that Rakic et al., having failed to find adult neurogenesis, couldn't accept that it existed. This psychohistorical view of the old guard clinging to dogma in the face of changing winds is weakened a bit by Altman's not having been a young anarchist, running amok in the archives. In fact, he is a bit older than Rakic and other principal skeptics. All of this needs to be adjudicated by historians, screenwriters, and soon, I hope, by the folks in Stockholm. Altman, who at the time of this writing is 89, published a 2011 memoir. Chapter .25. Parts of it have a plaintive, confused tone. Everyone was so excited at first. What happened? Maybe he spent too much time in the lab and too little marketing the discovery, he suggests. There's the ambivalence of someone who spent a long time as a scorned prophet, who at least got to be completely vindicated. He's philosophical about it. Hey, I'm a Hungarian Jew who escaped from a Nazi camp. You take things in stride after that. Some other domains of neuroplasticity. You've seen how an adult's experience can alter the number of synapses and dendritic branches, remap circuitry, and stimulate neurogenesis. 26. Collectively, these effects can be big enough to actually change the size of brain regions. For example, postmenopausal estrogen treatment increases the size of the hippocampus, probably through a combination of more dendritic branches and more neurons. Conversely, the hippocampus atrophies, producing cognitive problems in prolonged depression, probably reflecting its stressfulness and the typically elevated glucocorticoid levels of the disease. Memory problems and loss of hippocampal volume also occur in individuals with severe chronic pain syndromes, or with Cushing's syndrome, an array of disorders where a tumor causes extremely elevated glucocorticoid levels. Moreover, post-traumatic stress disorder is associated with increased volume, and as we know, hyperreactivity of the amygdala. In all of these instances, it is unclear how much the stress glucocorticoid effects are due to changes in neuron number or to changes in amounts of dendritic processes. One cool example of the size of a brain region changing with experience concerns the back part of the hippocampus, which plays a role in memory of spatial maps. Cab drivers use spatial maps for a living, and one renowned study showed enlargement of that part of the hippocampus in London taxi drivers. Moreover, a follow-up study imaged the hippocampus in people before and after the grueling Multer process of working and studying for the London cabbie license test, called the toughest test in the world by the New York Times. The hippocampus enlarged over the course of the process, in those who passed the test. 0.27. Thus, experience, health, and hormone fluctuations can change the size of parts of the brain in a matter of months. Experience can also cause long-lasting changes in the numbers of receptors for neurotransmitters and hormones, in levels of ion channels, and in the state of on-off switches on genes in the brain, to be covered in Chapter 8.28. With chronic stress, the nucleus accumbens is depleted of dopamine, biasing rats toward social subordination, and biasing humans toward depression. As we saw in the last chapter, if a rodent wins a fight on his home territory, there are long-lasting increases in levels of testosterone receptors in the nucleus accumbens and ventral tegmentum, enhancing testosterone's pleasurable effects. There's even a parasite called Toxoplasma gondii that can infect the brain over the course of weeks to months. It makes rats less fearful of the smell of cats and makes humans less fearful and more impulsive in subtle ways. Basically, most anything you can measure in the nervous system can change in response to a sustained stimulus. And importantly, these changes are often reversible in a different environment. Some conclusions. The discovery of adult neurogenesis is revolutionary, and the general topic of neuroplasticity in all its guises is immensely important, as is often the case when something the experts said couldn't be turns out to be. 0.29. The subject is also fascinating because of the nature of the revisionism. Neuroplasticity radiates optimism. Books on the topic are entitled The Brain That Changes Itself, Train Your Mind, Change Your Brain, and Rewire Your Brain, Think Your Way to a Better Life, hinting at the new neurology, i.e., no more need for neurology once we can fully harness neuroplasticity. There's can-do Horatio Alger spirit every which way you look. Amid that, some cautionary points. One recalls caveats aired in other chapters. The ability of the brain to change in response to experience is value-free. Axonal remapping in blind or deaf individuals is great, exciting, and moving. It's cool that your hippocampus expands if you drive a London cab. Ditto about the size and specialization of the auditory cortex in the triangle player in the orchestra. But at the other end, it's disastrous that trauma enlarges the amygdala and atrophies the hippocampus, crippling those with PTSD. Similarly, expanding the amount of motor cortex devoted to finger dexterity is great in neurosurgeons, but probably not a societal plus in safe crackers. The extent of neuroplasticity is most definitely finite. Otherwise, grievously injured brains and severed spinal cords would ultimately heal. Moreover, the limits of neuroplasticity are quotidian. Malcolm Gladwell has explored how vastly skilled individuals have put in vast amounts of practice. 10,000 hours is his magic number. Nevertheless, the reverse doesn't hold. 10,000 hours of practice does not guarantee the neuroplasticity needed to make any of us a Yo-Yo Ma or LeBron James. Manipulating neuroplasticity for recovery of function does have enormous, exciting potential in neurology. But this domain is far from the concerns of this book. Despite neuroplasticity's potential, it's unlikely that we'll ever be able to, say, spritz neuronal growth factors up people's noses to make them more open-minded or empathic, or to target neuroplasticity with gene therapy to blunt some jerk's tendency to displace aggression. So what's the subject good for in the realm of this book? I think the benefits are mostly psychological. This recalls a point from Chapter 2 in the discussion of the neuroimaging studies demonstrating loss of volume in the hippocampus of people with PTSD, certainly an example of the adverse effects of neuroplasticity. I sniped that it was ridiculous that many legislators needed pictures of the brain to believe that there was something desperately, organically wrong with veterans with PTSD. Similarly, neuroplasticity makes the functional malleability of the brain tangible, makes it scientifically demonstrated that brains change. That people change. In the time span considered in this chapter, people throughout the Arab world went from being voiceless to toppling tyrants, Rosa Parks went from victim to catalyst, Sadat and Begin from enemies to architects of peace, Mandela from prisoner to statesman. And you'd better bet that changes along the lines of those presented in this chapter occurred in the brains of anyone transformed by these transformations. A different world makes for a different worldview, which means a different brain. And the more tangible and real the neurobiology underlying such change seems, the easier it is to imagine that it can happen again. Adolescence, or Dude, Where's My Frontal Cortex? His chapter is the first of two focusing on development. We've established our rhythm, a behavior has just occurred. What events in the prior seconds, minutes, hours, and so on helped bring it about? The next chapter extends this into the developmental domain, what happened during that individual's childhood and fetal life that contributed to the behavior. The present chapter breaks this rhythm in focusing on adolescence. Does the biology introduced in the preceding chapters work differently in an adolescent than in an adult, producing different behaviors? Yes. One fact dominates this chapter. Chapter 5 did in the dogma that adult brains are set in stone. Another dogma was that brains are pretty much wired up early in childhood after all, by age 2, brains are already about 85% of adult volume. But the developmental trajectory is much slower than that. This chapter's key fact is that the final brain region to fully mature in terms of synapse number, myelination, and metabolism is the frontal cortex, not going fully online until the mid-20s. This has two screamingly important implications. First, no part of the adult brain is more shaped by adolescence than the frontal cortex. Second, nothing about adolescence can be understood outside the context of delayed frontocortical maturation. If by adolescence limbic, autonomic, and endocrine systems are going full blast while the frontal cortex is still working out the assembly instructions, we've just explained why adolescents are so frustrating, great, asinine, impulsive, inspiring, destructive, self-destructive, selfless, selfish, impossible, and world-changing. Think about this, adolescence and early adulthood are the times when someone is most likely to kill, be killed, leave home forever, invent an art form, help overthrow a dictator, ethnically cleanse a village, devote themselves to the needy, become addicted, marry outside their group, transform physics, have hideous fashion taste, break their neck recreationally, commit their life to God, mug an old lady, or be convinced that all of history has converged to make this moment the most consequential. The most fraught with peril and promise, the most demanding that they get involved and make a difference. In other words, it's the time of life of maximal risk-taking, novelty-seeking, and affiliation with peers. All because of that immature frontal cortex. The reality of adolescence. Adolescence real? Is there something qualitatively different distinguishing it from before and after, rather than being part of a smooth progression from childhood to adulthood? Maybe adolescence is just a cultural construct in the West as better nutrition and health resulted in earlier puberty onset and the educational and economic forces of modernity pushed for childbearing at later ages, a developmental gap emerged between the two. Voila! The invention of adolescence too. As we'll see, neurobiology suggests that adolescence is for real, that the adolescent brain is not merely a half-cooked adult brain or a child's brain left and refrigerated for too long. Moreover, most traditional cultures do recognize adolescence as distinct, i.e., it brings some but not all of the rights and responsibilities of adulthood. Nonetheless, what the West invented is the longest period of adolescence. What does seem a construct of individualistic cultures is adolescence as a period of intergenerational conflict. Youth of collectivist cultures seem less prone toward rolling at the dorkiness of adults, starting with parents. Moreover, even within individualistic cultures, adolescence is not universally a time of acne of the psyche, of Sturm und Drang. Most of us get through it just fine. The nuts and bolts of frontal cortical maturation. Delayed maturation of the frontal cortex suggests an obvious scenario, namely that early in adolescence the frontal cortex has fewer neurons, dendritic branches, and synapses than in adulthood, and that levels increase into the mid-20s. Instead, levels decrease. This occurs because of a truly clever thing evolved by mammalian brains. Remarkably, the fetal brain generates far more neurons than are found in the adult. Why? During late fetal development, there is a dramatic competition in much of the brain, with winning neurons being the ones that migrate to the correct location and maximize synaptic connections to other neurons. And neurons that don't make the grade, they undergo programmed cell death, genes are activated that cause them to shrivel and die, their materials then recycled. Neuronal overproduction followed by competitive pruning, which has been termed neural Darwinism, allowed the evolution of more optimized neural circuitry, a case of less being more. The same occurs in the adolescent frontal cortex. By the start of adolescence, there's a greater volume of gray matter, an indirect measure of the total number of neurons and dendritic branches, and more synapses than in adults. Over the next decade, gray matter thickness declines as less optimal, dendritic processes and connections are pruned away within the frontal cortex. The evolutionarily oldest subregions mature first. The spanking new, cognitive, dorsolateral PFC doesn't even start losing gray matter volume until late adolescence. The importance of this developmental pattern was shown in a landmark study, in which children were neuroimaged and IQ tested repeatedly into adulthood. The longer the period of packing on gray matter cortical thickness in early adolescence before the pruning started, the higher the adult IQ. Thus, frontal cortical maturation during adolescence is about a more efficient brain, not more brain. This is shown in easily misinterpreted neuroimaging studies, comparing adolescents and adults. Point 4. A frequent theme is how adults have more executive control over behavior during some tasks than do adolescents, and show more frontal cortical activation at the time. Now find a task where, atypically, adolescents manage a level of executive control equal to that of adults. In those situations adolescents show more frontal activation than adults. Equivalent regulation takes less effort in a well-pruned adult frontal cortex. That the adolescent frontal cortex is not yet lean and mean is demonstrable in additional ways. For example, adolescents are not at adult levels of competence at detecting irony and, when trying to do so, activate the DMPFC more than do adults. In contrast, adults show more activation in the fusiform face region. In other words, detecting irony isn't much of a frontal task for an adult. One look at the face is enough. Point 5. What about white matter in the frontal cortex, that indirect measure of myelination of axons? Here things differ from the overproduce, then prune approach to gray matter. Instead, axons are myelinated throughout adolescence. As discussed in Appendix 1, this allows neurons to communicate in a more rapid, coordinated manner. As adolescence progresses, activity in different parts of the frontal cortex becomes more correlated as the region operates as more of a functional unit. Point 6. This is important. When learning neuroscience, it's easy to focus on individual brain regions as functionally distinct, and this tendency worsens if you, then spend a career studying just one of them. As a measure of this, there are two high-quality, biomedical journals out there, one called Cortex, the other Hippocampus, each publishing papers about its favorite brain region. At neuroscience meetings attended by tens of thousands, there will be social functions for all the people studying the same obscure brain region, a place where they can gossip and bond and court. But in reality the brain is about circuits, about the patterns of functional connectivity among regions. The growing myelination of the adolescent brain shows the importance of increased connectivity. Interestingly, other parts of the adolescent brain seem to help out the underdeveloped frontal cortex, taking on some roles that it's not yet ready for. For example, in adolescents but not adults, the ventral striatum helps regulate emotions. We will return to this. Point 7. Something else keeps that tyrofrontal cortex off kilter, namely estrogen and progesterone in females and testosterone in males. As discussed in Chapter 4, these hormones alter brain structure and function, including in the frontal cortex, where gonadal hormones change rates of myelination and levels of receptors for various neurotransmitters. Logically, landmarks of adolescent maturation in brain and behavior are less related to chronological age than to the time since puberty onset. Point 8. Moreover, puberty is not just about the onslaught of gonadal hormones. It's about how they come online. Point 9. The defining feature of ovarian endocrine function is the cyclicity of hormone release. It's that time of the month. In adolescent females, puberty does not arrive full flower, so to speak, with one's first period. Instead, for the first few years, only about half of cycles actually involve ovulation and surges of estrogen and progesterone. Thus, not only are young adolescents experiencing these first ovulatory cycles, but there are also higher-order fluctuations in whether the ovulatory fluctuation occurs. Meanwhile, while adolescent males don't have equivalent hormonal gyrations, it can't help that their frontal cortex keeps getting hypoxic from the priapic blood flow to the crotch. Thus, as adolescence dawns, frontal cortical efficiency is diluted with extraneous synapses failing to make the grade, sluggish communication thanks to undermyelination, and a jumble of uncoordinated subregions working at cross purposes. Moreover, while the striatum is trying to help, a pinch hitter for the frontal cortex gets you only so far. Finally, the frontal cortex is being pickled in that ebb and flow of gonadal hormones. No wonder they act adolescent. Frontal cortical changes in cognition in adolescence. To appreciate what frontal cortical maturation has to do with our best and worst behaviors, it's helpful to first see how such maturation plays out in cognitive realms. During adolescence, there's steady improvement in working memory, flexible rule use, executive organization, and frontal inhibitory regulation, e.g., task shifting. In general, these improvements are accompanied by increasing activity in frontal regions during tasks, with the extent of the increase predicting accuracy 0.10. Adolescents also improve at mentalization tasks, understanding someone else's perspective. By this I don't mean emotional perspective, stay tuned, but purer cognitive challenges, like understanding what objects look like from someone else's perspective. The improvement in detecting irony reflects improvement in abstract, cognitive perspective taking. Frontal cortical changes in emotional regulation. Older teenagers experience emotions more intensely than do children or adults, something obvious to anyone who ever spent time as a teenager. For example, they are more reactive to faces expressing strong emotions. 11 In adults, looking at an effective facial display activates the amygdala, followed by activation of the emotion-regulating VMPFC as they habituate to the emotional content. In adolescence, though, the VMPFC response is less, thus the amygdaloid response keeps growing. Chapter 2 introduced reappraisal, in which responses to strong emotional stimuli are regulated by thinking about them differently. 12. Get a bad grade on an exam, and there's an emotional pull toward "I'm stupid." Reappraisal might lead you instead to focus on your not having studied, or having had a cold, to decide that the outcome was situational rather than a function of your unchangeable constitution. Reappraisal strategies get better during adolescence, with logical neurobiological underpinnings. Recall how in early adolescence, the ventral striatum, trying to be helpful, takes on some frontal tasks, fairly ineffectively, as it's working above its pay grade. At that age, reappraisal engages the ventral striatum, more activation predicts less amygdaloid activation and better emotional regulation. As the adolescent mature, the prefrontal cortex takes over the task and emotions get steadier. 13. Bringing the striatum into the picture brings up dopamine and reward, thus bringing up the predilection of adolescents for bungee jumping. Adolescent risk-taking. And the foothills of the Sierras are California caverns, a cave system that leads, after an initial narrow, twisting 30-foot descent down a hole, to an abrupt 180-foot drop, now navigable by rappelling. The Park Service has found skeletons, at the bottom dating back centuries, explorers who took one step too far in the gloom. And the skeletons are always those of adolescents. As shown experimentally, during risky decision-making, adolescents activate the prefrontal cortex less than do adults. The less activity, the poorer the risk assessment. This poor assessment takes a particular form, as shown by Sarah Jane Blakemore of University College, London. 14. Have subjects estimate the likelihood of some event occurring, winning the lottery, dying in a plane crash, then tell them the actual likelihood. Such feedback can constitute good news, i.e., something good is actually more likely than the person estimated, or something bad is less likely. Conversely, the feedback can constitute bad news. Ask subjects to estimate the likelihood of the same events again. Adults incorporate the feedback into the new estimates. Adolescents update their estimates as adults do for good news, but feedback about bad news barely makes a dent. Researcher, how likely are you to have a car accident if you're driving while drunk? Adolescent, 1 chance in a gazillion. Researcher, actually, the risk is about 50%. What do you think your own chances are now? Adolescent, hey, we're talking about me, 1 chance in a gazillion. We've just explained why adolescents have 2 to 4 times the rate of pathological gambling, as do adults. 0.15. So adolescents take more risks and stink at risk assessment. But it's not just that teenagers are more willing to take risks. After all, adolescents and adults don't equally desire to do something risky, and the adults simply don't do it because of their frontal cortical maturity. There is an age difference in the sensations sought. Adolescents are tempted to bungee jump, adults are tempted to cheat on their low-salt diet. Adolescence is characterized not only by more risking, but by more novelty seeking as well. 16. Novelty craving permeates adolescence. It is when we usually develop our stable tastes in music, food, and fashion, with openness to novelty declining thereafter. 0.17. And it's not just a human phenomenon. Across the rodent lifespan, it's adolescents who are most willing to eat a new food. Adolescent novelty seeking is particularly strong in other primates. Among many social mammals, adolescents of one sex leave their natal group, emigrating into another population, a classic means to avoid inbreeding. Among impalas there are groups of related females and offspring with one breeding male. The other males knock around disconsolately in bachelor herds, each scheming to usurp the breeding male. When a young male hits puberty, he is driven from the group by the breeding male, and to avoid some Oedipus nonsense, this is unlikely to be his father, who reigned many breeding males ago. But not among primates. Take baboons. Suppose 2 troops encounter each other at some natural boundary, say, a stream. The males threaten each other for a while, eventually get bored, and resume whatever they were doing. Except there's an adolescent standing at the stream's edge, riveted. New baboons, a whole bunch of them. He runs 5 steps toward them, runs back 4, nervous, agitated. He gingerly crosses and sits on the other bank, scampering back should any new baboon glance at him. So begins the slow process of transferring, spending more time each day with a new troop until he breaks the umbilical cord and spends the night. He wasn't pushed out. Instead, if he has to spend one more day with the same monotonous baboons he's known his whole life, he'll scream. Among adolescent chimps it's females who can't get off the farm fast enough. We primates aren't driven out at adolescence. Instead we desperately crave novelty. Thus, adolescence is about risk-taking and novelty-seeking. Where does the dopamine reward system fit in? Recall from Chapter 2 how the ventral tegmentum is the source of the mesolimbic dopamine projection to the nucleus accumbens, and of the mesocortical dopamine projection to the frontal cortex. During adolescence, dopamine projection density and signaling steadily increase in both pathways, although novelty seeking itself peaks at mid-adolescence, probably reflecting the emerging frontal regulation after that. 0.18. Changes in the amount of dopaminergic activity in the reward center of the brain following different magnitudes of reward. For the adolescents, the highs are higher, the lows lower. Visit bit.ly/203t/8 for a larger version of this graph. It's unclear how much dopamine is released in anticipation of reward. Some studies show more anticipatory activation of reward pathways in adolescents than in adults, while others show the opposite, with the least dopaminergic responsiveness in adolescents who are most risk-taking. 0.19. Age differences in absolute levels of dopamine are less interesting than differences in patterns of release. In a great study, children, adolescents, and adults in brain scanners did some task where correct responses produced monetary rewards of varying sizes. See figure above. 0.20. During this, prefrontal activation in both children and adolescents was diffuse and unfocused. However, activation in the nucleus accumbens in adolescents was distinctive. In children, a correct answer produced roughly the same increase in activity regardless of size of reward. In adults, small, medium, and large rewards caused small, medium, and large increases in accumbens activity. And adolescents. After a medium reward things looked the same as in kids and adults. A large reward produced a humongous increase, much bigger than in adults. And the small reward? Accumbens activity declined. In other words, adolescents experience bigger than expected rewards, more positively than do adults, and smaller than expected rewards as aversive. A gyrating top, nearly skittering out of control. This suggests that in adolescence strong rewards produce exaggerated dopaminergic signaling, and nice sensible rewards for prudent actions feel lousy. The immature frontal cortex hasn't a prayer to counteract the dopamine system like this. But there is something puzzling. Amid their crazy, unrestrained dopamine neurons, adolescents have reasoning skills that, in many domains of perceiving risk, match those of adults. Yet despite that, logic and reasoning are often jettisoned, and adolescents act adolescent. Work by Lawrence Steinberg of Temple University has identified a key juncture where adolescents are particularly likely to leap before looking when around peers. A. Peers, social acceptance, and social exclusion. Adolescent vulnerability to peer pressure from friends, especially peers they want to accept them as friends, is storied. It can also be demonstrated experimentally. In one Steinberg study, adolescents and adults took risks at the same rate in a video driving game. Adding two peers to egg them on had no effect on adults but tripled risk-taking in adolescents. Moreover, in neuroimaging studies, peers egging subjects on, by intercom, lessens VMPFC activity and enhances ventral striatal activity in adolescents but not adults. 21. Why do adolescents' peers have such social power? For starters, adolescents are more social and more complexly social than children or adults. For example, a 2013 study showed that teens average more than 400 Facebook friends, far more than do adults. 22. Moreover, teen sociality is particularly about affect and responsiveness to emotional signaling. Recall the greater limbic and lesser frontal cortical response to emotional faces in adolescence. And teens don't rack up 400 Facebook friends for data for their sociology doctorates. Instead, there is the frantic need to belong. This produces teen vulnerability to peer pressure and emotional contagion. Moreover, such pressure is typically deviance training, increasing the odds of violence, substance abuse, crime, unsafe sex, and poor health habits. Few teen gangs pressure kids to join them in tooth flossing followed by random acts of kindness. For example, in college dorms the excessive drinker is more likely to influence the teetotaling roommate than the reverse. The incidence of eating disorders in adolescence spreads among peers with a pattern resembling viral contagion. The same occurs with depression among female adolescents, reflecting their tendency to co-ruminate on problems, reinforcing one another's negative effect. Neuroimaging studies show the dramatic sensitivity of adolescents to peers. Ask adults to think about what they imagine others think of them, then about what they think of themselves. Two different, partially overlapping networks of frontal and limbic structures activate for the two tasks. But with adolescents the two profiles are the same. What do you think about yourself? Is neurally answered with whatever everyone else thinks about me. 23. The frantic adolescent need to belong is shown beautifully in studies of the neurobiology of social exclusion. Naomi Eisenberger of UCLA developed the fiendishly clever cyberball paradigm to make people feel snubbed. 24. The subject lies in a brain scanner, believing she is playing an online game with two other people. Naturally, they don't exist, it's a computer program. Each player occupies a spot on the screen, forming a triangle. The players toss a virtual ball among themselves. The subject is picking whom to throw to and believes the other two are doing the same. The ball is tossed for a while, then, unbeknownst to the subject, the experiment begins. The other two players stop, throwing the ball to her. She's being excluded by those creeps. In adults there is activation of the periaqueductal gray, anterior cingulate, amygdala, and insular cortex. Perfect. These regions are central to pain perception, anger, and disgust, and then, after a delay, the ventrolateral PFC activates. The more activation, the more the cingulate and insula are silenced and the less subjects report being upset afterward. What's this delayed VLPFC activation about? Why am I getting upset? This is just a stupid game of catch. The frontal cortex comes to the rescue with perspective, rationalization, and emotion regulation. Now do the study with teenagers. Some show the adult neuroimaging profiles. These are ones who rate themselves as least sensitive to rejection and who spend the most time with friends. But for most teenagers, when social exclusion occurs, the VLPFC barely activates, the other changes are bigger than in adults, and the subjects report feeling lousier. Adolescents lack sufficient frontal forcefulness to effectively hand-wave about why it doesn't matter. Rejection hurts adolescents more, producing that stronger need to fit in. 25. One neuroimaging study examined a neural building block of conformity. 26. Watch a hand moving, and neurons in premotor regions that contribute to moving your own hand become a bit active. Your brain is on the edge of imitating the movement. In the study, 10-year-olds watched film clips of hand movements or facial expressions. Those most vulnerable to peer influence, assessed on a scale developed by Steinberg, had the most premotor activation, but only for emotional facial expressions. In other words, kids who are more sensitive to peer pressure are more prepared to imitate someone else's emotionality. Given the age of the subjects, the authors framed their findings as potentially predictive of later teen behavior. This atomistic level of explaining conformity might predict something about which teens are likely to join in a riot. But it doesn't tell much about who chooses not to invite someone to a party because the cool kids think she's a loser. Another study showed neurobiological correlates of more abstract peer conformity. Recall how the adolescent ventral striatum helps the frontal cortex reappraise social exclusion. In this study, young adolescents most resistant to peer influence had the strongest such ventral striatal responses. And where might a stronger ventral striatum come from? You know the answer by now. You'll see in the remaining chapters. B. Empathy, sympathy, and moral reasoning. Why adolescents? People are typically pretty good at perspective taking, seeing the world as someone else would. That's usually when you'll first hear the likes of, well, I still disagree, but I can see how he feels that way, given his experience. Nonetheless, adolescents are not yet adults. Unlike adults, they are still better at first and third person perspective taking. How would you feel in her situation? Versus how does she feel in her situation? Point 27. Adolescent moral judgments, while growing in sophistication, are still not at adult levels. Adolescents have left behind children's egalitarian tendency to split resources evenly. Instead, adolescents mostly make meritocratic decisions with a smattering of utilitarian and libertarian viewpoints thrown in. Meritocratic thinking is more sophisticated than egalitarian, since the latter is solely about outcomes while the former incorporates thinking about causes. Nonetheless, adolescents' meritocratic thinking is less complex than adults. For example, adolescents are as adept as adults at understanding how individual circumstances impact behavior, but not at understanding systemic circumstances. As adolescents mature, they increasingly distinguish between intentional and accidental harm, viewing the former as worse. Point 28. When contemplating the latter, there is now less activation of three brain regions related to pain processing, namely the amygdala, the insula, and the premotor areas, the last reflecting the tendency to cringe when hearing about pain being inflicted. Meanwhile, there is increasing DLPFC and VMPFC activation when contemplating intentional harm. In other words, it is a frontal task to appreciate the painfulness of someone's being harmed intentionally. As adolescents mature, they also increasingly distinguish between harm to people and harm to objects, with the former viewed as worse. Harm to people increasingly activates the amygdala while the opposite occurs for harm to objects. Interestingly, as adolescents age, there is less differentiation between recommended punishment for intentional and unintentional damage to objects. In other words, the salient point about the damage becomes that, accidental or otherwise, the damn thing needs to be fixed. Even if there is less crying over spilled milk, there is no less cleaning required. What about one of the greatest things about adolescence with respect to this book's concerns, their frenzied, agitated, incandescent ability to feel someone else's pain, to feel everyone's pain, to try to make everything right? A later chapter distinguishes between sympathy and empathy, between feeling for someone in pain and feeling as that someone. Adolescents are specialists at the latter, where the intensity of feeling as the other can border on being the other. This intensity is no surprise, being at the intersection of many facets of adolescence. There are the abundant emotions and limbic gyrations. The highs are higher, the lows lower, empathic pain scalds, and the glow of doing the right thing makes it seem plausible that we are here for a purpose. Another contributing factor is the openness to novelty. An open mind is a prerequisite for an open heart, and the adolescent hunger for new experiences makes possible walking miles in lots of other people's shoes. And there is the egoism of adolescence. During my late adolescence I hung out with Quakers, and they'd occasionally use the aphorism, all God has is thee. This is the God of limited means, not just needing the help of humans to right a wrong, but needing you only, to do so. The appeal to egoism is tailor-made for adolescence. Throw an inexhaustible adolescent energy plus a feeling of omnipotence, and it seems possible to make the world whole, so why not? In Chapter 13 we consider how neither the most burning emotional capacity for empathy, nor the most highfalutin moral reasoning makes someone likely to actually do the brave, difficult thing. This raises a subtle limitation of adolescent empathy. As will be seen, one instance where empathic responses don't necessarily lead to acts is when we think enough to rationalize it's overblown as a problem, or someone else will fix it. But feeling too much has problems as well. Feeling someone else's pain is painful, and people who do so most strongly, with the most pronounced arousal and anxiety, are actually less likely to act prosocially. Instead, the personal distress induces a self-focus that prompts avoidance. This is too awful, I can't stay here any longer. As empathic pain increases, your own pain becomes your primary concern. In contrast, the more individuals can regulate their adverse empathic emotions, the more likely they are to act prosocially. Related to that, if a distressing, empathy-evoking circumstance increases your heart rate, you're less likely to act prosocially than if it decreases it. Thus, one predictor of who actually acts is the ability to gain some detachment, to ride, rather than be submerged by the wave of empathy. Where do adolescents fit in, with their hearts on their sleeves, fully charged limbic systems and frontal cortices straining to catch up? It's obvious. A tendency toward empathic hyperarousal that can disrupt acting, effectively. 29. This adolescent empathy frenzy can seem a bit much for adults. But when I see my best students in that state, I have the same thought: it used to be so much easier to be like that. My adult frontal cortex may enable whatever detached good I do. The trouble, of course, is how that same detachment makes it easy to decide that something is not my problem. Oh. Adolescent violence. Obviously, the adolescent years are not just about organizing bake sales to fight global warming. Late adolescence and early adulthood are when violence peaks, whether premeditated or impulsive murder, Victorian fisticuffs or handguns, solitary or organized, in or out of a uniform, focused on a stranger or on an intimate partner. And then rates plummet. As has been said, the greatest crime-fighting tool is a 30th birthday. On a certain level, the biology underlying the teenaged mugger is similar to that of the teen who joins the ecology club and donates his allowance to help save the mountain gorillas. It's the usual, heightened emotional intensity, craving for peer approval, novelty seeking, and that frontal cortex. But that's where similarities end. What underlies the adolescent peak in violence? Neuroimaging shows nothing particularly distinct about it versus adult violence. 30. Adolescent and adult psychopaths both have less sensitivity of the PFC and the dopamine system to negative feedback, less pain sensitivity, and less amygdaloid frontal cortical coupling during tasks of moral reasoning or empathy. Moreover, the adolescent peak of violence isn't caused by the surge in testosterone. Harking back to Chapter 4, testosterone no more causes violence in adolescence than it does in adult males. Moreover, testosterone levels peak during early adolescence, but violence peaks later. The next chapter considers some of the roots of adolescent violence. For now, the important point is that an average adolescent doesn't have the self-regulation or judgment of an average adult. This can prompt us to view teenage offenders as having less responsibility than adults for criminal acts. An alternative view is that even amid poorer judgment and self-regulation, there is still enough to merit equivalent sentencing. The former view has held in two landmark Supreme Court decisions. In the first, 2005's Roper v. Simmons, the court ruled 5-4 that executing someone for crimes committed before age 18 is unconstitutional, violating the Eighth Amendment ban on cruel and unusual punishment. Then in 2012's Miller v. Alabama, in another 5-4 split, the court banned mandatory life sentences without the chance of parole for juvenile offenders on similar grounds. 31. The court's reasoning was straight out of this chapter. Writing for the majority in Roper v. Simmons, Justice Anthony Kennedy said, "First, as everyone knows, a lack of maturity and an underdeveloped sense of responsibility are found in youth more often than in adults, and are more understandable among the young. These qualities often result in impetuous and ill-considered actions and decisions." 32. I fully agree with these rulings. But, to show my hand early, I think this is just window dressing. As will be covered in the screed that constitutes Chapter 16, I think the science encapsulated in this book should transform every nook and cranny of the criminal justice system. A final thought: why can't the frontal cortex just act its age? As promised, this chapter's dominant fact has been the delayed maturation of the frontal cortex. Why should the delay occur? Is it because the frontal cortex is the brain's most complicated construction project? Probably not. The frontal cortex uses the same neurotransmitter systems as the rest of the brain and uses the same basic neurons. Neuronal density and complexity of interconnections are similar to the rest of the fancy cortex. It isn't markedly harder to build frontal cortex than any other cortical region. Thus, it is not likely that if the brain could grow a frontal cortex as fast as the rest of the cortex, it would. Instead, I think there was evolutionary selection for delayed frontal cortex maturation. If the frontal cortex matured as fast as the rest of the brain, there'd be none of the adolescent turbulence, none of the antsy, itchy exploration and creativity, none of the long line of pimply adolescent geniuses who dropped out of school and worked away in their garages to invent fire, cave painting, and the wheel. Maybe. But this just so story must accommodate behavior evolving to pass on copies of the genes of individuals, not for the good of the species. Stay tuned for Chapter 10. And for every individual who scored big time, reproductively thanks to adolescent inventiveness, there've been far more who instead broke their necks from adolescent imprudence. I don't think delayed frontal cortical maturation evolved so that adolescents could act over the top. Instead, I think it is delayed so that the brain gets it right. Well, duh, the brain needs to get it right, with all its parts. But in a distinctive way in the frontal cortex. The point of the previous chapter was the brain's plasticity. New synapses form, new neurons are born, circuits rewire, brain regions expand or contract, we learn, change, adapt. This is nowhere more important than in the frontal cortex. An oft-repeated fact about adolescence is how emotional intelligence and social intelligence predict adult success and happiness better than do IQ or SAT scores. 33. It's all about social memory, emotional perspective taking, impulse control, empathy, ability to work with others, self-regulation. There is a parallel in other primates with their big, slowly maturing frontal cortices. For example, what makes for a successful male baboon in his dominance hierarchy? Attaining high rank is about muscle, sharp canines, well-timed aggression. But once high status is achieved, maintaining it is all about social smarts, knowing which coalitions to form, how to intimidate a rival, having sufficient impulse control to ignore most provocations and to keep displacement aggression to a reasonable level. Similarly, as noted in Chapter 2, among male rhesus monkeys a large prefrontal cortex goes hand in hand with social dominance. Adult life is filled with consequential forks in the road where the right thing is definitely harder. Navigating these successfully is the portfolio of the frontal cortex, and developing the ability to do this right in each context requires profound shaping by experience. This may be the answer. As we will see in Chapter 8, the brain is heavily influenced by genes. But from birth through young adulthood, the part of the human brain that most defines us is less a product of the genes with which you started life than of what life has thrown at you. Because it is the last to mature, by definition the frontal cortex is the brain region least constrained by genes and most sculpted by experience. This must be so, to be the supremely complex, social species that we are. Ironically, it seems that the genetic program of human brain development has evolved to, as much as possible, free the frontal cortex from genes. 7. Back to the crib, back to the womb. There journeying to planet adolescence, we resume our basic approach. Our behavior, good, bad, or ambiguous, has occurred. Why? When seeking the roots of behavior, long before neurons or hormones come to mind, we typically look first at childhood. C. Complexification. Childhood is obviously about increasing complexity in every realm of behavior, thought, and emotion. Crucially, such increasing complexity typically emerges in stereotypical, universal sequences of stages. Most child behavioral development research is implicitly stage-oriented, concerning a. the sequence with which stages emerge, b. how experience influences the speed and surety with which that sequential tape of maturation unreels, and c. how this helps create the adult a child ultimately becomes. We start by examining the neurobiology of the stage nature of development. T. A brief tour of brain development. Stages of human brain development make sense. A few weeks after conception, a wave of neurons are born and migrate to their correction locations. Around 20 weeks, there is a burst of synapse formation, neurons start talking to one another. And then axons start being wrapped in myelin, the glial cell insulation, forming white matter that speeds up action. Neuron formation, migration, and synaptogenesis are mostly prenatal in humans. 1. In contrast, there is little myelin at birth, particularly in evolutionarily newer brain regions. As we've seen, myelination proceeds for a quarter century. The stages of myelination and consequent functional development are stereotypical. For example, the cortical region central to language comprehension myelinates a few months earlier than that for language production. Kids understand language before producing it. Myelination is most consequential when enwrapping the longest axons in neurons that communicate the greatest distances. Thus myelination particularly facilitates brain regions talking to one another. No brain region is an island, and the formation of circuits connecting far-flung brain regions is crucial. How else can the frontal cortex use its few myelinated neurons to talk to neurons in the brain's subbasement to make you toilet trained too? As we saw, mammalian fetuses overproduce neurons and synapses. Ineffective or unessential synapses and neurons are pruned, producing leaner, meaner, more efficient circuitry. To reiterate a theme from the last chapter, the later a particular brain region mature, the less it is shaped by genes and the more by environment. 3. W. Stages. Stages of child development help explain the good and bad / in-between adult behavior that got the ball rolling in Chapter 1. The mother of all developmental stage theories was supplied in 1923, pioneered by Jean Piaget's clever, elegant experiments revealing 4 stages of cognitive development. 4. Sensory motor stage, birth to approximately 24 months. Thought concerns only what the child can directly sense and explore. During this stage, typically at around 8 months, children develop object permanence, understanding that even if they can't see an object, it still exists. The infant can generate a mental image of something no longer there. Preoperational stage, approximately 2 to 7 years. The child can maintain ideas about how the world works without explicit examples in front of him. Thoughts are increasingly symbolic, imaginary play abounds. However, reasoning is intuitive, no logic, no cause and effect. This is when kids can't yet demonstrate conservation of volume. Identical beakers A and B are filled with equal amounts of water. Pour the contents of beaker B into beaker C, which is taller and thinner. Ask the child, which has more water, A or C. Kids in the preoperational stage use incorrect folk intuition. The water line in C is higher than that in A, it must contain more water. Concrete operational stage 7 to 12 years. Kids think logically, no longer falling for that different-shaped beakers nonsense. However, generalizing logic from specific cases is iffy. As is abstract thinking. For example, proverbs are interpreted literally. Birds of a feather flock together means that similar birds form flocks. Formal operational stage, adolescence onward. Approaching adult levels of abstraction, reasoning, and metacognition. Kid playing hide and seek while in the if I can't see you, or even if I can't see you as easily as usual, then you can't see me, stage. Other aspects of cognitive development are also conceptualized in stages. An early stage occurs when toddlers form ego boundaries. There is a me, separate from everyone else. A lack of ego boundaries is shown when a toddler isn't all that solid on where he ends and mommy starts. She's cut her finger, and he claims his finger hurts. 5. Next comes the stage of realizing that other individuals have different information than you do. 9-month-olds look where someone points, as can other apes and dogs, knowing the pointer has information that they don't. This is fueled by motivation. Where is that toy? Where's she looking? Older kids understand more broadly that other people have different thoughts, beliefs, and knowledge than they. The landmark of achieving theory of mind, Tom. 6. Here's what not having Tom looks like. A 2-year-old and an adult see a cookie placed in box A. The adult leaves, and the researcher switches the cookie to box B. Ask the child, when that person comes back, where will he look for the cookie? Box B. The child knows it's there and thus everyone knows. Around age 3 or 4 the child can reason, they'll think it's in A, even though I know it's in B. Shazam, Tom. Mastering such false belief tests is a major developmental landmark. Tom then progresses to fancier insightfulness, e.g., grasping irony, perspective taking, or secondary Tom understanding person A's Tom about person, B.7. Various cortical regions mediate Tom, parts of the medial PFC, surprise. And some new players, including the precuneus, the superior temporal sulcus and the temporoparietal junction, TPJ. This is shown with neuroimaging, by Tom deficits. If these regions are damaged, autistic individuals who have limited Tom have decreased gray matter and activity in the superior temporal sulcus, and by the fact that if you temporarily inactivate the TPJ, people don't consider someone's intentions when judging them morally. 8. Thus there are stages of gaze following, followed by primary Tom, then secondary Tom, then perspective taking, with the speed of transitions influenced by experience, e.g., kids with older siblings achieve Tom earlier than average. 9. Naturally, there are criticisms of stage approaches to cognitive development. One is at the heart of this book. A Piagetian framework sits in a cognition bucket, ignoring the impact of social and emotional factors. One example to be discussed in Chapter 12 concerns preverbal infants who sure don't grasp transitivity. If AB and BC, then AC. Show a violation of transitivity in interactions between shapes on a screen. Shape A should knock over shape C, but the opposite occurs, and the kid is unbothered, doesn't look for long. But personify the shapes with eyes and a mouth, and now heart rate increases, the kid looks longer. Whoa, character C is supposed to move out of character A's way, not the reverse. Humans understand logical operations between individuals earlier than between objects. 10. Social and motivational state can shift cognitive stage as well. Rudiments of Tom are more demonstrable in chimps who are interacting with another chimp versus a human, and if there is something motivating, food, involved. 11. Emotion and affect can alter cognitive stage in remarkably local ways. I saw a wonderful example of this when my daughter displayed both Tom and failure of Tom in the same breath. She had changed preschools and was visiting her old class. She told everyone about life in her new school. Then, after lunch, we play on the swings. There are swings at my new school. And then, after that, we go inside and Carolee reads us a story. Then, after that, Tom, play on the swings. Wait, they don't know that my school has swings. I need to tell them. Failure of Tom, Carolee reads us a story. Carolee, the teacher at her new school. The same logic should apply, tell them who Carolee is. But because Carolee was the most wonderful teacher alive, Tom failed. Afterward I asked her, hey, why didn't you tell everyone that Carolee is your teacher? Oh, everyone knows Carolee. How could everyone not? Feeling someone else's pain. Tom leads to a next step. People can have different feelings than me, including pained ones. 12. This realization is not sufficient for empathy. After all, sociopaths who pathologically lack empathy use superb Tom to stay 3 manipulative, remorseless steps ahead of everyone. Nor is this realization strictly necessary for empathy, as kids too young for Tom show rudiments of feeling someone else's pain. A toddler will try to comfort someone feigning crying, offering them her pacifier, and the empathy is rudimentary in that the toddler can't imagine someone being comforted by different things than she is. Yes, very rudimentary. Maybe the toddler feels profound empathy. Or maybe she's just distressed by the crying and is self-interestedly trying to quiet the adult. The childhood capacity for empathy progresses from feeling someone's pain because you are them, to feeling for the other person, to feeling as them. The neurobiology of kid empathy makes sense. As introduced in Chapter 2, in adults the anterior cingulate cortex activates when they see someone hurt. Ditto for the amygdala and insula, especially in instances of intentional harm, there is anger and disgust. PFC regions including the emotional, VMPFC are on board. Observing physical pain, e.g., a finger being poked with a needle, produces a concrete, vicarious pattern. There is activation of the periaqueductal gray, PAG region central to your own pain perception, in parts of the sensory cortex receiving sensation from your own fingers, and in motor neurons that command your own fingers to move you clench your fingers. Work by Jean Desity of the University of Chicago shows that when 7-year-olds watch someone in pain, activation is greatest in the more concrete regions, the PAG and the sensory and motor cortices, with PAG activity coupled to the minimal VMPFC activation there is In older kids, the VMPFC is coupled to increasingly activated limbic structures. 13. And by adolescence, the stronger VMPFC activation is coupled to TOM regions. What's happening? Empathy is shifting from the concrete world of her finger must hurt, I'm suddenly conscious of my own finger, to Tomish focusing on the pokies, emotions and experience. Young kids' empathy doesn't distinguish between intentional and unintentional harm or between harm to a person and to an object. Those distinctions emerge with age, around the time when the PAG part of empathic responses lessens and there is more engagement of the VMPFC and TOM regions. Moreover, intentional harm now activates the amygdala and insula, anger and disgust at the perpetrator. This is also when kids first distinguish between self and other inflicted pain. More sophistication. By around age 7, kids are expressing their empathy. By ages 10 through 12, empathy is more generalized and abstracted, empathy for poor people, rather than one individual. Downside, this is also when kids first negatively stereotype categories of people. There are also hints of a sense of justice. Preschoolers tend to be egalitarians, e.g., it's better that the friend gets a cookie when she does. But before we get carried away with the generosity of youth, there is already in-group bias. If the other child is a stranger, there is less egalitarianism. 14. There is also a growing tendency of kids to respond to an injustice when someone has been treated unfairly. 15. But once again, before getting carried away with things, it comes with a bias. By ages 4 through 6, kids in cultures from around the world respond negatively when they are the ones being shortchanged. It isn't until ages 8 through 10 that kids respond negatively to someone else being treated unfairly. Moreover, there is considerable cross-cultural variability as to whether that later stage even emerges. The sense of justice in young kids is a very self-interested one. Soon after kids start responding negatively to someone else being treated unjustly, they begin attempting to rectify previous inequalities. He should get more now because he got less before. 16. By preadolescence, egalitarianism gives way to acceptance of inequality because of merit, or effort, or for a greater good. She should play more than him. She's better worked harder is more important to the team. Some kids even manage self-sacrifice for the greater good. She should play more than me, she's better. By adolescence, boys tend to accept inequality more than girls do on utilitarian grounds. And both sexes are acquiescing to inequality as social convention. Nothing can be done, that's the way it is. Moral development. With Tom, perspective-taking, nuanced empathy, and a sense of justice in place, a child can start wrestling with telling right from wrong. Piaget emphasized how much kids' play is about working out rules of appropriate behavior, rules that can differ from those of adults, and how this involves stages of increasing complexity. This inspired a younger psychologist to investigate the topic more rigorously, with enormously influential consequences. In the 1950s, Lawrence Kohlberg, then a graduate student at the University of Chicago and later, a professor at Harvard, began formulating his monumental stages of moral development. 17. Kids would be presented with moral conundrums. For example, the only dose of the only drug that will save a poor woman from dying is prohibitively expensive. Should she steal it? Why? Kohlberg concluded that moral judgment is a cognitive process, built around increasingly complex reasoning as kids mature. He proposed his famed 3 stages of moral development, each with 2 subparts. You've been told not to eat the tempting cookie in front of you. Should you eat it? Here are the painfully simplified stages of reasoning that go into the decision. Level 1, should I eat the cookie? Pre-conventional reasoning. Stage 1. It depends. How likely am I to get punished? Being punished is unpleasant. Aggression typically peaks around ages 2 through 4, after which kids are reined in by adults. Punishment: go sit in the corner, and peers, i.e., being ostracized. Stage 2. It depends. If I refrain, will I get rewarded? Being rewarded is nice. Both stages are ego-oriented, obedience and self-interest. What's in it for me? Kohlberg found that children are typically at this level up to around ages 8 through 10. Concern arises when aggression, particularly if callous and remorseless, doesn't wane around these ages. This predicts an increased risk of adult sociopathy, aka antisocial personality. Crucially, the behavior of future sociopaths seems impervious to negative feedback. As noted, high pain thresholds in sociopaths help explain their lack of empathy. It's hard to feel someone else's pain when you can't feel your own. It also helps explain the imperviousness to negative feedback. Why change your behavior if punishment doesn't register? It is also around this stage that kids first reconcile after conflicts and derive comfort from reconciliation, e.g., decreasing glucocorticoid secretion and anxiety. Those benefits certainly suggest self-interest motivating reconciliation. This is shown in another, realpolitik way. Kids reconcile more readily when the relationship matters to them. Level 2, should I eat the cookie? Conventional reasoning. Stage 3. It depends. Who will be deprived if I do? Do I like them? What would other people do? What will people think of me for eating the cookie? It's nice to think of others. It's good to be well regarded. Stage 4. It depends. What's the law? Are laws sacrosanct? What if everyone broke this law? It's nice to have order. This is the judge who, considering predatory but legal lending practices by a bank, thinks, I feel sorry for these victims. But I'm here to decide whether the bank broke a law. And it didn't. Conventional moral reasoning is relational, about your interactions with others and their consequences. Most adolescents and adults are at this level. Level 3, should I eat the cookie? Post-conventional reasoning. Stage 5, it depends. What circumstances placed the cookie there? Who decided that I shouldn't take it? Would I save a life by taking the cookie? It's nice when clear rules are applied flexibly. Now the judge would think, yes, the bank's actions were legal, but ultimately laws exist to protect the weak from the mighty, so signed contract or otherwise, that bank must be stopped. Stage 6, it depends. Is my moral stance regarding this more vital than some law, a stance for which I'd pay the ultimate price if need be? It's nice to know there are things for which I'd repeatedly sing, we will not be moved. This level is egoistic in that rules and their application come from within and reflect conscience, where a transgression exacts the ultimate cost, having to live with yourself afterward. It recognizes that being good and being law-abiding aren't synonymous. As Woody Guthrie wrote in Pretty Boy Floyd, I love a good man outside the law, just as much as I hate a bad man inside the law. Stage 6 is also egotistical, implicitly built on self-righteousness that trumps conventional petty bourgeois rule-makers and bean counters, the man, those sheep who just follow, etc. To quote Emerson, as is often done, when considering the post-conventional stage, every heroic act measures itself by its contempt of some external good. Stage 6 reasoning can inspire. But it can also be insufferable, premised on being good and being law-abiding as opposites. To live outside the law, you must be honest, wrote Bob Dylan. Kohlbergians found hardly anyone consistently at stage 5 or stage 6. Kohlberg basically invented the scientific study of moral development in children. His stage model is so canonical that people in the business diss someone by suggesting they're stuck in the primordial soup of a primitive Kohlberg stage. As we'll see in Chapter 12, there is even evidence that conservatives and liberals reason at different Kohlberg stages. Naturally, Kohlberg's work has problems. The usual, don't take any stage model too seriously. There are exceptions. Migurational transitions are not clean-cut, and someone's stage can be context-dependent. The problem of tunnel vision and wrong emphases. Kohlberg initially studied the usual on representative humans, namely Americans. And as we will see in later chapters, moral judgments differ cross-culturally. Moreover, subjects were male, something challenged in the 1980s by Carol Gilligan of NYU. The two agreed on the general sequence of stages. However, Gilligan and others showed that in making moral judgments, girls and women generally value care over justice, in contrast to boys and men. As a result, females tilt toward conventional thinking and its emphasis on relationships, while males tilt toward post-conventional abstractions. 18. The cognitive emphasis, are moral judgments more the outcome of reasoning, or of intuition and emotion? Kohlbergians favor the former. But as will be seen in Chapter 13, plenty of organisms with limited cognitive skills, including young kids and non-human primates, display rudimentary senses of fairness and justice. Such findings anchor social intuitionist views of moral decision-making associated with psychologists Martin Hoffman and Jonathan Haidt, both of NYU. 19. Naturally, the question becomes how moral reasoning and moral intuitionism interact. As we'll see, rather than being solely about emotion, moral intuition is a different style of cognition from conscious reasoning, and b, conversely, moral reasoning is often flagrantly illogical. Stay tuned. The lack of predictability. Does any of this actually predict who does the harder thing when it's the right thing to do? Are gold medalists at Colbergian reasoning the ones willing to pay the price for whistleblowing, subduing the shooter, sheltering refugees? Heck, forget the heroics, are they even more likely to be honest in dinky psych experiments? In other words, does moral reasoning predict moral action? Rarely, as we will see in Chapter 13, moral heroism rarely arises from super-duper frontal cortical willpower. Instead, it happens when the right thing isn't the harder thing. Marshmallows. The frontal cortex and its increasing connectivity with the rest of the brain anchors the neurobiology of kids' growing sophistication, most importantly in their capacity to regulate emotions and behavior. The most iconic demonstration of this revolves around an unlikely object, the marshmallow. 20. In the 1960s, Stanford psychologist Walter Mischel developed the marshmallow test to study gratification postponement. A child is presented with a marshmallow. The experimenter says, "I'm going out of the room for a while. You can eat the marshmallow after I leave. But if you wait and don't eat it until I get back, I'll give you another marshmallow," and leaves. And the child, observed through a two-way mirror, begins the lonely challenge of holding out for 15 minutes until the researcher returns. Studying hundreds of 3 to 6-year-olds, Mischel saw enormous variability. A few ate the marshmallow before the experimenter left the room. About a third lasted the 15 minutes. The rest were scattered in between, averaging a delay of 11 minutes. Kids' strategies for resisting the marshmallow's siren call differed, as can be seen on contemporary versions of the test on YouTube. Some kids cover their eyes, hide the marshmallow, sing to distract themselves. Others grimace, sit on their hands. Others sniff the marshmallow, pinch off an infinitely tiny piece to eat, hold it reverentially, kiss it, pet it. Various factors modulated kids' fortitude, shown in later studies described in Mischel's book where, for some reason, it was pretzels instead of marshmallows. Trusting the system mattered. If experimenters had previously betrayed on promises, kids wouldn't wait as long. Prompting kids to think about how crunchy and yummy pretzels are, what Mischel calls hot ideation, nuke self-discipline, prompts to think about a cold ideation, e.g., the shape of pretzels, or an alternative hot ideation, e.g., ice cream, bolstered resistance. As expected, older kids hold out longer, using more effective strategies. Younger kids describe strategies like, "I kept thinking about how good that second marshmallow would taste." The problem, of course, is that this strategy is about 2 synapses away from thinking about the marshmallow in front of you. In contrast, older kids use strategies of distraction, thinking about toys, pets, their birthday. This progresses to reappraisal strategies. This isn't about marshmallows. This is about the kind of person I am. To Mischel, maturation of willpower is more about distraction and reappraisal strategies than about stoicism. So kids improve at delayed gratification. Mischel's next step made his studies iconic. He tracked the kids afterward, seeing if marshmallow wait time predicted anything about their adulthoods. Did it ever? 5-year-old chance at marshmallow patients averaged higher SAT scores in high school, compared with those who couldn't wait, with more social success and resilience and less aggressive and oppositional behavior. 40 years post-marshmallow, they excelled at frontal function, had more PFC activation during a frontal task, and had lower BMIS. 21. A gazillion-dollar brain scanner doesn't hold more predictive power than one marshmallow. Every anxious middle-class parent obsesses over these findings, has made marshmallows fetish items. W. Consequences. You've now gotten a sense of various domains of behavioral development. Time to frame things with this book's central question. Our adult has carried out that wonderful or crummy, or ambiguous behavior. What childhood events contributed to that occurring? A first challenge is to truly incorporate biology into our thinking. A child suffers malnutrition and, as an adult, has poor cognitive skills. That's easy to frame biologically. Malnutrition impairs brain development. Alternatively, a child is raised by cold, inexpressive parents and, as an adult, feels unlovable. It's harder to link those two biologically, to resist thinking that somehow, this is a less biological phenomenon than the malnutrition cognition link. There may be less known about the biological changes, explaining the link between the cold parents and the adult with poor self-esteem than about the malnutrition cognition one. It may be less convenient to articulate the former biologically than the latter. It may be harder to apply a proximal biological therapy for the former than for the latter, e.g., an imaginary neural growth factor drug that improves self-esteem versus cognition. But biology mediates both links. A cloud may be less tangible than a brick, but it's constructed with the same rules about how atoms interact. How does biology link childhood with the behaviors of adulthood? Chapter 5's Neural Plasticity writ large and early. The developing brain epitomizes neural plasticity, and every hiccup of experience has an effect, albeit usually a minuscule one, on that brain. We now examine ways in which different types of childhoods produce different sorts of adults. Let's start at the very beginning, the importance of mothers. Nothing like a section heading stating the obvious. Everybody needs a mother. Even rodents separate rat pups from mom a few hours daily, and as adults, they have elevated glucocorticoid levels and poor cognitive skills, are anxious, and if male are more aggressive. 22. Mothers are crucial. Except that well into the 20th century, most experts didn't think so. The West developed child-rearing techniques where, when compared with traditional cultures, children had less physical contact with their mothers, slept alone at earlier ages, and had longer latencies to be picked up when crying. Around 1900, the leading expert, Luther Holt of Columbia University, warned against the vicious practice of picking up a crying child or handling her too often. This was the world of children of the wealthy, raised by nannies and presented to their parents before bedtime to be briefly seen, but not heard. This period brought one of history's strangest one-night stands, namely when the Freudians and the behaviorists hooked up to explain why infants become attached to their mothers. To behaviorists, obviously, it's because mothers reinforce them, providing calories when they're hungry. For Freudians, also obviously, infants lack the ego development to form a relationship with anyone other than mom's breasts. When combined with children should be seen but not heardism, this suggested that once you've addressed a child's need for nutrition, proper temperature, plus other odds and ends, they're set to go. Affection, warmth, physical contact? Superfluous. Such thinking produced at least one disaster. When a child was hospitalized for a stretch, dogma was that the mother was unnecessary, she just added emotional tumult, and everything essential was supplied by the staff. Typically, mothers could visit their children once a week for a few minutes. And when kids were hospitalized for extended periods, they wasted away with hospitalism, dying in droves from nonspecific infections and gastrointestinal maladies unrelated to their original illness. 23. This was an era when the germ theory had mutated into the belief that hospitalized children do best when untouched in antiseptic isolation. Remarkably, hospitalism soared in hospitals with newfangled incubators, adapted from poultry farming. The safest hospitals were poor ones that relied on the primitive act of humans actually touching and interacting with infants. In the 1950s, the British psychiatrist John Bowlby challenged the view of infants as simple organisms with few emotional needs. His attachment theory birthed our modern view of the mother-infant bond. 24. In his trilogy, Attachment and Loss, Bowlby summarized the no-brainer answers we'd give today to the question, what do children need from their mothers? Love, warmth, affection, responsiveness, stimulation, consistency, reliability. What is produced in their absence? Anxious, depressed, and/or poorly attached adults. Bowlby inspired one of the most iconic experiments in psychology's history, by Harry Harlow of the University of Wisconsin. It destroyed Freudian and behaviorist dogma about mother-infant bonding. 25. Harlow would raise an infant rhesus monkey without a mother, but with two surrogates instead. Both were made of a chicken wire tube approximating a torso, with a monkey-ish plastic head on top. One surrogate had a bottle of milk coming from its torso. The other had terry cloth wrapped around the torso. In other words, one gave calories, the other a poignant approximation of a mother monkey's fur. Freud and B.F. Skinner would have wrestled over access to chicken wire mom. But infant monkeys chose the terry cloth mom. Man cannot live by milk alone. Love is an emotion that does not need to be bottle or spoon-fed, wrote Harlow. Evidence for the most basic need provided by a mother comes from a controversial quarter Starting in the 1990s, crime rates plummeted across the United States. Why? For liberals, the answer was the thriving economy. For conservatives, it was the larger budgets for policing, expanded prisons, and three-strike sentencing laws. Meanwhile, partial explanation was provided by legal scholar John Donahue of Stanford and economist Stephen Levitt of the University of Chicago. It was the legalization of abortions. The author's state-by-state analysis of the liberalization of abortion laws and the demographics of the crime drop showed that when abortions become readily available in an area, rates of crime by young adults decline about 20 years later. Surprise, this was highly controversial, but it makes perfect depressing sense to me. What majorly predicts a life of crime? Being born to a mother who, if she could, would have chosen that you not be. What's the most basic thing provided by a mother? Knowing that she is happy that you exist. 26. Harlow also helped demonstrate a cornerstone of this book, namely what mothers and later peers provide as children grow. To do so, he performed some of the most inflammatory research in psychology's history. This involved raising infant monkeys in isolation, absent mother or peers. They spent the first months, even years, of their lives without contact with another living being, before being placed in a social group. Predictably, they'd be wrecks. Some would sit alone, clutching themselves, rocking autistically. Others would be markedly inappropriate in their hierarchical or sexual behaviors. There was something interesting. It wasn't that these ex-isolates did behaviors wrong. They didn't aggressively display, like an ostrich, make the sexually solicitive gestures of a gecko. Behaviors were normal but occurred at the wrong time and place, say, giving subordination gestures to pipsqueaks half their size, threatening alphas they should cower before. Mothers and peers don't teach the motoric features of fixed action patterns, those are hardwired. They teach when, where, and to whom, the appropriate context for those behaviors. They give the first lessons about when touching someone's arm or pulling a trigger can be among the best or worst of our behaviors. I saw a striking example of this among the baboons that I study in Kenya, when both a high-ranking and a low-ranking female gave birth to daughters the same week. The former's kid hit every developmental landmark earlier than the other, the playing field already on level. When the infants were a few weeks old, they nearly had their first interaction. Daughter of subordinate mom spotted daughter of dominant one, toddled over to say hello. And as she got near, her low-ranking mother grabbed her by the tail and pulled her back. This was her first lesson about her place in that world. You see her? She's much higher ranking than you, so you don't just go and hang with her. If she's around, you sit still and avoid eye contact and hope she doesn't take whatever you're eating. Amazingly, in 20 years those two infants would be old ladies, sitting in the savannah, still displaying the rank asymmetries they learned that morning. H. Any kind of mother in a storm. Harlow provided another important lesson, thanks to another study painful to contemplate. Infant monkeys were raised with chicken wire surrogates with air jets in the middle of their torsos. When an infant clung, she'd receive an aversive blast of air. What would a behaviorist predict that the monkey would do when faced with such punishment? Flee. But, as in the world of abused children and battered partners, infants held harder. Why do we often become attached to a source of negative reinforcement, seek solace when distressed from the cause of that distress? Why do we ever love the wrong person, get abused and return for more? Psychological insights abound. Because of poor self-esteem, believing you'll never do better. Or a codependent conviction that it's your calling to change the person. Maybe you identify with your oppressor, or have decided it's your fault and the abuser is justified, so they seem less irrational and terrifying. These are valid and can have huge explanatory and therapeutic power. But work by Regina Sullivan of NYU demonstrates bits of this phenomenon miles from the human psyche. Sullivan would condition rat pups to associate a neutral odor with a shock. 27. If a pup that had been conditioned at 10 days of age, or older pups, was exposed to that odor, logical things happened, amygdala activation, glucocorticoid secretion, and avoidance of the odor. But do the same to a younger pup and none of that would occur. Remarkably, The pup would be attracted to the odor. Why? There is an interesting wrinkle related to stress in newborns. Rodent fetuses are perfectly capable of secreting glucocorticoids. But within hours of birth, the adrenal glands atrophy dramatically, becoming barely able to secrete glucocorticoids. This stress hyporesponsive period SHRP wanes over the coming weeks. 28. What is the SHRP about? Glucocorticoids have so many adverse effects on brain development. Stay tuned that the SHRP represents a gamble. I won't secrete glucocorticoids in response to stress, so that I develop optimally. If something stressful happens, mom will handle it for me. Accordingly, deprive infant rats of their mothers, and within hours their adrenals expand and regain the ability to secrete plenty of glucocorticoids. During the SHRP, infants seem to use a further rule: if mom is around, and I thus don't secrete glucocorticoids, I should get attached to any strong stimulus. It couldn't be bad for me, mom wouldn't allow that. As evidence, inject glucocorticoids into the amygdala of young pups during the conditioning, and the amygdala would activate and the pups would develop an aversion to the odor. Conversely, block glucocorticoid secretion in older pups during conditioning, and they'd become attracted to the odor. Or condition them with their mother present, and they wouldn't secrete glucocorticoids and would develop an attraction. In other words, in young rats even aversive things are reinforcing in mom's presence, even if mom is the source of the aversive stimuli. As Sullivan and colleagues wrote, attachment by such an infant to the caretaker has evolved to ensure that the infant forms a bond to that caregiver regardless of the quality of care received. Any kind of mother in a storm. If this applies to humans, it helps explain why individuals abused as kids are as adults prone toward relationships in which they are abused by their partner. 29. But what about the flip side? Why is it that about 33% of adults who were abused as children become abusers themselves? Again, useful psychological insights abound, built around identification with the abuser and rationalizing away the terror. I love my kids, but I smack them around when they need it. My father did that to me, so he could have loved me too. But once again, something biologically deeper also occurs. Infant monkeys abused by their mothers are more likely to become abusive mothers. 30. Different routes to the same place. Anticipated that, with mothers now covered, we'd next examine the adult consequences of, say, paternal deprivation, or childhood poverty, or exposure to violence or natural disasters. And there'd be the same question: what specific biological changes did each cause in children that increased the odds of specific adult behaviors? But this plan didn't work. The similarities of effects of these very traumas are greater than the differences. Sure, there are specific links, e.g., childhood exposure to domestic violence makes adult antisocial violence more likely than does childhood exposure to hurricanes. But they all converge sufficiently that I will group them together, as is done in the field, as examples of childhood adversity. Basically, childhood adversity increases the odds of an adult having a. Depression, anxiety, and/or substance abuse, b. Impaired cognitive capabilities, particularly related to frontocortical function, c. Impaired impulse control and emotion regulation, d. Antisocial behavior, including violence, and e. Relationships that replicate the adversities of childhood, e.g., staying with an abusive partner. 31. And despite that, some individuals endure miserable childhoods just fine. More on this to come. We'll now examine the biological links between childhood adversity and increased risk of these adult outcomes. A. The biological profile. These forms of adversity are obviously stressful and cause abnormalities in stress physiology. Across numerous species, major early life stressors produce both kids and adults with elevated levels of glucocorticoids, along with CRH and ACTH, the hypothalamic and pituitary hormones that regulate glucocorticoid release, and hyperactivity of the sympathetic nervous system. 32. Basal glucocorticoid levels are elevated, the stress response is always somewhat activated, and there is delayed recovery back to baseline after a stressor. Michael Meany of McGill University has shown how early life stress permanently blunts the ability of the brain to rein in glucocorticoid secretion. As covered in Chapter 4, marinating the brain in excess glucocorticoids, particularly during development, adversely affects cognition, impulse control, empathy, and so on. 33. There is impaired hippocampal dependent learning in adulthood. For example, abused children who develop PTSD have decreased volume of the hippocampus in adulthood. Stanford psychiatrist Victor Karian has shown decreased hippocampal growth within months of the abuse. As a likely cause, glucocorticoids decrease hippocampal production of the growth factor BDNF, brain-derived neurotrophic factor. So childhood adversity impairs learning and memory. Crucially, it also impairs maturation and function of the frontal cortex. Again, glucocorticoids, by inhibiting BDNF, are likely culprits. The connection between childhood adversity and frontocortical maturation pertains to childhood poverty. Work by Martha Farah of the University of Pennsylvania, Tom Boyce of UCSF, and others demonstrates something outrageous. By age 5, the lower a child's socioeconomic status on the average, the higher the basal glucocorticoid levels, and/or the more reactive the glucocorticoid stress response, b, the thinner the frontal cortex and the lower its metabolism, and c, the poorer the frontal function concerning working memory, emotion regulation, impulse control, and executive decision-making. Moreover, to achieve equivalent frontal regulation, lower SES kids must activate more frontal cortex than do higher SES kids. In addition, childhood poverty impairs maturation of the corpus callosum, a bundle of axonal fibers, connecting the two hemispheres, and integrating their function. This is so wrong. Foolishly pick a poor family to be born into, and by kindergarten, the odds of your succeeding at life's marshmallow tests are already stacked against you. 0.34. Considerable research focuses on how poverty gets under the skin. Some mechanisms are human-specific. If you're poor, you're more likely to grow up near environmental toxins. 35 In a dangerous neighborhood with more liquor stores than markets selling produce, you're less likely to attend a good school or have parents with time to read to you. Your community is likely to have poor social capital, and you, poor self-esteem. But part of the link reflects the corrosive effects of subordination in all hierarchical species. For example, having a low-ranking mother predicts elevated glucocorticoids in adulthood in baboons. 0.36. Thus, childhood adversity can atrophy and blunt the functioning of the hippocampus and frontal cortex. But it's the opposite in the amygdala. Lots of adversity and the amygdala becomes larger and hyperreactive. One consequence is increased risk of anxiety disorders when coupled with the poor frontocortical development. It explains problems with emotion and behavior regulation, especially impulse control. 0.37. Childhood adversity accelerates amygdaloid maturation in a particular way. Normally, around adolescence the frontal cortex gains the ability to inhibit the amygdala, saying, "I wouldn't do this if I were you." But after childhood adversity, the amygdala develops the ability to inhibit the frontal cortex, saying, "I'm doing this and just try to stop me." Childhood adversity also damages the dopamine system, with its role in reward, anticipation, and goal-directed behavior in two ways. First, early adversity produces an adult organism more vulnerable to drug and alcohol addiction. The pathway to this vulnerability is probably threefold: A, effects on the developing dopamine system; B, the excessive adult exposure to glucocorticoids, which increases drug craving; C, that poorly developed frontal cortex. 0.38. Childhood adversity also substantially increases the risk of adult depression. Depression's defining symptom is anhedonia, the inability to feel, anticipate, or pursue pleasure. Chronic stress depletes the mesolimbic system of dopamine, generating anhedonia. The link between childhood adversity and adult depression involves both organizational effects on the developing mesolimbic system and elevated adult glucocorticoid levels, which can deplete dopamine. 0.39. Childhood adversity increases depression risk via second-hit scenarios, lowering thresholds so that adult stressors that people typically manage instead trigger depressive episodes. This vulnerability makes sense. Depression is fundamentally a pathological sense of loss of control, explaining the classic description of depression as learned helplessness. If a child experiences severe, uncontrollable adversity, the most fortunate conclusion in adulthood is those were terrible circumstances over which I had no control. But when childhood traumas produce depression, there is cognitively distorted overgeneralization, and life will always be uncontrollably awful. Two side topics. Varied types of childhood adversity converge in producing similar adult problems. Nonetheless, two types of adversity should be considered separately. Observing violence. What happens when children observe domestic violence, warfare, a gang murder, a school massacre? For weeks afterward there is impaired concentration and impulse control. Witnessing gun violence doubles a child's likelihood of serious violence within the succeeding 2 years. And adulthood brings the usual increased risks of depression, anxiety, and aggression. Consistent with that, violent criminals are more likely than non-violent ones to have witnessed violence as kids 40. This fits our general picture of childhood adversity. A separate topic is the effects of media violence on kids. Endless studies have analyzed the effects of kids witnessing violence on TV, in movies, in the news, and in music videos, and both witnessing and participating in violent video games. A summary. Exposing children to a violent TV or film clip increases their odds of aggression soon after 0.41. Interestingly, the effect is stronger in girls, amid their having lower overall levels of aggression. Effects are stronger when kids are younger or when the violence is more realistic and/or is presented as heroic. Such exposure can make kids more accepting of aggression. In one study, watching violent music videos increased adolescent girls' acceptance of dating violence. The violence is key. Aggression isn't boosted by material that's merely exciting, arousing, or frustrating. Heavy childhood exposure to media violence predicts higher levels of aggression in young adults of both sexes. Aggression, ranging from behavior in an experimental setting to violent criminality. The effect typically remains after controlling for total media watching time, maltreatment or neglect, socioeconomic status, levels of neighborhood violence, parental education, psychiatric illness, and IQ. This is a reliable finding of large magnitude. The link between exposure to childhood media violence and increased adult aggression is stronger than the link between lead exposure and IQ, calcium intake and bone mass, or asbestos and laryngeal cancer. 2 caveats. There is no evidence that catastrophically violent individuals, e.g., mass shooters, are that way because of childhood exposure to violent media. B. Exposure does not remotely guarantee increased aggression. Instead, effects are strongest on kids already prone toward violence. For them, exposure desensitizes and normalizes their own aggression. Bullying. Being bullied is mostly another garden-variety childhood adversity, with adult consequences on par with childhood maltreatment at home. 42. There is a complication, though. As most of us observed, exploited, or experienced as kids, bullying targets aren't selected at random. Kids with the metaphorical kick me signs on their backs are more likely to have personal or family psychiatric issues and poor social and emotional intelligence. These are kids already at risk for bad adult outcomes, and adding bullying to the mix just makes the child's future even bleaker. The picture of the bullies is no surprise either, starting with their disproportionately coming from families of single moms or younger parents with poor education and employment prospects. There are generally 2 profiles of the kids themselves. The more typical is an anxious, isolated kid with poor social skills who bullies out of frustration and to achieve acceptance. Such kids typically mature out of bullying. The second profile is the confident, unempathetic, socially intelligent kid with an imperturbable sympathetic nervous system. This is the future sociopath. There is an additional striking finding. You want to see a kid who's really likely to be a mess as an adult? Find someone who both bullies and is bullied, who terrorizes the weaker at school and returns home to be terrorized by someone stronger. 43. Of the 3 categories, bully, bullied, bully/bullied, they're most likely to have prior psychiatric problems, poor school performance, and poor emotional adjustment. They're more likely than pure bullies to use weapons and inflict serious damage. As adults, they're most at risk for depression, anxiety, and suicidality. In one study, kids from these 3 categories read scenarios of bullying. 44. Bullied victims would condemn bullying and express sympathy. Bullies would condemn bullying but rationalize the scenario, e.g., this time it was the victim's fault. And bullied kids? They would say bullying is okay. No wonder they have the worst outcome. The weak deserve to be bullied, so it's fine when I bully. But that means I deserve to be bullied at home. But I don't, and that relative bullying me is awful. Maybe then I'm awful when I bully someone. But I'm not, because the weak deserve to be bullied. A Mobius strip from hell. W. A key question. Eve now examined adult consequences of childhood adversity and their biological mediators. A key question persists. Yes, Childhood abuse increases the odds of being an abusive adult. Witnessing violence raises the risk for PTSD. Loss of a parent to death means more chance of adult depression. Nevertheless, many, maybe even most victims of such adversity turn into reasonably functional adults. There is a shadow over childhood, demons lurk in corners of the mind, but overall things are okay. What explains such resilience? As we'll see, genes and fetal environment are relevant. But most important, recall the logic of collapsing different types of trauma into a single category. What counts is the sheer number of times a child is bludgeoned by life and the number of protective factors. Be sexually abused as a child, or witness violence, and your adult prognosis is better than if you had experienced both. Experience childhood poverty, and your future prospects are better if your family is stable and loving than broken and acrimonious. Pretty straightforwardly, the more categories of adversities a child suffers, the dimmer his or her chances of a happy, functional adulthood. 45. W. A sledgehammer. That happens when everything goes wrong. No mother or family, minimal peer interactions, sensory and cognitive neglect, plus some malnutrition. 46. These are the Romanian institution kids, poster children for just how nightmarish childhood can be. In the 1980s, the Romanian dictator Nicolae Ceausescu banned contraceptives and abortions and required women to bear at least 5 children. Soon institutions filled with thousands of infants and kids, abandoned by impoverished families, many intent on reclaiming their child. When finances improved, kids were warehoused in overwhelmed institutions, resulting in severe neglect and deprivation. The story broke after Ceausescu's 1989 overthrow. Many kids were adopted by Westerners, and international attention led to some improvements in the institutions. Since then, children adopted in the West, those eventually returned to their families, and those who remained institutionalized have been studied, primarily by Charles Nelson of Harvard. As adults, these kids are mostly what you'd expect. Low IQ and poor cognitive skills. Problems with forming attachments, often bordering on autistic. Anxiety and depression galore. The longer the institutionalization, the worse the prognosis. And their brains. Decreased total brain size, gray matter, white matter, frontal cortical metabolism, connectivity between regions, sizes of individual brain regions. Except for the amygdala, which is enlarged. That pretty much says it all. C. Culture, with both a big and a little c. Chapter 9 considers the effects of culture on our best and worst behaviors. We now preview that chapter, focusing on 2 facts. Childhood is when culture is inculcated, and parents mediate that process. There is huge cultural variability in how childhood is experienced, how long and often kids are nursed, how often they are in contact with parents and other adults, how often they're spoken to, how long they cry before someone responds, at what age they sleep alone. Considering cross-cultural child rearing often brings out the most invidious and neurotic in parents. Do other cultures do a better job at it? There must be the perfect combo out there, a mixture of the quacudal baby diet, the Trobriand sleeping program, and the Inari approach to watching baby Mozart videos. But there is no anthropological ideal of child rearing. Cultures, starting with parents, raise children to become adults who behave in the ways valued by that culture, a point emphasized by the anthropologist Meredith Small of Cornell University. 47. We begin with parenting style, a child's first encounter with cultural values. Interestingly, the most influential typology of parenting style, writ small, grew from thinking about cultural styles, writ large. Amid the post-World War II ruins, scholars tried to understand where Hitler, Franco, Mussolini, Tojo, and their minions came from. What are the roots of fascism? 2 particularly influential scholars were refugees from Hitler, namely Hannah Arendt with her 1951 book, The Origins of Totalitarianism, and Theodor Adorno with the 1950 book, The Authoritarian Personality, co-authored with Else Frankel Brunswick, Daniel Levinson, and Nevitt Sanford. Adorno in particular explored the personality traits of fascists, including extreme conformity, submission to and belief in authority, aggressiveness and hostility toward intellectualism and introspection, traits typically rooted in childhood. 48. This influenced the Berkeley psychologist Diana Baumrind, who in the 1960s identified three key parenting styles in work since replicated and extended to various cultures. 49. First is authoritative parenting. Rules and expectations are clear, consistent, and explicable because I said so is anathema with room for flexibility. Praise and forgiveness trump punishment. Parents welcome children's input, developing children's potential and autonomy is paramount. By the standards of the educated neurotics who would read, let alone write. This produces a good adult outcome, happy, emotionally and socially mature and fulfilled, independent and self-reliant. Next is authoritarian parenting. Rules and demands are numerous, arbitrary, and rigid and need no justification. Behavior is mostly shaped by punishment. Children's emotional needs are low priorities. Parental motivation is often that, it's a tough, unforgiving world and kids better be prepared. Authoritarian parenting tends to produce adults who may be narrowly successful, obedient, conformist, often with an undercurrent of resentment that can explode, and not particularly happy. Moreover, social skills are often poor because, instead of learning by experience, they grew up following orders. And then there is permissive parenting, the aberration that supposedly let boomers invent the 1960s. There are few demands or expectations, rules are rarely enforced, and children set the agenda. Adult outcome, self-indulgent individuals with poor impulse control, low frustration tolerance, plus poor social skills, thanks to living consequence-free childhoods. Baumrind's trio was expanded by Stanford psychologists Eleanor, Maccabi and John Martin to include neglectful parenting. 50. This addition produces a 2x2 matrix. Parenting is authoritative, high demand, high responsiveness, authoritarian, high demand, low responsiveness, permissive, low demand, high responsiveness, or neglectful, low demand, low responsiveness. Importantly, each style usually produces adults with that same approach, with different cultures valuing different styles. Then comes the next way, cultural values are transmitted to kids, namely by peers. This was emphasized in Judith Rich Harris's The Nurture Assumption. Harris, a psychologist without an academic affiliation or doctorate, took the field by storm, arguing that the importance of parenting in shaping a child's adult personality is exaggerated. 51. Instead, once kids pass a surprisingly young age, peers are most influential. Elements of her argument included, parental influence is often actually mediated via peers. For example, being raised by a single mother increases the risk of adult antisocial behavior, but not because of the parenting. Instead, because of typically lower income, kids more likely live in a neighborhood with tough peers. B. Peers have impact on linguistic development, e.g., children acquire the accent of their peers, not their parents. C. Other young primates are mostly socialized by peers, not mothers. The book was controversial, partially because the theme begged to be distorted. Psychologist proves that parents don't matter, drawing criticism and acclaim as the dust has settled. Current opinion tends to be that peer influences are underappreciated, but parents still are plenty important, including by influencing what peer groups their kids experience. Why are peers so important? Peer interactions teach social competence, context-dependent behavior, when to be friend or foe, where you fit in hierarchies. Young organisms utilize the greatest teaching tool ever to acquire such information, play. 52. What is social play in the young? Writ large, it's an array of behaviors that train individuals in social competence. Writ medium, it's fragments of the real thing, bits and pieces of fixed action patterns, a chance to safely try out roles and improve motor skills. Writ small and endocrine, it's a demonstration that moderate and transient stress stimulation is great. Writ small and neurobiological, it's a tool for deciding which excess synapses to prune. The historian Johann Huizinga characterized humans as homo ludens, men the player with our structured, rule-bound play, i.e., games. Nevertheless, play is universal among socially complex species, ubiquitous among the young, and peaking at puberty, and all play involves similar behaviors after some ethological translating, e.g., a dominant dog signals the benevolence needed to initiate play by crouching, diminishing herself, translated into baboon, a dominant kid presents her rear to someone lower ranking. Play is vital. In order to play, animals forego foraging, expend calories, make themselves distracted and conspicuous to predators. Young organisms squander energy on play during famines. A child deprived of or disinterested in play rarely has a socially fulfilling, adult life. Most of all, Play is intrinsically pleasurable, why else perform a smidgen of a behavioral sequence in an irrelevant setting? Dopaminergic pathways activate during play. Juvenile rats, when playing, emit the same vocalizations as when rewarded with food. Dogs spend half their calories wagging their tails to pheromonally announce their presence and availability for play. As emphasized by the psychiatrist Stuart Brown, founder of the National Institute for Play, the opposite of play is not work, it's depression. A challenge is to understand how the brain codes for the reinforcing properties of the variety of play. After all, play encompasses everything from mathematicians besting each other with hilarious calculus jokes to kids besting each other by making hilarious fart sounds with their armpits. One significant type of play involves fragments of aggression, what Harlow called rough and tumble play, kids wrestling, adolescent impalas butting heads, puppies play biting each other. 53. Males typically do it more than females, and as we'll see soon, it's boosted by prenatal testosterone. Is rough and tumble play practice for life's looming status tournament, or are you already in the arena? A mixture of both. Expanding beyond peers, neighborhoods readily communicate culture to kids. Is there garbage everywhere? Are houses decrepit? What's ubiquitous? Bars, churches, libraries, or gun shops? Are there many parks, and are they safe to enter? Do billboards, ads, and bumper stickers sell religious or material paradises, celebrate acts of martyrdom or kindness and inclusiveness? And then we get to culture at the level of tribes, nations, and states. Here, briefly, are some of the broadest cultural differences in child-rearing practices. Collectivist versus individualist cultures. As will be seen in Chapter 9, this is the most studied cultural contrast, typically comparing collectivist East Asian cultures with uber-individualist America. Collectivist cultures emphasize interdependence, harmony, fitting in, the needs and responsibilities of the group. In contrast, individualist cultures value independence, competition, the needs and rights of the individual. On average, mothers in individualist cultures, when compared with those in collectivist ones, speak louder, play music louder, have more animated expressions. 54. They view themselves as teachers rather than protectors, abhor a bored child, value high-energy affect. Their games emphasize individual competition, urge hobbies involving doing rather than observing. Kids are trained in verbal assertiveness, to be autonomous and influential. Show a cartoon of a school of fish with one out front, and she'll describe it to her child as the leader. Mothers in collectivist cultures, in contrast, spend more time than individualist mothers soothing their child, maintaining contact and facilitating contact with other adults. They value low arousal affect and sleep with their child to a later age. Games are about cooperation and fitting in. If playing with her child with, say, a toy car, the point is not exploring what a car does, i.e., being automobile, but the process of sharing. Thank you for giving me your car, now I'll give it back to you. Kids are trained to get along, think of others, accept and adapt, rather than change situations. Morality and conformity are nearly synonymous. Show the cartoon of the school of fish and the fish out front must have done something wrong, because no one will play with him. Logically, kids in individualist cultures acquire Tom later than collectivist culture. Kids and activate pertinent circuits more to achieve the same degree of competence. For a collectivist child, social competence is all about taking someone else's perspective. 55. Interestingly, kids in collectivist Japan play more violent video games than do American kids, yet are less aggressive. Moreover, exposing Japanese kids to media violence boosts aggression less than in American kids. 56. Why the difference? 3 possible contributing factors. A. American kids play alone more often, a lone wolf breeding ground. B. Japanese kids rarely have a computer or TV in their bedroom, so they play near their parents. C. Japanese video game violence is more likely to have prosocial, collectivist themes. More in Chapter 9 on collectivist versus individualist cultures. Cultures of honor. These cultures emphasize rules of civility, courtesy, and hospitality. Taking retribution is expected for affronts to the honor of oneself, family, or clan. Failing to do so is shameful. These are cultures filled with vendettas, revenge, and honor killings. Cheeks aren't turned. A classic culture of honor is the American South, but as we'll see in Chapter 9, such cultures occur worldwide and with certain ecological correlates. A particularly lethal combo is when a culture of victimization, we were wronged last week, last decade, last millennium, is coupled with a culture of honors, ethos of retribution. Parenting in cultures of honor tends to be authoritarian. 57. Kids are aggressive, particularly following honor violations, and staunchly endorse aggressive responses to scenarios of honor violation. Class differences. As noted, an infant baboon learns her place in the hierarchy from her mother. A human child's lessons about status are more complex. There is implicit quing, subtle language cues, the cognitive and emotional weight of remembering the past when your grandparents emigrated here, they couldn't even. And hoping about the future, when you grow up, you're going to. Baboon mothers teach their young appropriate behavioral context. Human parents teach their young what to bother dreaming about. Class differences in parenting in Western countries resemble parenting differences between Western countries and those in the developing world. In the West, a parent teaches and facilitates her child exploring the world. In the toughest corners of the developing world, little more is expected than the awesome task of keeping your child alive and buffered from the menacing world. In Western cultures, class differences in parenting sort by Baumrein's typologies. In higher SES strata, parenting tends to be authoritative or permissive. In contrast, parenting in society's lower SES rungs is typically authoritarian, reflecting two themes. One concerns protecting. When are higher SES parents authoritarian? When there is danger. Sweetie, I love that you question things, but if you run into the street and I scream stop, you stop. A lower SES childhood is rife with threat. The other theme is preparing the child for the tough world out there. For the poor, adulthood consists of the socially dominant, treating them in an authoritarian manner. Class differences in parenting were explored in a classic study by the anthropologist Audrey Cousereau of St. Michael's College, who did fieldwork observing parents in three tribes: wealthy families on Manhattan's Upper East Side, a stable, blue-collar community, and a poor, crime-ridden one, the last two both in Queens. 58. The differences were fascinating. Parenting in the poor neighborhood involved hard defensive individualism. The neighborhood was rife with addiction, homelessness, incarceration, death, and parents' aim was to shelter their child from the literal and metaphorical street. Their speech was full of metaphors about not losing what was achieved, standing your ground, keeping up your pride, not letting others get under your skin. Parenting was authoritarian, toughening the goal. For example, parents teased kids far more than in the other neighborhoods. In contrast, working-class parenting involved hard offensive individualism. Parents had some socioeconomic momentum, and kids were meant to maintain that precarious trajectory. Parents' speech about their hopes for their kids contained images of movement, progress, and athletics, getting ahead, testing the waters, going for the gold. With hard work and the impetus of generations of expectations, your child might pioneer landfall in the middle class. Parenting in both neighborhoods emphasized respect for authority, particularly within the family. Moreover, kids were fungible members of a category, rather than individualized "you kids get over here." Then there was the soft individualism of upper middle class. Parenting children's eventual success, by conventional standards, was a given, as were expectations of physical health. Far more vulnerable was a child's psychological health. When children could become anything, parents' responsibility was to facilitate their epic journey toward an individuated fulfillment. Moreover, the image of fulfillment was often post-conventional. I hope my child will never work an unsatisfying job, just for money. This, after all, is a tribe giddied by tales of the shark in line to become CEO chucking it to learn carpentry or oboe. Parent speech brimmed with metaphors of potential being fulfilled, flowering, blooming, growing, blossoming. Parenting was authoritative or permissive, riddled with ambivalence about parent-child power differentials. Rather than "you kids, clean up this mess," there'd be the individuated, justifying request: "Caitlin, Zach, Dakota, could you clean things up a bit please? Malala is coming for dinner." We've now seen how childhood events, from the first mother-infant interaction to the effects of culture, have persistent influences, and how biology mediates such influences. When combined with the preceding chapters, we have finished our tour of environmental effects on behavior, from the second before a behavior occurs to a second after birth. In effect, we've done environment. Time for next chapter's genes. But this ignores something crucial: environment doesn't begin at birth. 9 long months. The cat in the hat in the womb. The existence of prenatal environmental influences caught the public's imagination with some charming studies demonstrating that near-term fetuses hear what's going on outside the womb, taste amniotic fluid, and remember and prefer those stimuli after birth. This was shown experimentally. Inject lemon-flavored saline into a pregnant rat's amniotic fluid, and her pups are born preferring that flavor. Moreover, some spices consumed by pregnant women get into amniotic fluid. Thus we may be born preferring foods our mothers ate during pregnancy. Pretty unorthodox cultural transmission. 59. Prenatal effects can also be auditory, as shown by inspired research by Anthony DeCosper of the University of North Carolina. 60. A pregnant woman's voice is audible in the womb, and newborns recognize and prefer the sound of their mother's voice. DeCosper used ethology's playbook to show this. A newborn can learn to suck a pacifier in 2 different patterns of long and short sucks. Generate one pattern, and you hear mom's voice, the other, another woman's voice. Newborns want mom's voice. Elements of language are also learned in utero. The contours of a newborn's cry are similar to the contours of speech in the mother's language. The cognitive capacities of near-term fetuses are even more remarkable. For example, fetuses can distinguish between 2 pairs of nonsense syllables, biba versus baby. How do you know? Get this, mom says biba, biba, biba repeatedly while fetal heart rate is monitored. Boring, or perhaps lulling, thinks the fetus, and heart rate slows. Then mom switches to baby. If the fetus doesn't distinguish between the two, heart rate deceleration continues. But if the difference is noted, whoa, what happened? Heart rate increases. Which is what DeCosper reported. 61. DeCosper and colleague Melanie Spence then showed, using the pacifier sucking pattern detection system, that newborns typically don't distinguish between the sounds of their mother, reading a passage from The Cat in the Hat and from the rhythmically similar The King, the Mice, and the Cheese. 62. But newborns whose mothers had read The Cat in the Hat out loud for hours during the last trimester preferred Dr. Seuss. Wow. Despite the charm of these findings, this book's concerns aren't rooted in such prenatal learning. Few infants are born with a preference for passages from, say, Mein Kampf. However, other prenatal environmental effects are quite consequential. W. Boy and girl brains, whatever that might mean. Start with a simple version of what environment means for a fetal brain: the nutrients, immune messengers, and, most important, hormones carried to the brain in the fetal circulation. Once the pertinent glands have developed in a fetus, they are perfectly capable of secreting their characteristic hormones. This is particularly consequential. When hormones first made their entrance in Chapter 4, our discussion concerned their activational effects that lasted on the order of hours to days. In contrast, hormones in the fetus have organizational effects on the brain, causing lifelong changes in structure and function. Around 8 weeks post-conception, human fetal gonads start secreting their steroid hormones, testosterone in males, estrogen and progesterone in females. Crucially, testosterone plus anti-Mullerian hormone also from the testes masculinize the brain. 3 complications of increasing messiness. In many rodents, the brain isn't quite sexually differentiated at birth, and these hormonal effects continue postnatally. A messier complication: surprisingly few testosterone effects in the brain result from the hormone binding to androgen receptors. Instead, testosterone enters target cells and, bizarrely, is converted to estrogen, then binds to intracellular estrogen receptors, while testosterone has its effects outside the brain either as itself or after intracellular conversion to a related androgen, dihydrotestosterone. Thus testosterone has much of its masculinizing effect in the brain by becoming estrogen. The conversion of testosterone to estrogen also occurs in the fetal brain. Wait. Regardless of fetal sex, fetal circulation is full of maternal estrogen, plus female fetuses secrete estrogen. Thus female fetal brains are bathed in estrogen. Why doesn't that masculinize the female fetal brain? Most likely it's because fetuses make something called alpha-fetoprotein, which binds circulating estrogen, taking it out of action. So neither mom's estrogen, nor fetal-derived estrogen masculinizes the brain in female fetuses. And it turns out that unless there is testosterone and anti-Mullerian hormone around, fetal mammalian brains automatically feminize. 63. Now for the uber messy complication. What exactly is a female or male brain? This is where the arguments begin. To start, male brains merely consistently drool reproductive hormones out of the hypothalamus, whereas female brains must master the cyclic secretion of ovulatory cycles. Thus, fetal life produces a hypothalamus that is more complexly wired in females. But how about sex differences in the behaviors that interest us? The question is, how much of male aggression is due to prenatal masculinizing of the brain? Virtually all of it, if we're talking rodents. Work in the 1950s by Robert Goy of the University of Wisconsin showed that in guinea pigs, an organizational effect of perinatal testosterone is to make the brain responsive to testosterone in adulthood. 64. Near-term pregnant females would be treated with testosterone. This produced female offspring who as adults appeared normal but were behaviorally masculinized. They were more sensitive than control females to an injection of testosterone, with a greater increase in aggression and male, typical sexual behavior, i.e., mounting other females. Moreover, estrogen was less effective at eliciting female, typical sexual behavior, i.e., a back-arching reflex, called lordosis. Thus prenatal testosterone exposure had masculinizing organizational effects, so that these females as adults responded to the activational effects of testosterone and estrogen as males would. This challenged dogma that sexual identity is due to social, not biological, influences. This was the view of sociologists who hated high school biology. And of the medical establishment as well. According to this view, if an infant was born with sexually ambiguous genitalia, roughly 1 to 2% of births, it didn't matter which gender they were raised as long as you decided within the first 18 months, just do whichever reconstructive surgery was more convenient. 65. So here's Goy reporting that prenatal hormone environment, not social factors, determines adult sex-typical behaviors. But these are guinea pigs, was the retort. Goy and crew then studied non-human primates. A quick tour of sexually dimorphic, i.e., differing by sex, primate behavior. South American species such as marmosets and tamarins who form pair bonds, show few sex differences in behavior. In contrast, most Old World primates are highly dimorphic, males are more aggressive, and females spend more time at affiliative behaviors, e.g., social grooming, interacting with infants. How's this for a sex difference? In one study, adult male rhesus monkeys were far more interested in playing with masculine human toys, e.g., wheeled toys, than feminine ones, stuffed animals, while females had a slight preference for feminine. 66. What next? Female monkeys prefer young adult, fantasy novels with female protagonists. Why should human toys be relevant to sex differences in monkeys? The authors speculate that this reflects the higher activity levels in males, and how masculine toys facilitate more active play. Male rhesus monkeys show a strong preference for playing with stereotypically masculine versus feminine human toys. Visit bit.ly/208ojel for a larger version of this graph. Goy studied highly sexually dimorphic rhesus monkeys. There were already hints that testosterone has organizational effects on their behavior. Within weeks of birth, males are more active than females and spend more time in rough and tumble play. This is long before puberty and its burst of testosterone secretion. Furthermore, even if you suppress their testosterone levels at birth, low, but nevertheless still higher than those of females, males still do more roughing and tumbling. This suggested that the sex difference arose from fetal hormone differences. Goy proved this by treating pregnant monkeys with testosterone and examining their female offspring. Testosterone exposure throughout pregnancy produced daughters who were pseudohermaphrodites, looked like males on the outside, but had female gonads on the inside. When compared with control females, these androgenized females did more rough and tumble play, were more aggressive, and displayed male-typical mounting behavior and vocalizations as much as males by some measures. Importantly, most but not all behaviors were masculinized, and these androgenized females were as interested as control females and infants. Thus, testosterone has prenatal organizational effects on some but not all behaviors. In further studies, many carried out by Goy's student Kim Wallen of Emory University, pregnant females received lower doses of testosterone and only in the last trimester. 67. This produced daughters with normal genitalia but masculinized behavior. The authors noted the relevance of this to transgender individuals, the external appearance of one sex, but the brain, if you will, of the other. And U.S. Initially it seemed clear that prenatal testosterone exposure is also responsible for male aggression in humans. This was based on studies of a rare disorder, congenital adrenal hyperplasia, CAH. An enzyme in the adrenal glands has a mutation, and instead of making glucocorticoids, they make testosterone and other androgens starting during fetal life. The lack of glucocorticoids causes serious metabolic problems requiring replacement hormones. And what about the excessive androgens in CAH girls, who are typically born with ambiguous genitals and are infertile as adults? In the 1950s, psychologist John Money of Johns Hopkins University reported that CAH girls had pathologically high levels of male typical behaviors, a paucity of female typical ones, and elevated IQ. That sure stopped everyone in their tracks. But the research had some problems. First, the IQ finding was spurious. Parents willing to enroll their CAH child in these studies averaged higher levels of education than did controls. And the gender-typical behaviors. Normal was judged by 1950s Aussie and Harriet standards. CAH girls were pathologically interested in having careers and disinterested in having babies. Oops, back to the drawing board. Careful contemporary CAH research has been conducted by Melissa Hines of the University of Cambridge. 68. When compared with non-CAH girls do more rough and tumble play, fighting, and physical aggression. Moreover, they prefer masculine toys over dolls. As adults, they score lower on measures of tenderness and higher in aggressiveness and self-report more aggression and less interest in infants. In addition, CAH women are more likely to be gay or bisexual or have a transgender sexual identity. Importantly, drug treatments begun soon after birth normalize androgen levels in these girls, so that the excessive androgen exposure is solely prenatal. Thus, prenatal testosterone exposure appears to cause organizational changes that increase the incidence of male typical behaviors. A similar conclusion is reached by an inverse of CAH, namely androgen insensitivity syndrome, AIs, historically called testicular feminization syndrome. 69. A fetus is male, XY chromosomes, testes that secrete testosterone. But a mutation in the androgen receptor makes it insensitive to testosterone. Thus the testes can secrete testosterone till the cows come home. But there won't be any masculinization. And often the individual is born with a female external phenotype and is raised as a girl. Along comes puberty, she's not getting periods, and a trip to the doctor reveals that the girl is actually a boy with testes typically near the stomach, plus a shortened vagina that dead ends. The individual usually continues with a female identity, but is infertile as an adult. In other words, when human males don't experience the organizational prenatal effects of testosterone, you get female atypical behaviors and identification. Between CAH and AIs, the issue seems settled. Prenatal testosterone plays a major role in explaining sex differences in aggression and various affiliative, prosocial behaviors in humans. Careful readers may have spotted two whopping big problems with this conclusion. 70. Remember that CAH girls are born with a something's very different. Post-it, the ambiguous genitalia, typically requiring multiple reconstructive surgeries. CAH females are not merely prenatally androgenized. They're also raised by parents who know something is different, have slews of doctors, mighty interested in their privates, and are treated with all sorts of hormones. It's impossible to attribute the behavioral profile solely to the prenatal androgens. Testosterone has no effects in AIS individuals because of the androgen receptor mutation. But doesn't testosterone have most of its fetal brain effects as estrogen interacting with the estrogen receptor? That aspect of brain masculinization should have occurred despite the mutation. Complicating things, some of the masculinizing effects of prenatal testosterone in monkeys don't require conversion to estrogen. So we have genetically and gonadally male individuals with at least some brain masculinization raised successfully as females. The picture is complicated further. AI's individuals raised female have higher than expected rates of being gay and of having an other than female or neither female nor male sex/gender self-identification. ARC. All we can say is that there is imperfect evidence that testosterone has masculinizing prenatal effects in humans as in other primates. The question becomes how big these effects are. Answering that question would be easy if you knew how much testosterone people were exposed to as fetuses. Which brings up a truly quirky finding, one likely to cause readers to start futzing awkwardly with a ruler. Weirdly, prenatal testosterone exposure influences digit length. 71. Specifically, while the second finger is usually shorter than the fourth finger, the difference, the 2D/FOURD ratio, is greater in men than in women, something first noted in the 1880s. The difference is demonstrable in third trimester fetuses, and the more fetal testosterone exposure as assessed by amniocentesis, the more pronounced the ratio. Moreover, females have a more masculine ratio, as do females who share their fetal environment and thus some testosterone with a male twin, while AI's males have a more feminine ratio. The sex difference in the ratio occurs in other primates and rodents. And no one knows why this difference exists. Moreover, this oddity is not alone. A barely discernible background, noise generated by the inner ear otoacoustic emissions, shows a sex difference that reflects prenatal testosterone exposure. Go explain that. The 2D/FOURD ratio is so variable, and the sex difference so small that you can't determine someone's sex by knowing it. But it does tell you something about the extent of fetal testosterone exposure. So what does the extent of exposure, as assessed by the ratio, predict about adult behavior? Men with more masculine 2D/FOURD ratios tend toward higher levels of aggression and math scores, more assertive personalities, higher rates of ADHD and autism, diseases with strong male biases, and decreased risk of depression and anxiety disorders with a female skew. The faces and handwriting of such men are judged to be more masculine. Furthermore, some reports show a decreased likelihood of being gay. Women having a more feminine ratio have less chance of autism and more of anorexia, a female bias disease. They're less likely to be left-handed, a male skewed trait. Moreover, they exhibit less athletic ability and more attraction to highly masculine faces. And they're more likely to be straight or, if lesbian, more likely to take stereotypical female sexual roles. 72. This constitutes some of the strongest evidence that fetal androgen exposure has organizational effects on adult behavior in humans as in other species, and b. that individual differences in the extent of such exposure predict individual differences in adult behavior. 73. Prenatal endocrine environment is destiny. Well, not exactly. These effects are small and variable, producing a meaningful relationship only when considering large numbers of individuals. Do testosterone's organizational effects determine the quality and/or quantity of aggression? No. How about the organizational plus the activational effects? Not those either. Expanding the scope of environment. Thus the fetal brain can be influenced by hormones secreted by the fetus. But in addition, the outside world alters a pregnant woman's physiology, which in turn affects the fetal brain. The most obvious version of this is how food ingested by a pregnant female influences what nutrients are delivered to the fetal circulation at an extreme. Maternal malnutrition broadly impairs fetal brain development. 74. Moreover, pathogens acquired by the mother can be passed to the fetus. For example, the protozoan parasite Toxoplasma gondii can infect someone pregnant, typically after exposure to infected cat feces, and eventually reach the fetal nervous system, potentially wreaking serious havoc. And this is also the world of maternal substance abuse producing heroin and crack babies or fetal alcohol syndrome. Importantly, maternal stress impacts fetal development. There are indirect routes. For example, stressed people consume less healthy diets and consume more substances of abuse. More directly, stress alters maternal blood pressure and immune defenses, which impact a fetus. Most important, stressed mothers secrete glucocorticoids, which enter fetal circulation, and basically have the same bad consequences as in stressed infants and children. Glucocorticoids accomplish this through organizational effects on fetal brain construction and decreasing levels of growth factors, numbers of neurons and synapses, and so on. Just as prenatal testosterone exposure generates an adult brain that is more sensitive to environmental triggers of aggression, excessive prenatal glucocorticoid exposure produces an adult brain more sensitive to environmental triggers of depression and anxiety. In addition, prenatal glucocorticoid exposure has effects that blend classical developmental biology with molecular biology. To appreciate this, here's a highly simplified version of the next chapter's focus on genes. Each gene specifies the production of a specific type of protein. B. A gene has to be activated for the protein to be produced and deactivated to stop producing it. Thus genes come with on-off switches. C. Every cell in our bodies contains the same library of genes. D. During development, the pattern of which genes are activated determines which cells turn into nose, which into toes, and so on. E. Forever after, nose, toes, and other cells retain distinctive patterns of gene activation. Chapter 4 discussed how some hormones have activational effects by altering on-off switches on particular genes, e.g., testosterone-activating genes related to increased growth in muscle cells. The field of epigenetics concerns how some hormonal organizational effects arise from permanently turning particular genes on or off in particular cells. 75. Plenty more on this in the next chapter. This helps explain why your toes and nose work differently. More important, epigenetic changes also occur in the brain. This domain of epigenetics was uncovered in a landmark 2004 study by Meany and colleagues, one of the most cited papers, published in the prestigious journal Nature Neuroscience. They had shown previously that offspring of more attentive rat mothers, those that frequently nurse, groom and lick their pups, become adults with lower glucocorticoid levels, less anxiety, better learning and delayed brain aging. The paper showed that these changes were epigenetic, that mothering style altered the on-off switch in a gene relevant to the brain's stress response. Mothering style alters gene regulation in pups' brains. Remarkably, Meany, along with Darlene Francis of the University of California, Berkeley, then showed that such rat pups as adults are more attentive mothers, passing this trait epigenetically to the next generation. Thus, adult behavior produces persistent molecular brain changes in offspring, programming them to be likely to replicate that distinctive behavior in adulthood. 76. More findings flooded in, many provided by Meany, his collaborator Moshe Seif, also of McGill, and Francis Champagne of Columbia University. 77. Hormonal responses to various fetal and childhood experiences have epigenetic effects on genes related to the growth factor BDNF, to the vasopressin and oxytocin system, and to estrogen sensitivity. These effects are pertinent to adult cognition, personality, emotionality, and psychiatric health. Childhood abuse, for example, causes epigenetic changes in hundreds of genes in the human hippocampus. Moreover, Stephen Suomi of the National Institutes of Health and Seif found that mothering style in monkeys has epigenetic effects on more than 1,000 frontocortical genes. This is totally revolutionary. Sort of. Which segues to a chapter summary. Conclusions. Epigenetic environmental effects on the developing brain are hugely exciting. Nonetheless, curbing of enthusiasm is needed. Findings have been overinterpreted, and as more researchers flock to the subject, the quality of studies has declined. Moreover, there is the temptation to conclude that epigenetics explains everything. Whatever that might be, most effects of childhood experience on adult outcomes probably don't involve epigenetics, and, stay tuned, most epigenetic changes are transient. Particularly strong criticisms come from molecular geneticists rather than behavioral scientists, who generally embrace the topic. Some of the negativity from the former, I suspect, is fueled by the indignity of having to incorporate the likes of rat mothers, licking their pups, into their beautiful world of gene regulation. But the excitement should be restrained on a deeper level, one relevant to the entire chapter. Stimulating environments, harsh parents, good neighborhoods, uninspiring teachers, optimal diets, all alter genes in the brain. Wow. And not that long ago the revolution was about how environment and experience change the excitability of synapses, their number, neuronal circuits, even the number of neurons. Whoa. And earlier the revolution was about how environment and experience can change the sizes of different parts of the brain. Amazing. But none of this is truly amazing. Because things must work these ways. While little in childhood determines an adult behavior, virtually everything in childhood changes propensities towards some adult behavior. Freud, Bowlby, Harlow, Meany, from their differing perspectives, all make the same fundamental and once revolutionary point: childhood matters. All that the likes of growth factors, on/off switches, and rates of myelination do is provide insights into the innards of that fact. Such insight is plenty useful. It shows the steps linking childhood, pointed to adult. Z. It shows how parents can produce offspring whose behaviors resemble their own. It identifies Achilles' heels that explain how childhood adversity can make for damaged and damaging adults. And it hints at how bad outcomes might be reversed and good outcomes reinforced. There is another use. In Chapter 2 I recounted how it required the demonstration of hippocampal volume loss in combat vets with PTSD to finally convince many in power that the disorder is real. Similarly, it shouldn't require molecular genetics or neuroendocrinology factoids to prove that childhood matters, and thus that it profoundly matters to provide childhoods filled with good health and safety, love and nurturance and opportunity. But insofar as it seems to require precisely that sort of scientific validation at times, more power to those factoids. 8. Back to when you were just a fertilized egg. I'm reminded of a cartoon where one lab-coated scientist is telling the other, you know how you're on the phone and the other person wants to get off, but won't say it, so they say, well, you probably need to get going, like you're the one who wants to get off, when it's really them. I think I found the gene for that. This chapter is about progress in finding the gene for that. Our prototypical behavior has occurred. How was it influenced by events when the egg and sperm that formed that person joined, creating their genome, the chromosomes, the sequences of DNA, destined to be duplicated in every cell in that future person's body? What role did those genes play in causing that behavior? Genes are relevant to, say, aggression, which is why we're less alarmed if a toddler pulls at the ears of a basset hound rather than a pit bull. Genes are relevant to everything in this book. Many neurotransmitters and hormones are coded for by genes, as are molecules that construct or degrade those messengers, as are their receptors. Ditto for growth factors guiding brain plasticity. Genes typically come in different versions. We each consist of an individuated orchestration of the different versions of our approximately 20,000 genes. This topic carries two burdens. The first reflects many people being troubled by linking genes with behavior. In one incident from my academic youth, a federally funded conference was canceled for suggesting that genes were pertinent to violence. This suspicion of gene behavior links exists because of the pseudoscientific genetics used to justify various isms, prejudice, and discrimination. Such pseudoscience has fostered racism and sexism, birthed eugenics and forced sterilizations, allowed scientifically meaningless versions of words like innate to justify the neglect of have-nots. And monstrous distortions of genetics have fueled those who lynch, ethnically cleanse, or march children into gas chambers. 1. But studying the genetics of behavior also carries the opposite burden of people who are overly enthusiastic about the subject. After all, this is the genomics era, with personalized genomic medicine, people getting their genomes sequenced, and popular writing about genomics giddy with terms like the Holy Grail and the Code of Codes. In a reductionist view, understanding something complex requires breaking it down into its components. Understand those parts, add them together, and you'll understand the big picture. And in this reductionist world, to understand cells, organs, bodies, and behavior, the best constituent part to study is genes. Overenthusiasm for genes can reflect a sense that people possess an immutable, distinctive essence, although essentialism predates genomics. Consider a study concerning moral spillover, based on kinship. Point 2. Suppose a person harmed people 2 generations ago. Are this person's grandchildren obliged to help his victim's grandchildren? Subjects viewed a biological grandchild as more obligated than one adopted into the family at birth. The biological relationship carried a taint. Moreover, subjects were more willing to jail 2 long-lost identical twins for a crime committed by one of them than to jail 2 unrelated, but perfect lookalikes. The former, raised in different environments, share a moral taint because of their identical genes. People see essentialism embedded in bloodlines, i.e., genes. This chapter threads between these two extremes, concluding that while genes are important to this book's concerns, they're far less so than often thought. The chapter first introduces gene function and regulation, showing the limits of genes' power. Next, it examines genetic influences on behavior in general. Finally, we'll examine genetic influences on our best and worst behaviors. Part 1, Genes from the Bottom Up. Start by considering the limited power of genes. If you are shaky about topics, such as the central dogma, DNA codes for RNA, which codes for protein sequence, protein structure determining function, the 3-nucleotide codon code, or the basics of point insertion and deletion mutations, first read the primer in Appendix 3. Do genes know what they are doing? The triumph of the environment. So genes specify protein structure, shape, and function. And since proteins do virtually everything, this makes DNA the holy grail of life. But no, genes don't decide when a new protein is made. Dogma was that there'd be a stretch of DNA in a chromosome, constituting a single gene, followed by a stop codon, followed immediately by the next gene, and then the next. But genes don't actually come one after another. Not all DNA constitutes genes. Instead, there are stretches of DNA between genes that are non-coding, that are not transcribed. And now a flabbergasting number: 95% of DNA is non-coding. 95%. What is that 95%? Some is junk, remnants of pseudogenes inactivated by evolution 3, but buried in that are the keys to the kingdom, the instruction manual for when to transcribe particular genes, the on-off switches for gene transcription. A gene doesn't decide when to be photocopied into RNA to generate its protein. Instead, before the start of the stretch of DNA coding for that gene is a short stretch called a promoter, the on switch. What turns the promoter switch on? Something called a transcription factor, TF, binds to the promoter. This causes the recruitment of enzymes that transcribe the gene into RNA. Meanwhile, other transcription factors deactivate genes. This is huge. Saying that a gene decides when it is transcribed is like saying that a recipe decides when a cake is baked. Thus, transcription factors regulate genes. What regulates transcription factors? The answer devastates the concept of genetic determinism, the environment. To start, unexcitingly, environment can mean intracellular environment. Suppose a hard-working neuron is low on energy. This state activates a particular transcription factor, which binds to a specific promoter, which activates the next gene in line, the downstream gene. This gene codes for a glucose transporter. More glucose transporter proteins are made and inserted into the cell membrane, improving the neuron's ability to access circulating glucose. Next, consider environment, including the neuron next door, which releases serotonin onto the neuron in question. Suppose less serotonin has been released lately. Sentinel transcription factors in dendritic spines sense this, travel to the DNA, and bind to the promoter upstream of the serotonin receptor gene. More receptor is made and placed in the dendritic spines, and they become more sensitive to the faint serotonin signal. Sometimes environment can be far-flung within an organism. A male secretes testosterone, which travels through the bloodstream and binds to androgen receptors in muscle cells. This activates a transcription factor cascade that results in more intracellular scaffolding proteins, enlarging the cell, i.e., muscle mass increases. Finally, and most important, there is environment, meaning the outside world. A female smells her newborn, meaning that odorant molecules that floated off the baby bind to receptors in her nose. The receptors activate and, many steps later in the hypothalamus, a transcription factor activates, leading to the production of more oxytocin. Once secreted, the oxytocin causes milk letdown. Genes are not the deterministic holy grail if they can be regulated by the smell of a baby's tushy. Genes are regulated by all the incarnations of environment. In other words, genes don't make sense outside the context of environment. Promoters and transcription factor introduce if-then clauses. If you smell your baby, then activate the oxytocin gene. Now the plot thickens. There are multiple types of transcription factors in a cell, each binding to a particular DNA sequence constituting a particular promoter. Consider a genome containing one gene. In that imaginary organism there is only a single profile of transcription, i.e., the gene is transcribed, requiring only one transcription factor. Now consider a genome consisting of genes A and B, meaning three different transcription profiles. A is transcribed, B is transcribed, A and B are transcribed, requiring three different TFS, assuming you activate only one at a time. 3 genes, 7 transcription profiles, A, B, C, A plus B, A plus C, B plus C, A plus B plus C, 7 different TFS. 4 genes, 15 profiles. 5 genes, 31 profiles. As the number of genes in a genome increases, the number of possible expression profiles increases exponentially, as does the number of TFS needed to produce those profiles. Now another wrinkle that, in the lingo of an ancient generation, will blow your mind. TFS are usually proteins coded for by genes. Back to genes A and B. To fully exploit them, you need the TF that activates gene A, and the TF that activates gene B, and the TF that activates genes A and B. Thus there must exist 3 more genes, each coding for one of those TFS, requiring TFS that activate those genes, and TFS for the genes coding for those TFS. Whoa! Genomes aren't infinite, instead TFS regulate one another's transcription, solving that pesky infinity problem. Importantly, across the species whose genomes have been sequenced, the longer the genome, i.e., roughly the more genes there are, the greater the percentage of genes coding for TFS. In other words, the more genomically complex the organism, the larger the percentage of the genome devoted to gene regulation by the environment. Back to mutations. Can there be mutations in DNA stretches constituting promoters? Yes, and more often than in genes themselves. In the 1970s, Alan Wilson and Mary Claire King at Berkeley correctly theorized that the evolution of genes is less important than the evolution of regulatory sequences upstream of genes, and thus how the environment regulates genes. Reflecting that, a disproportionate share of genetic differences between chimps and humans are in genes for TFS. Time for more complexity. Suppose you have genes 1, 10 and transcription factors A, B, and C. TFA induces the transcription of genes 1, 3, 5, 7, and 9. TFB induces genes 1, 2, 5, and 6. TFC induces 1, 5, and 10. Thus, upstream of gene 1 are separate promoters responding to TFS A, B, and C, thus genes can be regulated by multiple TFS. Conversely, each TF usually activates more than one gene, meaning that multiple genes are typically activated in networks. For example, cell injury causes a TF called NF-kappa B to activate a network of inflammation genes. Suppose the promoter upstream of gene 3 that responds to promoter TFA has a mutation, making it responsive to TFB. Result: gene 3 is now activated as part of a different network. Same network-wide outcome if there is a mutation in a gene for a TF, producing a protein that binds to a different promoter type. 4. Consider this: the human genome codes for about 1,500 different TFS, contains 4 million TF binding sites, and the average cell uses about 200,000 such sites to generate its distinctive gene expression profile. 5. This is boggling. Epigenetics. The last chapter introduced the phenomenon of environmental influences, freezing genetic on-off in one position. Such epigenetic changes were relevant to events, particularly in childhood, causing persistent effects on the brain and behavior. For example, recall those pair bonding prairie voles. When females and males first mate, there are epigenetic changes in regulation of oxytocin and vasopressin receptor genes in the Nucleus Accumbens, that target of mesolimbic dopamine, projection. 6. Let's translate the last chapter's imagery of freezing on-off switches into molecular biology. 7. What mechanisms underlie epigenetic changes in gene regulation? An environmental input results in a chemical being attached tightly to a promoter, or to some nearby structural protein surrounding DNA. The result of either is that TFS can no longer access or properly bind to the promoter, thus silencing the gene. As emphasized in the last chapter, epigenetic changes can be multigenerational. 8. Dogma was that all the epigenetic marks, i.e., changes in the DNA or surrounding proteins, were erased in eggs and sperm. But it turns out that epigenetic marks can be passed on by both, e.g., make male mice diabetic, and they pass the trait to their offspring via epigenetic changes in sperm. Recall one of the great punching bags of science history, the 18th century French biologist Jean-Baptiste Lamarck. 9. All anybody knows about the guy now is that he was wrong about heredity. Suppose a giraffe habitually stretches her neck to reach leaves high in a tree. This lengthens her neck. According to Lamarck, when she has babies, they will have longer necks because of acquired inheritance. Lunatic. Buffoon. Epigenetically mediated mechanisms of inheritance, now often called neo-Lamarckian inheritance, prove Lamarck right in this narrow domain. Centuries late, the guy's getting some acclaim. Thus, not only does environment regulate genes, but it can do so with effects that last days to lifetimes. The modular construction of genes, exons and introns. Time to do another dogma about DNA. It turns out that most genes are not coded for by a continuous stretch of DNA. Instead, there might be a stretch of non-coding DNA in the middle. In that case, the two separate stretches of coding DNA are called exons, separated by an intron. Many genes are broken into numerous exons, with, logically, one less intron than the number of exons. How do you produce a protein from an exonic gene? The RNA photocopy of the gene initially contains the exons and introns. An enzyme removes the intronic parts and splices together the exons. Clunky, but with big implications. Back to each particular gene coding for a particular protein. 10. Introns and exons destroy this simplicity. Imagine a gene consisting of exons 1, 2 and 3, separated by introns A and B. In one part of the body a splicing enzyme exists that splices out the introns and also trashes exon 3, producing a protein coded for by exons 1 and 2. Meanwhile, elsewhere in the body, a different splicing enzyme jettisons exon 2 along with the introns, producing a protein derived from exons 1 and 3. In another cell type a protein is made solely from exon 1. Thus alternative splicing can generate multiple unique proteins from a single stretch of DNA. So much for one gene specifies one protein. This gene specifies 7, A, B, C, A-B, A-C, BC, and A-BC. Remarkably, 90% of human genes with exons are alternatively spliced. Moreover, when a gene is regulated by multiple TFS, each can direct the transcription of a different combination of exons. Oh, and splicing enzymes are proteins, meaning that each is coded for by a gene. Loops and loops. Transposable genetic elements, the stability of the genome, and neurogenesis. Time to unmur another cherished idea, namely that genes inherited from your parents, i.e., what you started with as a fertilized egg, are immutable. This calls up a great chapter of science history. In the 1940s, an accomplished plant geneticist named Barbara McClintock observed something impossible. She was studying the inheritance of kernel color in maize, a frequent tool of geneticists, and found patterns of mutations unexplained by any known mechanism. The only possibility, she concluded, was that stretches of DNA had been copied, with the copy then randomly inserted into another stretch of DNA. Yeah, right. Clearly McClintock, with her derisively named jumping genes, had gone mad, and so she was ignored. Not exactly true, but this detracts from the drama. She soldiered on in epic isolation. And finally, with the molecular revolution of the 1970s, she was vindicated about her, now termed, transposable genetic elements, or transposons. She was lionized, canonized, Nobel-prized, and was wonderfully inspirational, as disinterested in acclaim as in her ostracism, working until her 90s. Transpositional events rarely produce great outcomes. Consider a hypothetical stretch of DNA coding for the fertilized egg is implanted in the uterus. There has been a transpositional event, where the underlying stretch of message was copied and randomly plunked down elsewhere. A fertilized eggterus is implanted in the uterus. Gibberish. But sometimes the fertilized egg is implanted in the uterus, becomes the fertilized eggplant is implanted in the uterus. Now, that's not an everyday occurrence. Plants utilize transposons. Suppose there is a drought, plants can't move to wetter pastures like animals can. Plant stress such as drought induces transpositions in particular cells, where the plant metaphorically shuffles its DNA deck, hoping to generate some novel savior of a protein. Mammals have fewer transposons than plants. The immune system is one transposon hotspot in the enormous stretches of DNA coding for antibodies. A novel virus invades, shuffling the DNA increases the odds of coming up with an antibody that will target the invader. The main point here is that transposons occur in the brain. 11. In humans, transpositional events occur in stem cells in the brain, when they are becoming neurons, making the brain a mosaic of neurons with different DNA sequences. In other words, when you make neurons, that boring DNA sequence you inherited isn't good enough. Remarkably, transpositional events occur in neurons that form memories in fruit flies. Even flies evolve such that their neurons are freed from the strict genetic marching orders they inherit. Chance. Finally, chance lessens genes as the code of codes. Chance, driven by Brownian motion, the random movement of particles in a fluid, has big effects on tiny things like molecules floating in cells, including molecules regulating gene transcription. 12. This influences how quickly an activated TF reaches the DNA. Splicing enzymes bump into target stretches of RNA, and an enzyme synthesizing something grabs the two precursor molecules needed for the synthesis. I'll stop here, otherwise I'll go on for hours. Some key points, completing this part of the chapter. A. Genes are not autonomous agents commanding biological events. B. Instead, genes are regulated by the environment, with environment consisting of everything from events inside the cell to the universe. C. Much of your DNA turns environmental influences into gene transcription, rather than coding for genes themselves. Moreover, evolution is heavily about changing regulation of gene transcription, rather than genes themselves. D. Epigenetics can allow environmental effects to be lifelong, or even multi-generational. E. And thanks to transposons, neurons contain a mosaic of different genomes. In other words, genes don't determine much. This theme continues as we focus on the effects of genes on behavior. L. Part 2, Genes from the Top Down, Behavior Genetics. Long before anything was known about promoters, exons, or transcription factors, it became clear that you study genetics top-down, by observing traits shared by relatives. Early in the last century, this emerged as the science of behavior genetics. As we'll see, the field has often been mired in controversy, typically because of disagreements over the magnitude of genetic effects on things like IQ or sexual orientation. First attempts. The field began with the primitive idea that, if everyone in a family does it, it must be genetic. This was confounded by environment running in families as well. The next approach depended on closer relatives having more genes in common than distant ones. Thus, if a trait runs in a family and is more common among closer relatives, it's genetic. But obviously, closer relatives share more environment as well. Think of a child and parent versus a child and grandparent. Research grew subtler. Consider someone's biological aunt, i.e., the sister of a parent, and the uncle who married the aunt. The uncle shares some degree of environment with the individual, while the aunt shares the same, plus genes. Therefore, the extent to which the aunt is more similar to the individual than the uncle reflects the genetic influence. But as we'll see, this approach has problems. More sophistication was needed. Twins, adoptees and adopted twins. A major advance came with twin studies. Initially, examining twins helped rule out the possibility of genetic determination of a behavior. Consider pairs of identical twins, sharing 100% of their genes. Suppose one of each pair has schizophrenia, does the twin as well? If there are any cases where the other twin doesn't, i.e., where the concordance rate is less than 100%, you've shown that the genome and epigenetic profile inherited at birth do not solely determine the incidence of schizophrenia. In fact, the concordance rate is about 50%. But then a more elegant twin approach emerged involving the key distinction between identical, monozygotic, or MZ, twins, who share 100% of their genes, and fraternal, non-identical, dizygotic, or DZ, twins, who, like all other sibling pairs, share 50% of their genes. Compare pairs of MZ twins with same-sex DZ twins. Each pair is the same age, was raised in the same environment, and shared a fetal environment. The only difference is the percentage of genes shared. Examine a trait occurring in one member of the twin pair. Is it there in the other? The logic ran that, if a trait is shared more among MZ than among DZ twins, that increased degree of sharing reflects the genetic contribution to the trait. Another major advance came in the 1960s. Identify individuals adopted soon after birth. All they share with their biological parents is genes. All they share with their adoptive parents is environment. Thus, if adopted individuals share a trait more with their biological than with their adoptive parents, you've uncovered a genetic influence. This replicates a classic tool in animal studies, namely cross-fostering, switching newborn rat pups between two mothers. The approach was pioneered in revealing a strong genetic component to schizophrenia. 13. Then came the most wonderful, amazing, totally awesome thing ever in behavior genetics, started by Thomas Bouchard of the University of Minnesota. In 1979, Bouchard found a pair of identical twins who were, get this, separated at birth and adopted into different homes, with no knowledge of each other's existence until being reunited as adults. 14. Identical twins separated at birth are so spectacular and rare that behavior geneticists swoon over them, want to collect them all. Bouchard eventually studied more than 100 such pairs. The attraction was obvious: same genes, different environments, and the more different the better. Thus, similarities in behavior probably reflect genetic influences. Here's an imaginary twin pair that would be God's gift to behavior geneticists: identical twin boys separated at birth. One, Shmuel, is raised as an Orthodox Jew in the Amazon. The other, Wolfie, is raised as a Nazi in the Sahara. Reunite them as adults and see if they do similar quirky things like, say, flushing the toilet before using it. Flabbergastingly, one twin pair came close to that. They were born in 1933 in Trinidad to a German Catholic mother and a Jewish father. When the boys were 6 months of age, the parents separated. The mother returned to Germany with one son, and the other remained in Trinidad with the father. The latter was raised there and in Israel as Jack Youth, an observant Jew whose first language was Yiddish. The other, Oscar Storr, was raised in Germany as a Hitler Youth zealot. Reunited and studied by Bouchard, they warily got to know each other, discovering numerous shared behavioral and personality traits including flushing the toilet before use. As we'll see, studies were more systematic than just documenting bathroom quirks. The flushing detail, however, always comes up in accounts of the pair. Behavior geneticists, wielding adoption and twin approaches, generated scads of studies, filling specialized journals like Genes, Brain and Behavior and Twin Research and Human Genetics. Collectively, the research consistently showed that genetics plays a major role in a gamut of domains of behavior, including IQ and its subcomponents, i.e., verbal ability and spatial ability, 15 schizophrenia, depression, bipolar disorder, autism, attention deficit disorder, compulsive gambling, and alcoholism. Nearly as strong genetic influences were shown for personality measures related to extroversion, agreeableness, conscientiousness, neuroticism, and openness to experience, known as the Big Five personality traits. 16. Likewise with genetic influences on degree of religiosity, attitude toward authority, attitude toward homosexuality, and propensities toward cooperation and risk-taking in games. Other twin studies showed genetic influences on the likelihood of risky sexual behavior and on people's degree of attraction to secondary sexual characteristics, e.g., musculature in men, breast size in women. 17. Meanwhile, some social scientists report genetic influences on the extent of political involvement and sophistication, independent of political orientation. There are behavior genetics papers in the American Journal of Political Science. 18. Genes everywhere. Large genetic contributions have even been uncovered, for everything from the frequency with which teenagers text to the occurrence of dental phobias. 19. So does this mean there is a gene for finding chest hair on guys to be hot, for likelihood of voting, for feelings about dentists? Vanishingly unlikely. Instead, gene and behavior are often connected by tortuous roots. 20. Consider the genetic influence on voter participation. The mediating factor between the two turns out to be sense of control and efficacy. People who vote regularly feel that their actions matter, and this central locus of control reflects some genetically influenced personality traits, e.g., high optimism, low neuroticism. Or how about the link between genes and self-confidence? Some studies show that the intervening variable is genetic effects on height. Taller people are considered more attractive and treated better, boosting their self-confidence. Damn it. In other words, genetic influences on behavior often work through very indirect routes, something rarely emphasized when news broadcasts toss out behavior genetics sound bites. Scientists report genetic influence on strategy when playing Candyland. The debates about twin and adoption studies. Many scientists have heavily criticized the assumptions in twin and adoption studies, showing that they generally lead to overestimates of the importance of genes. Most behavior geneticists recognize these problems but argue that the overestimates are tiny. 21. A summary of this technical, but important debate. Criticism number 1, twin studies are premised on MZ and same-sex DZ twin pairs sharing environment equally, while sharing genes to very different extents. This equal environment assumption is simply wrong. Starting with parents, MZ twins are treated more similarly than DZ twins, creating more similar environments for them. If this isn't recognized, greater similarity between Ms will be misattributed to genes. 22. Scientists such as Kenneth Kendler of Virginia Commonwealth University, a dean of the field, have tried to control for this by quantifying just how similar childhoods were for twins with respect to variables like sharing rooms, clothing, friends, teachers, and adversity. B. Examining cases of mistaken zygosity, where parents were wrong about their twins' MZ/DZ status, thus, for example, raising their DZ twins as if they were MZ, and C. Comparing full minus, half minus, and step siblings who were reared together for differing lengths of time. Most of these studies show that controlling for the assumption of Ms sharing more environment than do DZs doesn't significantly reduce the size of genetic influences. 23. Hold that thought. Criticism number 2, MZ twins experience life more similarly starting as fetuses. DZ twins are dichorionic, meaning that they have separate placentas. In contrast, 75% of MZ twins share one placenta, i.e., are monochorionic. Thus most MZ twin fetuses share maternal blood flow more than do DZ twins, and thus are exposed to more similar levels of maternal hormones and nutrients. If that isn't recognized, greater similarity in Ms will be misattributed to genes. Various studies have determined what the chorionic status was in different MZ pairs, and then examined endpoints related to cognition, personality, and psychiatric disease. By a small margin, most studies show that chorionic status does make a difference, leading to overestimates of genetic influence. How big of an overestimation? As stated in one review, small but not negligible. 24. Criticism number 3, recall that adoption studies assume that, if a child is adopted soon after birth, she shares genes but no environment with her biological parents. But what about prenatal environmental effects? A newborn just spent 9 months, sharing the circulatory environment with mom. Moreover, eggs and sperm can carry epigenetic changes into the next generation. If these various effects are ignored, an environmentally based similarity between mother and child would be misattributed to genes. Epigenetic transmission via sperm seems of small significance. But prenatal and epigenetic effects from the mother can be huge. For example, the Dutch hunger winter phenomenon showed that third trimester malnutrition increased the risk of some adult diseases more than tenfold. This confound can be controlled for. Roughly half your genes come from each parent, but prenatal environment comes from mom. Thus, traits shared more with biological mothers than with fathers argue against a genetic influence. The few tests of this, concerning the genetic influence on schizophrenia demonstrated in twin studies, suggest that prenatal effects aren't big. Criticism number 4, adoption studies assume that a child and adoptive parents share environment but not genes. 25. That might approach being true if adoption involves choosing adoptive parents randomly among everyone on Earth. Instead, adoption agencies prefer to place children with families of similar racial or ethnic background as the biological parents, a policy advocated by the National Association of Black Social Workers and the Child Welfare League. Thus, kids and adoptive parents typically share genes at a higher than chance level. If this isn't recognized, a similarity between them will be misattributed to environment. Researchers admit there is selective placement but argue over whether it's consequential. This remains unsettled. Bouchard, with his twins separated at birth, controlled for cultural, material, and technological similarities between the separate homes of twin pairs, concluding that shared similarity of home environments due to selective placement was a negligible factor. A similar conclusion was reached in a larger study carried out by both Kendler and another dean of the field, Robert Sapolsky of King's College London. These conclusions have been challenged. The most fire-breathing critic has been Princeton psychologist Leon Kamin, who argues that concluding that selective placement isn't important is wrong, because of misinterpretation of results, use of wimpy analytical tests, and over-reliance on questionable retrospective data. He wrote, "We suggest that no scientific purpose is served by the flood of heritability estimates generated by these studies." 26. Here's where I give up. If super smart people who think about this issue all the time can't agree, I sure don't know how seriously selective placement distorts the literature. Criticism number 5, adoptive parents tend to be more educated, wealthier, and more psychiatrically healthy than biological parents. 27. Thus, adoptive households show range restriction, being more homogeneous than biological ones, which decreases the ability to detect environmental effects on behavior. Predictably, attempts to control for this satisfy only some critics. So what do we know after this slog through the criticisms and counter-criticisms about adoption and twin studies? Everyone agrees that confounds from prenatal environment, epigenetics, selective placement, range restriction, and assumptions about equal environment are unavoidable. Most of these confounds inflate the perceived importance of genes. Efforts have been made to control for these confounds and generally have shown that they are of less magnitude than charged by many critics. Crucially, these studies have mostly been about psychiatric disorders, which, while plenty interesting, aren't terribly relevant to the concerns of this book. In other words, no one has studied whether these confounds matter when considering genetic influences on, say, people's tendency to endorse their culture's moral rules yet rationalize why those rules don't apply to them today, because they're stressed and it's their birthday. Lots more work to be done. The fragile nature of heritability estimates. Now starts a bruising, difficult, immensely important subject. I review its logic every time I teach it, because it's so unintuitive and I'm still always just words away from getting it wrong when I open my mouth in class. Behavior genetics studies usually produce a number, called a heritability score 0.28. For example, studies have reported heritability scores in the 40 to 60% range for traits related to prosocial behavior, resilience after psychosocial stress, social responsiveness, political attitudes, aggression, and leadership potential. What's a heritability score? What does a gene do? Is at least two questions. How does a gene influence average levels of a trait? How does a gene influence variation among people in levels of that trait? These are crucially different. For example, how much do genes have to do with people's scores, averaging 100 on this thing called an IQ test? Then how much do genes have to do with one person scoring higher than another? Or how much do genes help in explaining why humans usually enjoy ice cream? How much in explaining why people like different flavors? These issues utilize two terms with similar sounds but different meanings. If genes strongly influence average levels of a trait, that trait is strongly inherited. If genes strongly influence the extent of variability around that average level, that trait has high heritability. It is a population measure where a heritability score indicates the percentage of total variation attributable to genetics. The difference between an inherited trait and heritability generates at least two problems that inflate the putative influence of genes. First, people confuse the two terms. Things would be easier if heritability were called something like gene tendency and in a consistent direction. People often mistakenly believe that if a trait is strongly inherited, it's thus highly heritable. And it's particularly bad that confusion is typically in that direction, because people are usually more interested in variability of traits among humans than in average levels of traits. For example, it's more interesting to consider why some people are smarter than others than why humans are smarter than turnips. The second problem is that research consistently inflates heritability measures, leading people to conclude that genes influence individual differences more than they do. Let's slowly work through this, because it's really important. The difference between a trait being inherited and having a high degree of heritability. You can appreciate the difference by considering cases where they dissociate. First, an example of a trait that is highly inherited, but has low heritability offered by the philosopher Ned Block 29. What do genes have to do with humans averaging 5 fingers per hand? Tons. It's an inherited trait. What do genes have to do with variation around that average? Not much. Cases of other than 5 fingers on a hand are mostly due to accidents. While average finger number is an inherited trait, the heritability of finger number is low. Genes don't explain individual differences much. Or stated differently, say you want to guess whether some organism's limb has 5 fingers or a hoof. Knowing their genetic makeup will help by identifying their species. Alternatively, you're trying to guess whether a particular person is likely to have 5 or 4 fingers on his hand. Knowing whether he uses buzz saws while blindfolded is more useful than knowing the sequence of his genome. Next, consider the opposite, a trait that is not highly inherited, but which has high heritability. What do genes directly have to do with humans being more likely than chimps to wear earrings? Not much. Now consider individual differences among humans. How much do genes help predict which individuals are wearing earrings at a high school dance in 1958? Tons. Basically, if you had 2 X chromosomes, you probably wore earrings, but if you had a Y chromosome, you wouldn't have been caught dead doing so. Thus, while genes had little to do with the prevalence of earrings among Americans in 1958 being around 50%, they had lots to do with determining which Americans wore them. Thus, in that time and place, wearing earrings, while not a strongly inherited trait, had high heritability. The reliability of heritability measures. We're now clear on the difference between inherited traits and their degree of heritability, and recognize that people are usually more interested in the latter, you versus your neighbor, than the former, you versus a wildebeest. As we saw, scads of behavioral and personality traits have heritability scores of 40 to 60%, meaning that genetics explains about half the variability in the trait. The point of this section is that the nature of research typically inflates such scores 30. Say a plant geneticist sits in the desert, studying a particular species of plant. In this imaginary scenario, a single gene, gene 3127, regulates the plant's growth. Gene 3127 comes in versions A, B, and C. Plants with version A always grow to be 1 inch tall, version B, 2 inches, C, 3 inches. What single fact gives you the most power in predicting a plant's height? Obviously, whether it has version A, B, or C, that explains all the variation in height between plants, meaning 100% heritability. Meanwhile, 12,000 miles away in a rainforest, a second plant geneticist is studying a clone of that same plant. And in that environment, plants with version A, B, or C are 101, 102, or 103 inches tall, respectively. This geneticist also concludes that plant height in this case shows 100% heritability. Then, as required by the plotline, the two stand side by side at a conference, one brandishing 1/2/3 inch data, the other 101/102/103. They combine data sets. Now you want to predict the height of one example of that plant, taken from anywhere on the planet. You can either know which version of gene 3127 it possesses or what environment it is growing in. Which is more useful? Knowing which environment. When you study this plant species in two environments, you discover that heritability of height is minuscule. Neon lights. This is crucial. Study a gene in only one environment and, by definition, you've eliminated the ability to see if it works differently in other environments. In other words, if other environments regulate the gene differently. And thus you've artificially inflated the importance of the genetic contribution. The more environments in which you study a genetic trait, the more novel environmental effects will be revealed, decreasing the heritability score. Scientists study things in controlled settings to minimize variation in extraneous factors and thus get cleaner, more interpretable results, for example, making sure that the plants all have their height measured around the same time of year. This inflates heritability scores, because you've prevented yourself from ever discovering that some extraneous environmental factor isn't actually extraneous. Thus a heritability score tells how much variation in a trait is explained by genes in the environment S, in which it's been studied. As you study the trait in more environments, the heritability score will decrease. This is recognized by Bouchard. These conclusions derived from a behavior genetics study can be generalized, of course, only to new populations exposed to a range of environments similar to those studied. 31. Okay, that was slick on my part, inventing a plant that grows in both desert and rainforest, just to trash heritability scores. Real plants rarely occur in both of those environments. Instead, in one rainforest the three gene versions might produce plants of heights 1, 2, and 3 inches, while in another they are 1.1, 2.1, and 3.1, producing a heritability score that, while less than 100%, is still extremely high. Genes typically still play hefty roles in explaining individual variability, given that any given species lives in a limited range of environments. Capybaras stick to the tropics, polar bears to the Arctic. This business about heterogeneous environments driving down heritability scores is important only in considering some hypothetical species that, say, lives in both tundra and desert, in various population densities, in nomadic bands, sedentary farming communities, and urban apartment buildings. Oh, that's right, humans. Of all species, heritability scores in humans plummet the most when shifting from a controlled experimental setting to considering the species' full range of habitats. Just consider how much the heritability score for wearing earrings, with its gender split, has declined since 1958. Now to consider an extremely important complication. Gene/environment interactions. Back to our plant. Imagine a growth pattern in environment of 1, 1, and 1 for the three gene variants, while in environment B it's 10, 10, and 10. When considering the combined data from both environments, heritability is 0, variation is entirely explained by which environment the plant grew in. Now, instead in environment A it's 1, 2, and 3, while in environment B, it's also 1, 2, and 3. Heritability is 100%, with all variability in height explained by genetic variation. Now say environment A is 1, 2, and 3, and environment B is 1.5, 2.5, 3.5. Heritability is somewhere between 0% and 100%. Now for something different, environment A, 1, 2, 3. Environment B, 3, 2, 1. In this case, even talking about a heritability score is problematic, because different gene variants have diametrically opposite effects in different environments. We have an example of a central concept in genetics, a gene-environment interaction, where qualitative, rather than just quantitative, effects of a gene differ by environment. Here's a rule of thumb for recognizing gene-environment interactions. Translated into English, you are studying the behavioral effects of a gene in two environments. Someone asks, what are the effects of the gene on some behavior? You answer, it depends on the environment. Then they ask, what are the effects of environment on this behavior? And you answer, it depends on the version of the gene. It depends equals a gene-environment interaction. Here are some classic examples concerning behavior 3.2. The disease phenylketonuria arises from a single gene mutation. Skipping over details, the mutation disables an enzyme that converts a potentially neurotoxic dietary constituent, phenylalanine, into something safe. Thus, if you eat a normal diet, phenylalanine accumulates, damaging the brain. But eat a phenylalanine-free diet from birth, and there is no damage. What are the effects of this mutation on brain development? It depends on your diet. What's the effect of diet on brain development? It depends on whether you have this rare mutation. Another gene-environment interaction pertains to depression, a disease involving serotonin abnormalities. 33. A gene called 5-HTT codes for a transporter that removes serotonin from the synapse. Having a particular 5-HTT variant increases the risk of depression. But only when coupled with childhood trauma. What's the effect of 5-HTT variant on depression risk? It depends on childhood trauma exposure. What's the effect of childhood trauma exposure on depression risk? It depends on 5-HTT variant, plus loads of other genes, but you get the point. Another example concerns FADS2, a gene involved in fat metabolism. 34. One variant is associated with higher IQ, but only in breastfed children. Same pair of what's the effect, questions, same it depends, answers. One final gene-environment interaction was revealed in an important 1999 science paper. The study was a collaboration among 3 behavioral geneticists, one at Oregon Health Sciences University, one at the University of Alberta, and one at the State University of New York in Albany. 35. They studied mouse strains known to have genetic variants relevant to particular behaviors, e.g., addiction or anxiety. First, they ensured that the mice from a particular strain were essentially genetically identical in all 3 labs. Then the scientists did cartwheels to test the animals in identical conditions in the labs. They standardized everything. Because some mice were born in the lab, but others came from breeders, homegrounds were given bouncy van rides to simulate the jostling that commercially bred mice undergo during shipping, just in case that was important. Animals were tested at the same day of age on the same date at the same local time. Animals had been weaned at the same age and lived in the same brand of cage with the same brand and thickness of sawdust bedding, changed on the same day of the week. They were handled the same number of times by humans wearing the same brand of surgical gloves. They were fed the same food and kept in the same lighting environment at the same temperature. The environments of these animals could hardly have been more similar if the 3 scientists had been identical triplets separated at birth. What did they observe? Some gene variants showed massive gene-environment interactions with variants having radically different effects in different labs. Here's the sort of data they got. Take a strain called 129/Svevtak and a test measuring the effects of cocaine on activity. In Oregon, cocaine increased activity in these mice by 667 centimeters of movement per 15 minutes. In Albany, an increase of 701. Those are pretty similar numbers, good. And in Alberta, more than 5,000. That's like identical triplets pole vaulting, each in a different location. They've all had the same training, equipment, running surface, night's rest, breakfast, and brand of underwear. The first 2 vault, 18 feet and 18 feet 1 inch, and the third vaults 108 feet. Maybe the scientists didn't know what they were doing, maybe the labs were chaotic. But variability was small within each lab, showing stable environmental conditions. And crucially, a few variants didn't show a gene-environment interaction, producing similar effects in the 3 labs. What does this mean? That most of the gene variants were so sensitive to environment that gene-environment interactions occurred even in these obsessively similar lab settings where incredibly subtle and still unidentified environmental differences made huge differences in what the gene did. Citing gene/environment interactions is a time-honored genetics cliché. Point 36. My students roll their eyes when I mention them. I roll my eyes when I mention them. Eat your vegetables, floss your teeth. Remember to say, it's difficult to quantitatively assess the relative contributions of genes and environment to a particular trait when they interact. This suggests a radical conclusion. It's not meaningful to ask what a gene does, just what it does in a particular environment. This is summarized wonderfully by the neurobiologist Donald Hebb. It is no more appropriate to say things like characteristic A is more influenced by nature than nurture than to say that the area of a rectangle is more influenced by its length than its width. It's appropriate to figure out if lengths or widths explain more of the variability in a population of rectangles, but not in individual ones. As we conclude part 2 of this chapter, some key points. A gene's influence on the average value of a trait, i.e., whether it is inherited, differs from its influence on variability of that trait across individuals, its heritability. B. Even in the realm of inherited traits, say, the inheritance of 5 fingers as the human average, you can't really say that there is genetic determination in the classically hard-assed sense of the word. This is because the inheritance of a gene's effect requires not just passing on the gene, but also the context that regulates the gene in that manner. C. Heritability scores are relevant only to the environments in which the traits have been studied. The more environments you study a trait in, the lower the heritability is likely to be. D. Gene/environment interactions are ubiquitous and can be dramatic. Thus, you can't really say what a gene does, only what it does in the environments in which it's been studied. Current research actively explores gene-environment interactions. Point 37. How's this for fascinating? Heritability of various aspects of cognitive development is very high, e.g., around 70% for IQ in kids from high socioeconomic status (SES) families, but is only around 10% in low SES kids. Thus, higher SES allows the full range of genetic influences on cognition to flourish, whereas lower SES settings restrict them. In other words, genes are nearly irrelevant to cognitive development if you're growing up in awful poverty. Poverty's adverse effects trump the genetics. Similarly, heritability of alcohol use is lower among religious than non-religious subjects, i.e., your genes don't matter much if you're in a religious environment that condemns drinking. Domains like these showcase the potential power of classical behavior genetics. Part 3. So what do genes actually have to do with behaviors we're interested in? The marriage of behavior genetics and molecular genetics. Behavior genetics has gotten a huge boost by incorporating molecular approaches. After examining similarities and differences between twins or adoptees, find the actual genes that underlie those similarities and differences. This powerful approach has identified various genes relevant to our interests. But first, our usual caveats: not all of these findings consistently replicate. B. Effect sizes are typically small. In other words, some gene may be involved, but not in a major way. And C. The most interesting findings show gene-environment interactions. Studying candidate genes. Gene searches can take a candidate approach or a genome-wide association approach. Stay tuned. The former requires a list of plausible suspects, genes already known to be related to some behavior. For example, if you're interested in a behavior that involves serotonin, obvious candidate genes would include those coding for enzymes that make or degrade serotonin, pumps that remove it from the synapse, or serotonin receptors. Pick one that interests you, and study it in animals using molecular tools to generate knockout mice, where you've eliminated that gene, or transgenic mice, with an extra copy of the gene. Make manipulations like these only in certain brain regions, or at certain times. Then examine what's different about behavior. Once you're convinced of an effect, ask whether variants of that gene help explain individual differences in human versions of the behavior. I start with the topic that has gotten the most attention, for better or worse, mostly worse. The serotonin system. What do genes related to serotonin have to do with our best and worst behaviors? Plenty. Chapter 2 presented a fairly clear picture of low levels of serotonin fostering impulsive antisocial behavior. There are lower than average levels of serotonin breakdown products in the bloodstreams of people with that profile and of serotonin itself in the frontal cortex of such animals. Even more convincingly, drugs that decrease serotonergic tone, i.e., decreasing serotonin levels or sensitivity to serotonin, increase impulsive aggression. Raising the tone does the opposite. This generates some simple predictions. All of the following should be associated with impulsive aggression, as they will produce low serotonin signaling. A. Low activity variants of the gene for tryptophan hydroxylase. TH, which makes serotonin. B. High activity variants of the gene for monoamine oxidase. MAOA, which degrades serotonin. C. High activity variants of the gene for the serotonin transporter 5-HTT, which removes serotonin from the synapse. D. Variants of genes for serotonin receptors that are less sensitive to serotonin. An extensive literature shows that for each of those genes the results are inconsistent and generally go in the opposite direction, from low serotonin equals aggression, dogma. UG. Studies of genes for TH and serotonin receptors are inconsistent. Messes. 38. In contrast, the picture of 5-HTT, the serotonin transporter gene, is consistently in the opposite direction from what's expected. Two variants exist, with one producing less transporter protein, meaning less serotonin removed from the synapse, and counter to expectations, this variant, producing more serotonin in the synapse, is associated with more impulsive aggression, not less. Thus, according to these findings, high serotonin equals aggression, recognizing this as simplified shorthand. The clearest and most counterintuitive studies concern MAOA. It burst on the scene in a hugely influential 1993 science paper reporting a Dutch family with an MAOA gene mutation that eliminated the protein. 39. Thus serotonin isn't broken down and accumulates in the synapse. And counter to Chapter 2's predictions, the family was characterized by varied antisocial and aggressive behaviors. Mouse studies in which the MAOA gene was knocked out, producing the equivalent of the Dutch family's mutation, produced the same elevated serotonin levels in the synapse and hyperaggressive animals with enhanced fear responses. 40. This finding, of course, concerned a mutation in MAOA resulting in the complete absence of the protein. Research soon focused on low-activity MAOA variants that produced elevated serotonin levels. 41 people with that variant averaged higher levels of aggression and impulsivity and, when looking at angry or fearful faces, more activation of the amygdala and insula and less activation of the prefrontal cortex. This suggests a scenario of more fear reactivity and less frontal capacity to restrain such fear, a perfect storm for reactive aggression. Related studies showed decreased activation of frontal cortical regions during various attentional tasks and enhanced anterior, cingulate activity in response to social rejection in such individuals. So studies where serotonin breakdown products are measured in the body, or where serotonin levels are manipulated with drugs, say that low serotonin equals aggression. 42. And the genetic studies, particularly of MAOA, say high serotonin equals aggression. What explains this discrepancy? The key probably is that a drug manipulation lasts for a few hours or days, while genetic variants have their effects on serotonin for a lifetime. Possible explanations: the low-activity MAOA variants don't produce higher synaptic levels of serotonin all that consistently because the 5-HTT serotonin reuptake pump works harder at removing serotonin from the synapse, compensating, and maybe even overcompensating. There is evidence for this, just to make life really complicated. B. Those variants do produce chronically elevated serotonin levels in the synapse, but the postsynaptic neurons compensate, or overcompensate by decreasing serotonin receptor numbers, thereby reducing sensitivity to all that serotonin. There is evidence for that too. C. The lifelong consequences of differences in serotonin signaling due to gene variants, versus transient differences due to drugs, produce structural changes in the developing brain. There is evidence there as well, and in accordance with that, while temporarily inhibiting MAOA activity with a drug in an adult, rodent decreases impulsive aggression. Doing the same in fetal rodents produces adults with increased impulsive aggression. Yikes, this is complicated. Why go through the agony of all these explanatory twists and turns? Because this obscure corner of neurogenetics has caught the public's fancy, with, I kid you not, the low-activity MAUA variant being referred to as the warrior gene by both scientists and in the media 43, and that warrior who is worsened by the MAUA gene, being X-linked and its variants being more consequential in males than females. Amazingly, prison sentences for murderers have now been lessened in at least 2 cases because it was argued that the criminal, having the warrior gene variant of MAUA, was inevitably fated to be uncontrollably violent. OMG. Responsible people in the field have recoiled in horror at this sort of unfounded genetic determinism seeping into the courtroom. The effects of MAUA variants are tiny. There is non-specificity in the sense that MAUA degrades not only serotonin but norepinephrine as well. Most of all, there is non-specificity in the behavioral effects of the variants. For example, while nearly everyone seems to remember that the landmark MAUA paper that started all the excitement was about aggression, one authoritative review referred to the Dutch family with the mutation as notorious for the persistent and extreme reactive aggression demonstrated by some of its males. In actuality, members of the family with the mutation had borderline mental retardation. Moreover, while some individuals with the mutation were quite violent, the antisocial behavior of others consisted of arson and exhibitionism. So maybe the gene has something to do with the extreme reactive aggression of some family members. But it is just as responsible for explaining why other family members, rather than being aggressive, were flashers. In other words, there is as much rationale for going on about the drop your pants gene as the warrior gene. Probably the biggest reason to reject warrior gene determinism nonsense is something that should be utterly predictable by now. MAUA effects on behavior show strong gene-environment interactions. This brings us to a hugely important 2002 study, one of my favorites, by Avshalom Caspi and colleagues at Duke University. 44. The authors followed a large cohort of children from birth to age 26, studying their genetics, upbringing, and adult behavior. Did MAUA variant status predict antisocial behavior in 26-year-olds as measured by a composite of standard psychological assessments and convictions for violent crimes? No. But MAUA status coupled with something else powerfully did. Having the low-activity version of MAUA tripled the likelihood. But only in people with a history of severe childhood abuse. And if there was no such history, the variant was not predictive of anything. This is the essence of gene-environment interaction. What does having a particular variant of the MAUA gene have to do with antisocial behavior? It depends on the environment. Warrior gene, my ass. This study is important not just for its demonstration of a powerful gene-environment interaction but for what the interaction is, namely the ability of an abusive childhood environment to collaborate with a particular genetic constitution. To quote a major review on the subject, in a healthy environment, increased threat sensitivity, poor emotion control and enhanced fear memory. In MAUA LIE, the warrior variant men might only manifest as variation in temperament within a normal or subclinical range. However, these same characteristics in an abusive childhood environment, one typified by persistent uncertainty, unpredictable threat, poor behavioral modeling and social referencing, and inconsistent reinforcement for prosocial decision-making, might predispose toward frank aggression and impulsive violence in the adult. In a similar vein, the low-activity variant of the serotonin transporter gene was reported to be associated with adult aggressiveness. But only when coupled with childhood adversity. 45. This is straight out of the lessons of the previous chapter. Since then, this MAUA variant childhood abuse interaction has been frequently replicated, and even demonstrated with respect to aggressive behavior in rhesus monkeys. 46. There have also been hints as to how this interaction works. The MAUA gene promoter is regulated by stress and glucocorticoids. MAUA variants show other important gene-environment interactions. For example, in one study the low-activity MAUA variant predicts criminality, but only if coupled with high testosterone levels. Consistent with that, the MAUA gene also has a promoter responsive to androgens. In another study, low-activity MAUA participants in an economic game were more likely than high-activity ones to retaliate aggressively when exploited by the other player, but only if that exploitation produced a large economic loss. If the loss was small, there was no difference. In another study, low-activity individuals were more aggressive than others, but only in circumstances of social exclusion. Thus the effects of this genetic variant can be understood only by considering other, non-genetic factors in individuals' lives, such as childhood adversity and adult provocation. 47. The dopamine system. Chapter 2 introduced the role of dopamine in the anticipation of reward and in goal-directed behavior. Lots of work has examined the genes involved, most broadly showing that variants that produce lower dopamine signaling, less dopamine in the synapse, fewer dopamine receptors, or lower responsiveness of these receptors are associated with sensation-seeking, risk-taking, attentional problems, and extroversion. Such individuals have to seek experiences of greater intensity to compensate for the blunted dopamine signaling. Much of the research has focused on one particular dopamine receptor. There are at least 5 kinds, found in different parts of the brain, binding dopamine with differing strengths and duration, each coded for by a gene. 48. Work has focused on the gene for the D4 dopamine receptor. The gene is called DRD4, which mostly occurs in the neurons in the cortex and nucleus accumbens. The DRD4 gene is super variable, coming in at least 10 different flavors in humans. One stretch of the gene is repeated a variable number of times, and the version with 7 repeats, the 7R form, produces a receptor protein that is sparse in the cortex and relatively unresponsive to dopamine. This is the variant associated with a host of related traits: sensation and novelty-seeking, extroversion, alcoholism, promiscuity, less sensitive parenting, financial risk-taking, impulsivity, and, probably most consistently, ADHD, attention deficit/hyperactivity disorder. The implications cut both ways. The 7R could make you more likely to impulsively steal the old lady's kidney dialysis machine, or to impulsively give the deed of your house to a homeless family. Income gene-environment interactions. For example, kids with the 7R variant are less generous than average. But only if they show insecure attachment to their parents. Secure attachment 7Rs show more generosity than average. Thus 7R has something to do with generosity, but its effect is entirely context-dependent. In another study, 7R students expressed the least interest in organizations advocating for social causes, unless they were given a religious prime, in which case they were the most prosocial. One more 7Rs are worse at gratification postponement tasks, but only if they grew up poor. Repeat the mantra, don't ask what a gene does, ask what it does in a particular context. 49. Interestingly, the next chapter considers the extremely varied frequency of the 7R variant in different populations. As we'll see, it tells you a lot about the history of human migration, as well as about differences between collectivist and individualist cultures. 50. We shift now to other parts of the dopamine system. As introduced in Chapter 2, after dopamine binds to receptors, it floats off and must be removed from the synapse. 51. One route involves its being degraded by the enzyme catechylomethyltransferase, COMT. Among the variants of the COMT gene is one associated with a more efficient enzyme. More efficient equals better at degrading dopamine equals less dopamine in the synapse equals less dopamine signaling. The highly efficient COMT variant is associated with higher rates of extroversion, aggression, criminality, and conduct disorder. Moreover, in a gene-environment interaction straight out of the Mao playbook, that COMT variant is associated with anger traits, but only when coupled with childhood sexual abuse. Intriguingly, the variants seem pertinent to frontal regulation of behavior and cognition, especially during stress. In addition to degradation, neurotransmitters can be removed from the synapse by being taken back up into the axon terminal for recycling. 52. Dopamine reuptake is accomplished by the dopamine transporter, DAT. Naturally, the DAT gene comes in different variants, and those that produce higher levels of synaptic dopamine, i.e., transporter variants that are less efficient, in the striatum are associated with people who are more oriented towards social signaling, they're drawn more than average to happy faces, are more repelled by angry faces, and have more positive parenting styles. How these findings merge with the findings from the DRD4 and COMT studies, i.e., fitting risk-taking with a preference for happy faces, is not immediately apparent. Cool people with certain versions of these dopamine-related genes are more likely to engage in all sorts of interesting behaviors, ranging from the healthy to the pathological. But not so fast. These findings are not consistent, no doubt reflecting unrecognized gene-environment interactions. Again, why should the COMT world be related to sensation-seeking, while there are the DAT people and their happy faces? Both genes are about ending dopamine signaling. This is probably related to different parts of the brain differing as to whether DAT or COMT plays a bigger role. 53. The COMT literature is majorly messy, for the inconvenient reason that the enzyme also degrades norepinephrine. So COMT variants are pertinent to two totally different neurotransmitter systems. These effects are tiny. For example, knowing which DRD4 variant someone has explains only 3 to 4% of the variation in novelty-seeking behavior. The final piece of confusion seems most important but is least considered in the literature, probably because it would be premature. Suppose that every study shows with whopping clarity and consistency that a DRD4 variant is highly predictive of novelty seeking. That still doesn't tell us why for some people novelty seeking means frequently switching their openings in chess games, while for others it means looking for a new locale, because it's getting stale being a mercenary in the Congo. No gene or handful of genes that we are aware of will tell us much about that. The neuropeptides oxytocin and vasopressin. Time for a quick recap from Chapter 4. Oxytocin and vasopressin are involved in prosociality, ranging from parent-offspring bonds to monogamous bonds to trust, empathy, generosity, and social intelligence. Recall the caveats of sometimes these neuropeptides are more about sociality than prosociality, in other words, boosting social information gathering rather than acting prosocially with that information. B. They most consistently boost prosociality in people who already lean in that direction, e.g., making generous people more generous while having no effect on ungenerous people. And C. The prosocial effects are within groups, and these neuropeptides can make people crappier to outsiders, more xenophobic, and preemptively aggressive. Chapter 4 also touched on oxytocin and vasopressin genetics, showing that individuals with genetic variants that result in higher levels of either the hormones or their receptors tend toward more stable monogamous relationships, more actively engaged parenting, better skill at perspective taking, more empathy, and stronger fusiform cortex responses to faces. These are fairly consistent effects of moderate magnitude. Meanwhile, there are studies showing that one oxytocin receptor gene variant is associated with extreme aggression in kids, as well as a callous, unemotional style that foreshadows adult psychopathy. 54. Moreover, another variant is associated with social disconnection in kids and unstable adult relationships. But unfortunately these findings are uninterpretable, because no one knows if these variants produce more, less, or the usual amount of oxytocin signaling. Of course, there are cool gene-environment interactions. For example, having a particular oxytocin receptor gene variant predicts less sensitive mothering, but only when coupled with childhood adversity. Another variant is associated with aggression, but only when people have been drinking. Yet another variant is associated with greater seeking of emotional support during times of stress, among Americans, including first-generation Korean Americans, but not Koreans. Stay tuned for more in the next chapter. Genes related to steroid hormones. We start with testosterone. The hormone is not a protein. None of the steroid hormones are, meaning there isn't a testosterone gene. However, there are genes for the enzymes that construct testosterone, for the enzyme that converts it to estrogen, and for the testosterone androgen receptor. The most work has focused on the gene for the receptor, which comes in variants that differ in their responsiveness to testosterone. Intriguingly, a few studies have shown that among criminals, having the more potent variant is associated with violent crimes. 55. A related finding concerns sex differences in structure of the cortex, and adolescent boys with the more potent variant show more dramatic masculinization of the cortex. An interaction between receptor variant and testosterone levels occurs. High basal testosterone levels do not predict elevated levels of aggressive mood or of amygdaloid reactivity to threatening faces in males, except in those with that variant. Interestingly, the equivalent variant predicts aggressiveness in Akita dogs. How important are these findings? A key theme in Chapter 4 was how little individual differences in testosterone levels in the normal range predict individual differences in behavior. How much more predictability is there when combining knowledge of testosterone levels and of receptor sensitivity? Not much. How about hormone levels and receptor sensitivity and number of receptors? Still not much. But definitely an improvement in predictive power. Similar themes concern the genetics of the estrogen receptor. 56. For example, Different receptor variants are associated with higher rates of anxiety among women, but not men, and higher rates of antisocial behavior and conduct disorder in men, but not women. Meanwhile, in genetically manipulated mice, the presence or absence of the receptor gene influences aggression in females. Depending on how many brothers there were in the litter in utero, gene/environment again. Once again, the magnitude of these genetic influences is tiny. Finally, there is work on genes related to glucocorticoids, particularly regarding gene-environment interactions. 57. For example, there is an interaction between one variant of the gene for a type of receptor for glucocorticoids, for mavins, it's the MR receptor, and childhood abuse in producing an amygdala that is hyperreactive to threat. Then there is a protein called FKBP5, which modifies the activity of another type of receptor for glucocorticoids, the GR receptor. One FKBP5 variant is associated with aggression, hostility, PTSD, and hyperreactivity of the amygdala to threat, but only when coupled with childhood abuse. Buoyed by these findings, some researchers have examined two candidate genes simultaneously. For example, having both risk variants of 5-HTT and DRD4 synergistically increases the risk of disruptive behavior in kids, an effect exacerbated by low socioeconomic status. 58. Phew, all these pages and we've only gotten to thinking about two genes and one environmental variable simultaneously. And despite this, things still aren't great. The usual results aren't terribly consistent from one study to the next. The usual effect sizes are small. Knowing what variant of a candidate gene someone has, or even what variants of a collection of genes, doesn't help much in predicting their behavior. A major reason is that, after getting a handle on 5-HTT and DRD4 interactions, there are still roughly 19,998 more human genes and a gazillion more environments to study. Time to switch to the other main approach, looking at all those 20,000 genes at once. Fishing expeditions, instead of looking where the light is. The small effect sizes reflect a limitation in the candidate gene approach. In scientific lingo, the problem is that one is only looking where the light is. The cliché harks back to a joke: you discover someone at night, searching the ground under a street lamp. What's wrong? I dropped my ring, I'm looking for it. Trying to be helpful, you ask, were you standing on this side or that side of the lamp when you dropped it? Oh, no, I was over by those trees when I dropped it. Then why are you searching here? This is where the light is. With candidate gene approaches, you look only where the light is, examine only genes that you already know are involved. And with 20,000 or so genes, it's pretty safe to assume there are still some interesting genes that you don't know about yet. The challenge is to find them. The most common way of trying to find them all is with genome-wide association studies. 59. Examine, say, the gene for hemoglobin and look at the 11th nucleotide in the sequence. Everyone will pretty much have the same DNA letter in that spot. However, there are little hot spots of variability, single nucleotides where, say, two different DNA letters occur each in about 50% of the population, and where this typically doesn't change the amino acid being specified, because of DNA redundancy. There are more than 1 million of such SNP single nucleotide polymorphisms, scattered throughout the genome in stretches of DNA coding for genes, for promoters, for mysterious DNA junk. Collect DNA from a huge number of people, and examine whether particular SNPs associate with particular traits. If an SNP that's implicated occurs in a gene, you've just gotten a hint that the gene may be involved in that trait. A GWAS study might implicate scads of genes as being associated with a trait. Hopefully, some will be candidate genes already known to be related to the trait. But other identified genes may be mysterious. Now go check out what they do. In a related approach, suppose you have two populations, one with and one without a degenerative muscle disease. Take a muscle biopsy from everyone, and see which of the approximately 20,000 genes are transcriptionally active in the muscle cells. With this microarray or gene chip approach, you look for genes that are transcriptionally active only in diseased or in healthy muscle, not in both. Identify them, and you have some new candidate genes to explore. These fishing expeditions show why we're so ignorant about the genetics of behavior. 60. Consider a classic GWAS that looked for genes related to height. This was a crazy difficult study, involving examining the genomes of 183,727 people. 183,727. It must have taken an army of scientists just to label the test tubes. And reflecting that, the paper reporting the findings in Nature had approximately 280 authors. And the results? Hundreds of genetic variants were implicated in regulating height. A handful of genes identified were known to be involved in skeletal growth, but the rest was terra incognita. The single genetic variant identified that most powerfully predicted height explained all of 0.4%, 0.4% of the variation in height, and all those hundreds of variants put together explained only about 10% of the variation. Meanwhile, an equally acclaimed study was regarding body mass index, BMI. Similar amazingness, almost 250,000 genomes examined, even more authors than the height study. And in this case the single most explanatory, genetic variant identified accounted for only 0.3% of the variation in BMI. Thus both height and BMI are highly polygenic traits. Same for age of menarche, when girls menstruate for the first time. Moreover, additional genes are being missed because their variants are too rare to be picked up by current GWAS techniques. Thus these traits are probably influenced by hundreds of genes. 61. What about behavior? A superb 2013 GWAS study examined the genetic variants associated with educational attainment. 62. The usual over-the-top numbers: 126,559 study subjects, about 180 authors. And the most predictive genetic variant accounted for 0.02%, 0.02% of the variation. All the identified variants together accounted for about 2% of the variation. A commentary accompanying the paper contained this landmark of understatement. In short, educational attainment looks to be a very polygenic trait. Educational attainment, how many years of high school or college one completes, is relatively easy to measure. How about the subtler, messier behaviors that fill this book's pages? A handful of studies have tackled that, and the findings are much the same. At the end, you have a list of scores of genes implicated and can then go figure out what they do, logically, starting with the ones that showed the strongest statistical associations. Hard approaches that are still in their infancy. Made worse by GWAS missing more, subtly variable spots, meaning even more genes are likely involved. 63. As we conclude this section, some key points. 64. A. This review of candidate genes barely scratches even the surface of the surface. Go on PubMed, a major search engine of the biomedical literature, and search MAO. Gene/behavior, up come more than 500 research papers. Serotonin transporter gene/behavior, 1,250 papers. Dopamine receptor gene/behavior, nearly 2,000. B. The candidate gene approaches show that the effect of a single gene on a behavior is typically tiny. In other words, having the warrior gene variant of MAO probably has less effect on your behavior than does believing that you have it. C. Genome-wide survey approaches show that these behaviors are influenced by huge numbers of genes, each one playing only a tiny role. D. What this translates into is non-specificity. For example, serotonin transporter gene variants have been linked to risk of depression, but also anxiety, obsessive-compulsive disorder, schizophrenia, bipolar disorder, Tourette's syndrome, and borderline personality disorder. In other words, that gene is part of a network of hundreds of genes pertinent to depression, but also part of another, equally large and partially overlapping network relevant to anxiety, another relevant to OCD, and so on. And meanwhile, we're plugging away, trying to understand interactions of two genes at a time. E. And, of course, gene and environment. A. Conclusions. Long last, you and I have gotten to the end of this excruciatingly, but necessarily long chapter. Amid all these tiny effects and technical limitations, it's important to not throw out the genetic baby with the bathwater, as has been an agitated sociopolitical goal at times during my intellectual youth in the 1970s, sandwiched between the geologic periods of cranberry bell-bottoms and of John Travolta white suits was the genes have nothing to do with behavior ice age. Genes have plenty to do with behavior. Even more appropriately, all behavioral traits are affected to some degree by genetic variability. 65. They have to be, given that they specify the structure of all the proteins pertinent to every neurotransmitter, hormone, receptor, etc. that there is. And they have plenty to do with individual differences in behavior, given the large percentage of genes that are polymorphic, coming in different flavors. But their effects are supremely context-dependent. Ask not what a gene does. Ask what it does in a particular environment and when expressed in a particular network of other genes, i.e., gene/gene/gene/environment. Thus, for our purposes, genes aren't about inevitability. Instead, they're about context-dependent tendencies, propensities, potentials, and vulnerabilities. All embedded in the fabric of the other factors, biological and otherwise, that fill these pages. Now that this chapter's done, why don't we all take a bathroom break and then see what's in the refrigerator? 9. Centuries to millennia before. Let's start with a seeming digression. Parts of chapters 4 and 7 have debunked some supposed sex differences concerning the brain, hormones, and behavior. One difference, however, is persistent. It's far from issues that concern this book, but bear with me. A remarkably consistent finding, starting with elementary school students, is that males are better at math than females. While the difference is minor when it comes to considering average scores, there is a huge difference when it comes to math stars at the upper extreme of the distribution. For example, in 1983, for every girl scoring in the highest percentile on the math SAT, there were 11 boys. Why the difference? There have always been suggestions that testosterone is central. During development, testosterone fuels the growth of a brain region involved in mathematical thinking, and giving adults testosterone enhances some math skills. Oh, okay, it's biological. But consider a paper published in Science in 2008. When the authors examined the relationship between math scores and sexual equality in 40 countries, based on economic, educational, and political indices of gender equality. The worst was Turkey, the United States was middling, and, naturally, the Scandinavians were tops. Lo and behold, the more gender equal the country, the less of a discrepancy in math scores. By the time you get to the Scandinavian countries, it's statistically insignificant. And by the time you examine the most gender-equal country on Earth at the time, Iceland, girls are better at math than boys. El Guizzo et al., Culture, Gender, and Math, Sci 322,008, 1164. Visit bit.ly/2088s40 for a larger version of this graph. In other words, while you can never be certain, the Afghan girl pictured on top on the next page, seated next to her husband, is less likely than the Swedish girl pictured below her to solve the Erdos-Hagenel conjecture in graph theory. In other words, culture matters. We carry it with us wherever we go. As one example, the level of corruption, a government's lack of transparency, regarding use of power and finances in undiplomats' home countries, predicts their likelihood of racking up unpaid parking tickets in Manhattan. Culture leaves long-lasting residues. Shiites and Sunnis slaughter each other over a succession issue, 14 centuries old, across 33 countries. Population density, in the year 1500 significantly predicts how authoritarian the government was in 2000, over the course of millennia. Earlier adoption of the hoe over the plow predicts gender equality today. 2. And in other words, when we contemplate our iconic acts, the pulling of a trigger, the touching of an arm, and want to explain why they happened using a biological framework, culture better be on our list of explanatory factors. Thus, the goals of this chapter. Look at systematic patterns of cultural variation as they pertain to the best and worst of our behaviors. Explore how different types of brains produce different culture and different types of culture produce different brains. In other words, how culture and biology co-evolve. 3. See the role of ecology in shaping culture. C. Definitions, similarities, and differences. Culture, naturally, has been defined various ways. One influential definition comes from Edward Tyler, a distinguished 19th-century cultural anthropologist. For him, culture is that complex whole which includes knowledge, belief, art, morals, law, custom, and any other capabilities and habits acquired by man as a member of society. 4. This definition obviously is oriented towards something that is specific to humans. Jane Goodall blew off everyone's socks in the 1960s by reporting the now iconic fact that chimps make tools. Her study subjects modify twigs by stripping off the leaves and pushing them into termite mounds. Termites would bite the twig, still holding on when it was pulled out, yielding a snack for the chimps. This was just the start. Chimps were subsequently found to use various tools, wood or rock anvils for cracking open nuts, wads of chewed leaves to sponge up hard-to-reach water, and, in a real shocker, sharpened sticks for spearing bush babies. 5. Different populations make different tools. New techniques spread across social networks. Among chimps who hang with one another, kids learn the ropes by watching their moms. Techniques spread from one group to another. When someone emigrates, chimp nut-cracking tools in excess of 4,000 years old have been excavated. And in my favorite example, floating between tool use and accessorizing, a female in Zambia got it into her head to go around with a straw-like blade of grass in her ear. The action had no obvious function. Apparently she just liked having a piece of grass sticking out of her ear. So sue her. She did it for years, and over that time the practice spread throughout her group. A fashionista. In the decades since Gödel's discovery, tool use has been observed in apes and monkeys, elephants, sea otters, mongoose. 6. Dolphins use sea sponges to dig up fish burrowed into the sea floor. Birds use tools for nest building or food acquisition. Jays and crows, for example, use twigs to fish for insects, much as chimps do. And there's tool use in cephalopods, reptiles, and fish. E. van Leeuwen et al., a group-specific arbitrary tradition in chimpanzees, Pantroglodytes, Annal Cog 17 2014, 1421. All this is mighty impressive. Nonetheless, such cultural transmission doesn't show progression. This year's chimp nut cracking tool is pretty much the same as that of 4,000 years ago. And with few exceptions, more later, non-human culture is solely about material culture versus, say, social organization. So the classical definition of culture isn't specific to humans. 7. Most cultural anthropologists weren't thrilled with Gödel's revolution. Great. Next, the zoologists will report that Rafiki persuaded Simba to become the Lion King, and now often emphasize definitions of culture that cut chimps and other hoi polloi out of the party. There's a fondness for the thinking of Alfred Kreber, Clyde Kluckhohn, and Clifford Geertz, three heavyweight social anthropologists, who focused on how culture is about ideas and symbols, rather than the mere behaviors in which they instantiate, or material products like flint blades or iPhones. Contemporary anthropologists like Richard S. Wetter have emphasized a more effective, but still human-centric view of culture, as being about moral and visceral versions of right and wrong. And of course these views have been critiqued by postmodernists for reasons I can't begin to follow. Basically, I don't want to go anywhere near these debates. For our purposes we'll rely on an intuitive definition of culture that has been emphasized by Frans de Waal. Culture is how we do and think about things, transmitted by non-genetic means. Working with that broad definition is the most striking thing about the array of human cultures. The similarities or the differences depends on your taste. If the similarities seem most interesting, there are plenty after all. Multiple groups of humans independently invented agriculture, writing, pottery, embalming, astronomy, and coinage. At the extreme of similarities are human universals, and numerous scholars have proposed lists of them. One of the lengthiest and most cited comes from the anthropologist Donald Brown. 8. Here's a partial list of his proposed cultural universals: the existence of and concern with aesthetics, magic, males and females seen as having different natures, baby talk, gods, induction of altered states, marriage, body adornment, murder, prohibition of some type of murder, kinship terms, numbers, cooking, private sex, names, dance, play, distinctions between right and wrong, nepotism, prohibitions on certain types of sex, empathy, reciprocity, rituals, concepts of fairness, myths about afterlife. Music, color terms, prohibitions, gossip, binary sex terms, in-group favoritism, language, humor, lying, symbolism, the linguistic concept of int, tools, trade, and toilet training. And that's a partial list. For the purposes of this chapter, the staggeringly large cultural differences in how life is experienced, in resources and privileges available, in opportunities and trajectories, are most interesting. Just to start with some breathtaking demographic statistics born of cultural differences, a girl born in Monaco has a 93-year life expectancy, one in Angola, 39. Latvia has 99.9% literacy, Niger, 19%. More than 10% of children in Afghanistan die in their first year, about 0.2% in Iceland. Per capita GDP is $137,000 in Qatar, $609 in the Central African Republic. A woman in South Sudan is roughly 1,000 times more likely to die in childbirth than a woman in Estonia. 9. The experience of violence also varies enormously by culture. Someone in Honduras is 450 times more likely to be murdered than someone in Singapore. 65% of women experience intimate partner violence in Central Africa, 16% in East Asia. A South African woman is more than 100 times more likely to be raped than one in Japan. Be a school kid in Romania, Bulgaria, or Ukraine, and you're about 10 times more likely to be chronically bullied than a kid in Sweden, Iceland, or Denmark. Stay tuned for a closer look at this. 10. Of course, there are the well-known gender-related cultural differences. There are the Scandinavian countries approaching total gender equality and Rwanda, with 63% of its lower house parliamentary seats filled by women, compared with Saudi Arabia, where women are not allowed outside the house unless accompanied by a male guardian, and Yemen, Qatar, and Tonga, with 0% female legislators, and with the United States running around 20%. 11. Then there's the Philippines, where 93% of people say they feel happy and loved, versus 29% of Armenians. In economic games, people in Greece and Oman are more likely to spend resources to punish overly generous players than to punish those who are cheaters, whereas among Australians such antisocial punishment is non-existent. And there are wildly different criteria for prosocial behavior. In a study of employees throughout the world working for the same multinational bank, what was the most important reason cited to help someone? Population size, population density, population heterogeneity. The year 2008 marked a human milestone, a transition point 9,000 years in the making. For the first time, the majority of humans lived in cities. The human trajectory from semi-permanent settlements to the megalopolis has been beneficial. In the developed world, when compared with rural populations, city dwellers are typically healthier and wealthier. Larger social networks facilitate innovation. Because of economies of scale, cities leave a smaller per capita ecological footprint. 42. Urban living makes for a different sort of brain. This was shown in a 2011 study of subjects from a range of cities, towns, and rural settings who underwent an experimental social stressor while being brain scanned. The key finding was that the larger the population where someone lived, the more reactive their amygdala was during that stressor. 43. Most important for our purposes, urbanized humans do something completely unprecedented among primates, regularly encountering strangers who are never seen again, fostering the invention of the Anonymous Act. After all, it wasn't until 19th century urbanization that crime fiction was invented, typically set in cities. In traditional settings there's no whodunit, since everyone knows what everyone done. Growing cultures had to invent mechanisms for norm enforcement among strangers. For example, across numerous traditional cultures, the larger the group, the greater the punishment for norm violations, and the more cultural emphases on equitable treatment of strangers. Moreover, larger groups evolved third-party punishment. Stay tuned for more in the next chapter. Rather than victims punishing norm violators, punishment is meted out by objective third parties such as police and courts. At an extreme, a crime not only victimizes its victim, but also is an affront to the collective population. Hence the people versus Joe Blow. 44. Finally, life in larger populations fosters the ultimate third-party punisher. As documented by Ara Norenzian of the University of British Columbia, it is only when societies grow large enough that people regularly encounter strangers that big gods emerge, deities who are concerned with human morality and punish our transgressions. 45. Societies with frequent anonymous interactions tend to outsource punishment to gods. In contrast, hunter-gatherers' gods are less likely than chance to care whether we've been naughty or nice. Moreover, in further work across a range of traditional cultures, Norenzian has shown that the more informed and punitive people consider their moralistic gods to be, the more generous they are to correlationist strangers in a financial allocation game. Separate from the size of a population. How about its density? One study surveying 33 developed countries characterized each nation's tightness, the extent to which the government is autocratic, dissent suppressed, behavior monitored, transgressions punished, life regulated by religious orthodoxy, citizens viewing various behaviors as inappropriate, e.g., singing in an elevator, cursing at a job interview. 46. Higher population density predicted tighter cultures, both high density in the present and, remarkably, historically, in the year 1500. The issue of population density's effects on behavior gave rise to a well-known phenomenon, mostly well-known incorrectly. In the 1950s, John Calhoun at the National Institute of Mental Health asked what happens to rat behavior at higher population densities, research prompted by America's ever-growing cities. 47. And in papers for both scientists and the lay public, Calhoun gave a clear answer: high-density living produced deviant behavior and social pathology. Rats became violent, adults killed and cannibalized one another, females were aggressive to their infants, there was indiscriminate hypersexuality among males, e.g., trying to mate with females who weren't in estrus. The writing about the subject, starting with Calhoun, was colorful. The bloodless description of high-density living was replaced with crowding. Aggressive males were described as going berserk, aggressive females as Amazons. Rats living in these rat slums became social dropouts, autistic, or juvenile delinquents. One expert on rat behavior, A.S. Parks, described Calhoun's rats as unmaternal mothers, homosexuals and zombies, quite the trio you'd invite to dinner in the 1950s. 48. The work was hugely influential, taught to psychologists, architects, and urban planners. More than 1 million reprints were requested of Calhoun's original Scientific American report. Sociologists, journalists, and politicians explicitly compared residents of particular housing projects and Calhoun's rats. The take-home message sent ripples through the American heartland destined for the chaotic 60s. Inner cities breed violence, pathology, and social deviance. Calhoun's rats were more complicated than this, something underemphasized in his lay writing. High-density living doesn't make rats more aggressive. Instead, it makes aggressive rats more aggressive. This echoes the findings that neither testosterone, nor alcohol, nor media violence uniformly increases violence. Instead, they make violent individuals more sensitive to violence-evoking social cues. In contrast, crowding makes unaggressive individuals more timid. In other words, it exaggerates pre-existing social tendencies. Calhoun's erroneous conclusions about rats don't even hold for humans. In some cities, Chicago, for example, circa 1970, higher population density in neighborhoods does indeed predict more violence. Nevertheless, some of the highest density places on Earth, Hong Kong, Singapore, and Tokyo, have minuscule rates of violence. High-density living is not synonymous with aggression in rats or humans. The preceding sections examined the effects of living with lots of people, and in close quarters. How about the effects of living with different kinds of people? Diversity. Heterogeneity. Admixture. Mosaicism. Two opposite narratives come to mind. Mr. Rogers' neighborhood: when people of differing ethnicities, races, or religions live together, they experience the similarities rather than the differences and view one another as individuals, transcending stereotypes. Trade flows, fostering fairness and mutuality. Inevitably, dichotomies dissolve with intermarriage, and soon you're happily watching your grandkid in the school play on their side of town. Just visualize world peace. Sharks versus the Jets: differing sorts of people living in close proximity rub, and thus abrade, elbows regularly. One side's act of proud cultural identification feels like a hostile dig to the other side. Public spaces become proving grounds for turf battles, commons become tragedies. Surprise, both outcomes occur. The final chapter explores circumstances where intergroup contact leads to one rather than the other. Most interesting at this juncture is the importance of the spatial qualities of the heterogeneity. Consider a region filled with people from Albania and Kyrgyzstan, two hostile groups each providing half the population. At one extreme, the land is split down the middle, each group occupying one side, producing a single boundary between the two. At the other extreme is a microcheckerboard of alternating ethnicities, where each square on the grid is one person large, producing a vast quantity of boundaries between Albanians and Kyrgyz. Intuitively, both scenarios should bias against conflict. In the condition of maximal separation, each group has a critical mass to be locally sovereign, and the total length of border, and thus of the amount of intergroup elbow rubbing, is minimized. In the scenario of maximal mixing, no patch of ethnic homogeneity is big enough to foster a self-identity that can dominate a public space. Big deal if someone raises a flag between their feet and declares their square meter to be the Albanian Empire or a Kyrgyzstani Republic. But in the real world things are always in between the two extremes, and with variation in the average size of each ethnic patch. Does patch size and thus amount of border influence relations? This was explored in a fascinating paper from the aptly named New England Complex Systems Institute down the block from MIT 49. The authors first constructed an Elbonian-Kerplocki mixture with individuals randomly distributed as pixels on a grid. Pixels were given a certain degree of mobility, plus a tendency to assort with other pixels of the same type. As self-assortment progresses, something emerges: islands and peninsulas of Elbonians amid a sea of Kerplockus, or the reverse, condition that intuitively seems rife with potential intergroup violence. As self-assortment continues, the number of such isolated islands and peninsulas declines. The intermediate stage that maximizes the number of islands and peninsulas maximizes the number of people living within a surrounded enclave. The authors then considered a Balkanized region, namely the Balkans, ex-Yugoslavia, in 1990. This was just before Serbians, Bosnians, Croatians, and Albanians commenced Europe's worst war since World War II, the war that taught us the names of places like Srebrenica and people like Slobodan Milosevic. Using a similar analysis, with ethnic island size varying from roughly 20 to 60 kilometers in diameter, they identified the spots, theoretically most rife for violence. Remarkably, this predicted the sites of major fighting and massacres in the war. In the words of the authors, violence can arise due to the structure of boundaries between groups, rather than as a result of inherent conflicts between the groups themselves. They then showed that the clarity of borders matters as well. Good, clear-cut fences, e.g., mountain ranges or rivers between groups, make for good neighbors. Peace does not depend on integrated coexistence, but rather on well-defined, topographical and political boundaries separating groups, allowing for partial autonomy within a single country, the authors concluded. Thus, not only do size, density, and heterogeneity of populations help explain intergroup violence, but patterns and clarity of fragmentation do as well. These issues will be revisited in the final chapter. The residues of cultural crises. In times of crisis, the London Blitz, New York after 9/11, San Francisco after the 1989 Loma Prieta earthquake, people pull together, that's cool. But in contrast, chronic, pervasive, corrosive menace doesn't necessarily do the same to people or cultures. The primal menace of hunger has left historical marks. Back to that study of differences between countries' tightness, where tight countries were characterized by autocracy, suppression of dissent, and omnipresence and enforcement of behavior norms. 50. What sorts of countries are tighter? In addition to the high population density, correlates mentioned earlier, there are also more historical food shortages, lower food intake, and lower levels of protein and fat in the diet. In other words, these are cultures chronically menaced by empty stomachs. Cultural tightness was also predicted by environmental degradation, less available farmland or clean water, more pollution. Similarly, habitat degradation and depletion of animal populations worsens conflict in cultures dependent on bushmeat. And a major theme of Jared Diamond's Magisterial Collapse, How Societies Choose to Fail or Succeed, is how environmental degradation explains the violent collapse of many civilizations. Then there's disease. In Chapter 15 we'll touch on behavioral immunity, the ability of numerous species to detect cues of illness in other individuals. As we'll see, implicit cues about infectious disease make people more xenophobic. Similarly, historical prevalence of infectious disease predicts a culture's openness to outsiders. Moreover, other predictors of cultural tightness include having high historical incidence of pandemics, of high infant and child mortality rates, and of higher cumulative average number of years lost to communicable disease. Obviously, weather influences the incidence of organized violence. Consider the centuries of European wars taking a hiatus during the worst of winter and the growing season. 51. Even broader is the capacity of weather and climate to shape culture. The Kenyan historian Ali Mazrui has suggested that one reason for Europe's historical success, relative to Africa, has been the weather. Western-style planning ahead arose from the annual reality of winter coming. Larger-scale changes in weather are known to be consequential. In the tightness study, cultural tightness was also predicted by a history of floods, droughts, and cyclones. Another pertinent aspect of weather concerns the southern oscillation, known as El Niño, the multi fluctuation of average water temperatures in the equatorial Pacific Ocean. El Niños, occurring about every dozen years, involve warmer, drier weather, with the opposite during La Niña years, and are associated in many developing countries with droughts and food shortages. Over the last 50 years, El Niños have roughly doubled the likelihood of civil conflict, mostly by stoking the fires of pre-existing conflicts. The relationship between drought and violence is tricky. The civil conflict referred to in the previous paragraph concerned deaths caused by battle between governmental and non-governmental forces, i.e., civil wars or insurgencies. Thus, rather than fighting over a watering hole or a field for grazing, this was fighting for modern perks of power. But in traditional settings, drought may mean spending more time foraging or hauling water for your crops. Raiding to steal the other group's women isn't a high priority, and why rustle someone else's cows when you can't even feed your own? Conflict declines. Interestingly, something similar occurs in baboons. Normally, baboons in rich ecosystems like the Serengeti need forage only a few hours a day. Part of what endears baboons to primatologists is that this leaves them about 9 hours daily to devote to social machinations, trysting and jousting and backbiting. In 1984 there was a devastating drought in East Africa. Among baboons, while there was still sufficient food, it took every waking moment to get enough calories. Aggression decreased 0.52. So ecological duress can increase or decrease aggression. This raises the key issue of what global warming will do to our best and worst behaviors. There will definitely be some upsides. Some regions will have longer growing seasons, increasing the food supply and reducing tensions. Some people will eschew conflict, being preoccupied with saving their homes from the encroaching ocean or growing pineapples in the Arctic. But amid squabbling about the details in predictive models, the consensus is that global warming won't do good things to global conflict. For starters, warmer temperatures rile people up. In cities during the summers, for every 3-degree increase in temperature, there was a 4% increase in interpersonal violence and 14% in group violence. But global warming's bad news is more global: desertification, loss of arable land due to rising seas, more droughts. One influential meta-analysis projected 16% and 50% increases in interpersonal and group violence, respectively, in some regions by 2050.53. Oh, why not religion? For a quick hit and run about religion before considering it in the final chapter. Theories abound as to why humans keep inventing religions. It's more than a human pull toward the supernatural. As stated in one review, Mickey Mouse has supernatural powers, but no one worships or would fight, or kill, for him. Our social brains may help explain why children, the world over, are attracted to talking teacups, but religion is much more than that. Why does religion arise? Because it makes in groups more cooperative and viable. Stay tuned for more in the next chapter. Because humans need personification, and to see agency and causality when facing the unknown. Or maybe inventing deities is an emergent byproduct of the architecture of our social brains. 54. Amid these speculations, far more boggling is the variety of the thousands of religions we've invented. They vary as to number and gender of deities, whether there's an afterlife, what it's like and what it takes to enter, whether deities judge or interfere with humans, whether we are born sinful or pure, and whether sexuality changes those states, whether the myth of a religion's founder is of sacredness from the start, so much so that, say, wise men visit the infant founder, or of a Sybarite who reforms, e.g., Siddhartha's transition from palace life to being the Buddha, whether the religion's goal is attracting new followers, say. With exciting news, e.g., an angel visited me in Manchester, New York, and gave me golden plates, or retaining members, we've got a covenant with God, so stick with us. On and on. There are some pertinent patterns amid this variation. As noted, desert cultures are prone toward monotheistic religions, rainforest dwellers, polytheistic ones. Nomadic pastoralists' deities tend to value war and valor in battle as an entree to a good afterlife. Agriculturalists invent gods who alter the weather. As noted, once cultures get large enough that anonymous acts are possible, they start inventing moralizing gods. Gods and religious orthodoxy dominate more in cultures with frequent threats, war, natural disasters, inequality, and high infant mortality rates. Before turfing this subject to the final chapter, 3 obvious points. A religion reflects the values of the culture that invented or adopted it and very effectively transmits those values. B. Religion fosters the best and worst of our behaviors. C. It's complicated. We've now looked at various cultural factors, collectivism versus individualism, egalitarian versus hierarchical distribution of resources, and so on. While there are others to consider, it's time to shift to the chapter's final topic. This is one that has generated shitstorms of debate as old as the weathered layers of Oljavai Gorge and as fresh as a newborn baby's tush, a topic that has scientists who study peace at one another's throats. Why? Hobbes or Rousseau? Those guys. To invoke some estimates, anatomically modern humans emerged about 200,000 years ago, and behaviorally modern ones about 40,000 to 50,000 years ago. Animal domestication is 10,000 to 20,000 years old, agriculture around 12,000. After plant domestication, it was roughly 5,000 more years until history began with civilizations in Egypt, the Mideast, China, and the New World. When in this arc of history was war invented? Does material culture lessen or worsen tendencies toward war? Do successful warriors leave more copies of their genes? Has the centralization of authority by civilization actually civilized us, providing a veneer of socially contractual restraint? Have humans become more or less decent to one another over the course of history? Yes, it's short/nasty/brutish versus noble savage. In contrast to the centuries of food fights among philosophers, contemporary Hobbes versus Rousseau is about actual data. Some of it is archaeological, where researchers have sought to determine the prevalence and antiquity of warfare from the archaeological record. Predictably, half of each conference on the subject consists of definitional squabbles. Is war solely organized and sustained violence between groups? Does it require weapons? A standing army, even if only seasonally? An army with hierarchy and chain of command? If fighting is mostly along lines of relatedness, is it a vendetta or clan feud instead of a war? Fractured bones. For most archaeologists, the operational definition has been streamlined to numerous people, simultaneously meeting violent deaths. In 1996, the archaeologist Lawrence Keeley of the University of Illinois synthesized the existing literature in his highly influential War Before Civilization, The Myth of the Peaceful Savage, ostensibly showing that the archaeological evidence for war is broad and ancient. 55. A similar conclusion comes in the 2011 book, The Better Angels of Our Nature, Why Violence Has Declined, by Harvard's Steven Pinker. 56. Cliché police be damned. You can't mention this book without calling it monumental. In this monumental work, Pinker argued that violence and the worst horrors of inhumanity have been declining for the last half-millennium, thanks to the constraining forces of civilization, and b. the warfare and barbarity preceding that transition are as old as the human species. Keeley and Pinker document savagery galore in prehistoric tribal societies, mass graves filled with skeletons, bearing multiple fractures, caved-in skulls, parrying fractures, which arise from raising your arm to fend off a blow, stone projectiles embedded in bone. Some sites suggest the outcome of battle, a preponderance of young adult male skeletons. Others suggest indiscriminate massacre, butchered skeletons of both sexes and all ages. Other sites suggest cannibalism of the vanquished. In their separate surveys of the literature, Keeley and Pinker present evidence of pre-state, tribal violence comes from sites in Ukraine, France, Sweden, Niger, India and numerous pre-contact American locations. 57. This collection of sites includes the oldest such massacre, the 12,000 to 14,000-year-old Jebel Sahaba site along the Nile in northern Sudan, a cemetery of 59 men, women and children, nearly half of whom have stone projectiles embedded in their bones. And it includes the largest massacre site, the 700-year-old Crow Creek in South Dakota, a mass grave of more than 400 skeletons, with 60% showing evidence of violent deaths. Across the 21 sites surveyed, about 15% of skeletons showed evidence of death in warfare. One can, of course, be killed in war in a way that doesn't leave fractures, or projectiles embedded in bone, suggesting that the percentage of deaths due to warfare was higher. Otzi, in his current state, left, and in an artist's reconstruction, right. Note, his killer, still at large, probably looked pretty much the same. Keeley and Pinker also document how prehistoric settlements frequently protected themselves with defensive palisades and fortifications. And, of course, as the poster child for prehistoric violence, there is Otzi, the 5,300-year-old Tyrolean iceman found in a melting glacier in 1991 on the Italian-Austrian border. In his shoulder was a freshly embedded arrowhead. Thus, Keeley and Pinker document mass casualties of warfare long predating civilization. Just as important, both, starting with Keeley's subtitle, suggest a hidden agenda among archaeologists to ignore that evidence. Why had there been, to use Keeley's phrase, pacification of the past? In Chapter 7 we saw how World War II produced a generation of social scientists, trying to understand the roots of fascism. In Keeley's view, the post-World War II generations of archaeologists recoiled from the trauma of the war by recoiling from the evidence that humans had been prepping a long time for World War II. For Pinker, writing from a younger generation's perspective, the current whitewashing of prehistoric violence has the flavor of today's archaeological graybeards being nostalgic about getting stoned in high school and listening to John Lennon's Imagine. Keeley and Pinker generated a strong backlash among many notable archaeologists, who charged them with war-ifying the past. Most vocal has been R. Brian Ferguson of Rutgers University, with publications with titles like Pinker's List, exaggerating prehistoric war mortality. Keeley and Pinker are criticized for numerous reasons. 5-8. A. Some of the sites supposedly presenting evidence for warfare actually contain only a single case of violent death, suggesting homicide, not war. B. The criteria for inferring violent death include skeletons in close proximity to arrowheads. However, many such artifacts were actually tools used for other purposes, or simply chips and flakes. For example, Fred Wendorf, who excavated Jebel Sahaba, considered most of the projectiles associated with skeletons to have been mere debris. 59. C. Many fractured bones were actually healed. Instead of reflecting war, they might indicate the ritualized club fighting scene in many tribal societies. D. Proving that a human bone was gnawed on by a fellow human instead of another carnivore is tough. One tour de force paper demonstrated cannibalism in a Pueblo village from around the year 1100. Human feces there contained the human version of the muscle-specific protein myoglobin. 60. In other words, those humans had been eating human meat. Nonetheless, even when cannibalism is clearly documented, it doesn't indicate whether there was exer. Endocannibalism, i.e., eating vanquished enemies versus deceased relatives, as is done in some tribal cultures. E. Most important, Keeley and Pinker are accused of cherry-picking their data, discussing only sites of putative war deaths, rather than the entire literature. When you survey the thousands of prehistoric skeletal remains from hundreds of sites worldwide, rates of violent deaths are far lower than 15%. Moreover, there are regions and periods lacking any evidence of warlike violence. The glee in refuting the broadest conclusions of Keeley and Pinker is unmistakable, e.g., Ferguson in the previously cited work, For 10,000 Years in the Southern Levant, there is not one single instance where it can be said with confidence, war was there. His emphasis, am I wrong? Name the place. Thus these critics conclude that wars were rare prior to human civilizations. Supporters of Keeley and Pinker retort that you can't ignore bloodbaths like Crow Creek or Jebel Sahaba, and that the absence of proof of early war in so many of these sites is not proof of absence. This suggests a second strategy for contemporary Hobbes versus Rousseau debates, namely to study contemporary humans in pre-state tribal societies. How frequently do they war? Prehistorians in the flesh. Well, if researchers endlessly argue about who or what gnawed on a 10,000-year-old human bone, imagine the disagreements about actual living humans. Keeley and Pinker, along with Samuel Bowles of the Santa Fe Institute, conclude that warfare is nearly universal in contemporary non-state societies. This is the world of headhunters in New Guinea and Borneo, Maasai and Zulu warriors in Africa, Amazonians on raiding parties in the rainforest. Keeley estimates that, in the absence of pacification enforced by outside forces such as a government, 90 to 95% of tribal societies engage in warfare, many constantly, and a much higher percentage are at war at any time than is the case for state societies. For Keeley, the rare peaceful tribal societies are usually so because they have been defeated and dominated by a neighboring tribe. Keeley charges that there has been systematic underreporting of violence by contemporary anthropologists intent on pacifying living relics of the past. Clockwise, top left, New Guinea, Maasai, Amazonian, Zulu. Keeley also tries to debunk the view that tribal violence is mostly ritualistic, an arrow in someone's thigh, a head or two bashed with a war club, and time to call it a day. Instead, violence in non-state cultures is lethal. Keeley seems to take pride in this, documenting how various cultures use weapons designed for warfare, meant to cause festering damage. He often has an almost testy, offended tone about those pacifying anthropologists who think indigenous groups lack the organization, self-discipline, and Puritan work ethic to inflict bloodbaths. He writes about the superiority of tribal warriors to westernized armies, e.g., describing how in the Anglo-Zulu War, Zulu spears were more accurate than 19th-century British guns, and how the Brits won the war, not because they were superior fighters, but because their logistical sophistication allowed them to fight prolonged wars. Like Healy, Pinker concludes that warfare is nearly ubiquitous in traditional cultures, reporting 10 to 30% of deaths as being war-related in New Guinea tribes, such as the Djibuzi and Mainga, and a 35 to 60% range for Wairani and Hivaro tribes in the Amazon. Pinker estimates rates of death due to violence. Europe currently is in the range of 1 death per 100,000 people per year. During the crime waves of the 1970s and 1980s, the United States approached 10, Detroit was around 45. Germany and Russia, during their 20th century wars, averaged 144 and 135, respectively. In contrast, the 27 non-state societies surveyed by Pinker average 524 deaths. There are the Grand Valley Dani of New Guinea, the Piegan Blackfoot of the American Great Plains, and the Dinka of Sudan, all of whom in their prime approached 1,000 deaths, roughly equivalent to losing one acquaintance per year. Taking the gold are the Kato, a California tribe that in the 1840s crossed the finish line near 1,500 deaths per 100,000 people per year. No tour of violence in indigenous cultures is complete without the Yanomamo, a tribe living in the Brazilian and Venezuelan Amazon. According to Dogma, there is almost always raiding between villages. 30% of adult male deaths are due to violence, nearly 70% of adults have had a close relative killed by violence, 44% of men have murdered. 61. Fun folks. The Yanomamo are renowned because of Napoleon Chagnon, one of the most famous and controversial anthropologists, a tough, pugnacious, no-holds-barred academic brawler who first studied them in the 1960s. He established the Yanomamo's street cred with his 1967 monograph Yanomamo, The Fierce People, an Anthropology Classic. Thanks to his publications and his ethnographic films about Yanomamo violence, both their fierceness and his are well-known tropes in anthropology. A central concept in the next chapter is that evolution is about passing copies of your genes into the next generation. In 1988, Chagnon published the remarkable report that Yanomamo men who were killers had more wives and offspring than average, thus passing on more copies of their genes. This suggested that, if you excel at waging it, war can do wonders for your genetic legacy. Thus, among these non-state tribal cultures standing in for our prehistoric past, nearly all have histories of lethal warfare, some virtually non-stop, and those who excel at killing are more evolutionarily successful. Pretty grim. And numerous anthropologists object strenuously to every aspect of that picture. 6.2. Again with the cherry picking. In Pinker's analysis of violence among hunter-horticulturalists and other tribal groups, all but one of his examples come from the Amazon or the New Guinea highlands. Global surveys yield much lower rates of warfare and violence. Pinker had foreseen this criticism by playing the Keeley pacification of the past card, questioning those lower rates. In particular, he has leveled this charge against the anthropologists, whom he somewhat pejoratively calls anthropologists of peace, somewhat akin to believers in the Easter Bunny, who have reported on the remarkably non-violent semi-people of Malaysia. This produced a testy letter to science from this group that, in addition to saying that they are peace anthropologists, not anthropologists of peace, stated that they are objective scientists who study the semi without preconceived notions rather than a gaggle of hippies. They even felt obliged to declare that most of them are not pacifists. Pinker's response was, it is encouraging that anthropologists of peace now see their discipline as empirical rather than ideological, a welcome change from the days when many anthropologists signed manifestos on which their position on violence was correct and censured, shut down, or spread libelous rumors about colleagues who disagreed. Woof, accusing your academic adversaries of signing manifestos is like a sharp knee to the groin. 63. Other anthropologists have studied the Yanomamo, and no one else reports violence like Chagnon has. 64. Moreover, his report of increased reproductive success among more murderous Yanomamo has been demolished by the anthropologist Douglas Fry of the University of Alabama at Birmingham, who showed that Chagnon's finding is an artifact of poor data analysis. Chagnon compared the number of descendants of older men who had killed people in battle with those who had not, finding significantly more kids among the former. However, Chagnon did not control for age differences. The killers happened to be an average of more than a decade older than the non-killers, meaning more than a decade more time to accumulate descendants. More important, this was the wrong analysis to answer the question posed. The issue isn't the reproductive success of elderly men who had been killers in their youth. You need to consider the reproductive success of all killers, including the many who were themselves killed as young warriors, distinctly curtailing their reproductive success. Not doing so is like concluding that war is not lethal, based solely on studies of war veterans. Moreover, Chagnon's finding does not generalize. At least 3 studies of other cultures fail to find a violence/reproductive success link. For example, a study by Luke Glowacki and Richard Wrangham of Harvard examined a nomadic pastoralist tribe, the Nyangatom of southern Ethiopia. Like other pastoralists in their region, the Nyangatom regularly raid one another for cattle. 65. The authors found that frequent participation in large, open battle raiding did not predict increased lifelong reproductive success. Instead, such success was predicted by frequent participation in stealth raiding, where a small group furtively steals cows from the enemy at night. In other words, in this culture, being a warrior on roids does not predict amply passing on your genes. Being a low-down sneaky varmint of a cattle rustler does. These indigenous groups are not stand-ins for our prehistoric past. For one thing, many have obtained weapons more lethal than those of prehistory. A damning criticism of Chagnon is that he often traded axes, machetes, and shotguns to Yanomamo for their cooperation in his studies. For another, these groups often live in degraded habitats that increase resource competition, thanks to being increasingly hemmed in by the outside world. And outside contact can be catastrophic. Pinker cites research showing high rates of violence among the Amazonian Ake and Hiwi tribes. However, in examining the original reports, Fry found that all of the Ake and Hiwi deaths were due to killings by frontier ranchers intent on forcing them off their land. 66. This tells nothing about our prehistoric past. Both sides in these debates see much at stake. Near the end of his book, Keeley airs a pretty weird worry. The doctrines of the pacified past unequivocally imply that the only answer to the mighty scourge of war is a return to tribal conditions, and the destruction of all civilization. In other words, unless this tomfoolery of archaeologists pacifying the past stops, people will throw away their antibiotics and microwaves, do some scarification rituals, and switch to loincloths, and where will that leave us? Critics on the other side of these debates have deeper worries. For one thing, the false picture of, say, Amazonian tribes as ceaselessly violent has been used to justify stealing their land. According to Stephen Corey of Survival International, a human rights organization that advocates for indigenous tribal peoples, Pinker is promoting a fictitious, colonialist image of a backward brutal savage, which pushes the debate back over a century, and is still used to destroy tribes. 67. Amid these roiling debates, let's keep sight of what got us to this point. A behavior has occurred that is good, bad, or ambiguous. How have cultural factors stretching back to the origins of humans contributed to that behavior? And rustling cattle on a moonless night, or setting aside tending your cassava garden to raid your Amazonian neighbors, or building fortifications, or butchering every man, woman, and child in a village is irrelevant to that question. That's because all these study subjects are pastoralists, agriculturalists, or horticulturalists, lifestyles that emerged only in the last 10,000 to 14,000 years after the domestication of plants and animals. In the context of hominin history stretching back hundreds of thousands of years, being a camel herder or farmer is nearly as newfangled as being a lobbyist advocating for legal rights for robots. For most of history, humans have been hunter-gatherers, a whole different kettle of fish. War and hunter-gatherers, past and present. Roughly 95 to 99% of hominin history has been spent in small, nomadic bands that foraged for edible plants and hunted cooperatively. What is known about hunter-gatherer, for sanity's sake, henceforth violence? Given that prehistoric HGS didn't have lots of material possessions that have lasted tens of thousands of years, they haven't left much of an archaeological record. Insight into their minds and lifestyle comes from cave paintings dating back as much as 40,000 years. Though paintings from around the world show humans hunting, hardly any unambiguously depict inner human violence. The paleontological record is even sparser. To date, there has been discovered one site of an HG massacre dating back 10,000 years in northern Kenya. This will be discussed later. What to do with this void of information? One approach is comparative, inferring the nature of our distant ancestors by comparing them with extant non-human primates. Early versions of this approach were the writings of Konrad Lorenz and of Robert Ardrey, who argued in his 1966 bestseller, The Territorial Imperative, that human origins are rooted in violent territoriality. 68. The most influential modern incarnation comes from Richard Wrangham, particularly in his 1997 book with Dale Peterson, Demonic Males, Apes and the Origins of Human Violence. For Wrangham, chimps provide the clearest guide to the behavior of earliest humans, and the picture is a bloody one. He essentially leapfrogs HGS entirely, so we come back to the Yanomamo. Do they suggest to us that chimpanzee violence is linked to human war? Clearly they do. Wrangham summarizes his stance. The mysterious history before history, the blank slate of knowledge about ourselves before Jericho, has licensed our collective imagination and authorized the creation of primitive Edens for some, forgotten matriarchies for others. It is good to dream, but a sober, waking rationality suggests that if we start with ancestors like chimpanzees and end up with modern humans building walls and fighting platforms, the 5-million-year-long trail to our modern selves was lined, along its full stretch, by a male aggression that structured our ancestors' social lives and technology and minds. It's Hobbes all the way down, plus Keeley-esque contempt for pacification of the past dreamers. This view has been strongly criticized. We're neither chimps nor their descendants. They've been evolving at nearly the same pace as humans since our ancestral split. Wrangham picks and chooses in his cross-species linkages. For example, he argues that the human evolutionary legacy of violence is rooted not only in our close relationship to chimps but also in our nearly as close kinship with gorillas, who practice competitive infanticide. The problem is that, overall, gorillas display minimal aggression, something Wrangham ignores in linking human violence to gorillas. As the most significant species cherry-picking, Wrangham pretty much ignores bonobos with their far lower levels of violence than chimps, female social dominance and absence of hostile territoriality. Crucially, humans share as much of their genes with bonobos as with chimps, something unknown when Demonic Males was published, and, notably, Wrangham has since softened his views. For most in the field, most insights into the behavior of our HG ancestors come from study of contemporary HGS. Once, the world of humans consisted of nothing but HGS. Today the remnants of that world are in the few remaining pockets of peoples who live pure HG lives. These include the Hadza of northern Tanzania, Mbuti Pygmies in the Congo, Batwa in Rwanda, Gunwingu of the Australian outback, Andaman Islanders in India, Batak in the Philippines, Simeng in Malaysia, and various Inuit cultures in northern Canada. To start, it was once assumed that among HGS, women do the gathering while men supply most of the calories by hunting. In actuality, the majority of calories come from foraging. Men spend lots of time talking about how awesome they were in the last hunt and how much awesomer they'll be in the next. Among some Hadza, maternal grandmothers supply more calories to families than do the men, the hunter men. 69. The arc of human history is readily equated with an arc of progress, and key to the latter is the view that agriculture was the best thing humans ever invented. I'll rant about that later. A cornerstone of the agriculture lobby is the idea that primordial HGS were half-starved. In reality, HGS typically work fewer hours for their daily bread than do traditional farmers, and are longer-lived and healthier. In the words of anthropologist Marshall Salins, HGS were the original affluent society. Clockwise from top, Hadza, Mbuti, Andaman, Simeng. There are some demographic themes shared among contemporary HGS. 70. Dogma used to be that HG bands had fairly stable group membership, producing considerable in-group relatedness. More recent work suggests less relatedness than thought, reflecting fluid fusion fission groupings in nomadic HGS. The Hadza show one consequence of such fluidity, namely that particularly cooperative hunters find one another and work together. More on this in the next chapter. What about our best and worst behaviors in contemporary HGS? Up into the 1970s, the clear answer was that HGS are peaceful, cooperative, and egalitarian. Interband fluidity serves as a safety valve preventing individual violence, i.e., when people are at each other's throats, someone moves to another group, and nomadicism as a safety valve preventing intergroup violence, i.e., rather than warring with the neighboring band, just hunt in a different valley from them. The standard bearers for HG grooviness were the Kalahari Kung. 70 won the title of an early monograph about them. Elizabeth Marshall Thomas's 1959 The Harmless People says it all Kung are to the Unimamo as Joan Baez is to Sid Vicious and the Sex Pistols. Naturally, this picture of the Kung in particular and HGS in general was ripe for revisionism. This occurred when field studies were sufficiently long-term to document HGS killing one another as summarized in an influential 1978 publication by Carol Ember of Yale. 72. Basically, if you're observing a band of 30 people, it will take a long time to see that, on a per capita basis, they have murder rates approximating Detroit's, the standard comparison always made. Admitting that HGS were violent was seen as a purging of 60s anthropological romanticism, a bracing slap in the face for anthropologists who had jettisoned objectivity in order to dance with wolves. By the time of Pinker's synthesis, HG violence was established, and the percentage of their deaths attributed to warfare averaged around 15%, way more than in modern Western societies. Contemporary HG violence constitutes a big vote for the Hobbesian view of warfare and violence permeating all of human history. Kalahari Kung hunter-gatherers. Time for the criticisms. 7.3. Mislabeling. Some HGS cited by Pinker, Keeley, and Bowles are, in fact, hunter-horticulturalists. Many instances of supposed HG warfare on closer inspection were actually singular homicides. Some violent Great Plains HG cultures were untraditional in the sense of using something crucial that didn't exist in the Pleistocene, domesticated horses ridden into battle. Like non-Western agriculturalists or pastoralists, contemporary HG are not equivalent to our ancestors. Weapons invented in the last 10,000 years have been introduced through trade. Most HG cultures have spent millennia being displaced by agriculturalists and pastoralists, pushed into ever tougher, resource-sparse ecosystems. Once again, the cherry-picking issue, i.e., failure to cite cases of peaceful HGS. Most crucially, there's more than one type of HG. Nomadic HGS are the original brand, stretching back hundreds of thousands of years. 74. But in addition to HG 2.0 equestrians, there are complex HGS who are different, violent, not particularly egalitarian, and sedentary, typically because they're sitting on a rich food source that they defend from outsiders. In other words, a transitional form from pure HGS. And many of the cultures cited by Ember, Keeley, and Pinker are complex HGS. This difference is relevant to Naidarik, that northern Kenyan site of a 10,000-year-old massacre, skeletons of 27 unburied people, killed by clubbing, stabbing, or stone projectiles. The victims were sedentary HGS, living alongside a shallow bay on Lake Turkana, prime beachfront property with easy fishing and plentiful game animals coming to the water to drink. Just the sort of real estate that someone else would try to muscle in on. The most thoughtful and insightful analyses of HG violence come from Fry and from Christopher Baim of the University of Southern California. They paint a complex picture. D.P. Fry and P. Soderberg, Lethal Aggression in Mobile Forager Bands and Implications for the Origins of War, Sci 341 2013, 270. Visit bit.ly/2eg96t for a larger version of this graph. Fry has provided what I consider the cleanest assessment of warfare in such cultures. In a notable 2013 science paper, he and Finnish anthropologist Patrik Soderberg reviewed all cases of lethal violence in the ethnographic literature in pure nomadic HGS, i.e., well studied before extensive contact with outsiders and living in a stable ecosystem. The sample consisted of 21 such groups from around the world. Fry and Soderberg observed what might be called warfare, defined by the fairly unstringent criterion of conflict producing multiple casualties in only a minority of the cultures. Not exactly widespread. This is probably the best approximation we'll ever get about warfare in our HG ancestors. Nonetheless, these pure HGS are no tidy pacifists. 86% of the cultures experienced lethal violence. What are their causes? In his 2012 book Moral Origins, The Evolution of Virtue, Altruism, and Shame, Baim also surveys the literature using slightly less stringent criteria than Fry uses, producing a list of about 50 relatively pure nomadic HE cultures heavily skewed toward Inuit groups from the Arctic. 0.75. As expected, violence is mostly committed by men. Most common is killing related to women, two men fighting over a particular woman, or attempts to kidnap a woman from a neighboring group. Naturally, there are men killing their wives, usually over accusations of adultery. There's female infanticide and killing arising from accusations of witchcraft. There are occasional killings over garden variety stealing of food or refusals to share food. And lots of revenge killings by relatives of someone killed. Both Fry and Baim report killings akin to capital punishment for severe norm violations. What norms do nomadic HDS value most? Fairness, indirect reciprocity, and avoidance of despotism. Fairness. As noted, HDS pioneered human cooperative hunting and sharing among non-relatives. 0.76. This is most striking with meat. It's typically shared by successful hunters with unsuccessful ones, and their families. Individuals playing dominant roles in hunts don't necessarily get much more meat than everyone else. Crucially, the most successful hunter rarely decides how the meat is divided. Instead, this is typically done by a third party. There are fascinating hints about the antiquity of this. Big game hunting by hominids, 400,000 years ago, has been documented. Bones from animals butchered, then show cut marks that are chaotic, overlapping at different angles, suggesting a free-for-all. But by 200,000 years ago, the contemporary HE pattern is there. Cut marks are evenly spaced and parallel, suggesting that single individuals butchered and dispensed the meat. This does not mean, though, that sharing is effortless for pure HES. Bain notes how, for example, the Kung perpetually kvetch about being shortchanged on meat. It's the background hum of social regulation. Indirect reciprocity. The next chapter discusses reciprocal altruism between pairs of individuals. Baim emphasizes how nomadic HDS specialize instead in indirect reciprocity. Person is altruistic to be social obligation now isn't necessarily as much being altruistic to as paying the altruism forward to C pays it forward to D, etc. This stabilizing cooperation is ideal for big game hunters, where two rules hold: A, your hunts are usually unsuccessful, and B, when they are successful, you typically have more meat than your family can consume, so you might as well share it around. As has been said, an A's best investment against future hunger is to put meat in other people's stomachs now. Avoidance of despotism. As also covered in the next chapter, there's considerable evolutionary pressure for detecting cheating when someone reneges on their half of a reciprocal relationship. For nomadic HDS, policing covert cheating is less of a concern than overt evidence of intimidation and powermongering. HDS are constantly on guard against bullies throwing their weight around. HD societies expend lots of collective effort on enforcing fairness, indirect reciprocity, and avoidance of despotism. This is accomplished with that terrific norm enforcement mechanism, gossip. HDS gossip endlessly, and as studied by Polly Wiesner of the University of Utah, it's mostly about the usual norm violation by high-status individuals. 0.77. People magazine around the campfire, gossiping serves numerous purposes. It helps for reality testing. Is it just me, or was he being a total jerk? Passing news to guesses who just happened to get a foot cramp during the hairiest part of the hunt today, and building consensus something needs to be done about this guy. Gossip is the weapon of norm enforcement. HD cultures take similar actions, collectively subjecting miscreants to criticism, shaming and mockery, ostracizing and shunning, refusing to share meat, non-lethal physical punishment, expulsion from the group, or as a last resort, killing the person, done either by the whole group or by a designated executioner. Bain documents such judicial killings in nearly half the pure HD cultures. What transgressions merit them? Murder, attempts at grabbing power, use of malicious sorcery, stealing, refusal to share, betrayal of the group to outsiders, and of course breaking of sexual taboos. All typically punished this way after other interventions have failed repeatedly. So, Hobbes or Rousseau? Well, a mixture of the two, I say unhelpfully. This lengthy section makes clear that you have to make some careful distinctions. A. HGS versus other traditional ways of making a living. B. Nomadic HGS versus sedentary ones. C. Data sets that canvass an entire literature versus those that concentrate on extreme examples. D. Members of traditional societies, killing one another versus members being killed by gun-toting, land-grabbing outsiders. E. Chimps as our cousins versus chimps erroneously viewed as our ancestors. F. Chimps as our closest ancestors versus chimps and bonobos as our closest ancestors. G. Warfare versus homicide, where lots of the former can decrease the latter in the name of in-group cooperation. H. Contemporary HGS living in stable, resource-filled habitats with minimal interactions with the outside world versus contemporary HGS pushed into marginal habitats and interacting with non-HGS. Once you've done that, I think a pretty clear answer emerges. The HGS who peopled Earth for hundreds of thousands of years were probably no angels, being perfectly capable of murder. However, war, both in the sense that haunts our modern world and in the stripped-down sense that haunted our ancestors, seems to have been rare until most humans abandoned the nomadic HD lifestyle. Our history as a species has not been soaked in escalated conflict. And ironically, Keeley tacitly concludes the same. He estimates that 90 to 95% of societies engage in war. And whom does he note as the exceptions? Nomadic HDS. Which brings us to agriculture. I won't pull any punches. I think that its invention was one of the all-time human blunders, up there with, say, the New Coke debacle and the Edsel. Agriculture makes people dependent on a few domesticated crops and animals instead of hundreds of wild food sources, creating vulnerability to droughts and blights and zoonotic diseases. Agriculture makes for sedentary living, leading humans to do something that no primate with a concern for hygiene and public health would ever do, namely living in close proximity to their feces. Agriculture makes for surplus and thus almost inevitably, the unequal distribution of surplus, generating socioeconomic status differences that dwarf anything that other primates cook up with their hierarchies. And from there it's just a hop, skip, and a jump until we've got Mr. McGregor persecuting Peter Rabbit and people incessantly singing Oklahoma. Maybe this is a bit over the top. Nonetheless, I do think it is reasonably clear that it wasn't until humans began the massive transformation of life that came from domesticating teosinte and wild tubers, aurochs and einkorn, and of course wolves, that it became possible to let loose the dogs of war. Some conclusions. The first half of the chapter explored where we are, the second, how we most likely got here. Where we are is awash in cultural variation. From our biological perspective, the most fascinating point is how brains shape cultures, which shape brains, which shape. That's why it's called coevolution. We've seen some evidence of coevolution in the technical sense, where there are significant differences between different cultures in the distribution of gene variants pertinent to behavior. But those influences are pretty small. Instead, what is most consequential is childhood, the time when cultures inculcate individuals into further propagating their culture. In that regard, probably the most important fact about genetics and culture is the delayed maturation of the frontal cortex, the genetic programming for the young frontal cortex to be freer from genes than other brain regions, to be sculpted instead by environment to sop up cultural norms. To hark back to a theme from the first pages of this book, it doesn't take a particularly fancy brain to learn how to motorically, say, throw a punch. But it takes a fancy, environmentally malleable frontal cortex to learn culture-specific rules about when it's okay to throw punches. In another theme from the first half, cultural differences manifest themselves in monumentally important, expected ways, say, whom it is okay to kill, an enemy soldier, a cheating spouse, a newborn of the wrong sex, an elderly parent too old to hunt, a teenage daughter who is absorbing the culture around her rather than the culture, her parents departed. But the manifestations can occur in unlikely places, e.g., where your eyes look within milliseconds of seeing a picture, or whether thinking of a rabbit prompts you to think of other animals, or of what rabbits eat. Another key theme is the paradoxical influence of ecology. Ecosystems majorly shape culture, but then that culture can be exported and persist in radically different places for millennia. Stated most straightforwardly, most of Earth's humans have inherited their beliefs about the nature of birth and death and everything in between, and thereafter from proliterate Middle Eastern pastoralists. The second half of the chapter, just concluded, addresses the key issue of how we got here. Has it been hundreds of thousands of years of Hobbes or of Rousseau? Your answer to that question greatly shapes what you'll make of something we'll consider in the final chapter, namely that over the last half millennium people have arguably gotten a lot less awful to one another. A. 10. The evolution of behavior. Last we reached the foundations. Genes and promoters evolve, as do transcription factors, transpososes, and splicing enzymes. As has every trait touched by genetic influences, i.e., everything. In the words of the geneticist Theodosius Dobzhansky, nothing in biology makes sense except in the light of evolution. Including this book. Point 1. E. Evolution 101. Evolution rests on 3 steps. A. Certain biological traits are inherited by genetic means. B. Mutations and gene recombination produce variation in those traits. C. Some of those variants confer more fitness than others. Given those conditions, over time the frequency of more fit gene variants increases in a population. We start by trashing some common misconceptions. First, that evolution favors survival of the fittest. Instead, evolution is about reproduction, passing on copies of genes. An organism living centuries but not reproducing is evolutionarily invisible. The difference between survival and reproduction is shown with antagonistic pleiotropy, referring to traits that increase reproductive fitness early in life, yet decrease lifespan. For example, primates' prostates have high metabolic rates, enhancing sperm motility. Upside, enhanced fertility; downside, increased risk of prostate cancer. Antagonistic pleiotropy occurs dramatically in salmon, who epically journey to their spawning grounds to reproduce and then die. If evolution were about survival rather than passing on copies of genes, there'd be no antagonistic pleiotropy. Point 2. Another misconception is that evolution can select for pre-adaptations, neutral traits that prove useful in the future. This doesn't happen. Selection is for traits pertinent to the present. Related to this is the misconception that living species are somehow better adapted than extinct species. Instead, the latter were just as well adapted until environmental conditions changed sufficiently to do them in. The same awaits us. Finally, there's the misconception that evolution directionally selects for greater complexity. Yes, if once there were only single-celled organisms, and there are multicellular ones now, average complexity has increased. Nonetheless, evolution doesn't necessarily select for greater complexity. Just consider bacteria decimating humans with some plague. The final misconception is that evolution is just a theory. I will boldly assume that readers who have gotten this far believe in evolution. Opponents inevitably bring up that irritating canard that evolution is unproven, because, following an unuseful convention in the field, it is a theory like, say, germ theory. Evidence for the reality of evolution includes numerous examples where changing selective pressures have changed gene frequencies in populations within generations, e.g., bacteria evolving antibiotic resistance. Moreover, there are also examples, mostly insects, given their short generation times, of a species in the process of splitting into two. Voluminous fossil evidence of intermediate forms in numerous taxonomic lineages. Molecular evidence. We share approximately 98% of our genes with the other apes, approximately 96% with monkeys, approximately 75% with dogs, approximately 20% with fruit flies. This indicates that our last common ancestor with other apes lived more recently than our last common ancestor with monkeys, and so on. Geographic evidence. To use Richard Dawkins's suggestion for dealing with a fundamentalist insisting that all species emerged in their current forms from Noah's Ark, how come all 37 species of lemurs that made landfall on Mount Ararat in the Armenian highlands hiked over to Madagascar, none dying and leaving fossils in transit? Unintelligent design, oddities explained only by evolution. Why do whales and dolphins have vestigial leg bones? Because they descend from a four-legged terrestrial mammal. Why should we have erector pili muscles in our skin that produce thoroughly useless goose flesh? Because of our recent speciation from other apes whose erector pili muscles were attached to hair, and whose hair stands up during emotional arousal. Enough. Don't get me started. Evolution sculpts the traits of an organism in two broad ways. Sexual selection selects for traits that attract members of the opposite sex. Natural selection, for traits that enhance the passing on of copies of genes through any other route, e.g., good health, foraging skills, predator avoidance. The two processes can work in opposition. Point 3. For example, among wild sheep one gene influences the size of horns in males. One variant produces large horns, improving social dominance, a plus for sexual selection. The other produces small horns, which are metabolically cheaper, allowing males to live and mate, albeit at low rates, longer. Which wins, transient but major reproductive success, or persistent but minor success? An intermediate form, or consider male peacocks paying a price in terms of natural selection for their garish plumage. It costs a fortune metabolically to grow, restricts mobility, and is conspicuous to predators. But it sure boosts fitness via sexual selection. Importantly, neither type of selection necessarily selects for the most adaptive version of a trait, which replaces all others. There can be frequency-dependent selection, where the rarer version of two traits is preferable, or balanced selection, where multiple versions of traits are maintained in equilibrium. Oh. Behavior can be shaped by evolution. Organisms are amazingly well adapted. A desert rodent has kidneys that excel at retaining water, a giraffe's huge heart can pump blood up to its brain, elephant's leg bones are strong enough to support an elephant. Well, yes, it has to work that way. Desert rodents whose kidneys weren't great at retaining water didn't pass on copies of their genes. Thus there is a logic to evolution, where natural selection sculpts traits into adaptiveness. Importantly, natural selection works not only on anatomy and physiology but on behavior as well. In other words, behavior evolves, can be optimized by selection into being adaptive. Various branches of biology focus on the evolution of behavior. Probably best known is sociobiology, premised on social behavior, being sculpted by evolution to be optimized, just as biomechanical optimization sculpts the size of a giraffe's heart. 4. Sociobiology emerged in the 1970s, eventually generating the offshoot evolutionary psychology, the study of the evolutionary optimization of psychological traits. As we'll see, both have been plenty controversial. As a simplifying convenience, I'll refer to people who study the evolution of social behavior as sociobiologists. W. The demise of group selection. Start by grappling with an entrenched misconception about the evolution of behavior. This is because Americans were taught about the subject in the 1960s by Marlin Perkins on the TV program Mutual of Omaha's Wild Kingdom. It was great. Perkins would host. Jim, his sidekick, did dangerous things with snakes. And there were always seamless segues from the program to ads from Mutual of Omaha. Just as lions mate for hours, you'll want fire insurance for your home. Unfortunately, Perkins espoused wildly wrong evolutionary thinking. Here's how it looked on the program. It's dawn on the savannah. There's a herd of wildebeest on a river's edge. The grass is greener on the other side, and everyone wants some, but the river teems with predatory crocodiles. The wildebeest are hemming and hawing in agitation, when suddenly an elderly wildebeest pushes to the front, says, "I sacrifice myself for you, my children," and leaps in. And while the crocs are busy with him, the other wildebeest cross the river. Why would the old wildebeest do that? Marlin Perkins would answer with patrician authority, because animals behave for the good of the species. Yes, behavior evolves by group selection, for the good of the species. This idea was championed in the early 1960s by V.C. Wynne Edwards, whose wrongness made him modern evolutionary biology's Lamarck 5. Animals don't behave for the good of the species. But what about that wildebeest? Look closely and you'll see what really happens. Why did he wind up saving the day? Because he was old and weak. Good of the species, my caster. They pushed the old guy in. Group selection was done in by theoretical and empirical studies showing patterns of behavior incompatible with it. Key work was done by two gods of evolutionary biology, George Williams of SUNY Stony Brook and Oxford's Bill W. D. Hamilton. 6. Consider eusocial insects, where most individuals are non-reproductive workers. Why forgo reproduction to aid the queen? Group selection, obviously. Hamilton showed that eusocial insects' unique genetic system makes a colony of ants, bees, or termites a single superorganism. Asking why worker ants forgo reproduction is like asking why your nose cells forgo reproduction. In other words, eusocial insects constitute a unique type of group. Williams then elaborated on how the more standard genetic system in species from non-eusocial insects to us was incompatible with group selection. Animals don't behave for the good of the species. They behave to maximize the number of copies of their genes passed into the next generation. This is the cornerstone of sociobiology and was summarized in Dawkins's famed soundbite that evolution is about selfish genes. Time to see its building blocks. P. Individual selection. Passing on lots of copies of one's genes is accomplished most directly by maximizing reproduction. This is summarized by the aphorism, a chicken is an egg's way of making another egg. Behavior is just an epiphenomenon, a means of getting copies of genes into the next generation. Individual selection fares better than group selection in explaining basic behaviors. A hyena bears down on some zebras. What would the nearest one do if she's a group selectionist? Stand there, sacrificing herself for the group. In contrast, an individual selectionist zebra would run like hell. Zebras run like hell. Or consider hyenas that have just killed a zebra. Group selection mindset, everyone calmly takes turns eating. Individual selection, frenzied free-for-all. Which is what occurs. But wait, says the group selectionist, wouldn't the zebra species benefit if it is the fastest animals who survive and pass on those fast-running genes? Ditto for the group benefits of the fiercest hyena getting the most food. As more nuances of behavior are observed, clinging to group selection requires increasingly tortuous arguments. But one single observation devastates group selection. In 1977, the Harvard primatologist, Sarah Blaffer-Hurry, documented something remarkable. Langur monkeys in the Mount Abu region of India kill one another. 7. People already knew that some male primates kill one another, fighting for dominance. Okay, makes sense, boys will be boys. But that's not what Hurry reported. Male langurs were killing infants. Once people believed her careful documentation, there was an easy answer: since babies are cute and inhibit aggression, something pathological must be happening. 8. Maybe the Abu langur population density was too high and everyone was starving, or male aggression was overflowing, or infanticidal males were zombies. Something certifiably abnormal. Hurry eliminated these explanations and showed a telling pattern to the infanticide. Female langurs live in groups with a single resident breeding male. Elsewhere are all male groups that intermittently drive out the resident male. After infighting, one male then drives out the rest. Here's his new domain, consisting of females with the babies of the previous male. And crucially, the average tenure of a breeding male, about 27 months, is shorter than the average interbirth interval. No females are ovulating, because they're nursing infants, thus this new stud will be booted out himself before any females wean their kids and resume ovulating. All for nothing, none of his genes passed on. What, logically, should he do? Kill the infants. This decreases the reproductive success of the previous male and, thanks to the females ceasing to nurse, they start ovulating. That's the male perspective. What about the females? They're also into maximizing copies of genes passed on. They fight the new male, protecting their infants. Females have also evolved the strategy of going into pseudoestrus, falsely appearing to be in heat. They mate with the male. And since males know squat about female langur biology, they fall for it. Hey, I mated with her this morning and now, she's got an infant, I am one major stud. They'll often cease their infanticidal attacks. Despite initial skepticism, competitive infanticide has been documented in similar circumstances in 119 species, including lions, hippos, and chimps. 9. A variant occurs in hamsters because males are nomadic. Any infant a male encounters is unlikely to be his, and thus he attempts to kill it. Remember that rule about never putting a pet male hamster in a cage with babies. Another version occurs among wild horses and gelato baboons. A new male harasses pregnant females into miscarrying. Or suppose you're a pregnant mouse and a new, infanticidal male has arrived. Once you give birth, your infants will be killed, wasting all the energy of pregnancy. Logical response. Cut your losses with the Bruce effect, where pregnant females miscarry if they smell a new male. 10. Thus competitive infanticide occurs in numerous species, including among female chimps, who sometimes kill infants of unrelated females. 11. None of this makes sense outside of gene-based individual selection. Individual selection is shown with heartbreaking clarity by mountain gorillas, my favorite primate. 12. They're highly endangered, hanging on in pockets of high-altitude rainforest on the borders of Uganda, Rwanda, and the Democratic Republic of the Congo. There are only about 1,000 gorillas left, because of habitat degradation, disease caught from nearby humans, poaching, and spasms of warfare rolling across those borders. And also because mountain gorillas practice competitive infanticide. Logical for an individual intent on maximizing the copies of his genes in the next generation, but simultaneously pushing these wondrous animals toward extinction. This isn't behaving for the good of the species. T. Kin selection. Understand the next foundational concept, reflect on what it means to be related to someone and to pass on copies of your genes. Suppose you have an identical twin with the same genome as you. As a startling, irrefutable fact, in terms of the genes being passed on to the next generation, it doesn't matter if you reproduce or sacrifice yourself so that your twin reproduces. What about a full sibling who isn't an identical twin? Recall from Chapter 8 that you'd share 50% of your genes with him, thus reproducing once and dying so that he reproduces twice are evolutionarily identical. Half-sibling, 25% of genes in common, calculate accordingly. The geneticist J.B.S. Haldane, who when asked if he'd sacrifice his life for a brother is credited to have quipped, "I'll gladly lay down my life for 2 brothers or 8 cousins." You can leave copies of your genes in the next generation by reproducing, but also by helping relatives reproduce, especially closer relatives. Hamilton formalized this with an equation factoring in the costs and benefits of helping someone, weighted by their degree of relatedness to you. This is the essence of kin selection. This explains the crucial fact that in countless species, whom you cooperate with, compete with, or mate with depends on their degree of relatedness to you. Mammals first encounter kin selection soon after birth, reflecting something monumentally obvious: females rarely nurse someone else's infants. Next, among numerous primates, the mother of a newborn and an adolescent female may commence a relationship fraught with pluses and minuses. The mother occasionally lets the adolescent care for her offspring. For the mother, the plus is getting time to forage without baby on board. The minus is that the babysitter may be incompetent. For the adolescent, the plus is getting mothering experience, the minus, the effort of child care. Lynn Fairbanks of UCLA has quantified the pluses and minuses of such allomothering, including that adolescents who practiced mothering have a better survival rate for their own kids. And who is a frequent allomother? The female's kid sister. 13. An extension of allomothering is the cooperative breeding of New World monkeys like marmosets. In their social groups, only one female breeds, while the others, typically younger relatives, help with child care. 14. The extent to which a male primate cares for infants reflects his certainty of paternity. 15. Among marmosets, who form stable pair bonds, males do most of the child care. In contrast, among baboons, where a female mates with multiple males during her estrus cycle, it's only the likely fathers, i.e., males who mated on the female's most fertile day when she had her most conspicuous estrus swelling, who invest in the well-being of the child, aiding him in a fight. Among many primates, how often you groom someone depends on how closely related they are to you. Among baboons, females spend their whole life in their natal troop, whereas males migrate to a new troop at puberty. As a result, adult females have complex, cooperative kinship relations and inherit their dominance rank from their mother. Among chimps it's the opposite: females leave home at puberty, and kin-based adult cooperation occurs only among males, for example, where groups of related males attack solitary males from neighboring groups. And among langurs, when a female defends her infant against a new male, she most often is helped by elderly female relatives. Moreover, primates understand kinship. Dorothy Cheney and Robert Sapolsky of the University of Pennsylvania, studying wild vervet monkeys, have shown that if animal A is crummy to animal B afterward, B is more likely to be crummy to A's relatives. And if A is lousy to B, B's relatives are more likely to be crummy to A. Furthermore, if A is lousy to B, B's relatives are more likely to be crummy to A's relatives. 16. In beautiful playback experiments, Cheney and Sapolsky first recorded vocalizations from each vervet in a group. They'd place a speaker in some bushes, and when everyone was sitting around, they'd play a recording of some kid giving a distress call. And the females would all look at the kid's mother. "Hey, that's Madge's kid. What's she going to do?" Note that this also shows that monkeys recognize voices.